Welcome to AP Biology 7.1, Introduction to Natural Selection. I'm Gail Pfizer from Darnell Cookman, Jacksonville, Florida.
Hi, my name is Allison Kate. I teach AP Biology in California. Today, Ms. Pfizer and I will both be teaching parts of this lesson, but going forward, we will trade off with Ms. Pfizer teaching some lessons and me teaching others. Before Ms. Pfizer gives you the lesson overview for today, I wanted to provide you with the reasoning behind our approach to the online lesson planning. Today, the topic we are covering is one that colleges have asked all AP students to study, even if it will not be on the exam. Colleges are supporting AP students this year by allowing this more flexible approach to the exam, where it will focus on a lesser amount of content than in other years. But in turn, they have asked that AP students learn today's material. To make this relevant to your upcoming exam, we will also include some exam prep in today's lesson. For all lessons, it will be helpful to have pen and paper nearby, and for some of our lessons, we will request that you also have a calculator handy. In a week or so, after we've covered some of the key outstanding topics, we will be posting a full schedule of review topics for the remainder of this course, so that you can choose which topics you want to prioritize for review and thus which classes you want to attend. We hope that you will attend all of the AP Biology lessons.
And now for a lesson overview. Today, we will be introducing you to the principles of natural selection and an overview of Science Practice Skill 2A, visual representation. You will gain proficiency in describing a given model or representation and communicating the biological meaning it represents. You should already be familiar with genetic variation and understand that genetic variation is necessary for natural selection and using visual models and representations to explain or illustrate biological process. By the end of this lesson, you will also be able to describe the causes of natural selection and explain how natural selection affects population. And now we'll have a warm-up. Okay, so in the future, the warm-up will be the homework from the previous lesson, but tonight we're going to start off with a skill 2A, the same skill that we'll be using tonight, but we're going to take it back to when it applied to cells. So I'm going to tell you how I teach my kids and how I go through either a multiple choice question and FRQ. So what I first tell them is to decide or to figure out what the diagram, graph, or table is telling them. It's going to tell them a story. It's going to help them answer questions. So in this particular diagram, I would see three cells. There's a cell at 5 minutes, at 15 minutes, and at 30 minutes. And I can see that it has a nucleus, an endoplasmic reticulum, it has a Golgi apparatus, secretory vesicles in each. And I can also see that these black dots are moving through this cell. And those black dots, I can see it's labeled figure 1, radioactively labeled polypeptide. So I can determine that there are black dots moving from the endoplasmic reticulum to, and if you look in the second cell at the 15 minutes, you can see one little tiny vesicle has a dark dot. So they're moving through the vesicle to the Golgi apparatus. And then the dark dots are leaving the Golgi apparatus and to another vesicle, and that vesicle is fusing with that plasma membrane. And then the polypeptides are being released outside the cell. So I ask them to go through and get the story of what the diagram is telling them. And then I ask them to read the question so they'll know what they're going to be trying to answer. So the question is using the experimental results, describe the pathway that secretory proteins take from their synthesis to their release from the cell. And then I ask them to read the passage if there's anything they think they still need after looking at the diagram. In an experiment investigating the mechanism of protein secretion, researchers track the movement of radioactively labeled polypeptides in pancreatic cells. At various times after the labeling, samples of the cells were observed using an electron microscope to determine the location of the radioactively labeled polypeptide. Figure 1 summarizes results at 5 minutes, 15 minutes, and 30 minutes. The dark dots in the figure represent the radioactively labeled polypeptide. So I'm going to give you a minute to, on your pen and paper, with your pen and paper, write the answer to this FRQ question. Using experimental results, describe the pathway that secretory proteins take from their synthesis to their release from the cell. And I'll give you just a minute to do that on your own. Okay, I hope that was enough time. Let's see what the scoring guideline says. And then you compare your answer with the scoring guideline. So the scoring guideline, you will get 1 point. This is a 1 point because there's one task verb involved, and I'll talk about those in a little bit. So if you said that it went from the endoplasmic reticulum to the Golgi to the vesicle to exocytosis, fusion with the plasma membrane, and outside the cell, you would have scored 1 point. Now, if you'll notice, there are words that are written in parentheses. Those are optional. Those are not required. Like if you did not say rough ER or rough endoplasmic reticulum, you would have not lost that point as long as you said endoplasmic reticulum. And the same goes with that second stage, which was the vesicle between the ER and the Golgi. So I hope that you scored that point. Okay, so the framework of the AP Biology course. We've been working through different units this year: biomolecules, cells, photosynthesis, and we have broken those units into small topics. Each topic has questions called the learning objectives. This is what we need to be able to do. This is what you need to be able to do on the exam. And we need to know content in order to answer these questions. So the content is called essential knowledge. So you need to know the essential knowledge to be able to answer the learning objective. And then each topic also has associated science practice. And this is how we're going to show that we know the answer to the question, which is the learning objective. The science practice that is associated with natural selection is visual representation. You've probably been doing this in class since the beginning of the year. Okay, so Ms. Kate is now going to talk to you about what you need to know, the essential knowledge.
Hi, so I'm going to take you through the content to our topic today, which is an introduction to natural selection. And I thought a great place to start would be with the definition of natural selection. So I'm going to read it aloud, and I'd like for you guys to read it along with me. Natural selection is a process in which individuals that have certain inherited traits tend to survive and reproduce at higher rates than other individuals because of those traits. Now I'd like you to take a look at the graphics and notice that what we're going to be looking at is a prey-predator relationship, and there are 3 panels, and arrows moving from right to left are indicating time. So we're going to start at the first generation, and we see a population of mice has moved into a new area where the rocks are very dark. Due to natural genetic variation, some of the mice are black and others are tan. In the middle panel, we see that the tan mice are more visible to the predatory birds than the black mice. Thus, the tan mice are eaten at a higher frequency than the black mice. And since only surviving mice can reach reproductive age and leave offspring, in the final panel, we see that the black mice, because they had a higher chance of leaving offspring than the tan mice, the next generation contains a higher fraction of black mice than the previous generation. So just to take us back, this concept, this mechanism of how natural selection happens was first proposed by Charles Darwin in 1859. And if you look to the right of your screen, you're going to see what we affectionately call Darwin's finches. And when you look at these birds, you can see that these 4 birds all have very different beaks. They have different beak sizes and beak shapes. And so we're going to go through, using that as an example, the components that are needed for the mechanism of natural selection as proposed by Darwin. So the first thing is variation, and this exists naturally in populations, and I just went through the beak variations. We're going to talk more about how that variation gets introduced into populations in the next slide. The next component is reproduction, and the idea here is that more offspring will be produced than the environment can support. And that sets us up for the third component, which is competition. If there's more offspring than the environment can support, then there's competition for food, mates, nesting sites, and to be able to escape predators. The fourth component is the idea of fitness. In this case, fitness means that these individuals are more likely to survive and reproduce. So successful traits that certain individuals inherit mean that they are more likely to survive and reproduce. Finally, evolution occurs when these adaptations become more common in a population. So looking at the sources of variation in a population, the first source is mutations, which are random changes to DNA. The second source of variation in a population is due to sexual reproduction. When organisms reproduce sexually, there is a new arrangement of alleles in every offspring, and these new combinations of allele mean new phenotypes. And the selective process works on the phenotypes, the traits that are visible. So again, this mechanism of natural selection was originally proposed by Charles Darwin in 1859. So let's put it all together. Let's look at the components that are necessary for natural selection, keeping in mind what Darwin's observations were and applying it to this scenario. So let's look at the starting population. We're looking at, again, another prey-predator relationship, and we see that there's a snail snail species, and it lives on a brown sandy beach, and the snails are eaten by hungry birds that spot them from above. There's overproduction, differential reproduction, and you can see that there are more juvenile snails that are born that can survive on this beach. There's not enough food or space for all of them to live. Some of the snails are always spotted by the birds and eaten. And as you can see, there's natural variation in this population. Not all the snails are brown. Some of the snails are red, some of them are green, some of them are blue. And so all the snails on the beach are competing to stay hidden from the birds and not get eaten. Here's where that adaptation piece comes in. The brightly colored snails really stand out against that brown sandy beach, and that makes them easier to spot by the predators lying above. So the brown snails that match the color of their environment are more protected, making it easier for them to hide. The differential survival piece comes into play when, because of their color, the brown snails are better at avoiding the birds, and since they're eaten left, they can reproduce more. And in the final image, you can see that that brown sandy beach is mostly inhabited by brown snails. That means that they had a higher fitness and were able to survive and reproduce much better than the other variations in the population. And now what I'd like to do is introduce you to a real-life example of natural selection. We're going to see the exact same thing that we saw in our previous example, but this is a real-life example. I'm not sure if you're getting sound. I'm not hearing sound.
I'm not hearing sound either, but I can't. It's different. I didn't hear myself talk either.
Okay.
She can't hear it.
Okay,
In
so
Mexico, a battlefield to find one of the tiniest souls.
one moment, please.
there we go. Okay. Sorry, guys.
It's a volcanic eruption that occurred about 1,000 years ago. The eruption spewed a river of lava more than 40 miles long across the desert. As the molten rock cooled, it darkened, leaving any creature dependent on camouflage in serious trouble. In the complex battle of life, one of the constant struggles between seeing and not being seen, the evolutionary game of hide and seek. And we come here to the Valley of Fire in New Mexico, a battlefield, to find one of the tiniest soldiers and what it can teach us about how evolution works. On the desert sands, the rock pocket mouse blends in perfectly, its light-colored fur concealing it from predators. But on dark lava, the same fur makes the mouse stand out, attracting the many creatures that see it as food.
These mice are the Snickers bar of the desert. They're eaten by foxes and coyotes and rattlesnakes and certainly by owls and maybe even occasionally hawks. And most of those predators are visual predators.
So what happened to the pocket mice that found themselves on this new terrain? My accompanying biologist Michael knocked them onto the lava. It doesn't take long to find out.
Oh, this one's closed.
Knockman has been collecting mice unharmed in traps. And it's a dark one.
It is, yeah.
Now, are most of the ones you find up here dark?
Almost all.
Not only have the mice here evolved to be as dark as the rock, the color change has occurred precisely where it will conceal them from hunters.
Now, they have a white underbelly too. That's right. All of the dark ones here and a lot of those have a white underbelly. And presumably there's no selection for dark underbelly because predators are coming to stuff.
Left to themselves, the mice show no preference for light or dark rocks. It's the predators that have made the difference.
The change in color over evolutionary time in the population is driven by predators weeding out the mice that don't match their background.
But how did the dark mice arise in the first place?
When a black mouse appears in a light population of mice, that is usually going to be due to a new mutation.
Okay, so that is the video clip just to give you a real life example of how protective coloration can be a protection for predation in that mouse population. So what I'd like to do is take you through a check for understanding, and this is the type of multiple choice question that you might have seen on the exam. So let's take a look at these side-by-side graphs, and the next slide will have 4 multiple choice questions, and you will need to select the right answer. So when looking at the graphs, you want to make sure you look at the x-axis and see that it's showing you fur colors. You're going to look at the y-axis and it's showing you the frequency, and then in the right-hand graph, it's the same x and y axes, but it's the fur color distribution after 20 years compared to the one on the left, which is the fur color distribution after immediate release. So let's take a look at the questions. We'll have an opportunity to come back and look at the graph before you give your final answer. So I'm going to read you the question. I'd like you to read it along with me, and then also take a look at 4 answer choices. A group of mice was released into a large field to which no other mice had access. Immediately after the release, a representative sample of the mice was captured, and the fur color of each individual in the sample was observed and recorded. The mice were then returned to the field. After 20 years, another representative sample of the mice was captured, and the fur color of each individual in the sample was again recorded. Which of the following best explains the change in the frequency distribution of the fur color phenotypes in the mouse population as shown in the figures above? So let's go back and take a look at those figures, and then you can come back and choose your answer.
Okay, can you describe that to them?
Okay, so
if I go backwards, I'm going to mess things up today.
Okay, so we're unable to actually move backwards. So there were 3 color choices. There was gray, brown, and black. And in the first one, you can see that the gray bar was lower, and in this after 20 year, the gray bar was the highest. So hopefully that will help you with your answer choice. So let's read them. Choice A, the allele for gray fur color is unstable, and over 20 years, most of those alleles mutated to become alleles for black fur. Choice B, the field was composed primarily of light-colored soil and little vegetation, affording gray mice protection from predators. Choice C, sexual selection led to increased mating frequency of black and brown versus gray and brown. And choice D, the gray mice were the hardest to capture, and so were underrepresented in the 20-year sample. So I hope that you selected choice B, that because there was primarily light-colored soil, the gray mice had protection from predators. And one final thing, while we're on the topic of natural selection, all of the components that I've been talking to you about were first described by Charles Darwin. There was another scientist by the name of Lamarck who also had a view of the mechanism of natural selection, and his view was called the inheritance of acquired characteristics. He said that physical changes that occur during an organism's lifetime could be inherited by that organism's offspring. And the classic example is of giraffes and their long necks. So what Lamarck said was that as the giraffes stretched their necks during their lifetime to reach the leaves on tall trees, they would acquire those long necks. And when they had offspring, those offspring would be born with longer necks. While it's an interesting observation, it turns out that there's no data to support this. And I'll give you an example. So let's say you love Charles Darwin as I do, and you wanted to get a tattoo on your bicep. Then when you go to have children, your child will not be born with a tattoo of Charles Darwin on their arm. So that's the Lamarckian example.
Okay, on with the science skills. And today, visual representation and describe is the task verb that we're going to talk about. And describe characteristics of a biological concept, process, or model represented visually is the skill. And describe is a task verb. And we've used this particular one all throughout all the units of the curriculum, not just evolution. And when they're in a free response question, they're going to be bolded and they're going to be worth 1 point on the exam. So when you describe something, this is all you need to do. You just need to list the characteristics, patterns, or trends of a representation. That representation could be a diagram, it could be a graph, it could be a table. And a lot of people get describe and explain confused. So I'm going to give you an example of each. If you describe mitosis, you could say mitosis occurs in a sequential series of steps: prophase, metaphase, anaphase, and telophase. But if you were explaining mitosis, you could say something like this. You could say during mitosis, sister chromatids separate during anaphase, ensuring the transfer of the genome to genetically identical daughter cells. So explain is more like how or the why of something. Okay, and I'm going to take you through a little guided practice on using the skill. So again, the first thing that I suggest suggest you do, and I suggest to my kids, well, I kind of make them do it, but I suggest that you look at what the representation is telling you. And so let's look at the x-axis here. We have average big depth of parents in millimeters, and on the y we have percent of pairs successfully producing at least one offspring. So if I'm going to describe the trend illustrated, then all I would say is that I see a higher percent of pairs successfully producing at least one offspring at 5 millimeters. Okay, let me read the passage just to see if there's anything else we need to know. Scientists investigated the role that big depth plays in the ability of one species of seed-eating finch to reproduce. The scientists calculated the average big depth of finches in mating pair, and then observed whether or not the pairs produced at least one offspring that survived to the next season. The data are represented in figure 1. So what we need to answer is describe the trend illustrated. I'll give you just a moment because it kind of led you there for the most part. You should be able to write this down. Okay, so the answer, compare your answer, and hopefully you said something similar to this. Parental pairs with a specific depth of 5 had the highest reproductive success. Does yours look like that? I hope so. Okay, let's do another one as we're still under guided practice. I'll let you out on your own in just a little while. Okay, hemoglobin is the iron-containing protein used to transport oxygen in the blood. Since iron is important for growth, S. aureus have evolved the ability to absorb the iron from the host hemoglobin. Okay, now that I'm reading that, I need to look at this table first. Okay, so at the table, S. aureus strain, I see 4 strains, 1, 2, 3, 4, and I see an amino acid sequence. And you'll see some letters that you might not be familiar with representing amino acids. Like the Q is glutamine. You don't need to know that right now, but it can't be G because there's already another G. Anyway, host species, you have species A, species B, macaque, species C. And what we see in this table is that strain 1 prefers host A, and strain 2 prefers B, strain 3 prefers macaque, and strain 4 prefers species C. Okay, and what we're going to be looking for in the question, describe the process responsible for the different amino acid strains found on different hosts. So let's see if there's something in the passage that can help us answer this question. All right, different S. aureus strains preferentially affect different hosts and have different amino acid sequences at their hemoglobin binding domains, and that's in table 1, which I just talked about. In an experiment, different S. aureus strains were mixed with hemoglobin from macaque monkeys, and their binding ability was measured, and that's in figure 1. And I'm not showing you figure 1 yet, and I'll tell you why in just a few minutes. All right, the differences in amino acid sequences contributed to the differential binding abilities observed. So when you get an FRQ question, some of them have 3 or 4 different representations. They might have a diagram, they might have a table, a graph. They'll have all of them, maybe 2 or 3 graphs. But each, when you answer part A, B, C, or D, it doesn't mean that you need to refer to all of them. There may be something that's very specific to your task verb, and you only need to go to that one. So that's why I just showed you this particular table. All right, so describe the process responsible for the different amino acid strains found on different hosts. So I'll give you a minute to write your response to that describe. Okay, now I'm not going to give you the answer to A just yet because now we have B, describe the trend illustrated. So in this graph, this kind of looks like what we just did just a few minutes ago. So on our X, we have S. aureus strains, 1, 2, 3, 4, and on our Y, we have percent hemoglobin binding. And so I'm going to give you just a moment, transfer the skill that you just did on previous question to now this question. Okay, and I'll bring up the answers to both. Okay, so your answer A should be similar to each strain is best adapted to a specific host species, and then B, strain 3 has the highest percent hemoglobin binding. Pretty easy, I think. Describe is going to be one of those that you need not leave that point behind because all you need to do is talk about the characteristics of what you're seeing. Okay, so now for a debrief and summary of today. Hopefully we were successful with the learning objectives for today. You should be able to describe the causes of natural selection. You should be able to explain how natural selection affects population. The key takeaways: evolution is characterized by change in the genetic makeup of a population over time, and it's supported by multiple lines of evidence. Natural selection is the mechanism of evolution. It was proposed by Charles Darwin in 1859. There are many examples of natural selection, and only some were presented here in this presentation. To describe, you should list the characteristics, patterns, or trends of a representation. Now I want to talk about common errors and misunderstandings. In fact, there's a lot of them I'm going to talk about in the next several minutes. Hopefully we'll learn something along the way, and then we'll get to the misunderstanding at the end of this conversation. So this was a question of the 2015 read. It was the first exam. It was my first year as a reader, and I had question number 1, and this was it. And it consumes all of us by the end of the week. So, and I'll tell you why. Anyway, so let's look at our diagram. We have a mouse on the left that, well, we have 2 running wheels, and we have a mouse that's inactive and a mouse that's active. And then if we look underneath those mice, we're going to see what's called an actogram. And an actogram has the days on the y-axis, and then it has the time and hours on the x. Now, if you've never seen an actogram, my kids have never seen an actogram before this day either. And you're going to do that. There's going to be an exam where there is a new type of graph that you've never seen before, a new type of diagram, something you've never seen before, and your job is to interpret it. And once you have that skill set, you can transfer it from one diagram, table, or graph to another. So we can see that in the morning hours from 0 to 12, there is blank. And then in the evening hours from 12 to 24, there is dark bars. So we just look at that mouse. He's running on that running wheel, and it makes the dark black line. So we can see that the actogram has a black line with activity. All right, and that was at the evening hours. So that's what we should have pulled together from interpreting that diagram. All right, so let me read you part of the question. Many species have circadian rhythms that exhibit an approximately 24-hour cycle. Circadian rhythms are controlled by both genetics and environmental conditions, including light. Researchers investigated the effect of light on mouse behavior by using a running wheel with a motion sensor to record activity on actogram as shown in figure 1. Okay, here's another part. So here we have another image, and we have another actogram, and we can see that there's some variables there's a variable being introduced in this particular scenario eventually. Okay, so now we added time. We went over for 2 weeks. We have 14 days on our y-axis. We still have a 24-hour period, and we have most of our dark lines, our bold dark lines, are in the evening hours. So we can see our mouse is still running and being active in the dark. All right, so for the investigation, adult male mice were individually housed in cages soundproof at 25 degrees Celsius. Each mouse was provided with adequate food, water, bedding material, and a running wheel. The mice were exposed to daily periods of 12 hours of light, and that will be represented with an uppercase L, and 12 hours of dark, which will be represented with an uppercase D. So L12, D12 means 12 hours in the light, 12 hours in the day. And they did this for 14 days, and they monitored their activity for those 14 days. Okay, now we have another actogram, and so now this one has extended to 30 days, and we can see that there's a dark bar across. So it looks like there's going to be continuous darkness, and there's something cyclical maybe going on, but we can say that there is some activity going on in the morning hour and in the evening hour. Okay, after 14 days in L12, D12, the mice were placed in continuous dark DD, and their activity on the running wheel was recorded at 4. The activity data under DD conditions are shown in figure 3. Okay, so let's take a moment to think about what we just saw. The mice are active when it's dark and not when it's light. Okay, in nature, mice are potential prey for some predatory birds that hunt during the day. Describe 2 features of a model that represent how the predator-prey relationship between the birds and the mice may have resulted in the evolution of the observed activity pattern of the mice. So make sure you answer what the question's asking you. So you're going to describe it, which means list characteristics of this representation, and it wants 2 features of it. So there's going to be 2 parts. I don't want to tell you what they are until you think about it yourself. But there's a predator-prey relationship. We have that, and what that has to do with how this activity pattern has evolved. So we have a bird eating a mouse, and we have birds eating mice, and then we have activity during the dark. So I want you to take a few minutes and write 2 features, describe 2 features of what would have happened along the way to make this happen. Okay. Okay, let's see what the scoring guidelines. Okay, so here's the scoring guidelines, and there are the 2 features that they wanted you to talk about were the selected advantage and the reproductive success. So if you're a mouse, or if you're a population of mice, and you are awake and are active during the day, the birds are going to eat you. Okay, so those mice active during the day will be selected against. If you are a population of mice that are active at night, then the birds will not be able to see you, and so you'll be selected for if you are active at night. So that's a selected advantage. So if you are dead during the light hours, you're not reproducing. But if you are living because you have a selected advantage in the nighttime hour, then you are the ones that are going to reproduce, and you'll have generations of offspring following, and you will be the fit in that environment. Okay, so that's where they were heading with that question. So we went over a lot. We reviewed again natural selection. But my point to this, I want you to read what you wrote on that piece of paper, and I want you to look and see if you said anything that looks like the mouse evolved, like this mouse who decided like I better stay up at night, okay, or I'm going to be eating, and I just need to change my routine until I eat. The mouse evolved. That's what you're kind of saying. So that's called the Markian, and we don't accept that when you write about populations. You need to say the mouse population evolved to they were selected for, and then they had reproductive success. Okay. All right. Next is an AP exam practice. You're not going to do this right this minute. This is going to be your homework. Even though if you're all home now, I'm not really sure what homework is called. Anyway, if somebody has an idea, you can send that to our way on Instagram or Twitter or whatever it is that you want to talk about this on. Anyway, so I'm not going to, what I want you to do. I want you to go look at that diagram and figure it out, and then I want you to go find the passwords, what I need to do, read the passage, and then answer the question. And then tomorrow night, well, I won't say tomorrow night for some of you who will be watching it another time, but on the next lesson, I'll be starting off with this particular question as our warm up. So you can, after this is, I'll give you some time after, well, the video will be saved, and then you'll be able to rewatch it to get all the details of this question again. And thanks for watching. We hope you'll join us again. Be sure to complete the practice question, and we'll review the answer in the next lesson.
Thank you. Bye-bye.
Welcome to AP Biology, topic 7.2, natural selection. I'm Gail Pfizer. I'm from Darnell Cookman Middle High School in Jacksonville, Florida. That's the Turner College Board. Today, the topic we are covering is one that colleges have asked that all AP students study, even if it will not be on the exam. Colleges are supporting AP students this year by allowing this more flexible approach to the exam, where it will focus on a lesser amount of content than in other years. But in turn, they have asked that AP students learn today's material. To make this relevant to your upcoming exam, we'll also include some exam prep in today's lesson. In a week or so, after we've covered some of these key outstanding topics, we'll be posting a full schedule of review topics for the remainder of this course so that you can choose which topics you want to prioritize for review and thus which classes you'll want to attend. So first, I'm going to go over a lesson overview. So today, I will be introducing you to the principles of natural selection, focusing on phenotypical variation, and then we'll also be going over science skill 1B, which has to do with explanation. You'll be gaining proficiency in explaining biological concepts and processes. You should already be familiar with the causes of natural selection, how natural selection affects populations, explaining biological concepts and processes. By the end of this lesson, you'll also be able to describe the importance of phenotypic variation in a population and explain biological phenomena. So we'll have our warm-up first. So this is our homework from our last lesson, which was 7.1. And so we're going to start off every lesson with a warm-up, and it will be from the homework from the previous lesson. So this will be from 7.1. And before I will give you time after I go over it, for those that are just joining us for the first time tonight, I'll go ahead and tell you my strategy of going through questions, whether it be multiple choice or FRQ. I take an approach of looking at the diagram, and then I want an interpretation of that diagram to tell me the story of what's happening before I go deep into the question. So an example of me doing that and teaching my students to do this would be I would start with, I see 3 boxes. I see that we have blood, epithelial layer, intestinal lumen. I see there's a cell in the epithelial layer, and then there on the right is the intestinal lumen. So I must be in the, this must be a cell in the intestine, and then on the other side has to do with the absorption into the blood. So now that I know where I am, I can see the lie on the cell. Now I can look at the sodium. I see high sodium in the lumen and then low in the sodium. That's already ringing a bell with me that we're going to be moving sodium from a high concentration to a low concentration. And then I see sodium high in the blood. So we'll be going from a low sodium to a high sodium from the epithelial layer to the blood. And that's going to tell me that we're going against the gradient, and then I'm going to have to use some ATP to make that happen. Then I see the glucose. There's a medium amount of glucose in the lumen. There's high in the epithelial layer, and there's low in the blood. And so I know that the high glucose to low glucose, that will move in a concentration gradient, but somehow I have to get this medium glucose in the lumen to that cell. So now I'm looking at the diagram, and I see that sodium is moving from a high concentration to a low concentration through a transport protein. And while it's doing that, glucose is moving also. So it's acting as a co-transport into the cell. And now once we're in the cell, the glucose is now at a high glucose amount, and it's going to move out of the cell from high to low concentration, still through a transport protein. Now the sodium on the inside, I said earlier, that was low sodium, so we're going to have to use the sodium potassium pump and use some ATP to go against that gradient. So now that I kind of know the story and what's going on, now I'm going to see what the question is going to ask me. Generally, I would read the question, and then if there was a long passage, that I would go into the text last, the passage last. But this is a very short one, so I'm just going to do it all at one time. Okay, it says glucose and sodium move from the lumen of the small intestine into the blood via transport proteins in the epithelial cells lining the small intestine. Based on figure 1, describe the direct source of energy used to move glucose into the epithelial cell from the intestinal lumen. And now there's another, that was our first task word, that was describe. We went over describe in the previous lesson a lot. And then we also covered explain in just a small amount. So explain how the system maximizes glucose absorption from the intestinal lumen into the epithelial cells and from, and that and is key, because not only we're explaining the first half of that sentence, we're going to need to explain the second part too, and from the epithelial cells in the blood. And there's also another key word in there, explain how this system, so how this glucose is working from the lumen to the cell and back and out to the blood altogether. So I'm going to give you a minute, and I'm going to time you, and you go ahead and start answering that question. On every warm-up, you're going to need a pen and paper available. So because I'm going to, I'll wait on you for a minute, and then I'll tell you when I'm going to count down to 10 seconds and see how that works today. So go ahead and give the FRQ question a try. Okay, I'm going to give you 10 more seconds. Okay, now I know that wasn't time enough to answer this question, and what I will suggest is that after this live viewing of this video, that you watch it in pieces and slow it down at your own pace and redo everything that we're doing, and so you can work through them at a more comfortable pace. So check your answers against the scoring guideline. There are 3 points here. There's a describe point, which you should have addressed that the energy from the sodium gradient is what is the source of energy to move the glucose into the cell. Okay, and then there's 2 explains. Remember that and. So there were 2 places to explain. So there's 2 points here, and you should have addressed that the sodium potassium pump maintains the sodium concentration gradient and allows for co-transport of the glucose. And then you should have also addressed that the import of the inflow of glucose maintains the glucose concentration gradient between the epithelial cell and the blood and allows for facilitated diffusion of the glucose. All right, I hope you did a good job. All right, so I'm going to do this for the first few videos that we have. So as people start to jump on and be regular, then I won't tell you this every single time. But this is how the framework of the course. And so we've been working through different units this year: biomolecules, cells, photosynthesis, and we've broken those units into small topics. And each topic has questions called the learning objectives. And this is what you need to be able to do. And we need you to know content in order to be able to do that. So the content is called the essential knowledge, and each topic has an associated science skill that will be tested along with it. So this is how we're going to show we know the answer to the question. So the science skill that is associated with natural selection is visual. The suggested science skill for this lesson is visual representation. And you've probably been doing this in class all year anyway. So today we'll topic skill pair. We'll do natural selection and explain together. So what you need to know. Okay, this is my favorite part. Is I had to stop putting in more material today because there's just so much that I enjoy that we're about to talk about. So as from our previous lesson, we learned that natural selection is a mechanism of evolution. And also now tonight, we're going to talk about natural selection, phenotypic variation, and selective pressures. And I like to teach by examples. And so I have 2 examples that I'm going to talk about. I'm going to talk about the peppered moth, and I'm also going to talk about sickle cell anemia. And the peppered moth is it's a very simple story. In fact, I did have a video that was connected to this, but I was afraid of music copyright laws of getting me. So anyway, about the second week that my kids were separated from me and we were not in school, that I started missing them, and I was thinking I need to teach them some science, and I need to make it in as harsh and not harsh, in a very sweet and soft way that they'll enjoy and think it's fun. So what I'm about to show you is at a very elementary level, but it tells the story perfectly. I do have a video on YouTube that I did send them if you can find it. Anyway, so we're going to talk about the peppered moth. So what we have here, we have moths that have two phenotypes. We have peppered and a dark moth. And a phenotype, remember, is the observable physical trait, okay, or characteristic if you want to call it that. Now, their environment of the story that I'm about to tell you is on a light-colored birch tree with lichen. So can you see them? This peppered moth population has adaptation selected for in this environment. If you can see them, there are 6 of them that I placed on there. There are a higher number of peppered moths than dark moths. Birds are able to see the dark moths on light-colored trees. The dark moth has a phenotype selected against in this environment. And there is a lot of research about what I'm about to tell you. And this is the birch tree before the Industrial Revolution. And this is the birch tree turned dark from burning coal during the Industrial Revolution. Now, the peppered moths were detectable by the birds on this dark birch tree covered in coal. The birds now began eating the peppered moth, and now there were more dark moths reproducing than pepper. The dark moth phenotype was selected for in this environment. The peppered moth was selected against. The dark, wait, hold on just a second. Sorry about that. The dark moth phenotype was selected for in this environment. The peppered moth was selected against. I didn't see my period. All right, the dark moth population had adaptations that increased their survival and reproduction. Now, the peppered moths now had a lower population than the dark, and then that's when efforts to clean the air began. Now, the lighter environment allowed the birds to see the dark moth again. The dark moths were selected against in this environment again. And to take away from this example, natural selection acts on phenotypic variations in population. Environments can change and apply new selective pressures to population. Some phenotypic variations significantly increase or decrease fitness. This is reproductive success of the organism in particular environments. So I sent my this is really sweet and cute in this fast little video that I made for my students on this topic. All right, so now let's get to sickle cell anemia, and there is so much science that I love in this topic. First, we have 2 different phenotypes of red blood cell here. We have a normal red blood cell and we have a sickle red blood cell. And if you look in this blood smear over to the left, you can see that there are sickle cells that you can pick out. Okay, and this is what happens is that what a patient feels like. I used to be a nurse a long time ago before I was a teacher. Anyway, so there is the sickling that's going to happen. I'm going to tell you how that happens. But anyway, the membrane, there are characteristics of the membrane that change, not only just in the shape, and I'll tell you how that shape changes, but it starts sticking to the endothelium, and they start sticking together, and they end up blocking the good red blood cells that are able to carry oxygen. And so any cells past that point, they start having a decrease in availability of oxygen, and so they are starving of oxygen, and then sometimes they actually go through infarction and they die, and then those cells are unable to function anymore, and there's a lot of pain involved in that. So this is how it all starts. Okay, so this blue strand is DNA, and that is a template strand. Okay, what you're not seeing is the coding strand because you already see the messenger RNA complementary to that template strand. So there's the DNA, and you can see that there's one that's normal hemoglobin and one that's mutated hemoglobin. Now, in the normal hemoglobin, in the coding strand, okay, you should have a GAG for a particular codon. Well, there's this one single point mutation which happens, and it ends up being a GUG. The thing is, with the GAG, it codes for glutamic acid, and the GUG codes for valine. So what should be there in a normal hemoglobin is a glutamic acid, but ends up with a valine instead. Now, that single point mutation on chromosome 11 is responsible. Thymine is substituted for an adenine. So what you're seeing is this. I don't know if you can see any of this cursor, but what you're seeing is a U. Okay, where they're talking about a thymine is substituted for an adenine, but that would be on the coding strand if I had a coding strand here. Okay, ultimately responsible for producing the hemoglobin protein. This one single point mutation results in the production of the valine amino acid instead of glutamic acid. So this is called missense. Now, this is I'm going to give you a question later that this diagram is from. So let's go through what's on this diagram. There's a ribosome, and I see a large subunit and a small subunit, and I can tell that my messenger RNA is going through my ribosome, and I can tell it's heading in a 5' to 3' direction going in because this is codon 2, 3, 4, and 5. Okay, and this was my amino terminal end, and I have amino acid 1, 2, 3, and 4. So these are transfer RNAs, and here we have our GAG. So I'm talking about normal hemoglobin. So this right here is what's supposed to be here. This codon is for GAG, and if we look on our genetic code down at G, this is how you read it. There's a G, and there's a G, and there's a G. So there's GAG right down here, and that's going to code for glutamic acid, and that's what is supposed to be there. Okay, that's normal hemoglobin. So now let's look at an example with the mutation. I'm sorry. All right, so same situation. We have ribosome, we have transfer RNAs, we have messenger RNA going through the ribosome, and here we have our GUG. Okay, so there we have that mutation that you should not be there. That should be an A, right? Okay, so now we're going to be complementary to that anticodon, which is CAC, and that anticodon is responsible for bringing the amino acid valine into the ribosome to be part of the next or the polypeptide that it's building. So let's look at that. Here's a G, U, and a G. We have valine. So that's how we ended up with the wrong amino acid. All right, so amino acids have different properties, and they'll cause them to interact differently. So what happens is, if you see this long fiber, these guys are not supposed to be in a rod like this. They're supposed to be single hemoglobin units. Anyway, these mutated hemoglobin molecules now stick together and form a fiber rod, which gives the blood cell the sickle shape. So think about putting a growing rod in this red blood cell and how it would eventually push the plasma membrane around, and that's how we're forming the sickle shape. Now I talked about them having the amino acids having different properties. So let's go over the basics of this amino acid because it's probably been around a long time since if you started with biomolecules at the beginning of the year. So here we have amino acid, and we have the amine functional group on one side, and we have the carboxyl functional group on the other. There's our central carbon, and here is our R group, is what we're going to call it. Okay, here's our amine end, carboxyl end, here's our central carbon, and our R group, which is very different. Each of the 20 have a different R group, and they have they're grouped into different properties. And if you look at the glutamic acid, it's hydrophilic, and the valine is hydrophobic. And so what I tell my students is, if we all stood up and we were amino acids in a protein, where would the hydrophobic members be? And they say, well, we'd be in the inside of the group, right? Because as a cell, we're surrounded by water. So the hydrophilic ones would have to be on the outside. So I said, what if we changed the hydrophilic on the outside and gave them a substitution and then made them hydrophobic? What would happen? And they say, oh, they would all move to the center, right? So that's going to change the structure. And when you change the structure of a protein, then you're going to change the function as well. Okay, so I added a little bit about how you get sickle cell through and genetic inheritance of it. And so I drew a little simple Punnett square. And so I could say uppercase S, lowercase s, but I'm going to just do it like I usually do in my room. And we have a big S, little s. Okay, so the father's heterozygous and the mother's heterozygous. And if we have offspring from them, we can expect that 25% of them would be homozygous dominant, okay, with a normal hemoglobin. S in the uppercase is going to represent the normal hemoglobin, and the lowercase is going to represent the mutated hemoglobin. All right, so we have a 50% chance of having a heterozygous offspring, and we have a 25% of having a recessive offspring. Okay, variation of fitness. Homozygous dominant, you have 2 normal alleles. If you have 2 normal alleles, then the parasite responsible for malaria can use your red blood cells to reproduce, and then you'll have malaria, and you'll be very sick. Homozygous recessive, there's 2 abnormal alleles. If you have 2 abnormal alleles, then you will get sickle cell anemia and no malaria. The parasite cannot reproduce in the sickle cell. And then you have the heterozygous carrier. You have 1 normal allele and 1 abnormal allele, and if you have 1 normal and 1 abnormal allele, then you're a carrier, and you'll have the heterozygous advantage. No malaria, no sickle cell. So there are different outcomes for different phenotypes. Homozygous recessive gets the sickle cell anemia that I just talked about, and oxygen is not delivered efficiently. Your blood cells die young, and you become anemic. Tissues die, stroke, and organ failure can happen. When you're homozygous dominant, then you're you're able to get malaria. And if you have the heterozygous advantage, then you won't get either. If you're in an area with a high incidence of malaria, being homozygous recessive or heterozygous would protect you from getting malaria. Okay, so now for the science skill explanation. Today's task verb is explain. So we're going to learn how to explain biological concepts and processes. All right, explain. Explain is how or why a process occurs. It can also include comparing patterns and trends and representation, and or predicting patterns and trends and representation. So let's have a little check for understanding. And I want you to take a minute to tell me. I'm going to get my phone out. You can start writing now, and I'm going to time it. I'll tell you when you have 10 seconds left. Okay, and okay, go. I want you to explain the effect of color on the fitness of the peppered moth and the dark moth. Okay, 10 more seconds. And I know you need more time, perhaps. Okay. All right. So let's see how your answer compares. All right. Some things you might have said. There's not just one right answer. You may have said environmental changes with different selective pressures on the moth population. Dark moths have increased fitness or reproductive success. I always want you to follow up fitness with talking about what it means on dark trees because birds will not see them and eat them. Peppered moths will have increased fitness on white trees. Dark moths will have decreased fitness on dark trees. Phenotypic variation can increase or decrease an organism's reproductive success in particular environments, and natural selection acts on phenotypic variations in population. So let's work through some problems. I have a couple of multiple choice that we're going to go through, and it's still even though your exam is All right. So even though your exam is going to be FRQs, interpreting a multiple choice is the same thing. Okay. We don't have a diagram or table or graph in this particular question, but we still have text to interpret and find out what the story is telling us. So the bird Paris major is commonly found in man-made habitats. During the summer, these birds primarily feed on insects, and in the winter, when insects are scarce, they forage in gardens and bird feeders for seeds, nuts, and berries. Recently, climate change has led to a decrease in food available from gardens, and scientists have observed that the average beak length of the birds has increased. Which of the following best explains the directional shift in beak length of these birds? Okay, so what did I just read? I just read that in the summer they eat insects, in the winter they have to go to gardens for bird feeders for seeds, and now that's not there. So they need to find something else to eat, right? And their beak is and the average beak of the population, not just a bird, because we're not just going to say a bird has another beak, and you know why. Anyway, so which of the following best explains the directional shift in beak length in these birds? Our options are A, longer beaks allow the birds to better fend off predators. B, longer beaks increase the fitness of the birds in wild habitats. C, longer beaks allow the birds to better access seeds and bird feeders. And D, longer beaks decrease the fitness of the birds in wild habitats. I'll give you just a minute. I don't think you need a minute for this. Okay, 10 seconds. Okay, I hope that you chose answer B. Okay, here's the second question. It has to do with climate change also. All right, climate change is disrupting the length of winter in various ecosystems, resulting in less snowfall and milder winter. Scientists in Finland have been observing the tawny owl, a fairly large owl species that's commonly found in woodlands across much of Europe and Asia. Typically, the feathers of the upper body are either brown, and I'm going to say big B, and little b, or gray, little b. Scientists study tawny owls in Finland and have found that the percentage of tawny owls that have brown feathers has increased over the past 28 years. Which of the following best explains why the brown feather phenotype in tawny owl has increased over the past 28 years? Think about the length of the winter and less snowfall. Oh, I'm going to read you those. Okay, A, since the brown genotype is dominant, it will eventually eliminate the recessive gray genotype from the population. B, darker feather color in tawny owls makes them less visible to predators as they sit in the trees. C, with milder winter, the advantage of gray feathers then blend into a snowy environment that has decreased. And D, the directional selection toward a darker phenotype is random and likely due to a bottleneck effect. All right, take just a moment. I think we'll start at 30 seconds on this, so 10 more seconds. Okay, I hope you chose answer C. All right, now you need to write a little bit, just a sentence or two. All right, so now Charles Darwin proposed that evolution by natural selection was the basis for the differences that he saw in similar organisms as he traveled and collected specimens in, I just learn how to do that. All right, as he traveled and collected specimens in South America and on the Galapagos Islands. And now you need to explain, and explain is our verb now, okay? So remember that was how or why. The theory of evolution by natural selection as presented by Darwin. So explain the theory of evolution by natural selection, okay? I'm gonna give you a whole minute to do that. Okay, 10 seconds. Okay. All right, let's see what the scoring guidelines said for this one. Okay, there are a number of things that you could talk about. Okay. You could talk about reproductive potential, which is the ability to overproduce. You could talk about variability, inheritable changes or mutations linked to variability, limited resources, whether biotic or abiotic, competition, interspecific struggle for existence, differential reproduction, reproductive success of the variants, generations, time needed for evolution to occur, and elaboration. You could have expanded on Darwin's ideas such as effects of environmental change or artificial selection, or given a good linked example of something that I want you when you're talking about reproduction. Another good thing to always add in is about that it's not just happened, that it's multiple generations. Sometimes that's a good thing to add when you're talking about evolution and also experimental design. Okay, so I love this question. I just found it this morning. Anyway, I love it. And so I'm going to look at the diagram and I'll talk you through the diagram. And then it has 4 parts to it. And I want you to spend 20 minutes on it at home, but not now. And so anyway, we'll just run through it real quick and I'll just guide you through it. Okay. All right. So my diagram, I can see that there are 3 steps to what the question wants me to answer. I see a large ribosomal unit and a small ribosomal unit. I see that I have my messenger RNA. I see that we have some transfer RNAs bringing in some amino acids. We have codons 1, 2, 3, 4, and 5 as in triplets, as in those codons are those are representing 3 nucleotides. Okay. All right. So they're called a codon or a triplet. I sometimes use that. All right. So if you have number 3 if you look above it, it's at the 3rd amino acid and there are 2 above it, which means number 1 and number 2, their amino acid has been taken care of. It's already in the polypeptide chain. Now we have, if you remember, you have an A site, a P site, and an E site. And I tell my kids in a rival site, a polypeptide building site, and an exit site. So in the second one, I still see them and say the same location in step 2. I see a transfer RNA bringing in that it has just attached. What has just happened is that the amino acid that came in on codon 4 has now been added to the rest. Okay, and now it is on the codon 4's transfer RNA that's complementary to that. So you see that number 3 that they have shifted. Now they're in the polypeptide place, and then in the elongation, and then you have them in the exit without amino acid because now it has become part of this building of this polypeptide. So now let me read you the story. A processed eukaryotic messenger RNA lacking introns. So pay close attention to those words that I just said. If eukaryotic messenger RNA is lacking introns because there's been a nucleus, okay, and inside that nucleus there has went from messenger RNA through some processing steps, and the introns are still were taken out or cut out inside the nucleus before the messenger RNA, which was a process, came out and exited the nucleus. Now the messenger RNA is introduced into the bacteria. So now we're talking about eukaryotic messenger RNA being put into bacteria, so that the bacteria can produce a large quantity of the encoded polypeptide. Translation of the messenger RNA to produce a polypeptide involves the binding of the large and small ribosomal unit to a messenger RNA. Each complete ribosome has 3 transfer RNA binding sites that align with the 3 sequential codons in the messenger RNA. 3 steps in the process of translation elongation. The addition of amino acids to a growing polypeptide chain are illustrated in figure 1. And we just looked at that, so we already know what's going on in that diagram. All right, so let's look at A and look at B, and then we're going to give you a few moments to answer those two, and then we have a C and D. But I just want you to get a kind of a planning stage outline. You're not going to be able to write this because we can't spend 20 minutes here, but I do want you to try to spend more time at home on these FRQs that I've included. Okay, so here we have our describe. A has described why translation of a messenger RNA can start before transcription of messenger RNA is complete in prokaryotes, but not in eukaryotes. And I gave you every clue in that first part of that sentence. Okay, and then B, referring to figure 1, explain the process illustrated between step 1 and step 2. So that's going to be a how. Okay, and describe is going to be what are the characteristics of what that question is asking about. So I'll give you a couple minutes on that. I'll give you 2 minutes. Okay, because it's 2 questions. Okay, I have 30 seconds. Okay, all right. So let's move on to C and D, and then I'll reveal all of the answers at one time. Okay, now let's look. We've seen this before, right? So we already know what this is, and that's why I gave you that image before, so you can kind of get used to it. All right. So we have a large ribosomal subunit, small messenger RNA headed through 5 to 3' way. We have codons 2, 3, 4, 5. We have a building amino polypeptide chain, and we have transfer RNA. But now we have blank boxes. Okay, so this is your job now. In C, fill in the boxes in the template to it. Oh, and there's also a genetic code at the bottom, and I'm just go ahead and put a hint word in there. Universal genetic code. Okay, fill in the boxes in the template to identify the nucleotide sequence from 3' to 5' of the transfer RNA anticodon that will recognize codon 5. Okay, codon 5 is right here. All right, and the transfer RNA anticodon that will recognize codon 510, and that's right here of the messenger RNA template. And to identify the amino acids that will be added at these 2 positions, a codon table is provided for reference. I'm going to have to give you a minute to work on that before I read the next one. Okay, let's go 10 seconds. Okay, all right, let's read D. Referring to the figures provided, explain why the polypeptide produced when the transformed bacteria translate the introduced eukaryotic messenger RNA provides evidence for the common ancestry of all living organisms. Okay, so you have eukaryotic messenger RNA, and you have a bacteria ribosome. And ribosomes, remember, eukaryotic cell and prokaryotic cells have ribosomes. So give it a try for D. Okay, about 10 seconds. All right, let's take a look. I'm trying to give you 10 seconds. Okay, let's look at the scoring guidelines. So your answer for A should resemble this. Prokaryotes do not have a nuclear envelope that separates the genome from ribosomes in the cytoplasm of the cell, whereas eukaryotes do. I try to give you a hint about that nucleus. All right, the growing polypeptide chain attached to the transfer RNA aligned with the codon 3 of the messenger RNA is transferred to the amino terminus of amino acid 4 attached to the transfer RNA aligned with the codon 4 of the messenger RNA. So all you need to do was write down each step of the way what exactly was happening to answer B. All right, for codon 5, the anticodon sequence is GCC, and the amino acid added is arginine. The response also indicates that codon 510 there will be no transfer RNA with a complementary anticodon because UAG in the messenger RNA is a translation stop sequence, and no amino acid will be added, and translation terminates. Okay, and then D should resemble the polypeptide produced by translation of a processed eukaryotic messenger RNA by a eukaryotic cell or prokaryotic cell into which it has been introduced. Should have the same amino acid sequence because the genetic code is almost universal, and the same codon chart can be used for both prokaryotic and eukaryotic organisms. All right, I hope you. I know you didn't have enough time because it's clearly not enough time to answer an FRQ of that size. So go back to it after the video or when you have time to work on it. All right, so this is what we were saying. Why do I have this here? Okay, maybe to explain what was just going on. Yes. Okay, so there is that codon 5. There's CGG, and so there we have CGG on our genetic code, and then we have arginine here, and that's why that transfer RNA. So CGG and the complementary anticodon is GCC. Now the first thing that half of my kids want to do is look for GCC on this genetic code, and then they get the wrong amino acid. So make sure you're looking for the codon for the messenger RNA when you're figuring out what amino acid you should be bringing in. And then at 510, we have a UAG, and that is complementary to AUC. And if you look for UAG, I have it in aqua here, you'll see that that's where that stop is. Okay. All right. Short debrief and summary today. I know I have went through a lot of information. I really didn't have that much information about. That would have been a really quick video about the pepper moths and the sickle cell. So I added some things that could help you review along the way from some stuff you had at the beginning of the year and maybe the middle of the year also. Anyway, so the takeaways for today: phenotypic variation significantly increase or decrease fitness, and that's reproductive success of the organism in particular environments. And you should be able to describe the importance of phenotypic variation in a population. You should also be able to explain biological concepts and processes. The common error, misunderstanding that could come from the natural selection phenotype variation part of this today is when you use the word fitness. If you're writing about this and you just write the word fitness, you're not going to score a point. You have to in your words show that you are understanding what fitness means, and that you need to talk about reproductive success and that there's offspring, and use those kind of vocabulary words to define what you're talking about. You should define a lot of things that you're talking about when you're writing. If you think there's any question about whether you understand, the reader will understand that you understand. Okay. All right. So you're going to make reference to offspring inheriting genes for particular traits, and remember, don't speak Lamarckian. Now you do have homework. All right. It's short, very short. Now it's a one task per certain human genetic conditions such as sickle cell anemia result from single base pair mutations in DNA, and you're going to explain how a single base pair mutant in DNA can alter the structure and some cases that function of a protein. So now we're talking about the sickle cell anemia. That was the trait of phenotypic variation that we started this off with, and we moved this towards single base pair mutations and wrapped all of this biology together. So that's your homework for tonight, and I have 2 more slides I need to share with you. And when this gets saved, then just go back into it, and then you'll have that homework right at the very end. Okay. All right. So this is new from the College Board. Device and internet access. We know that not all students have access to the internet or a device. We're working on solutions to help students get what they need to show their best work. If you need mobile tools or connectivity, or know someone who does, you can reach us directly to let us know. And here is the address cb.org/tech. And thanks for watching. We hope you'll join us again. Be sure to complete your practice question. Ms. Kate will be tomorrow night with 7.3, and that's artificial selection. And we'll review the answer to the homework question. She'll start with that tomorrow in the next lesson. Okay, thank you.
And thank you for joining us today for AP Biology Lesson 3. My name is Allison Kate, and I'm teaching you today from my home in El Cerrito, California. I wanted to start with some things that I've been hearing throughout conversations in the AP Biology community about why we're teaching lessons from Units 7 and 8 if they're not going to be tested on this year's modified exam. And the reason for our lesson plans is that colleges have asked that all AP students are familiar with this content, even if it will not be on the exam this year. Please remember that each of the lessons also contain a skill, and these science skills can be paired with any content, so they're relevant across the AP Biology curriculum. Finally, each lesson has a practice exam, practice both in terms of practicing free response questions and also practice using the task verbs. In a week or so after we have covered some of these key outstanding topics, we'll be posting a full schedule of review topics for the remainder of the course. Then you can choose which topics you want to prioritize for review and which classes you want to attend. Of course, we hope you'll attend all of the AP Biology lessons. On Friday, April 3rd, an announcement will be made that will explain the new exam format and answer many of the questions you have about this year's modified exam, so please stay tuned. So we're going to start today with a warm-up, and the warm-up is from yesterday's lesson 7.2, which was taught by my colleague Gail Pfizer. And what we'll do going forward is the warm-up will be the last slide of the lesson, and we'll begin each new lesson by reviewing that warm-up. So in case case you didn't turn in yesterday, you still are going to have an opportunity to do this question. This might be a good time for me to remind you that it's always good to have pen and paper handy so that you can participate in these lessons rather than just have me talking at you. And I know it's kind of weird for both of us. This is the question from yesterday. The topic was natural selection. Certain human genetic conditions, such as sickle cell anemia, result from single base pair mutations in the DNA. You're asked to explain how a single base pair mutant in DNA can alter the structure and, in some cases, the function of a protein. So if you did this for homework last night, good for you. Have your answer handy because I'm going to go over the scoring guidelines and you can compare your response to the scoring guidelines. If you're just tuning in today, go ahead and try to answer this question kind of quickly. Okay, so here are the scoring guidelines. This question asked you to consider two things: what happens to the DNA and what happens to the protein. And this was a 4-point question. Each of the bullet points you see represent one way to earn a point. So for the DNA side of the answer to this question, you could have defined mutation, which we know is a change in the bases of A, C, G, or T. We all know that that's shorthand for adenine, cytosine, guanine, or thymine. You could have also described the different types of mutations. There's a list there, but it's not an exhaustive list. So you could have talked about a duplication, for example, or a frame shift, or a nonsense, deletion, or substitution. Not all of it, by the way. You could have also earned a point by describing the central dogma. So the central dogma explains that the flow of genetic information is from DNA to RNA to protein. Or you could have actually gotten into the process of the central dogma and explained that RNA is transcribed from DNA and proteins are translated from RNA. Another couple of ways to earn points were by talking about the translation of codons, which are 3 nucleotide sequences, and each of those 3 nucleotides or codon codes for 1 amino acid. And finally, you could have earned a point if you said redundancy exists in the genetic code, that we have 64 codons that code for the 20 amino acids, so that there's more than one codon for an amino acid. Those are the 3 points. I hope you did well on the DNA side of things. Let's look at the protein side of things. Remember that you were asked to either describe the change of protein function or structure. So let's look at the protein function answer first. And again, this was 1 point, so only one bullet point. You would only earn 1 point for either of those bullet points. So you could have described the function change. When you have the mutation, it could have altered the shape or conformation of the active site, which might result in differential binding. In this case, we're talking about the hemoglobin protein, so we're talking about oxygen binding may be changed. If you went to the structural side of the question, you could have talked about the fact that the change may have altered interactions. So those interactions could have been through hydrophobic or hydrophilic amino acid interactions, could have altered disulfide bond formation, it could have interfered with R group interactions, or with hydrogen bonding. So compare your scores to your answers to the scoring guideline, and I hope everybody got 4 out of 4. Remember that we'll end this lesson with a warm-up question or a practice question that you will do as your warm-up on Monday, since today is Friday. Okay, so overview of today's lesson. Today we're going to be introducing you to the principles of artificial selection, which is topic 7.3 in the course and exam description, and I'll talk a little bit more about that in a later slide and about where you can find that as a resource. And we're going to also be talking about science skill for BC, which is describing data from a data table or graph, including the relationship between the variables. And when I say that, I'm talking about the independent and dependent variables. We're going to practice today by describing data from a data table. We'll look at a sample multiple choice question that requires data analysis. We'll also look at a free response practice question that combines both the content of artificial selection and the skill of describing data from a data table. If you've tuned into the last 2 lessons, you should be familiar with natural selection. I'm sure you're familiar with that already from your classes. We are also going to ask that you be familiar with interpreting data tables and graphs and have an understanding of the statistics standard error of the mean. At the end of this lesson, you should be able to explain that through artificial selection, humans affect variation in other species. You should be able to explain that convergent evolution occurs when similar selective pressures result in similar phenotypic adaptations in different populations. And you should also be able to analyze data tables and graphs. So I also want to just talk a little bit about where we are today in the large framework of our ecobiology course. So we're currently working in Unit 7, which is called Natural Selection. The units are broken down into smaller topics, which are lessons. And today we're working on topic 7.3, as I already mentioned. Each topic has learning objectives, or LOs. And this is what you, the students, need to be able to do with the content knowledge. Today we're going to be working on 2 LOs. One of them is to be able to explain how humans can affect diversity in a population. And the second one is to explain relationships between changes in the environment and evolutionary changes in populations. And the last thing I'd like you to know is that about the essential knowledge. This is the knowledge that's required to perform the learning objective. So the essential knowledge for today's lesson is going to be about the evolutionary change and how humans influence that. Today's topic skill pair is artificial selection and being able to describe data. So here's what you need to know. This is the teaching part. And so again, if you haven't grabbed that pen and paper, now's the time. We're going to start with the definition of artificial selection, and it is defined as the selective breeding of domesticated plants and animals to encourage the occurrence of desirable traits. So I'm going to give you a chance to show what you already know about artificial selection. I'm going to give you 30 seconds to write down as many examples of artificially bred organisms that you can think of. So grab that pencil. Ready? Go. All right, let's see what you came up with. So, did you come up with cow breeding? Cows have been selectively bred across many generations to produce offspring with improved milk production. Farmers have also targeted breeding cows with a mutation, resulting in increased muscle mass. The resulting stock of cattle, which are called Belgian blues, have excessive bulk and produce more edible, lean meat. So, if you look at the image on the left, you can see a normal cow, and if you look at the image on the right, you see a Belgian blue. And it's kind of the Arnold Schwarzenegger, if you will, of the cow breeds. And you can see lots of lean meat there. How about this one? Did you come up with horse breeding? Horses have been selectively bred across many generations to produce variation according to a targeted function. So, I've given you 2 examples. We've got racehorses and draft horses. Racehorses have been bred for speed, and so are typically leaner, lighter, taller, and quicker, whereas draft horses have been bred for power and endurance, so they're sturdier and stockier. And finally, my personal favorite, dog breeding. So, dog breeds show an enormous amount of variety due to targeted selection of particular traits by humans. So we can see that we get hunting dogs and herding dogs and racing dogs and toy dogs. So an example of a hunting dog is a beagle, like Snoopy, and they were typically bred to be smaller in stature so they could enter foxholes. Herding dogs, like sheepdogs, were bred for heightened intelligence in order to follow herding commands. And just a personal note, I've had several cattle dogs in my own home, and they do herd humans. They've herded me and my children. There are also racing dogs like greyhounds that are specifically bred to be sleek and fast, and toy dogs like chihuahuas that are selectively bred for their small or diminutive size. So let's take a look at this image here, and you can see that there's a bunch of really cute cartoon domesticated dogs. They all were bred for particular traits, and their common ancestor is a wolf. So your key takeaway here is that through artificial selection, humans can affect variation in other species. Just so you know that artificial selection is not just done in animals. Artificial selection can also be done in plants, and many of our crops that our farmers grow today have been artificially selected for certain traits. So my example here is wild mustard. It is kind of the common ancestor. It's the wolf, if you will, in this example. And by selecting for certain traits, you can see that a variety of vegetables that we're familiar with can be grown. So I'll start with kale because it's kind of the vegetable right now, super healthy, and the leaf has been selected for in kale. If you look at Brussels sprouts, which I happen to love, but I know a lot of people don't, they've been selected for their lateral buds. And then if you skip over to the left side of this image and look at cauliflower and broccoli, which have been bred selectively for their flower and stems, you can see how you can get a wide variety of different types of plant species from from this ancestor, the wild mustard. Again, our key takeaway is that through artificial selection, humans affect variation in other species. One other thing about while we're on this topic of artificial selection is this concept of convergent evolution. The evolution, which is the evolution of similar features in independent evolutionary lineages. In this case, the environment selects the traits rather than humans, like we saw in artificial selection. So my example here is I've given you 4 different organisms. They all have different evolutionary lineages. We can see that there's a shad, which is a fish. We have a dolphin, which is a mammal, an ichthyosaur, which is a reptile, and a puffin, which is a bird. So they're all different in terms of their lineages, but they all share a streamlined body plan, and that's because they all live in water. So that streamlined body shape is adapted for life underwater. So again, kind of the key difference here is that in artificial selection, humans are selecting the trait, whereas in convergent evolution, organisms that are really different come to resemble one another because the environment is selecting those traits. So I wanted to kind of tie in a lab that goes along with this topic of artificial selection. This is from the AP Bio College Board lab manual. It's lab 1, and it's called artificial selection. Maybe some of you have already done this in your classes, and if you did do this lab, then this is probably a good opportunity for you to review it. And if you haven't done it, I'm going to share enough background so that you can at least see the objectives and how the data collection would be handled in a classroom situation. And then we're going to actually look at the data and do our skill, which is being able to describe data from data tables and graphs. So what you're looking at in these images are 4 different images of a plant that is known as a Wisconsin bass plant. And the reason it gets that name is that it goes through its generation from seed to seed quite quickly. So we are able to grow several generations in a relatively short period of time. And when I say that, this lab in the classroom really takes at least a month, maybe more. So this is kind of an ongoing investigation that you might see in your AP biology classrooms back at your schools. So the question that this lab is trying to answer is, can extreme selection change the expression of a trait that can be quantified in a population over a short period of time? Remember that we humans are going to select the trait. We are going to then track these populations over time, over 3 generations, and see if we've made a difference in that trait so that we can quantify it. If you were doing this in the lab, you need to make sure that you check your water levels of your plants every day and to be gentle when handling these plants and making sure that you're kind of not touching them if you don't have to. So the objectives of this lab are to investigate artificial selection as a mechanism of evolution. So this is different than natural selection, but it still results in a change in the phenotype of this population over time. So we also want to apply mathematical methods to the data from a real population to make some predictions about what could happen to the population in the future. And we also want to investigate how this selection acts on a phenotypic variation in a population. So you may be wondering what the trait or phenotype is that we're selecting for. And it's a trait called trichromes. And if you look carefully at the image that I have on the screen, you can see there are little hairs that are coming out of the main stem and petioles. Those are the trichromes. So that's the trait that we're going to be selecting for in this investigation. The term trichome comes from the Greek meaning hair, and they are fine outgrowths or appendages on plants. And their functions are several. First of all, they are going to interfere with the feeding of small herbivores. So if an herbivore comes up to this plant and it's got these hairs that are sticking out, it may deter them. It also keeps frost away from living cells and breaks up airflow across the plant surfaces and reflects sunlight in hot, dry, open habitats. So it's adaptive against protecting from being eaten as well as weather conditions. And so once you do this lab, you collect your data. You're going to want to use your class data. And if you have more than one AP biology section at your school, oftentimes teachers will combine that because more data is better data. You have a larger sample size. So after you did that, you've collected your data and recorded it. You're going to make a bar graph of this data. And I want to point out some things that you want to make sure you do when you construct your bar graph. So let's first start with the axes. On the x-axis, that is where you're going to put your independent variable. In this case, it's generation. And this experiment ran for 3 generations. On the y-axis, we have our dependent variable. This is what we're measuring. We're measuring the mean number of trichromes. And then you can see that we've given it a title. In this case, the title is just artificial selection for trichromes, but you could also simply say generation versus the mean number of trichromes. And then the last thing that you're going to do to your bars on your bar graph is add your standard error of the mean error bars. So in order to construct this graph, you would have had to calculate your sample size, your means, your standard deviation, and your standard error of the mean. Those are different skills that we'll address in future lessons, but I do want to say at this point that all of the formulas that you are going to need will be available to you. They're on the AP Biology formula sheet. It is also found in the course and exam description, and it will be made available to you on your exam day. So let's do a check for understanding, kind of putting it all together. So we're going to look at a free response question. This was a question that was given on the 2014 exam. And the idea here is that we're focusing on the skill of describing data from a data table, but we're also going to bring in our content, what we've learned about artificial selection. So let me read the question. I'd like for you guys to read along with me, and then let's kind of take a look at the data table together, and then I'm going to give you an opportunity to answer this question. So as we just learned, trichromes are hair-like outgrowths of the epidermis of plants that are thought to provide protection against being eaten by herbivores, which is called herbivory. In a certain plant species, stem trichrome density is genetically determined. To investigate variation in stem trichrome density within the plant species, a student counted the number of plants on the stems of 6 plants in each of 3 different populations. The student used the data to calculate the mean trichrome density, which is the numbers of hairs per square centimeter for each population. The results are provided in the table below. So again, I want you to take a look at the independent variable, which is the far left column. That's the population over time, and we have 3 populations. Okay, and then we have our dependent variable. We have 6 plants, but they have been we have calculated the mean trichrome density from those 6 plants, and you can see that each of those populations had a standard error of the mean that we calculated, and that is equal to 1. So the question now is going to ask you, based on the sample means and the standard error of the mean, identify the 2 populations that are most likely to have statistically significant differences in mean stem trichrome density. Once you've identified those 2 populations, you're asked to justify your response. So again, I'm just going to give you a little bit of time to think about this, and if you are watching this on a recorded version, you might want to stop here and give yourself an opportunity to answer this question. Okay, let's take a look at the scoring guidelines for this one. So I've given you the question again so you can see the question as you think about your answer, and the identification was worth 1 point and the justification was worth 1 point. The identification is populations 1 and 3 are the 2 populations that are most likely to have statistically significant differences, and let's talk about the justification, then I'll go back to the data table. The justification is that in these 2 populations, the error bars or 95% confidence intervals do not overlap. Okay, so let's back. Now, what I mean by that is if you look at the mean of population 1, it has a standard error of the mean of 1, which means that those error bars are going to extend from 8 to 10. If you look at the mean of population 3, it also has a standard error of 1, so the error bars will extend from 13 to 15, so that there's no overlap in those error bars from populations 1 and 3. If you look at population 2, the error bars would extend from 10 to 12, and therefore population 2 would overlap with 1 and 3. I hope that makes sense. For practice, if you graph the data, the mean data and the standard error of the means, you'll have more practice with that, and then you can also convince yourself that those error bars between populations 1 and 3 do not overlap. So I want to say a word about task verbs. We'll be talking about these throughout our lessons. The complete list of task verbs is also in the course and exam description. Today's question, when we were practicing, had 2 task verbs. It had identify and justify. So let's just talk a little bit about that and have some practice with these task verbs. The first one is identify. Identify means to indicate or provide information about a specific topic without elaboration or explanation. So if you think about the last question, you were asked to identify those 2 populations, and all you needed to say was populations 1 and 3. Okay? So to identify something, you could name it, right? So let's practice with the task verb identify. This image is also from a free response question from a previous AP Biology Ecology exam. And you can see that there is a graphic that shows a cell, and inside the cell there are several organelles. You can see that there's a nucleus, an endoplasmic reticulum, a lysosome, a Golgi apparatus, and a mitochondrion. You can also see that the cell is surrounded by a plasma membrane. The question is an identified task verb. I always tell my students that these are like your command terms. They're information about what this question wants you to do. So this question wants you to identify the most likely cellular location of the ribosomes that synthesize the CFTR protein. You could just omit that CFTR protein. It was specific for this question, and just identify the most likely cellular location of the ribosomes that synthesize protein. Hopefully that makes this one a little bit easier. So think back to your lessons on cell structure and function. Remember the organelle that makes the organelle that makes protein. Where is that organelle located? Okay? And if you answered the rough endoplasmic reticulum, you just earned yourself an identify point. Good job. All right, let's practice with the task verb justify. Justify means to provide evidence to support, qualify, or defend a claim, and or provide reasoning to explain how that evidence supports or qualifies the claim. So to justify something, you could support a statement with evidence, or use specific data to support a statement. Okay, so data is given. Really want to use that specific data to justify your response. So let's practice. This again is an image from a short FRQ from a prior exam, and you can see that there's a graph. And I'm just going to read what Figure 1, what the legend says. Figure 1 shows the effect of exercise on blood prolactin levels in adult males. The data represents the mean plus or minus 2 standard errors of the mean. So getting skill in describing this data that's shown on the graph, you can see on the x-axis we have with exercise and without exercise. We see two bars. The open bar is T at 0 hours, and the shaded bar is T at 1 hour. The y-axis, or what we're measuring, the dependent variable, is blood plasma prolactin, which is measured in nanomoles per liter. Okay, you can also see that there are error bars, as was indicated in that legend. Okay, so let's read the question. Researchers conducted a study to investigate the effect of exercise on the release of prolactin into the blood. The researchers measured the concentration of prolactin in the blood of 8 adult males at T equals 0, so it's before exercise, and after 1 hour vigorous exercise, which is T equals 1 hour. As the control, the researchers measured the concentration of blood prolactin in the same group of individuals at the same times of day 1 week later, but without having them exercise. You are asked to justify the use of the without exercise treatment as the control in the study design. Okay, so again, we're practicing using the task verb justify. Okay, so let's take a look at what you said about how you justify using the without exercise treatment as the control for the study. So the justification for either one of these bullet points would have earned you the point. You could have said that you attribute the changes to the concentration of blood prolactin to exercise only, or you could have said that you wanted to rule out any normal fluctuations in the blood prolactin levels or release. So task verbs are important. They're going to be used throughout all units of the AP Biology curriculum, not just in evolution. You can find them on page 199 of the course and exam description, which we call the CED, and the course and exam description and much more can be found on AP Central. And I'm just telling you guys about this because since you're not in school and you're not having access to your teachers in the same way that you did previously, if you go to AP Central and navigate to the AP Biology homepage, there's a lot of information, including announcements and information about the new exam that you'll be taking this year. So please visit it. All right, so now I'm going to give you some guided practice. We're going to do a multiple choice question that will give you some more practice with the skill of describing data from a table. So this is a data table that shows a trend of increasing milk yield for cows. Part of the increase in milk production can be attributed to the improved nutrition and health protocols for dairy cows. But if you look at this, you're looking at the year and you're looking at the average annual milk production in kilograms per cow. The question asks you, based on the information provided, which of the following best describes the source of the rapid increase in milk production from the years 1976 to 2015. So take a minute and analyze that data, and then select your answer choice from one of those 4 answer choices, A, B, C, and D. Okay. If everybody said B, the correct answer is starting in the 1970s, farmers increased the selective breeding of cows and bulls for offspring that produce more milk. All right, so now we're at the debrief and summary. So hopefully, after listening to today's lesson, you should be able to explain how humans can affect diversity within a population. You should be able to explain the relationship between changes in the environment and evolutionary changes in the population. Further, you should be able to explain that convergent evolution occurs when similar selective pressures result in similar phenotypic adaptations in different populations or species, and you should be able to analyze data, tables, and graphs. There are 2 key takeaways. These are the things that I'd like you to remember after this lesson is over. That evolution is characterized by a change in the genetic makeup of a population over time and is supported by multiple lines of evidence. In this lesson, artificial selection is a line of evidence. And through artificial selection, we humans affect variation in other species. Think about domesticated dogs and the vegetables that you have for your dinner. Before I leave you with homework, I wanted to talk about some common errors and misunderstandings that students have when it comes to describing data. And in order to do this, we're going to take a look at another short FRQ from the 2014 exam. And first, we're going to take a look at the question. Then we'll look at the scoring guidelines for this question. And then we'll look at the common errors and misunderstandings that were seen by the AP readers when this question was scored. So this is a more global view of how all of the AP biology students approach this question on the 2014 exam. And hopefully we can look at what most students or many students did and avoid making those same mistakes. So before we actually get into the text of the question, let's look at the graph because we're working on the skill of describing data from a graph. Okay. And again, I want you guys to look at the axes. So if you look at the x-axis, we've got our independent variable, which is time in months. On the y-axis, we have our dependent variable, which is the mean number of spots per adult male guppy. I also want you to notice that the lines on the graph are different. We've got a solid line that's representing when the predator is absent, and we have a dashed line that is representing when predators are present. So there's already a lot going on. And then on top of that, we have standard error of the mean error bars. So it takes a while to kind of digest what is going on in the graph, and you'll notice that some of those standard error bars are much larger than others. And so you want to pay attention to the height of those error bars to give you some more information about what is going on with that data. Okay. So now let's take a look at the first part of the question, which just gives you kind of background and a setup of what's going on, and then it tells you about how this data was generated. So I'm going to read it, but you guys should read along with me. Adult male guppies exhibit genetically determined spots, while juvenile and adult female guppies lack spots. In a study of selection, male and female guppies from genetically diverse populations were collected from different mountain streams and placed together in an isolated environment containing no predators. The study population was maintained for several generations in the isolated area before being separated into 2 groups. One group was moved to an artificial pond containing a fish predator, while the second group was moved to an artificial pond containing no predators. The 2 groups went through several generations in their new environments. At different times during the experiment, the mean number of spots per adult male guppy was determined, as shown in the figure below. The vertical bars in the figure represent 2 standard errors of the mean. So these are the questions, and also let me read them, and then let's go back and I'll talk about what's important about what you should look for when you read these questions. So in part A, you were asked to describe the change in genetic variation of the population between 0 and 6 months and provide reasoning for your description based on the means and the standard error of the means. In part B, you were asked to propose one type of mating behavior that could have resulted in the observed change in the number of spots per adult male guppy between 6 and 20 months. And finally, in part C, you were asked to propose an evolutionary mechanism that explains the change in the average number of spots between 6 and 20 months in the presence of a predator. So I'm just putting in a plea to ask you guys to read these questions very carefully. Pay attention to your task verbs. In part A, you have describe and provide reasoning. And also notice that in each section of this question, they're asking you to look at a different aspect. So in the first one, you're looking at the change in genetic variation between 0 and 6 months, and then to try to provide reasoning why. And it's directing you, when you provide the reasoning, to look at the means and the standard error of the mean. The second part is asking you with the task verb proposed to look at a mating behavior that could have resulted in the change in those number of spots. But now the time frame is different. It's between 6 and 20 months. So if you were kind of hurrying to get your question read and answered, you might not have paid attention to that difference in the time frame. And then finally, you have another proposed, and this time it's an evolutionary mechanism rather than a mating behavior that would explain the change in the average number of spots, same time period, right, between 6 and 20 months, but this time looking only at the line that has the presence of the predator. So you were asked to do a different task in each part of this question. So let's look at the scoring guidelines. The first part A, asks you to describe the change and provide reasoning. So they were each worth 1 point, and the change is that the genetic variation was decreasing. And the reason we know that, looking at the standard error of the mean, it gets smaller. So look at the size of those error bars. In part B, to propose one type of mating behavior, it was also worth 1 point, and you could have earned a point for either of those bullet points. The first one is that there was sexual selection for individuals with more spots, so more spots were favored by one of the sexes, or it could have been random mating behavior that resulted in more spots by chance. So remember, this is a fairly small population. And finally, in part C, proposed was worth 1 point as well, and any of these 4 statements would have earned the point. So the first one is directional selection against individuals with large numbers of spots, or another way of saying this is directional selection for individuals with fewer spots. The result is the same. You could have earned a point if you use natural selection in context. And here I want to go back to something that we discussed in a prior lesson. You don't want to say anything about, the guppies wanted to have more spots, right? Because that's Lamarckian language. You also want to make sure that you include the key phrase survive and reproduce, right? So that is important when talking about natural selection, and that's what it means with used in context. And finally, because these were small populations, you could have earned a point for, say, genetic drift resulted in several generations of decreased numbers of spots. Okay, so takeaway, these are the kind of the global trends that were noticed when this question was scored. Many students were unable to appropriately analyze the data that was provided in the question. The question showed the relationship between the mean number of spots per male guppy and time, but the y-axis units were misinterpreted by many students as the number of individuals instead the mean number of spots. So again, please read that y-axis carefully. Other errors included confusing the lines representing the population in the absence of predators with the line representing the population of presence of predators. So that's that solid line versus the dashed line. Students also struggled to correctly assess the variation in the number of spots and frequently responded that the genetic variation of the population was increasing because the slope of the line was positive. And finally, students were unable to speak clearly about the mechanism of natural selection as it relates to evolution, including that organisms chose to evolve. And I mentioned that previously. So you want to avoid any Lamarckian evolution language. My key takeaway for you guys is to, when you are trying to describe data from a graph, make sure you look at the x-axis and remember that it's for the independent variable and that's what's changing in the experiment. And then you want to look at the y-axis for the dependent variable, which is what is being measured. And pay attention when reading the question and make sure you're answering what is asked. So we've come to the fun part of today's lesson, which is your AP exam practice, aka your homework. Since you're at home, it's just going to be AP exam practice. And this is also a prior AP exam question. So it is practice using describing data from a table. All right. So again, before I read you the question, you can either take a screenshot of this right now, or once it's recorded, you can navigate to this and take your time to answer the question. Ms. Pfizer will be with you on Monday for the next lesson, and she will begin her lesson by debriefing this practice form. Okay. So to determine the likelihood that a patient with a bacterial infection who is in the intensive care unit, which is ICU of a hospital, could be successfully treated with antibiotic therapies, researchers investigated the frequency of antibiotic resistance in bacteria isolated from patients in ICUs in the United States from 1994 to 2000. The data from the study are shown in the table, along with the overall change in the frequency of bacterial resistance from patient samples from years 1994 to 2000. So you can see your antibiotic list in the far column. In the middle column, you can see the percent of all samples demonstrating antibiotic resistance in the year 2000. And then in the far right column, you can see the overall change in percent resistance to antibiotics from 1994 to 2000. Here's your task. A patient was admitted to the ICU in December 2000 with an uncharacterized bacterial infection. Based on the study, identify the following. First, the antibiotic that was most likely to be effective for treating the infection, and second, the antibiotic that was least likely to be effective for treating the infection. So that's your homework. And again, Ms. Pfizer will go over that as her warm-up on Monday. I'd like to thank you for watching. I hope that you'll join us again. Be sure to complete this practice question that will be reviewed on Monday in the next lesson. I have one more slide that I'd like to share with you. This is on behalf of the College Board. We know that not all students have access to the internet or a device. College Board is working on solutions to help students get what they need to show their best work. If you need mobile tools or connectivity or know someone who does, you can reach us directly to let us know. And you can reach out at cb.org/tech. So obviously, if you don't have access, you're probably not watching this. So if you have friends or you know students, anyone who needs a little bit of support, please encourage them to reach out. Thanks so much for watching, and we'll see you next week.
Welcome to AP Biology. I'm Gail Pfizer from Darnell Cookman Middle High School and Florida State College in Jacksonville, Florida. Today, the topic we're covering is one that colleges have asked that all AP students study, even if it will not be on the exam. Colleges are supporting AP students this year by allowing this more flexible approach to the exam, where we'll focus on a lesser amount of content than in other years. But in turn, they have asked that AP students learn today's material. To make this relevant to your upcoming exam, we'll also include some exam prep in today's lesson. Soon, after we've covered some of these key outstanding topics, we'll be posting the full schedule of review topics for the remainder of this course so that you can choose which topics you want to prioritize for review, and thus which classes you'll want to attend. Now for an overview of our lesson. So today we're going to be talking about topic 7.4, which is population genetics. I'll be introducing you to population genetics, science practice skill 3B, which is questions and methods, and we're going to be stating the null or alternative hypotheses and predicting the results of an experiment. You should already be familiar with the causes of natural selection, how natural selection affects populations, the importance of phenotypic variation in a population, and explaining biological concepts, processes, and phenomena. By the end of this lesson, you'll also be able to explain how random occurrences affect the genetic makeup of a population, describe the role of random processes in the evolution of a specific population, and describe the change in the genetic makeup of a population over time. So now for our warm-up. If you've been watching, you know that the homework from the previous night will be the warm-up for the next lesson. So this is from the homework from 7.3. Let's take a look at our data table and see what this is about first. Okay, incidence of antibiotic resistant in patients admitted to ICU from 1994 to 2000. Okay, we have a list of antibiotics, ciprofloxacin, gentamicin, ampicillin, imipidine, tazobactam, and we have the percent of all samples demonstrating antibiotic resistance in 2000, and then an overall change in percent resistance to antibiotics from 1994 to 2000. Okay, let's read the story. To determine the likelihood that a patient with a bacterial infection who is in the intensive care unit, the ICU of a hospital, could be successfully treated with antibiotic therapies, researchers investigated the frequency of antibiotic resistance in bacteria isolated from patients in ICUs in the United States from 1994 to 2000. The data from the study are shown in the table, along with the overall change in the frequency of bacterial resistance in patient samples from 1994 to 2000. Okay, what is our job to do? A patient was admitted to ICU in December 2000 with an uncharacterized bacterial infection. Based on the data from the study, identify the following: the antibiotic that was most likely to be effective for treating the infection, and then identify two identifies here: the antibiotic that was least likely to be effective for treating the infection. I'll give you about 20 seconds to do that if you are just joining us for the first time. Okay, I'll give you 10 more seconds. Okay, let's see what the scoring guideline indicated. All right, so there's 2 points here because you identify 2 medications, 2 antibiotics. And when you identify, it can be as simple as saying the word, okay? So if you put imipidine is the most effective and ampicillin is the least effective, you got both of those points. Okay, I'm going to go through this for a few more times in this amount of detail, and then probably by the time we start reviewing, I'll start dropping this, or then just narrow it down to what topic we're going to be reviewing. Okay, so the topic of tonight, we're doing population genetics, and that falls under the unit evolution or natural selection, and each topic has questions called learning objectives, and this is what we need to be able to do, and we need to know content in order to answer these questions. That content is called essential knowledge. Each topic also has an associated science skill, and this is how we're going to show that we know the answer to the question, the learning objective. The science skill is associated with population genetics, questions and methods, and this includes a null hypothesis or an alternate hypothesis and predicting the results of the experiment. Today we'll topic skill pair population genetics with questions and methods. Okay, here's what you need to know. All right, so the learning objective: explain how random occurrences affect the genetic makeup of a population. We're going to learn about mutations, genetic drift, 2 types. We're going to talk about bottleneck, and we're going to talk about founder effect, and then we're going to talk about gene flow. The first occurrence that we're going to talk about is mutation. Mutations are changes in DNA sequences. They can happen spontaneously in replication and result from environmental factors such as radiation or chemicals. Mutations can result in a recessive disorder or a dominant disorder. A change in the DNA sequence caused by mutations may affect the structure and function of proteins. Mutations result in genetic variation, which provides phenotypes on which natural selection acts. This changes the genetic makeup of a population over time. The next topic I want to talk about is genetic drift, and we're going to talk about 2 examples of that. We're going to do bottleneck and founder effect just a moment. All right, so genetic drift is a non-selective process occurring in small populations. The smaller the number of organisms, the more likely the representation of an allele will change or disappear. There are 2 types of genetic drift that I'm going to talk about: bottleneck and founder effect. Okay, so for bottleneck, we're going to talk about the great prairie chicken, and I'm going to tell you the story. I read some research articles to pull all this together. In the mid to late 1800s, in the state of Illinois, there were millions of great prairie chickens. In 1993, there were only 50. The great prairie chickens lost natural habitats of prairie lands because of human activity, such as creating farmland. This reduced population had lost genetic diversity and variation. The genetic diversity of the great prairie chickens had declined over the years to this bottleneck. To prove there had been a decrease in the number of alleles, researchers used the DNA of great prairie chicken specimens from a museum as a control for the pre-bottleneck alleles. They extracted DNA from museum specimens and chickens from the central Illinois and other surrounding areas. They analyzed the DNA from the samples taken from the feather root, and they looked at 6 loci for differences in the number of alleles. To do this, they ran PCR and gel electrophoresis. Their results indicated there were 5 missing alleles. So genetic diversity had in fact decreased from human activity. Okay, here is the state of Illinois. And this is not to scale, and these great prairie chickens do not represent any number. This is just a representation of there was a lot of prairie land for them, and there were a lot of great prairie chickens, and then that was reduced, and they were down to just 50 in 1993 of great prairie chickens. So if I said some words that you didn't understand, or you were thinking about 2 alleles, I added this part to help you out. Okay, so here we have a homologous pair. I'm going to give a mouse so I don't know if you can see that. No, I think I can do it with my finger. Okay, so here's a homologous pair of chromosomes. So this is one came from your mom and one came from your dad, and so here is these dark bands. Those are actually genes, and they have a location, and we're going to call that a loci. And so if you, it's just like living on your street. Okay, if this was your street, then you would have an address on that street, and that would be your location, right? So that's the loci that we're talking about. So you can have more than 2 alleles, and they tested the great prairie chickens at 6 different loci, and that means that probably on 6 different chromosomes, a particular area on each one. So the number of alleles went from 22 pre-bottleneck to 17 post-bottleneck. They lost 5 alleles during this time. All right, the next example is founder effect, and I'll share something with you when I get through talking about this. Okay, founder effect has to do with a small group of individuals leading a population and establishing a new small population. This results in decreased variety of alleles carried to this new small population. Their new gene pool is different from their original population. One example of founder effect is polydactyly. Polydactyly is a symptom of a few different syndromes. Polydactyly means extra digits. These syndromes are commonly found in the old order Amish of Pennsylvania. Recessive genes are responsible for the traits of these disorders. In 1750s to 1850s, a small group of Amish people came to America and settled in Pennsylvania. They left Germany and other areas due to differences in religious ideas. They only brought the gene pool of those individuals. In addition, the Amish marry within their own community. So founder effect traits such as polydactyly can be seen in descendants. Well, I'll share a story with you. When I have polydactyly, and when I was 5 years old, my mother had my extra digit cut off of my hand, and I also have both hands. And it was a type that's what's called an ulnar type, and it's a type B. So it wasn't a full form bone finger, and it was only like a tag of a finger-like part of the skin. Okay, that resembled a small finger. And anyway, so I have been sequenced, and I have 2 variants and 2 genes that have to do with Ellis-Van-Creveld syndrome. So I didn't know all this until I started working with this for you. And so that's a good day at work for me when I can put something together and learn. Okay, the next way, let's see, we're talking about gene flow. Okay, so this is my favorite way to talk about gene flow. This is a immigrants on an Atlantic liner, and they're coming to America. And so when people move into a new city, they bring their genes with them, and that's called gene flow. All right, these random processes, mutations, genetic drift, and gene flow can reduce the genetic variation of a given population and can increase the differences between populations of the same species. So our science skill today is going to be about questions and methods, and the task verb today that we're going to focus on is state. And this is what we'll be stating. The null hypothesis is what we're going to be working on today. Okay, and we're going to be predicting the results of an experiment. This is an FRQ from the very first year that I taught AP biology. And what you see in the diagram, you see a choice chamber, and on one side it has a glucose-soaked cotton ball, and on the other side it has a dry cotton ball. And so let's read what the story tells. All right, in an investigation of fruit fly behavior, a covered choice chamber is used to test whether the spatial distribution of flies is affected by the presence of a substance placed at one end of the chamber. To test the flies' preference for glucose, 60 flies are introduced into the middle of the choice chamber at the insertion point indicated by the arrow in the figure above. A cotton ball soaked with a 10% glucose solution is placed at one end of the chamber, and a dry cotton ball with no solution is placed at the other end. The positions of the flies are observed and recorded every minute for 10 minutes. Predict the distribution of flies in the chamber after 10 minutes and justify your prediction. So take just 20 seconds and you make a prediction of where those 60 flies are going to be. All right, how about 10 more seconds? Okay, I'm going to show you my prediction. Okay, I know those are bees and they're not flies, but it's the only shape that was even resembled being close enough to put in here. Okay, so you see we have our choice chamber down the bottom, the glucose soaked, the dry, and my prediction, I predicted 58 glucose will be on the glucose soaked end, 1 in the middle, and 1 on the dry cotton. So let's take this another step because I did that because they would want sugar, is what I was thinking. Okay, before we move on, I want to ask you a question, and my question to you, and I hope you know the answer, is what would you do differently here? What did you see as an initial problem in this diagram and the initial start of this experiment? I see that there is a dry cotton ball and there's a glucose soaked cotton ball. So I think that it would have been better to start with not a dry, but maybe a soaked cotton ball with sterile water. So it wouldn't be the wet or dry that could be interfering. It would be about the glucose. I just wanted to see if you maybe caught that on the first round. Okay, so here's B, and I'll give you a couple minutes to do B. The experiment described above is repeated with ripe bananas at one end and unripe bananas at the other end. Once again, the positions of the flies are observed and recorded every minute for 10 minutes. The positions of the flies after 1 minute and after 10 minutes are shown in the table below. All right, I'm going to go to the next and show you the table. Okay, so here's the distribution of flies in the choice chamber at 1 minute and at 10 minutes. And there's at the end with the ripe banana. At 10 minutes, we were 45. In the middle, we had 3. And on the end, we had 12. Now, there is the chi-square formula, because you're going to be doing chi-square calculations today. You're going to write a null hypothesis, and you're going to be doing chi-square. So let's go through this formula for a minute. All right, so this symbol right here means summation, okay? And this O is talking about observed. So I would have observed 45, and the next one would be I observed 3, and then I observed 12. So expected, I'm going to leave that to you because you're going to be writing a null hypothesis. And so I hope that you understand what that means. And if you don't, I'll make it really clear when we get done. But one other thing about this formula, see that chi right there, chi-square, that is not an x squared. So don't find this and then think that's x squared and then get the square root of this. Don't do that. Okay, I'm going to give you 2 minutes to work through this. And in the beginning, I should have told you to get some paper and a pen, and in the future, some graph paper and calculator too. Okay, how about another 30 seconds? And 10. And okay, let's take a look at the answer. Okay, so to write a proper null, okay, this is what I do. And I think there's 3 things I really need to say to cover all my bases when I write this. And I always think, well, null has 3 letters, right? Okay, so I need to say that the number of flies will be equal in all 3 sections. Or I could have said it like the flies will have the same amount of preference for each area. Okay, then there is no statistical difference. And then any difference is due to chance. So I use this as a framework because I can substitute anything I'm talking about into that first sentence. And then I will be able to know what else I need to say and have some frame of reference to check myself back. Okay, what was no statistical difference with no chance or chance. But if you do it this first time when you write your null, you will be able to control which way you're writing and that you don't make an error and change things up as you're writing. So let's take a look at this map. We had 45, observed 3, 12, and totaling 60. And then if we didn't expect any difference in their choices, okay, or their preferences, then there would be 20, 20, and 20. And then you would have done the calculations and come up with a chi-square value of 48.9. All right, so here's our chi-square formula, and I'm going to run through how to do the math, and then it's going to go really, really fast. Okay, I hope you were looking at the map and not trying to look at my fingers. All right, so here is our next step. We're going to take this a little bit further. This is a degree of freedom table, and this is only a small section compared to various tables. I mean, there's a lot of information can be on a table. It can be a different orientation. The p-value can be at the top. The degrees of freedom can be on the left, the right. It can have all kinds of differences, but this is the one that the College Board has on the exam. So we're in biology. We're going to use our p-value of 0.05, and now we're going to look at our degree of freedom. We had 3 categories, right? We had the right, we had the middle, and then we had the not right. So when we're determining our degree of freedom, we're going to do our number of categories that we're looking at -1. So this would be 2 degrees of freedom. Now our chi-square value is 48.9. So let's look and see under 2. There's no 48.9, is there? So what's happening with the numbers underneath it? So here it is at 0.05. Our critical value is 5.99. So we have 9.21. So what are those numbers doing? They're increasing. So 48, not 0.9 would be way down here. So as the chi-square value increases, okay, we're also going to have, let's look at our p-value. Here's p 0.05, 0.01, and as our p-value decreases, right, then our chi-square value increases. So if our p is less than 0.05, we're going to reject our null. This is not due to chance, okay? Less than 0.05, not due to chance. Okay, so this was the scoring guidelines. We predicted there was 1 point for just predicting the right number of expected in each, and then 1 point for justifying the prediction, which is our null hypothesis. That was a separate point. The flies will be evenly distributed across the 3 different parts of the choice chamber. So that said the same thing as they prefer each equally, okay, or there would be the same number in each, and that would be because of the glucose. All right, so check for understanding. I have a multiple choice. In fact, I think I have 2 in here, and I looked up how to pronounce I think it's Tarisha. I think it's Tarika. All right, the California. Let's just go with the California. All right, lives in the coastal areas around Los Angeles. Which of the following is a valid null hypothesis relating fitness to survival of a bottleneck event in a coastal area where a small isolated population of California newts reside? Okay, A. Only those salamanders with the lowest evolutionary fitness will be eliminated by the bottleneck effect, lowering their allelic frequencies of their traits. B. Only those salamanders with the highest evolutionary fitness will survive the bottleneck event, raising their allelic frequencies of their traits. C. Surviving the bottleneck event will be random, so any change in the allelic frequencies of the salamander population is not attributed to fitness. And D. Surviving the bottleneck event will be random, so there will be no changes to the allelic frequencies due to the bottleneck event. Okay, I'll give you, let's go 15 seconds. Okay, I hope you chose C. Surviving the bottleneck event will be random, so any change in the allelic frequencies of the salamander population is not attributed to fitness. All right, now for some guided practice. All right, so in this table we have some salamanders, and we have their phenotype. They're a black phenotype, dark brown, light brown, and we have a flood involved. Okay, there is a distribution before the flood. There's 13, 25, and 6, and then after the flood there's 7, 14, and 3. Okay, so which of the following is appropriate null hypothesis regarding the phenotypic frequencies of this population of salamanders before and after the flood? All right, so A. More black salamanders survive because they could better hide from predators on the dark muddy soil, so the frequency of the black phenotype significantly increased in the population after the flood. B. Fewer light brown salamanders survived because they had reduced chance of finding mates due to the high water levels, so the frequency of the light brown phenotype in the population decreased after the flood. C. More dark brown salamanders migrated to a new drier area because of the flood, and fewer of them remained in their original habitat, so the frequency of the dark brown salamander significantly decreased in the population after the flood. And D. The proportions of black, dark brown, light brown salamanders before and after the flood are not significantly different, and any observed differences are due to random chance. Okay, I'll give you 15 seconds. Okay, and I hope you, this is practically what I told you earlier to write as a null. I hope you chose D. The proportions of black, dark brown, and light brown salamanders before and after the flood are not significantly different, and any observed differences are due to chance, to random chance. Okay, this one is so fun. All right, so you're going to need, this has to do with doing, you're going to draw some diagrams yourself, and there's a lot of science to learn in this drawing, in this example. So let's look at our diagrams. We have an aquarium, and we have a negative control aquarium, and we have an experimental aquarium. And what else do we have? We have a male flower, we have a female flower, and we have invertebrates. Okay, let's read the story, and we're going to work on A. Okay, all right. Seagrasses are aquatic plants that reproduce sexually. Male seagrass flowers produce sticky pollen that is carried by circulating water to female flowers, resulting in fertilization. A researcher claims that the mobile aquatic invertebrates can also transfer pollen from male to female flowers in the absence of circulating water. To investigate this claim, the researchers set up aquariums to model the possible interactions between the invertebrates and the seagrasses. Using the symbols below and the template aquariums to demonstrate the experimental design for testing the researchers' claim, the mobile aquatic invertebrates can pollinate seagrass in the absence of circulating water. Now, draw the appropriate symbols in the negative control aquarium and the experimental aquarium, and don't use any symbol more than once in each aquarium. Okay, I'm going to give you a minute to do that. About 15 more seconds. Okay, let's take a look. All right, so look at the aquariums at the bottom. So you should have a male and a female in your negative control aquarium, and you should have a male and a female and an invertebrate in the treatment aquarium. Okay, so the purpose of the control. Okay, so I think that everybody can say the control is the no treatment and the experiment is the one with the treatment. But what is the purpose of having the control? The purpose of having the control is they said that this invertebrate was responsible for carrying pollen from that male to that female. So we have to, in the negative control, see without water if it's possible to transfer pollen from that male to that female without that invertebrate variable that was added. Okay, so that was the purpose of the negative control, and that's the purpose of a control. You have to see what happens without the treatment. Okay, and so you can claim that the treatment was responsible. All right, now let's do B. Identify the dependent variable in the experiment. Predict the experimental results that would support the researcher's claim that mobile aquatic invertebrate can also transfer pollen from male to female flowers in the absence of circulating water. So now that you've seen what the right answer for A is, now what is the dependent variable, and predict what the outcome is going to be of this experiment to support the researcher's claim. Okay, I'll give you just a minute to do that. Okay, let's take a look at B. All right, so your dependent result should be, your dependent variable is the results, right? And that should be that fertilization occurs, okay? And what's going to happen is you should have no fertilization in the control tank and treatment and fertilization in the treatment tank. That the male is not going to be able to fertilize the female in the no treatment tank, and the male will be able to fertilize the female via the invertebrates carrying the pollen over there, okay? All right, to the female. So that should have been your dependent variable, the results, which is fertilization, and then fertilization no, and the negative control yes, and the treatment tank. So this is what the rubric looks like for this particular. And when you have one of these, if you have a prediction involved, it has to be in support of your what you identified. It can't jump. You can't have one in the third column on the bottom here supporting one at the top column over here. Okay, so was I right? Fertilization was what I identified as my dependent variable, and we're going to have fertilization is seen in the experimental aquarium. I want you to also follow up with that if you were to write that with no fertilization is seen in the control, okay? Because we have to see if it's able to happen in the control tank without that invertebrate. Okay, all right, debrief and summary. Okay, today's takeaway: evolution is also driven by random occurrences, mutations, genetic drift, two types of genetic drift, or bottleneck and founder effect. I hope you enjoyed learning about the great prairie chicken and polydactyly, and then the gene flow. I hope that you also could explain to someone else these random occurrences, and I hope you're really a whole lot better at stating a null hypothesis and predicting experimental results. Common errors and misunderstandings. A prediction must compare the experimental group to the control group. Why do we need the control? To show the result is not occurring without the treatment. The difference between a negative control and a positive control, and my kids had in my classroom had the same question, but we had one particular lab that kind of made it all clear. In a negative control, no response is expected. No treatment is added. The dependent variable results you're looking for will not occur. Okay, and a positive control will reflect a known result. And a good example of this is when you're running gel electrophoresis. A positive control will tell you if a result is in fact negative because it's really negative and not just due to operator error. If the positive result was missing, then you know it's not really a negative result, but something else in your procedure probably went wrong. All right, we have some homework left. All right, so tonight, and this is another one of my favorite questions. In fact, I start my kids off at their very first day of school every year on day one. We do this. We learn how to graph, and we learn how to place error bars and just get that right from day one. So you have a table, you have bees involved in this, okay, and you have caffeine, and you're going to look at putting error bars plus or minus 2 standard error to mean, and you have 10 minutes and 24 hours, okay, that they had an average probability. All right, so in flowering plants, pollination is a process that leads to fertilization of an egg and the production of seeds. Some flowers attract pollinators such as bees, usual visual and chemical cues. When a bee visits a flower, in addition to transferring pollen, the bee can take nectar from the flower and use it to make honey for the colony. Nectar contains sugar, but certain plants also produce caffeine in the nectar. Caffeine is a bitter tasting compound that can be toxic to insects at high concentration. To investigate the role of caffeine in nectar, a group of researchers studied the effect of 0.1 millimole caffeine on bee behavior. The results of an experiment to test the effect of caffeine on bees' memory of a nectar source are shown in table 1. Design an experiment using artificial flowers to investigate potential negative effects of increasing caffeine concentration in nectar on the number of floral visits by the bees. I'm going to give you the road to not go down, okay? I don't want you to talk about the bees overdosing on caffeine, okay? I don't want them dying. I just want you to increase those caffeine concentrations and have a nice experimental design. All right, identify the null hypothesis and appropriate control treatment and the predicted results that can be used to reject the null hypothesis. Now, I added this to this because we've got to get this skill down. I want you to make a graph also, okay? And tomorrow night when the next lesson, when Ms. Kate starts her lesson, it will start off with the answer to this homework, okay? Enjoy that question. All right, device and internet access. We know that not all students have access to the internet or a device. We're working on solutions to help students get what they need to show their best work. If you need mobile tools or connectivity or know someone who does, you can reach us directly to let us know. cv.org/tech. And I want to give a shout out to Margaret from Alaska and to Mila. Thank you for your questions this week. Thanks for watching. We'll hope you'll join us again. Be sure to complete the practice question and review the answer in the next lesson.
Hi, welcome to our 5th AP Biology lesson. Today our lesson will be on new material in Unit 7. On Friday, April 3rd, an announcement will be made providing details about the 2020 AP Biology exam. Beginning on April 7th, we will be presenting lessons that will review the first 6 units in the AP Biology course. All lessons, including these with new material, will include practice with science skills, which are relevant with any content. We will also provide practice with free response questions in each lesson. My name is Allison Kate. I'm affiliated with both UC Berkeley and Redwood High School, and today I'm teaching you from my home in El Cerrito, California. So we're going to start today with a warm-up, and in case you're just joining us, these warm-ups are presented as homework in the previous lesson. So this is homework from yesterday's lesson, topic 7.4, which is about population genetics. And so let's take a look at the question. It's got a data table, which is titled Table 1, the effect of 0.1 millimolar caffeine on memory of bees. And you can see that in the far left column, there is the independent variable or treatment, which is caffeine, and it is compared to a control. And then we see the dependent variable is memory, which is measured by the average probability of revisiting a nectar source, plus or minus 2 standard errors of the mean. And you can see that data was collected at 2 time periods, 10 minutes and 24 hours. So now let's look at the question. And if you're just joining us today, please grab a pen and paper, and you can try to jot down an answer as I'm talking about this, but I will be debriefing it in the next slide. In flowering plants, pollination is a process that leads to the fertilization of an egg and the production of seeds. Some flowers attract pollinators, such as bees, using this visual and chemical cues. When a bee visits a flower, in addition to transferring pollen, the bee can take nectar from the flower and use it to make honey for the colony. Nectar contains sugar, but certain plants also produce caffeine in the nectar. Caffeine is a bitter tasting compound that can be toxic to insects at high concentrations. To investigate the role of caffeine in nectar, a group of researchers studied the effect of 0.1 micromolar caffeine on bee behavior. The results of this experiment to test the effect of caffeine on bees' memory of a nectar source are shown in Table 1. That's what I started out discussing with you. So you are asked in this question to design an experiment using artificial flowers to investigate the potential negative effects of increasing caffeine concentrations in nectar on the number of floral visits by bees. In that experimental design, you are asked to identify the null hypothesis, and appropriate control treatment and the predicted results that could be used to reject the null hypothesis. And also you were asked to make a graph of this data to give you more practice with that skill. So if you did this last night, grab your homework, and if you're just joining us today, try to think about your experimental design. You might not have time to actually do the graph. You can go back and do that later. All right, so the scoring guidelines for this question show that your experimental design piece was worth 3 points, and each row would have earned you 1 point. So the null hypothesis in this particular case is that increasing caffeine concentration has no effect on the number of floral visits by bees. The control would be nectar or flowers with no caffeine. And the predicted results, you could have earned a point with either of those bullet points. The number of floral visits by bees is different at increasing caffeine concentrations, or the number of floral visits by bees is different than the control. So each one of those parts of the experimental design was worth 1 point. Now let's take a look at the graph. I hope you did it. If you didn't, you should practice this because this is an important skill that can be applied to any content throughout the course. I have 2 images here because you could have earned full credit for either one of these ways of setting this graph up. So let's take a look at the graph on the left first. You can see that the dependent variable on the y-axis is memory, or the average probability of revisiting the nectar source. And then the x-axis is the independent variable, and it is the treatment. And the first student showed both the control and the caffeine next to each other at 10 minutes, and then the control and the caffeine treatment at 24 hours. You can also see that the student correctly used the plus or minus 2 standard errors of the mean by showing those error bars. And there's a lovely title on this graph as well, the effect of caffeine on memory of bees. So that's a full point question, which is 3 points for the graph. So just a little variation that still would have earned full credit. If you look at the graph on the right, we have the same dependent variable, probability of revisiting the nectar source. This student chose to display the data a little bit differently. We still have the time at 10 minutes and 24 hours, but the treatments are shown as either shaded or open. So the shaded is the control and the open is the caffeine. And there's a key to indicate which is which. And again, this student also has error bars done correctly and the title. So both of these graphs were full credit graphs. All right, so today's lesson overview. First thing, don't forget your pen and paper. They're going to be used today, and today you need a calculator. So you can use your phone or you can get your calculator out. The concept today is Hardy-Weinberg equilibrium, and the science practice skill is 5AA, which is performing mathematical equations, including mathematical equations in the curriculum. And we're going to gain proficiency by looking at sample multiple choice questions requiring the use of the Hardy-Weinberg equation. We're also going to look at FRQ practice questions that combine the content and skill. So as we embark on this lesson today, you should be familiar with the concepts of population genetics. My colleague Ms. Weiser presented a lesson on that yesterday. You should also be familiar with the terms allele, genotype, phenotype, and frequency. And by the end of this lesson, you will be able to do the following. You should be able to describe the conditions under which allele and genotype frequencies will change in populations. You should be able to explain the impacts on the population if any of the conditions of the Hardy-Weinberg are not met, and you should be able to perform mathematical calculations using the Hardy-Weinberg equation. So here's what you need to know. This is just a pinpoint where we are in the large AP biology curriculum to our lesson today. So we are currently working in Unit 7, which is about natural selection. The units are broken into small topics. Each topic has a learning objective, and this is what the students need to be able to do with your content knowledge. And the essential knowledge is the knowledge required to perform the learning objective. So today we are working on topic 7.5, which is the Hardy-Weinberg equilibrium. There are 2 learning objectives that we're working on. The first one is to describe the conditions under which allele and genotype frequencies will change in population, and the second is to explain the impacts on the population if any of the conditions of Hardy-Weinberg are not met. The essential knowledge, which is the knowledge required to be able to do those learning objectives, is that first, the Hardy-Weinberg is a model for describing and predicting allele frequencies in a non-evolving population. And there are 5 conditions for population to be in Hardy-Weinberg equilibrium. So the last thing today to remember is that today's topic skill pair is Hardy-Weinberg equilibrium and forming mathematical equations. All right, so a little background about the Hardy-Weinberg model. Hardy and Weinberg were 2 different people. Hardy was an English mathematician and Weinberg was a German physician. And independently, at the same time, they both worked out the effects of random mating in successive generations on the frequencies of alleles in population. So we give both of these men credit for coming up with a model that describes these effects. This is important for biologists because it's the basis of a hypothetical stability from which real change can be measured. So it is the null hypothesis. It is when a population is not evolving so that we can use it to compare an evolving population to. So this model also allows us to figure out the frequency of genotypes from phenotypes and to track any changes in allele frequency, which means evolution is occurring. So the Hardy-Weinberg model again describes a hypothetical situation. It serves as the null hypothesis, which in this case is a non-evolving population. The model is for when all agents of evolutionary change are absent. So I'm going to go through these, and then the next slide, I'm going to present what those agents of evolutionary change are, so you can see it from both sides. So in the model, there can be no genetic drift, and that means no small population, so you have to have a large population size. There can be no gene flow, which means no migration of alleles into or out of the population. There can be no mutations, which are changes to the DNA. Mating must be random. There can be no sexual selection, no mate preference, and there can be no natural selection, which means that there's equal survival rates. And again, this is a model. This seldom occurs in natural populations, which is why we can compare what happens in natural populations to this model. So here are the 5 agents of evolutionary change. So let's take a look at each one of these. The first one is mutation. And mutation is simply changes to the DNA. The second one is gene flow, which is migration. The third one is non-random mating. There can be no sexual selection. Genetic drift means that you can not have small populations. And then selection, which means that there is differential survival of individuals in the population. So putting it all together, remember, the 5 conditions required for Hardy-Weinberg equilibrium to hold true are a large population, in other words, no genetic drift, isolation from other populations, no gene flow, no net mutations, mating must be random, and no natural selection. Remember these, okay? You just have to remember these 5 conditions, and you have to remember that these conditions are seldom met in natural populations. So let's take a look at the math. We're going to take a look at the Hardy-Weinberg equations, and these equations are from the AP Biology equations and formula sheet found in Appendix 5.1 in the AP Biology course and exam description. This is also available to you on exam day. So there are 2 equations that I'd like you to get familiar with. We're going to practice with these in the next several slides. The first equation says p squared plus 2pq plus q squared is equal to 1. Okay, so p represents the frequency of one allele in population, and q represents the frequency of the second allele in population. The first equation, p squared plus 2pq plus q squared equals 1, is used to determine genotype frequencies. And the reason we have equals 1 is that all of those genotypes have to add up to all of the genotypes in the population. The second equation is p plus q equals 1. This is a really useful equation. These represent the allele frequencies, and all of the alleles have to add up to 1. So kind of the cool thing about Hardy-Weinberg is you can always check your math. If you get some weird number and it doesn't add up to 1, you know you've done your calculation incorrectly. So a couple things to remember about the components of this equation. To calculate allele frequency, you use p plus q equals 1. P represents the dominant allele, and that's usually usually symbolized with a capital letter. Q represents the recessive allele, and that's symbolized with a lowercase letter. To calculate the genotype frequency, you use the p squared plus 2pq plus q squared equation, where p squared represents the homozygous dominant genotype, and 2pq represents the heterozygous genotype, and q squared represents the homozygous recessive genotype. Don't worry, we're going to practice with this. So let's take a look at how the Hardy-Weinberg equilibrium, how Hardy and Weinberg actually thought about this. So there are two images. Let's look at the image on the left, and we're looking at some flowers, and we're looking at phenotypes. And you may recall from genetics that there are two ways to have the dominant phenotype. The first way is to be homozygous, right? So you're homozygous dominant, which is displayed or represented by capital A, and you can also have the same phenotype if you're heterozygous, because as long as you have one dominant allele, you express that phenotype. So there are two ways to be a pink flower. The white flower is the homozygous recessive. So let's say that we have got a population of plants where the total number of plants in this population is 500. And we know that 320 of them are homozygous dominant. Maybe we did a test cross to figure that out. And 160 are heterozygous, and 20 are homozygous recessive. So if you have the total number of plants and you know the genotype frequencies, you can figure out the frequency of each genotype. I'm sorry, I said that wrong. Let me check that. If you know the total number of plants and you know the total number of a particular genotype, then you can determine the frequency of each of those genotypes. So in the first case, the homozygous dominant genotype would be 320 out of 500, which is 0.64. Now you can look at the heterozygote. The heterozygote is 160 out of 500, or a frequency of 0.32. And the homozygous recessive is 20 out of 500, or a frequency of 0.04. If you look at the number of alleles, you have to remember that each individual has two alleles. And so what happens is when you're figuring this out, and you'll see this when we get to the heterozygote, you have to multiply the number of alleles by 2. So we had 320, multiply that by 2, and that's 640 dominant alleles out of the total number of alleles in the gene pool, which is 500 times 2, or 1,000. And so now we get the allele frequency of the dominant allele is 0.8, and that is represented by P, Hardy-Weinberg language. Now let's look at the heterozygote. Remember, the heterozygote has half, 160 of the dominant allele, and half, or 160 of the recessive allele. So that is where we get the 800. We had 640 from the dominant genotype, and then 160 from the heterozygote. We look at the recessive allele, little a, which is represented by Q in Hardy-Weinberg language. We have 160 of those recessive alleles from the heterozygote, and 40 from the homozygous recessive. Add those together, and that's 200 out of the total number of alleles, 1,000, and you get your frequency of 0.2. And again, the beauty of Hardy-Weinberg is you can check your math. 0.8 plus 0.2 equals 1. That's all the alleles in the population. The image on the right just shows why that 2pq comes about. If you take the gametes from the parent, you see the egg and sperm, and you can see that there's two ways to get that heterozygous genotype. There's little a, big A, and there's big A, little a. So that's where the 2pq comes from. Okay, so things to know about the Hardy-Weinberg equation. The convention when solving Hardy-Weinberg problems is to use decimals to show the frequencies. You may see coming up some questions that ask you to determine the percent of a genotype or an individual. And since we're using decimals, all you would need to do is multiply by 100 to get that percent. And we'll get some practice with that in a minute. Knowing that p plus q equals 1 allows you to determine the other allele value if you know 1. For example, if p is 0.7, then q is 1 minus p, or 1 minus 0.7, q equals 3. Add them up, 0.7 plus 0.3 equals 1. And my personal favorite is when you begin to solve a Hardy-Weinberg problem, you always want to start with solving for q or q squared, because you know the homozygous recessive phenotype. If you know the phenotype, you know it has to be the genotype, because with those homozygous recessives, what you see is what you get. All right, so let's get some practice. I'm going to go through a couple of problems, and then I'm going to let you practice on your own. Okay, so the key takeaway for this first one is that p and q represent the allele frequencies of the dominant and recessive alleles. Let's look at a practice problem. The allele, capital W for white wool, is dominant over the allele, lowercase w for black wool. In a sample of 900 sheep, 891 are white and 9 are black. Calculate the allele frequencies in this sample. So you always want to begin with what you know, and then you need to figure out what you're solving for. So they're asking us to calculate allele frequencies. So we know that there are 891 out of 900 white sheep. Remember, we don't know if they're homozygous dominant white or heterozygous white, so we don't want to start there. But we also know that 9 out of 900 are black sheep. And since black is recessive, we know that 9 out of 900, or 0.01, is the homozygous recessive genotype. So that's q squared. So if q squared is 9 out of 900, then q has to be the square root of that, or the square root of 0.01. So q equals 0.1. Now that we know q, we can easily determine p, because p equals 1 minus q. So p is 0.9. So here's our answers. The frequency of the dominant allele p is 0.9, and the frequency of the recessive allele q is 0.1. And again, you can check your answer by adding those together, and they equal 1, or all of the alleles in the sheep population. So let's do the same thing, but this time let's look at genotype frequencies. Your key takeaway is that p squared, 2pq, and q squared represent the genotype frequencies of the homozygous dominant, heterozygous, and homozygous recessive genotypes. So let's do one more together. In a given Hardy-Weinberg equilibrium with allele frequencies where the dominant allele, capital A, is 0.6, and the recessive allele, lowercase a, is 0.4, determine the frequencies of the three genotypes. So again, begin with what you know. Since we're solving for the genotype frequencies, we must use the correct formula, which is p squared plus 2pq plus q squared equals 1. Since we're given the frequency of both of those alleles, we know that the dominant allele p is 0.6 and the recessive allele q is 0.4. We just need to plug them into the equation. So for p squared, it's 0.6 squared. For 2pq, it's 2 times 0.6 times 0.4. And for q squared, it's 0.4 squared. So our genotype frequencies are 0.36 for homozygous dominant, 0.48 for the heterozygous, and 0.16 for the homozygous recessive. And again, if you add those frequencies together, you get 1, or all of the genotypes in the population. So now you're going to try it, all right? Get that calculator, get your pen and paper handy. In a population that's in Hardy-Weinberg equilibrium, the frequency of the Z allele is 0.6. What percentage of the population is heterozygous at this locus? So first of all, you're solving for the heterozygous genotype. You're given that one allele is 0.6, and this is a percentage problem. So whatever your final answer is in decimal form, you need to convert to percentage by multiplying by 100. Okay, so take a minute and solve this problem. Don't worry, I'll go over the answer with you. Okay, I know that that's not a lot of time, but let's move on. So you know that one allele, Z equals 0.6. So that means you know the other allele because p plus q equals 1. So your allele frequencies are 0.6, which was given, and 0.4, which was easy to determine. Probably didn't even need to use your calculator for that one. And now you're asked to solve for the heterozygous genotype. So you're plugging in 0.6 and 0.4 into the 2pq part of the genotype frequency equation. So that 2 times 0.6 times 0.4 is 0.48. And because the question asks for percentage, you simply multiply 0.48 by 100, and your answer is 48% of the population is heterozygous. Hope you got that answer. All right, let's do a couple more practice questions in just a minute. I wanted to bring you back into the classroom because if you were in the classroom, your teacher may choose to do a lab investigation on this topic. And I'm going to start with the AP Bio lab investigation to you from the College Board lab manual. And that is a mathematical modeling activity in which you use Excel to model Hardy-Weinberg allele frequencies. In this case, you can build your own Excel spreadsheet and then play with the variables to look at how those frequencies change over time. Or there are some already built spreadsheets that allow you to change the parameters and see how those allele frequencies change. Another one is called fishy frequencies. And this one is done using goldfish crackers and a prey-predator simulation to model the null hypothesis, which is no selection, and then evolution, which is selection against one of the goldfish phenotypes. And the fun thing about this activity is you are the predator, so you get to select against one of those goldfish cracker phenotypes. This can also be done with any two different phenotypes. So you could use animal crackers, or you could use M&Ms of two different colors. It's fun because you get to eat your data, which is always always a good thing. And then finally, Raw Pocket Mouse has an activity that uses authentic data. And let me just remind you that in our very first lesson, we showed a little video clip of the Raw Pocket Mouse. And this is a modern day example of how protective coloration is a selective advantage. And this investigation uses that actual data that was collected by Dr. Michael Nachman to determine the frequencies of light and dark pocket mice in a population over time. And this is done right in the southwestern desert of the United States. One little personal note, turns out that Dr. Nachman and I were classmates back in the Paleozoic era at UC Berkeley. We took a vertebrate zoology course together. There was a recent reunion, so I was kind of a fangirl and got my picture taken with him. So that was pretty cool. All right, so any of these three are labs that you could do to investigate the Hardy-Weinberg equation. All right, so let's do a check for understanding. We're going to really focus on the skill of performing mathematical equations, including the mathematical equations in the curriculum. And I just can't stress this enough. If I were in my classroom with my students, I would just tell them that the key to Hardy-Weinberg problems is to practice, practice, practice. So let's practice some more so we can master this skill. This question tells you that 9% of an African population is born with a severe form of sickle cell anemia. So this is the homozygous recessive genotype, or little s, which is also known as Q squared in Hardy-Weinberg language. The question is asking what percentage of the population will be resistant to malaria because they are heterozygous, big S, little s, for the sickle cell gene. And this is something that you'll see over and over in AP Bio. This example of sickle cell anemia comes up in a variety of content topics. But in this case, the folks who are heterozygous for this particular allele do have protection from malaria. So in solving a Hardy-Weinberg problem, you know that 9% represents Q squared. It means that Q squared is 9 out of 100 because it's percent, or 0.09. So Q is the square root of 0.09, or Q is equal to 0.3. Once you know Q, you can solve for P. Again, you probably don't need your calculators. P is 1 minus Q, or 0.7. And now we just plug those values, P and Q, back into our equation for the heterozygous genotype, which is 2PQ. So 2 times 0.7 times 0.3 is 0.42. Now here's what we gotta watch out for. Remember that the question asked you what percentage. So you don't want to leave that in decimal form. You want to convert that to percent by multiplying by 100. So your answer is 42%. All right, now you get to try it. And I have 3 slides for you. They're all multiple choice Hardy-Weinberg problems. You need that calculator, and I want you to try these on your own. Don't worry, I'll provide you with the correct answer so you can check your work. And I'll also not just tell you it's answer choice B, but I'm going to show you how that works. Are you ready? All right, so here's your first practice. A particular gene has 2 alleles, a dominant allele, capital A, and a recessive allele, lowercase a. The frequency of the dominant allele is 0.55. If the population is in Hardy-Weinberg equilibrium with respect to the gene, then what is the expected expected frequency of genotype big A, little a? Okay, so you're solving for 2PQ. Got it? Okay, in this case, the correct answer was D. You all got that, right? And here's how we came up with that correct answer. We are solving for big A, little a, or 2PQ. We know that for big A, or P, is equal to 0.55. They gave that to us in the question. So Q has to be 0.45 because we know that P plus Q equals 1. So now we just put those values in to the 2PQ part of the equation and solve the question. 2 times 0.55 times 0.45 equals 0.495. And again, it's kind of a weird number. Don't let that throw you off because those values add up to 1. Okay, ready for the next one? Might be a little bit hard. All right, so Gaucher disease, or type 1, or GD1, is a recessive genetic disease that affects 1 in 900 individuals in a particular population. GD1 is caused by mutation in the enzyme glucocerosidase. I'm sure I didn't say that right. Assuming the population is in Hardy-Weinberg equilibrium, calculate the frequency of the wild type or non-mutant allele for the enzyme. I'm going to try it again. Glucocerebrodase. Awesome. Okay, so now that I've massacred the enzyme name, you are calculating the frequency of the non-mutant allele, and you are given the recessive genetic disease affects 1 in 900 individuals. All right, go. If you need again, think about what they're giving you in the question. Okay, so that one's a little harder, not much, but let's take a look at the answer. So in this case, the correct answer is C. They gave us Q squared in the question. So remember how my advice was to always start with Q or Q squared? They gave us Q squared. So we know that 1 out of 900, or 0.00111, is equal to Q squared because GD1 is a recessive genetic disease. The question is asking us to solve for the P, which is the non-mutant allele. And we start by finding Q, which is the square root of Q squared, right? So it's the square root of 0.00111, or 0.033, and now we can solve for P because we know that P plus Q equals 1. So P equals 0.967, or answer choice C. All right, one more. Okay, so humans vary in their ability to taste the bitter chemical compound phenothiocarbamide, PTC. The taster phenotype is dominant to the non-taster phenotype. Researchers tested the PTC taster phenotype of individuals from an isolated population. In the sample, 780 individuals were able to taste PTC, and 70 individuals were not able to taste PTC. Assuming the population is in Hardy-Weinberg equilibrium, what is the frequency of the non-taster allele? Again, think about what they're giving you, think about what they're asking you to solve for, do your math, and come up with an answer choice. Okay, so this question asked us to solve for the non-taster allele, so we're solving for Q. We also know that 70 out of the total number of individuals, which is 850, so 780 plus 70 is 850, are equal to Q squared. So Q squared is equal to 0.082. So to get this answer, all we have to do is take the square root of 0.082, and that is 0.286 rounded up to 0.29, and that's answer choice B. So once you get the hang of these, they're pretty fun. I think they're pretty fun. Practice with Hardy-Weinberg problems. All right, so our debrief and summary, you should now be able to describe the conditions under which allele and genotype frequencies will change populations. You should be able to explain the impacts on the population if any of these conditions of Hardy-Weinberg are not met. You should be able to perform mathematical calculations using the Hardy-Weinberg equation. Your key takeaways is that evolution is characterized by a change in the genetic makeup of a population over time and is supported by multiple lines of evidence. In this case, evolution is supported by a change in the allele frequency. Hardy-Weinberg is a model for describing and predicting allele frequencies in a non-evolving population. So it is only a model that we can compare natural populations to see if they are evolving. So let's talk about some common errors and misunderstandings, and also just some reminders for how to use this and how to solve Hardy-Weinberg problems. Each individual has 2 alleles for any given gene, 1 from each parent, so that the total number of alleles in a population is the number of individuals times 2. We had some practice with that in the last practice, multiple choice practice question. And also if you go back to the graphic I showed you of how this was derived with the flowers, it really illustrates that nicely. You cannot calculate genotype or allele frequencies from phenotype alone unless you know the phenotype of the homozygous recessive. In this case, what you see, the phenotype is the genotype, and often they will give you that in the problem. If you're given the number of individuals with particular phenotypes, you can only be sure of the genotype of the individuals with the recessive phenotype. So start by figuring out Q squared and Q before anything else. You cannot start knowing the phenotype of the dominant homozygous dominant because you don't know if it's homozygous dominant or if it's heterozygous, and I hope that makes sense. So you cannot use Q squared or any other, like the P squared or the 2PQ, to determine the allele frequencies. You have to count the alleles to get allele frequencies. So I'm going to leave you with these. You need to know and tell the difference, don't mix them up, between allele and genotype frequencies. In other words, mind your P's and Q's. Single letters such as Q, P, R, etc. represent allele frequencies. And remember, there could be more than 2 alleles for a particular gene. The allele frequency then is equal to the number of a given allele divided by the total number of alleles in a population. So that's allele frequency. Any letter that is not a single letter, like p squared, q squared, r squared, 2pq, represents the genotype frequency, which is equal to the number of individuals with this phenotype divided by the total number of individuals. You can only count the alleles if you know the number of individuals with a given genotype. So that's a lot of try to remember, mind your P's and Q's, must knows. Again, once you start solving a lot of these problems in practice, these will make a lot of sense. Okay, so let's give you some AP exam practice. This is your homework for tonight, and Ms. Pfizer will go over this with you as your warm-up tomorrow. So this is from a FRQ from a previous exam, and it reads, evolution is one of the unifying themes of biology. Evolution involves change in the frequencies of alleles in population. For a particular genetic locus in a population, the frequency of the recessive allele, A, is 0.4, and the frequency of the dominant allele is 0.6. You're asked to do 3 separate things. In part A, what is the frequency of each genotype? So you need to calculate the genotype frequencies for big A, little a, and little a in the population, and then you need to calculate the frequency of the dominant phenotype. Part B, how can the Hardy-Weinberg principle of genetic equilibrium be used to determine whether this population is evolving? And in part C, you're asked to identify a particular environmental change and describe how it might alter allele frequencies in this population. And I'll give you a hint. Think about the conditions that have to be present for Hardy-Weinberg to hold true. And I would set the timer for about 20 minutes as you work through this problem. Remember, if you were given this problem on the AP exam, you would have the formula sheet and you would have a calculator. So that takes that stress away. All right. So this is your homework for tonight, and it will be debriefed tomorrow as your warm-up for the next lesson. One more thing before I say goodbye today. We know that not all students have access to the internet or to a device. We're working on solutions to help students get what they need to show their best work. If you need mobile tools or connectivity or know someone who does, you can reach out to us directly and let us know. And you can do that at cb.org/tech. I'd like to thank you for joining me today. I hope you'll join us for all of our lessons. Be sure to complete that practice question because we're going to review it in the next lesson. Have a great rest of your day.
Welcome to AP Biology. I'm Ms. Pfizer from Darnell-Cookman Middle High School and Florida State College in Jacksonville, Florida. Today, the topic we are covering is one that colleges have asked that all AP students study, even if it will not be on the exam. Colleges are supporting AP students this year by allowing this more flexible approach to the exam, where we'll focus on a lesser amount of content than in other years. But in turn, they have asked that AP students learn today's material to make this relevant to your upcoming exam. We'll also include some exam prep in today's lesson. Soon, after we've covered some of these key outstanding topics, we'll be posting the full schedule of review topics for the remainder of this course so that you can choose which topics you'll want to prioritize for review and thus which classes you'll want to attend. So now for an overview of our lesson today. Today, I'll be introducing you to Evidence of Evolution, topic 7.6, Common Ancestry, topic 7.7, and Continuing Evolution, which is topic 7.8. You should already be familiar with mechanisms of evolution, natural selection, mutations, genetic drift, and gene flow, and describing, explaining, identifying, constructing hypotheses, and predicting. By the end of this lesson, you'll also be able to explain how morphological, biochemical, and geological data provide evidence that organisms have changed over time. Describe the fundamental molecular and cellular features shared across all domains of life, which provide evidence of common ancestry. Explain how evolution is an ongoing process in living organisms, and use the following science skills: argumentation, questions and methods, and representing and describing data. I hope you have a pen and pencil and paper and calculator, and if you have graphic paper, that would be great. All right, so for the warm-up. The warm-up is the homework from topic 7.5, which was the previous lesson to tonight, since we're 7.6, 7, and 8. I'll go ahead and, if you're just joining us for the first time, I'm going to read it, and then I'll give you a minute, and hopefully those that watch the last lesson have their homework ready. All right, evolution is one of the unifying themes of biology. Evolution involves change in the frequencies of alleles in a population. For a particular genetic locus in a population, the frequency of the recessive allele, little a, is 0.4, and the frequency of the dominant allele, big A, is 0.6. So A is going to ask us 2 questions. What is the frequency of each genotype, big A, little a, in this population? And what is the frequency of the dominant phenotype? B, how can the Hardy-Weinberg principle of genetic equilibrium be used to determine whether this population is evolving? And C, identify a particular environmental change and describe how it might alter allelic frequencies in this population. I'm going to give you a moment if you're new to our videos, and if you have your homework ready, you can go ahead and start reviewing that, and I'll show you the answer in just a moment. Okay, let's make it 45 seconds. That's 10 more seconds. Okay, now I'm going to show you my homework because Ms. Kate assigned you homework, and I do homework with my students all the time. When they write FRQs, I sit down with them and I write those FRQs mainly because I enjoy it, but I want to be in the moment with them just as much as possible. So this is my homework. I started by reading the questions, and as I'm reading the questions, I'm writing down information that will help me stay organized in my thoughts along the way. So I took the text told me that the recessive allele, the little a, okay, was little a, and so I know that is my Q, which is 0.4, and then my dominant allele is big A, and my P was 0.6. I know my equations, P plus Q equals 1, and then I also know that P squared plus 2PQ plus Q squared equals 1. So if I just substitute in those values, I will have for P squared, I have 0.6 squared plus 2 times 0.4 times 0.6 for my 2PQ, and then plus 0.04 squared for my Q squared. Now, what I get is a frequency of 0.36 for big A, 0.48 for big A, little a, and 0.16 for little a. Now, if I was dealing with a population of 1,000 people, that would mean that 36% of the population was big A, so that would be 360 people. And if it was for big A, 480 of those 1,000 would be heterozygous, and then 160 of them would be recessive. Now, the second part of that question asks for a dominant phenotype. So I have 0.36 is my big A, and 0.48 is my big A, so I have a 0.84, which is 84% of the population would have the dominant phenotype. Now, I wrote a very simple answer for B and C, and I'm not going to take you through the rubric on this. I'm going to show it to you and show where it is, so you can go back and look at it. It's very busy, and it's an older style, and I don't want to spend 2 minutes on it, but I put it in so you can look at it later and you'll get a glance at it in a second. So B, I said if the allelic frequency changes, then evolution has occurred. And for C, if environmental selection pressures. And see, when I first wrote it, I did not put down words that I wanted to put in there, and so I came back and I kind of squeezing things in, and that's okay. So if environmental selection pressures change against a recessive allele, then that would mean that natural selection was occurring. So this is the rubric. See what I was talking about? Very, very busy. And so if you want to go, our numbers are correct, and if you want to go down and read all the other options and strategies for B and C, you can do that at your leisure. All right, the framework of the AP Biology course. We're in the evolution unit. We're in natural selection. The unit is actually called natural selection. Our topic is evidence of evolution, common ancestry, and continuing evolution. Each topic has questions called the learning objectives. That is what we need to be able to do, and we need to know content in order to answer these questions, and that's called our essential knowledge. And each topic also has an associated science skill. This is how we're going to show that we know the answer to the question, which is the learning objective. The science skills that are associated with our topics are representing and describing data, argumentation, and questions and methods. And today we'll focus on a topic skill pair of evidence of evolution and representing and describing data. So what do you need to know to do this? So some examples of types of data that provide evidence that organisms have changed over time are geological, morphological, and biochemical. Here's some geological evidence. It can provide evidence for evolution. And okay, so radioactive decay can be measured to determine the age of rocks. The length of time required for the atom to change into another stable element is called its half-life. And iron interacting with oxygen can indicate where levels of oxygen were or are available. And cyanobacteria produced the earliest fossil evidence in stromatolites and 3.5 billion years ago. And if you haven't seen stromatolites, these are stromatolites. All right, fossils. The age of fossils can be determined by the age of rocks where a fossil is found, which are called index fossils. I wore my dinosaur dupe necklace for this occasion. It's called copperlite, and I have I have a big rock in my classroom, which I can't get to right now, but I have actually a fossil of dinosaur waste, and then I have this polished version on a necklace. All right, so homologies. Similar morphology, shared by common ancestor. So homologous means the same set of bones organized in a similar way. So if you look at this human arm, okay, you have your humerus, you have your radius, your ulna, your carpals, your phalanges, and if you look at this dog, bird, and whale, there's the same organization pattern in those bones, and they function in a similar way. All right, vestigial structures. So vestigial means reduced or obsolete function, and some examples are bone structure for legs and some snakes, and the hip bone structures in whales. And in this particular diagram, you'll see that it says it's a mutant male, and he has vestigial wings. Well, those wings will not allow him to fly, yet he still has wings. Okay, fundamental molecular and cellular features are shared, conserved across all domains of life, and provide evidence of ancestry. Structural and functional evidence supports the relatedness of organisms in all domains. Organisms use the same biomolecules. Organisms have the same DNA codes for the same 20 amino acids, and conserved protein sequences of amino acids have a distinct function in different organisms. So I'm going to help you process some information that's actually going to help you tomorrow or the next lesson, which is 7.9, and it has to do with phylogeny and phylogenetic trees. And so this is a table that kind of will help you grow towards being able to do that. So here we have some evidence from data supporting evolution. And this is biochemical because we're talking about amino acid differences. So this is a table, and if you look, there's in the first column, there's a species 1, 2, 3, 4, and 5, and then across in that row, there's species 1, 2, 3, 4, and 5. Now you can either work this from a row with the most information in it, or a column. I'm a column person, so I'm going to tell you how I process this information. All right, so I look at species 5, and I can see that it is 5 amino acids different than species 1. Okay, that's 5. And then the next value is 20, and I can see that I am 20 amino acid differences away from 2, and 10 away from 3, and 12 away from 4. So I saw something right here that these 2, 3, and 4 are almost equal in their distance away from species 5. So here we have, if I'm going to give you a second, I want you to think about which one would be the closest relative, and which one would be most closely related, and then which one would be the farthest away, and which 2 go in the middle. And I'll give you just 10 seconds to think about that. Maybe 5. Okay, so the more amino acid differences among the 5 different mammal species means the more time that has passed since their divergence. And the divergence means they're in common ancestor, and mutations happen, and these began their journey to becoming a different species. Okay, so if you look, this would be the closest one. There's only 5 amino acids different between 1 and 5, and between 2 and 5 there's 20. So this one's the farthest away, and 10 and 12 will be somewhere in the middle. And that's from just some data on a table in that organization is what I wanted you to see. All right, here's another representation of this. Has to do with both actually DNA nucleotide sequences and protein amino acid sequences, because it involves both, because you wouldn't have that amino acid sequence without that DNA sequence. So let's look at our first column. We have species 1, 2, 3, and 4, and then at the top it says relative amino acid position. So if this was a polypeptide, this would be amino acid 1, amino acid 2, 3, 4, and so on. So if you look at amino acid 1 in each of the species 1, 2, 3, 4, they all have valine. Okay, if you look at amino acid position 2, everything is histidine, and then 3 leucine, 4 xaline. Oh, and but there's something different. There's one amino. Keep that in mind. There's one amino acid different there in species 2, and then we have glutamic acid, and then arginine, glutamic acid, tryptophan, histidine, alanine, valine, cysteine. Something's going on in 4 that's totally different than everything else. So let me give you another view. So this is called a sequence alignment. This is a very elementary sequence alignment. You can once you proceed in biology, you'll start using the National Center for Biotechnology, and there's alignment tools that you'll be using, and it's quite interesting. Okay, so let's look at there's valine, histidine, leucine, and then we have that one different amino acid. So what happened there? There was another amino acid coded for, so there probably was a single point mutation that occurred there. Now in position 5, we all of a sudden have arginine, and then we have tryptophan. Everything all of a sudden is not the right amino acid from position 4, and then starting in 5. So what I think happened there must be a frame shift, and we'll talk about that in the review period in the next videos that are coming up in the coming weeks. And then the other one, all of a sudden we dropped 2 amino acids, so all of a sudden it stops before it was supposed to. So then we have nonsense. So if we had to pick one that was closest related, it would be the one with the least amount of change, the least amount of differences in amino acids. So 2 would be a closer relative to 1 than 3 or 4 if we were looking at it from that perspective. Okay, so let's talk about membranes. They are evidence of common ancestry also, and this is, I'm going to talk about endosymbiosis, and I'm also going to tie in endomembrane system at the same time. So here we have a prokaryotic cell. This cell, this is called the cell. This will be cell 1. All right, so we have a prokaryotic cell, and we can see that there's not a nucleus, but we have a circular, one circular chromosome in the middle. Okay, and then over here, in the next, which would be cell 2, we have this invagination of this membrane coming in, and that looks like it's going to be forming a nucleus, a nuclear membrane around, right? And so it eventually will, and then that will also have a connection with the endoplasmic reticulum, vesicles, Golgi apparatus, or vesicles, and the plasma membrane. All those are made out of these same plasma membrane materials. Okay, now if we look at cell 3, we have our nucleus, right? And now we have this engulfing of this prokaryotic cell that has these respiratory function, and then it's coming into the cell. And now the next cell, we have an engulfing of a prokaryotic cell that has photosynthetic function, and now both of them are in the cell, and now they are going to be functioning as a mitochondria and a chloroplast. Okay, now so you can wrap your head around this double membrane stuff that I'm about to talk about. I drew this visual at the bottom. So this was a cell, and it was engulfing this other cell, which is this would be the cell right here, and then this would be the prokaryote. Notice that this plasma membrane of that first cell would be around there, and then this would be the and then this would be the cell, the plasma membrane of the prokaryote cell. So now it has 2 membranes, a double membrane. All right, so evidence supporting the theory of endosymbiosis: double membrane of mitochondria and chloroplast. Mitochondria and chloroplast size are the same as prokaryotes. Mitochondria and chloroplast have their own DNA, and mitochondria and chloroplast replicate through binary fission. All right, evolution is an ongoing process in all living organisms. Population of organisms continue to evolve. An example would be the finch beaks of the Galapagos Islands. Genomic changes occur over time. There is continuous change in the fossil record. Organism populations evolve and gain resistance to antibiotics, pesticides, herbicides, and chemotherapy drugs. And then pathogens evolve and cause emergent disease. This is currently happening as a virus that causes the illness COVID-19 is mutating. So let's talk about basic antibiotic resistance. So let's say that these are bacteria in your body, the little nuts and little snowflakes, okay? And this particular antibody will kill the bacteria, but it's only killing those that are sensitive to it. There are some bacteria that are resistant to this antibiotic. So then that allows the resistant bacteria to reproduce, and then eventually they can reproduce and over and over and over, and this antibiotic will not kill them. And then they can also get up this resistance to other bacteria through conjugation and transduction. All right, so let's talk about, we're going to just going to have content, and now we're going into our science skill. So we're going to describe data from a table or graph, including identifying specific data points, and some task verbs that we're going to use today: support, describe, explain, calculate. There are a couple more. There are many, and what I'm about to talk about. All right, so here's another table that I wanted to familiarize you. I can't even say that. All right, so we're looking at a table, and it has cell wall composed of cellulose, vascular tissue, roots, seeds enclosed in fruit, and flagellated sperm cells. Now across the top we have green alga, moss, spruce tree, and apple tree. So this is using symbols that we haven't used before. We've been using percents difference or number differences, and now we have plus or minuses. So let's read our text. Students analyzed several photosynthetic eukaryotic organisms in the laboratory to determine their phylogenetic relationships. The organisms were Spirogyra, a green alga, a moss, a spruce tree, and an apple tree. Table 1 compares several characteristics in the organism. A plus indicates the trait is present, and a minus indicates the trait is absent. Okay, let's read the question. Which of the following rows of data listed in table 1 best supports the possibility of a common ancestor for the organisms listed there? And we have choices: A, vascular tissue; B, seeds enclosed in fruit; C, cell wall composed of cellulose; and D, flagellated sperm cells. So which of the following rows of data listed in table 1 best supports the possibility of a common ancestor for the organisms listed there? So which row? These are rows. Okay, go across. I'll give you just a minute to think about it. Okay, and I hope you chose C, cell wall composed of cellulose. If you'll notice, each of these have this trait. So they are, I'll let Ms. Katay talk to you more about this in the next lesson, but they all have this trait, and then only 2 of them have this trait, and 2 of them have that trait. So there's a divergence that's going to happen that they all don't have those traits, okay? All right, more of that to come tomorrow. All right, so now let's look at this data represented in another way. The data below summarizes the number of differences in amino acid sequences of cytochrome C found in selected species. So if we're looking at humans to chimpanzees, there's 0 differences in amino acid sequences. That's what we're talking about in this particular protein. And humans have 1 difference from rhesus monkeys in this particular cytochrome C. And between humans and horses, donkeys are 7. Humans and cows or pigs or sheep are 7. Humans and rabbits 7. Birds and reptiles 10 to 15. Fish 18 to 20. Plants 45 to 48. Okay, interpretation of the data supports which of the following statements? Okay, follow along with me. So rhesus monkeys are more closely related to rabbits than they are to horses. So look at where they are. Here's rhesus monkeys, and there's rabbits. Okay, and there's horses. So are they more closely related to rabbits? No, because they're the same. But that doesn't mean it's the same changes in amino acids. It just means the amount of the same. Okay. All right. Horses and cows have identical amino acid sequences in their cytochrome C. Okay, so the horses and cows both have 7, but we don't know that which 7. It doesn't mean it's the same 7. Humans are more closely related to rabbits than they are rhesus monkeys. So humans to rabbits 7. Humans to rhesus monkeys 1. So humans are more closely related to rabbits. No. All right. Plants and animals have no similarities at all. Yes, they do, because if there are 45 to 48 differences, that means that there are similarities also. And mammals are more closely related to reptiles than they are fish. We have mammals to reptiles. There's only 10 to 15 differences, and mammals to fish there's 18 to 20. So we're more closely related to birds and reptiles than to fish. So the answer to that is E. Okay, so now let's make sure that you understand how to use these tables with this one multiple choice question. You have species 1, 2, 3, 4, 5, and you have columns, and you have crazy looking titles in each where the E's, it's all different. Anyway, hemoglobin is a highly conserved protein used by all mammals to transport oxygen. Table 1 was constructed by comparing amino acid sequence and hemoglobin molecules from 5 different mammal species. Okay, so what do we know how to do? I'm going to give you I'm going to give you a minute to think about it. Oh, let me read it to you. Which of the following best describes the importance of the number of amino acid differences indicated in table 1? A, they reflect the different environments where the organisms were captured. B, they reflect the number of base pair changes in the DNA. C, they reflect random changes due to translation errors. And D, they reflect the number of oxygen molecules each hemoglobin molecule carries. Okay, I hope you chose B. All right, let's look at those answers. They reflect the different environments where organisms were captured. This table doesn't tell you anything about an environment. Okay, so not the answer. They reflect random changes due to translation errors. No, that would be a better choice if it had said maybe replication errors. All right, they reflect the number of oxygen molecules each hemoglobin molecule carries. No, it's not about that at all. It's about amino acid differences. So how do we code for an amino acid? It has to do with the DNA. Okay, it has to do with those 3 letters of the 3 nucleotides that are going to be a codon for that particular amino acid to become part of that polypeptide. All right, so before I flip this, I don't want you to freak out when you see this, because this is what kids do as soon as they see something that looks massive like I'm about to show you. Okay, all right, here we go. Oh my gosh, right? It has writing. There's so much text. I have to read through this. So let's take this in baby steps. I want you to not be afraid of a question that looks like this. So first thing I do is, where's the table? Where's the graph? I've got to have something to look at first. So I'm going to flip this page here. And so here I have a table. It says, and I'm going to assume the role of a student right now. All right, table 1, IC50. I have no idea what that is. Okay, for common antimalarial drugs tested on artesunate sensitive, oh my gosh, another more big words, ARTS and artesunate resistant ARTR plasmodium strains. And then I see there's drugs, amodiaquine, chloroquine, DHA, fluvoquine, and quinine. And then I see we have some data. We have some ARTS and we have some ARTR, and we have the IC50 again, and there's a plus or minus. Okay, something I can recognize, right? You have your SEM. So can I just do this if right now I'm assuming the role of a student that does not know what IC50 is and can't decipher what these ARTs are? I can't do this with just looking at this table. So I'm going to have to go back and read that text. And I'm going to have to, sorry about that, I'm going to have to engage in reading this much text. And as I'm going, I'm writing myself any notes that I need to carry with me so I don't have to reread it. Okay, but read it good, read it, and engage in it, and so you don't have to reread it. All right, malaria is a disease that is caused by the eukaryotic parasite Plasmodium and is transmitted between individuals by the bite of infected mosquitoes. Different species of Plasmodium cause different disease in different vertebrate species, but in all cases, the parasite infects and destroys red blood cells. The repeated emergence of drug-resistant strains of Plasmodium parasite responsible for human infection has caused researchers to continually search for new drugs or combination of drugs that can provide an effective treatment. A drug that has now proven effective in recent years is artesunate, but now artesunate-resistant Plasmodium strains have also appeared. Researchers investigated whether artesunate-resistant Plasmodium strains could be effectively treated by other drugs. All right, so they're looking for something's not working anymore, and they're looking for something else. All right, in a mouse model of malaria, the researchers determined that a particular strain of artesunate-sensitive Plasmodium, and now we can refer that to as ARTS, could be largely eliminated from the mice at a dose of 0.15 milligrams per kilogram by repeated exposure of the artesunate-sensitive strain, the ARTS, to increasing concentrations of artesunate. The researchers developed a Plasmodium strain that is highly resistant to artesunate, ARTR, and requires a minimum dose of 240 milligrams per kilogram to kill the resistant to eliminate parasites. Okay, compared to the 0.15. All right, so the researchers then compared how effectively several common antimalarial drugs eliminate the ARTS and the ARTR strains in vitro. The researchers infected human red blood cells with the ARTS and ARTR Plasmodium strain and incubated the infected cells in tissue culture medium. They added serial dilution of the drugs to replicate cultures and determined the IC50 for each drug, the concentration of the drug that reduced the parasite load in the red blood cells by 50%. Okay, so what I know to take forward to this table with me is that ARTS, that's my sensitive one. ARTR, that is my resistant strain. And then with IC50, that's what I need. That's the dose that it takes to kill half of the resistant strain. Okay, now let's look at the table. And now we can see that we have different values ranging from 11 to 50 in our sensitive strain. And then look at this, ranging from 39 as our lowest point to 216. Oh, but look at that SEM 81. So when you get to the graph, that's going to become important. What those numbers, the range of those numbers. All right, so let's read first what A wants us to do. Plasmodium is an organism with both sexual and asexual life cycle. Describe the most likely cause of heritable phenotypic change in some members of the population of asexually reproducing organisms. Explain how repeated exposure of the artesunate-sensitive Plasmodium strain to increasing concentrations concentration of the artesunate led to the development of the artesunate resistant plasmodium strain. Okay, so there's two things you need to do: describe the likely cause of this phenotypic change, which is heritable from one to another, one offspring to next generation, and explain how then it keeps it's developed, how you go from some resistant to a population of resistant. Okay, I'll give you let's do a minute and a half on that. I'll tell you when you have about 15 seconds left. Okay, let's stop in about 15 seconds. Okay, your describe answer should be similar to this. The response indicates that the most likely cause is random mutation. In parentheses, it says, however, differential gene activation in some organisms under different environmental conditions is also possible. But primarily, most people would have answered the response indicates that most likely cause is random mutation. Okay, so they explain. The response indicates that each increasing concentration of artesunate selected for only those members of the artesunate sensitive plasmodium population that could survive and reproduce, and eventually only the artesunate resistant organisms remain to reproduce, just like in the petri dishes that I showed you a few minutes ago. All right, now we're going to graph the data on that table, and then we're going to answer a question about it. So let's go ahead and let's take the table. Let's do the table first and graph it, and I'm going to go back and bring up the data again, and I'm going to tell you a little bit as you graph that. Okay, so don't just jump in. Well, first of all, if you have graph paper, you possibly have a whole sheet, and just don't jump in and start throwing numbers on there. Put your eyes on this table and look and see what your range is going to be, and go ahead and start thinking about what is the results. Okay, these are results in these two columns. So if these are results in these two columns, those are going to be on your y-axis. Okay, because that's the dependent variable. So when you label, make sure all of this, because this is your results and this is your unit, that it goes with you. Okay, and then you have to also say and, because you got all this on one graph. So put all of this information as your label on your, and don't be lazy about it, and just write it all. Okay, all right, and then you have on your x. So what you're changing, what's the variable here, is that you're changing the drug. Okay, so you can write drugs, and then you can put the amodiaquine, chloroquine, DHA, methylaquine, and quinine all on the x-axis. But when you start scaling, look at this. This is your lowest value, 11.3, and this is your highest value, 216.9 and 81. What is that? 297, 298. You're going to need a range from 0 to 300 to pull this off. So scale correctly. You can't jump around and make different rows or different amounts. Okay, so go ahead and start that. You need that back. Okay, so I'll give you a minute and a half to at least start your graph. You won't be able to do the whole thing because that's going to take some time, but you can come back and look at this video and do it again, because that's what my kids in my classroom will be doing tomorrow morning. You should be graphing. Okay, I'm going to go ahead and stop you, and then we're going to take a look at my graphing. Okay, so here's my scaling. There's my 0 to my 300. That's where I started. And then I took a look at how much room I had on my X, and I know I had 5 drugs involved. You can look really close and you can see my little dots of how I want this graph to be proportional and look nice when I get finished, so it's easy to interpret. All right, so I started, and then I finished with my labeling. See, here's what I'm talking about. Everything is over there, okay, on my Y-axis, and then everything is here on my X. And then because I'm extra sometimes, I actually put a title on. You wouldn't have to do that on an AP exam, but sometimes if this isn't real clear and you set it up here, then it might score you that point. All right, and then I added the data, and I added my error bars. And so I actually, you don't have to do this, but I do this just for clarity. There's my mean values, okay, and then I took my mean plus my 132.4 plus that 16.9. That brought me to 150. Now I'm not going to do the math for each side of this because I have a pretty good eyeball on being even, but you can just subtract 16.9 to get the other side. Now, excuse me. All right, so here's the second part. So we have there's a sensitive resistant of each of the drugs. So based on the data, determine the antimalarial drugs that are likely to be significantly less. So I'm using that significantly less effective word. Okay, at treating disease caused by the ARTR strain than by the ARTS strain. Okay, so now let's look at our error bars. That's going to help us decide which of these 3 are less effective on the resistance. Okay, that's what it's asking for. Take a few seconds to do that. Have you chosen 3? Okay, so we're looking for less effective, and so that would be that. Look at this. Look at this quinine. We have error bars not overlapping, and look at the difference in the two. It takes all of this value to do the job, okay, than it does when they in the sensitive strain. So this is one, and let's see, we have these two. We have some overlapping error bars, so there's nothing significant going on there. Yes, here on the DHA, no on the chloroquine, and yes on the amodiaquine. All right, so when you're graphing, make sure you have your axis labeled, and your scaling has to be right, your units have to be right, and all of your data plotted correctly. All right, and then the labeling, so that should have been on each side, and we did a good job on that. We use the error bars, and we have that amodiaquine, DHA, and quinine have significantly higher IC50s for ARTR than ARTS, and are therefore likely to be less effective at treating disease by the ARTR than the ARTS. All right, so let's go over what we did today. Takeaways for today: geological, morphological, biochemical, and mathematical data provide evidence of evolution. You should also be able to describe fundamental molecular and cellular features shared across all domains of life, which provide evidence of common ancestry. Explain how evolution is an ongoing process in all living organisms. Use the following science skills: argumentation, questions, and methods, and representing and describing data. Common errors and misunderstandings. I kept this brief because I knew I'll be running out of time. Don't lose points with your graphing. That's 3 points that you can. There's no reason to lose those. Don't be lazy with your labeling. Okay, put the units there. Put everything there. Okay, just don't leave anything behind. And then your error bars, you need to be accurate above and below. Your data points in the right place, and scaling. Can't jump around. Can't say all of a sudden this one is 10 and then the next one is 50. Can't do that. All right, but don't leave those 3 points behind. And here's your homework for tonight. According to very short FRQ, and we're going to use the word describe. We're going to talk about endosymbiotic theory. According to the endosymbiotic theory, some organelles are believed to have evolved through a symbiotic relationship between eukaryotic and prokaryotic cells. Describe 3 observations that support the endosymbiotic theory. You can leave this behind you, and you can look at the video later to look at it again, and then start writing that for tomorrow night's warm-up. All right, device and internet access. We know that not all students have access to the internet or device. We're working on solutions to help students get what they need to show their best work. If you need mobile tools or connectivity, or know someone who does, you can reach us directly to let us know at cb.org/tech. And thanks for watching, and I hope you come back for the next lesson, which will be 7.9 Phylogeny. We have this today. Be sure to do your homework. Okay, bye.
Hello, welcome to our 7th AP Biology lesson. Today our lesson will be on new material in Unit 7. On Friday, April 3rd, an announcement will be made providing details about the 2020 AP Biology exam. Beginning on April 7th, we will be presenting lessons that will review the first 6 units in the AP Biology course. All lessons, including these with new material, will include practice with science skills, which are relevant with any content. We will also provide practice with free response questions in each lesson. My name is Allison Kate. I'm affiliated both with UC Berkeley and Redwood High School, and I'm teaching today from my home in El Cerrito, California. So let's get started with our warm-up today. If you're just joining us, the warm-up is a question from the previous lesson. It's given for homework at the end of the previous lesson, which yesterday was lesson 7.8. And in that lesson, one of the content topics was about the endosymbiotic theory. So this question is about that. If you're are just joining us, go ahead and grab a pen and paper and see if you can answer it as I debrief it. And if you did this last night for homework, good for you. And go ahead and get that out as I go over the scoring guidelines. All right, so let's read the question together. According to the endosymbiotic theory, some organelles are believed to have evolved through a symbiotic relationship between eukaryotic and prokaryotic cells. Describe 3 observations that support the endosymbiotic theory. Okay, so you're asked for 3, and you're asked to use that command term or task verb describe. So what you're looking at on the scoring guidelines is a lot of bullet points. The ones on the left were the bullet points that were most often used to answer this question. The ones on the right also appear, but least often or less often. You need to come up with 3 descriptions, and so each bullet point would have earned you 1 point. Remember, we're trying to provide evidence for endosymbiosis. So either mitochondria or chloroplasts containing their own DNA, like prokaryotes do, would have earned a point. Mitochondria and chloroplasts can both self-replicate, like prokaryotes. Either one of those would have earned you a point. And mitochondrial chromosomes and chloroplast chromosomes are circular, like prokaryotic chromosomes. Finally, one way most often response was that mitochondrial chromosomes lack histones, as do prokaryotic chromosomes. And then I'm not going to read through the other long list, but you can take a look at those and see if you came up with any of those. So that was a 3-point question. I hope you did really well on that one. All right, so today's lesson overview. Today we will be introducing you to the concept of phylogeny and to science practice scale 2DC, which if you don't have your CED in front of you, deals with visual representations. And so when we look at phylogeny, we're going to see that both phylogenetic trees and cladograms are models of the history of a species that represent this visually. So we're going to look at diagrams of both. We're also going to look at sample multiple choice and free response practice questions that combine the content of phylogeny and the skill of explaining relationships in models that are represented visually. You should already be familiar with the concepts of species and speciation and the terms phylogenetic tree and cladogram. And if you haven't been exposed to those before, you will hear them a lot in this lesson today. By the end of this lesson, you'll be able to describe the types of evidence that can be used to infer an evolutionary relationship. You'll be able to explain how a phylogenetic tree and cladogram can be used to infer evolutionary relatedness, and you'll be able to explain the relationship between different characteristics of biological concepts, processes, or models that are represented visually. So don't forget your pen and paper, and let's get started. I want to tell you where we are today in terms of the overall framework of the biology course. Again, today we're working on topic 7.9, which is in the natural selection unit. We're working on the idea of phylogeny, and there are 2 learning objectives which I read to you out loud in the previous slide. You can see there. And our essential knowledge for today's lesson is that phylogenetic trees and cladograms show evolutionary relationships among lineages. So today our topic scale pair is phylogeny and explain relationships that are represented visually. So here's what you need to know. First of all, what is phylogeny? Phylogeny is the evolutionary history of a species, and the way that we represent those relationships visually is either using a phylogenetic tree or a cladogram. And we'll talk about the differences between those. So phylogenetic trees will show the amount of change over time that is calibrated by both fossil evidence or a molecular clock. And if you look at the phylogenetic tree image, you can see that we have a history of life. We get the 3 domains: bacteria, archaea, and eukarya, and it shows kind of that common ancestor at the root at the bottom of that tree. Okay, point that out as pointer, okay? And then the cladograms on the right is kind of a blurry image, but I'm going to be showing you a bunch more cladograms as we proceed through this lesson. So phylogeny, there are some important vocabulary words or concepts that you should understand when we are going to be constructing these diagrams. And so you can use traits that are either gained or lost during evolution to construct these phylogenetic trees and cladograms. But let's take a look at some of the important ideas here. First of all, shared characteristics are present in more than one lineage. Shared derived characteristics indicate common ancestry and are informative for the construction of phylogenetic trees and cladograms. And then finally, the outgroup represents the lineage that is least closely related to the remainder of the organisms in that phylogenetic tree or cladogram. And I'm going to give you some practice so you can see what that outgroup looks like. You'll see it a couple of times during today's lesson. A few other points about phylogeny and the type of data that can be used to construct these visual representations. Molecular data typically provide more accurate and reliable evidence than morphological traits. That is how an organism looks when constructing these trees and cladograms. Phylogenetic trees and cladograms can be used to illustrate speciation, whether it has occurred or not. The nodes are a really important concept. The nodes on a tree represent the most recent common ancestor of any 2 groups or lineages. I'll be pointing some of those out to you as we do our practice. Phylogenetic trees and cladograms can be constructed from morphological similarities of living or fossil species and from DNA and protein sequence similarities. And in just a moment, I'm going to take you through a lab that you might do if you were in class right now. That will have you start off with morphological characteristics and then refine it using molecular evidence. In this case, it'll be DNA sequence evidence. And finally, phylogenetic trees and cladograms represent hypotheses and are constantly being revised based on evidence. So the lab that I take you through will also give you practice at revising your original hypothesis. So the lab that I'm talking about is from the College Board AP Biology Lab Manual, which you can access on AP Central. This is also a lab that if you have access to a computer, you can do on your own. I'm going to be providing you with some screenshots so that you can work on this after you watch this lesson and give it a try because it's kind of fun. So this is AP Biology Investigation 3, and it uses a tool called BLAST, and I'll be explaining what BLAST is in a minute, but it allows you to compare DNA sequences to understand evolutionary relationships. So BLAST is short for Basic Local Alignment Search Tool. It's kind of a mouthful, so we just call it BLAST. BLAST is a bioinformatics tool that allows us to search a very large molecular database quickly to determine evolutionary relationships that are based on either DNA or protein similarities. In this particular lab, the objective is to determine the organism that is most closely related to one discovered in an unknown fossil. So this lab starts off with you being a paleontologist, and you go out and find this fossil. And the first thing that you're asked to do is to construct a cladogram that represents your hypothesis based on morphological characteristics alone. So if I were back in my classroom, I would take this image that you see in figure 3 of the fossil that you guys just collected out in the field, and I would project it, and I would ask the students to see if they can identify any morphological characteristics. So I know it's kind of small, but if you look closely, you can see that there are some limbs. I see 2 for sure, maybe 3, maybe 4. It's a little hard to tell. I can see that this organism has a tail, and I can see that it has some backbones or vertebrae, maybe even some ribs. Your eyes are probably better than mine. And I can also see that there is an opening in the skull. So you can take a look at this and see how many characteristics you can come up with. And then the first thing that you're going to do, and we'll do that together in the next slide, is to place this organism on a cladogram based on those traits alone. Then I'm going to teach you how to use the BLAST tool, and I'm just going to have to do that through a series of screenshots. And then finally, you're going to be given 4 gene sequences that come from this fossil, and you're going to use those to compare the fossil genes to extant or living gene sequences. And then finally, you'll use that information to revise your cladogram in light of the molecular evidence that you get using this BLAST tool. So the first thing we want to do is indicate where on this cladogram we might hypothesize this organism should be placed. So we know that it has vertebrae, so we know that we're going to place it above this node because that would be the last common ancestor. Some of you may, based on how it looks, may want to travel up and take a look and see maybe it belongs in the bird lineage or the crocodilian lineage. We don't have any evidence of fur, but it would be unlikely that that would be preserved in fossil form anyway. So I mean, we're kind of thinking that it was going to be in that area. Okay, so that's your working hypothesis based on the traits that you can observe or see. So now we're going to use the BLAST tool. Again, that's the Basic Local Alignment Search tool. We're going to see how well our fossil gene sequences align to living gene sequences. And so this is the home page of this tool, and you can Google the NCBI GenBank or BLAST, and it will come up. We are going to be using the nucleotide blast, but you could also do a protein blast, which you can see on the lower right side of your screen. So what you're going to be doing is beginning with one of the fossil gene sequences, and you're going to upload them one at a time. So when you do that, you're going to see your job title, and that's your AP Biology fossil specimen gene 1. You'll upload it into this box, and what you'll see is a series of A's, C's, T's, and G's. That's your gene sequence from this fossil. And then you're going to look down here, and you're going to choose which database you want to search. If you only wanted to search the human genome database, you would click that button. If you wanted to know mouse, we want to know all of them. This is a drop-down menu that you can also optimize, and we want to look at gene sequences, not these RNA sequences. So we would include that as well. So that's getting set up. And then the last thing we want to do is we want to optimize for highly similar sequences. We're going to get a lot of hits. We want the hits that are most closely related to the sequence that we put into the tool. And then you hit blast, this button here, so you blast off. And then what happens is literally in a minute or two, this database is searched for those sequences that most closely match the one that you put in. So you might see results that look like this. And again, it just looks like a gene sequence. So what the heck does that mean? So you can look at what is a little bit more information about that gene sequence. You can see that it comes from an organism with this scientific name coding for this protein. So this is collagen, and it codes for one of the chains. And we did indicate from the morphological traits that we saw some vertebrate bones, limbs, other bones, and collagen would be an important component of protein for an organism that has bones. If you don't know what the heck this scientific name is, you can come down here and you can see that this is in fact a turkey. That's the common name of this organism. And then you can get the full classification for this turkey. And then if you think about it, when we go back to that cladogram that we hypothesized, we put it after the vertebrae. We weren't sure if it was going to be on the bird lineages or the crocodile lineage, but we were pretty close with our hypothesis. Now we have some more evidence. This is molecular evidence that is probably related to a bird. Then you would go ahead and you would upload the other 3 genes and see what information they provide. The other thing that's pretty cool about this tool is that it gives you a cladogram. They call it a distance tree of results. And so you would click on that, and what you would see is something like this. And you can see that there's a lot of information here. You can see going back to the nearest common ancestor. Let's enlarge this, and your gene of interest is highlighted. And then you can see here that this is most closely related to a Gallus-Gallus collagen, the code for Gallus-Gallus collagen. And if you're not sure what Gallus-Gallus is, you can see that it's color coded. So you can look at the color coding, and again, that turns out to be a chicken, and a chicken is a bird. And so we know that we're on the right track placing that organism in that area. So then you go back and you would refine your hypothesis based on this new molecular evidence. Okay, so if you want, go ahead and try this on your own. It should be fun. All right, so some key takeaways. Phylogeny is the evolutionary history of a species. Both phylogenetic trees and cladograms are visual representation that shows these evolutionary relationships. The difference is that phylogenetic trees show the amount of change over time, and that's calibrated using fossil evidence or molecular clocks. And finally, molecular data typically provide more accurate and reliable evidence than morphological traits in the construction of phylogenetic trees and cladograms. So we're going to investigate that coming up, why that might be. So let's get some practice. My first practice is showing you a phylogenetic tree, and this figure shows relationships of organisms based on catalase structure. Catalase is a protein enzyme that catalyzes the breakdown of hydrogen peroxide, which is H2O2, into water and oxygen gas in the cells of many living organisms. And you may have worked with this in your enzyme lab when you did your enzyme lab in class. Students used an online database which used the structure of catalase from different species to generate the phylogenetic tree shown here. Okay, so let's look at what the question is asking us to do. It's asking us which of the following student claims is best supported by the data provided. Okay, so the first claim is that mushrooms and potatoes are most closely related since the node they share gave rise to all of the other organisms. So now you need to look at mushrooms and potatoes. The node they share, did it give rise to all of the other organisms? Is that the data that is going to support that claim? Cattle, which we see here, are equally related to pigs and sheep because phylogeny places the cattle between pigs and sheep. C, yeast and mushrooms are most closely related because they are both fungi. And finally, D, cattle and sheep are separated by only one node, and they are separated from the other organisms by more than one node. So this is the node. The cattle and sheep are separated by one, and the others are separated by more than one. Okay, so you have your answer. I hope you chose D. D is the correct response. Cattle and sheep are separated by only one node, and they are separated from the others by more than one node. All right, let's try one more multiple choice. Now we're looking at a phylogenetic tree of some island birds, and you may want to think back to an earlier lesson about Darwin's finches. And when we're talking about the morphology in this case, we're talking about the beak. How does the beak look? And you can see that it's different in each one of these species. So a group of birds is found in an island chain that have all descended from a single ancestral species, and that would be here. Okay, so this is the common ancestor of all 10 species. And the question is, which of the following best explains the divergence in morphology in the birds? In other words, why do they all have different beaks? Okay, so here are the choices. A, the original habitat contained mutagens, leading to the rise of new species as a result of accumulated mutations. B, the original habitat had many available unoccupied ecological niches that were colonized by populations of the original species. C, the original habitat contained many predators, and the species diversified in order to avoid extinction. And D, the original habitat was tropical, which increased the rate of mutation and led to rapid diversification. So in this case, I want you to think about some of the things that we have studied in previous lessons. I want you to think about Lamarckian language. I want you to think about habitat and mutations. So the correct answer in this case is B, the original habitat had many available unoccupied ecological niches that were colonized by populations of the original species. So as the original species was able to occupy a different niche, they were able to exploit a different food source, and so adaptations that helped them obtain that food source would allow them to survive and reproduce, and over time they would develop those different morphologies in their beak structure, so they'd look different. All right, let's take a look at FRQ. And in this particular FRQ, we're only going to look at part B, and I know that in yesterday's lesson, Ms. Pfizer had you take a look at a data table that looked a lot like this. So hopefully you can apply that to this question. So again, the statement that phylogeny is the evolutionary history of a species kind of points us into what we're going to need to do. In part B, we are asked to do 4 different things. So look at those task verbs. The first thing is asking us to use the data in the table. So based on the data in the table below, draw a phylogenetic tree that reflects the evolutionary relationships of the organisms based on the differences in their cytochrome C amino acid sequences. So you are being asked to draw a visual representation of these 5 species based on the data in this data table. Then you're asked to explain the relationships of the organisms. In other words, why did you draw the tree the way you did? And then you're asked, based on the data again, identify which organism is most closely related to the chicken and explain why you selected that organism. So let's get into this data table, which is showing us the number of amino acid differences in cytochrome C among various organisms. So we're talking about the protein cytochrome C, and we're looking at the amino acid differences in a horse, donkey, chicken, penguin, and snake. And so let's take a look at this row. So there's 1 amino acid difference between the donkey and the horse, so that tells you that they're probably pretty closely related to one another. There are 11 between the chicken and the horse, and 10 between the chicken and the donkey. The penguin has 13 between the horse and the penguin, 12 between the donkey and the penguin, and then only 2 between the penguin and the chicken. Okay, and then finally, is that right? 2, 3, and then the snake has the greatest number of amino acid differences from the horse. Okay, so let's start with that information and look at how we could construct our phylogenetic tree. So the scoring guideline shows us a phylogenetic tree over here with a root showing its common ancestry, and then this shows it in that cladogram form. And again, the snake and the horse have the greatest number of amino acid differences, so they're going to be the ones that are least related. The donkey and the horse were the most closely related, so they're going to be next to each other on this tree. And then you can fill in based on the number of amino acid differences from the chart. So either one of these would have earned 1 point for correctly drawing the tree. The second part asks you to explain explain how you constructed that tree. And you could have said this either way. You could have said that the more amino acid differences, the less closely related they are. So the horse and the snake had 21. Or you could have said the fewer differences, the more closely related they are. So the horse and the donkey had 1. So they were most closely related. Clearly the penguin is most closely related to the chicken, and because it had the fewest amino acid differences compared to the snake and the donkey and the horse. Okay, so that's a little practice at both drawing a visual representation and kind of understanding how to use that data table, showing you those amino acid differences. And again, that's molecular data, not morphological data. All right, so in this case, 2 of the task verbs were explained, so I wanted to just get some practice with that task verb. Explain means to provide information about how or why a relationship, process, pattern, position, situation, or outcome occurs, and it wants you to use evidence and or reasoning to support or qualify a claim. So we just did that on our last FRQ practice. If you get an explain how, it typically requires analyzing the relationship, so analysis. And if it's explain why, it requires analysis of motivations or reasons for the relationship, process, pattern, etc. So pay close attention to that task verb if it's just explain or explain how or explain why. So let's get some practice with that. This is a really long prompt, and then after this prompt, there's going to be a visual representation, a model, a phylogenetic tree that we're going to be asked to use to explain something. Okay, so I'm going to read this out loud, and I'd like you guys to read along with me so that when we get to that phylogenetic tree, you have your background. All right, so river dolphins are morphologically distinct from other dolphins and are primarily restricted to freshwater ecosystems in Asia and South America. Determining the phylogeny of these animals based on morphology has been difficult because they have a large number of derived characters, including a long, narrow rostrum, which is a scientific name for beak. They have a low dorsal fin, a flexible neck, and eyes that are greatly reduced. River dolphins were originally classified together based on their morphology, but molecular studies have shown they are less closely related than previously thought. Scientists analyzed the nucleotide sequences of mitochondrial genes, including the genes for cytochrome B and 2 ribosomal RNAs. By combining the nucleotide sequence data with analyses of embryonic development and morphology, the scientists have proposed the phylogenetic tree shown in figure 1. And in this case, we're going to see that baleen whales, they're filter feeders, and they're going to be used as our outgroup in this phylogenetic tree. So you can get your example of an outgroup. All right, so here's the phylogenetic tree of cetaceans, which is the family that includes both toothed whales and baleen whales. And you can see that the baleen whales are an outgroup because they do not contain teeth, okay? So they feed differently. I also want you to see that we're mostly concerned with the river dolphins. So we've got a Southeast Asian river dolphin, Chinese river dolphin, Amazon river dolphin, and La Plata river dolphin. And you can see they're all in their own lineage. D, this is part D. This is a whole question, but we're only looking at part D. It says that molecular data indicate that river dolphins do not form a monophyletic group. That means that we don't see them all in one lineage. Explain why these animals nevertheless have morphological similarities. Before we go over the answer, I want you to think about something that we talked about in a previous lesson. When we talked about lesson 7.3 on artificial selection, we talked about how humans select for certain traits, but that in convergent evolution, the environment selects the traits. And so that sometimes individuals who are not related, they have different evolutionary lineages, they can resemble one another due to the environmental pressures. So please keep that in mind as we go through our choices or our thought process. Okay, so this is a free response question that's asking you to explain why these animals look the same even though genetically they're different. And your answer should include some way of getting in that the similarities in morphology are likely due to convergent evolution. And the reason for that is that river dolphins inhabit similar habitats and are subject to the pressures of those environments. So reduced visibility in most large rivers occurs due to silt, and that would lead to a reduction in vision or eye size. Long noses are an adaptation for searching for food in that environment, and streamlined bodies and dorsal fins are not as critical for stability in rivers as they are for some of the other organisms in that tree that live in the ocean, like some of the other whales. So this kind of puts it all together, looking at convergent evolution and how that can be shown in a visual representation such as a phylogenetic tree. All right, so just a reminder about task verbs. We're going to practice these in all of our lessons. Task verbs are not just in evolution; they're used through all units in the curriculum. You can find them in the course and exam description on page 199. And again, you find that course and exam description on AP Central. And again, since you're not in the classroom and your teacher, don't have your teacher kind of guiding you through these things, I think it might be a good idea for you to look at those on your own. Okay, so let's do a couple of checks for understanding. So remember the skill that we're practicing is to explain the relationships between different characteristics of biological concepts, processes, or models that are represented visually. So again, in this content, phylogeny, we're going to be talking about cladograms of phylogenetic trees. So what you're looking at is a cladogram of selected vertebrate groups. It looks a lot like the one that I showed you on the very first slide that was kind of blurry. And this question reads, whales, bony fish, and cartilaginous fish all have similar adaptations that enable them to live in the ocean. This figure shows the phylogenetic relationships of selected vertebrates. Your question is way down here. Based on the phylogenetic relationships shown in figure 1, which of the following best explains the similarities exhibited by whales, bony fish, and cartilaginous fish? So we're looking at whales, bony fish, and cartilaginous fish, and you want to kind of look at the lineage. Okay, so these nodes represent the last common ancestor. Okay, so your answer choices: A, convergent evolution because these organisms do not share a recent common ancestor for a shared recent common ancestor on that tree. Adaptive radiation because all 3 groups evolved from a unique shared common ancestor from the cartilaginous fish group. Is that true? C, mutation because shared traits can be traced back to a recent common ancestor. And D, founder effect because all 3 groups previously existed together as one population that was then disrupted, causing speciation. So look at your choices. Think about what we've already talked about today. Look at your phylogenetic tree or cladogram. Select your choice. Okay, the correct answer is A, convergent evolution. These organisms do not share a recent common ancestor. So look at those nodes for the common ancestors and look at where that recent common ancestor would be, and you don't see it. All right, so let's debrief, and then we're going to get some practice with some more visual representation. So you should now be able to describe the types of evidence that can be used to infer evolutionary relationships, right? Morphological traits or molecular traits. You should be able to explain how a phylogenetic tree and or cladogram can be used to infer evolutionary relatedness. You want to look for shared characteristics and shared derived traits. You want to look at the node that represents the most recent common ancestors. And finally, you should be able to explain between different characteristics of biological concepts, processes, or models that are represented visually. Your key takeaways: evolution is characterized by a change in genetic makeup of a population over time and is supported by multiple lines of evidence. Phylogenetic trees and cladograms are both visual representation of evolutionary relationships. So let's take a look at FRQ. I believe this was from last year's AP exam. And we can take a look at some common errors and misconceptions that student had in their responses on this question. So we're looking again at that data table, which you've had some practice with, and we're looking at molecular evidence. We're looking at the divergence. These are percents of mitochondrial DNA sequences among 5 primate species. So you can see we have humans, gorillas, orangutans, gibbons, and chimps. And again, if you kind of look across, you can see that we see the gibbon at 18.1% has the greatest difference from the human. Okay, the orangutan has 16.1, which is the next greatest difference from human. You can look at the gorilla at 10.3%, for 10.3%, which would be the next, and then the one that has the fewest differences in percent, 8.8% from the human, is the chimp. Okay, so you're given the template on the question, and they also provided an outgroup for you. So this outgroup is the gibbon, and it would be the one that is least related to the other ones. It's on its own lineage. And so looking at our task verbs, using data in the table, construct a cladogram on the template provided, and then provide reasoning for the placement of gibbons as the outgroup on the cladogram. And then finally, it asks students to draw a circle around all the species indicated by the node in the square that are related. Okay, so think about how you would answer that based on these differences in the percent divergent. These primate species, even have that pencil and paper and draw your cladogram. And then the final thing is you want to think about the organisms that are going to be related to the species that are shown by this node with the square around it. Okay, so from the top to bottom, the human, the chimp, the gorilla, the orangutan, and the given. And the reason for that is that they are the human and the chimp have the least divergence, the human and the gorilla the next least, as I went over on the data table, and the human and the orangutan had the most on those ones. And then compared to the outgroup, it had the most. So the reasoning why the given is the outgroup is that it is the least similar to all of the other species, or you could assess the one that's most different from all of the other species, or you could have used the term divergence, the one that is most divergent from the other species. And students did pretty well with that. However, where the error and misconception came, so you were supposed to correctly circle the 3 species in the first 3 positions of the cladogram to show the ones that are descended from the species indicated by this note. And so it's all of the ones that are above that, which would be the gorilla, the chimp, and human. And students had a difficult time doing that. So that is was a knowledge gap on this one. So here's your homework for tonight. This question will be the warm up tomorrow, and my colleague Ms. Pfizer will go over this with you. It gives you more practice with the data table and more practice constructing a phylogenetic tree. So again, this is based on molecular data. The amino acid sequence of cytochrome C was determined for 5 species of vertebrates, and the table below shows the number of differences in the sequences between each pair of species. This time you're given some scientific names. It doesn't totally matter. In some ways, it may be a little bit harder to not know them, but really what you're looking at is the difference, right? So you can see that these guys it's 0 and 1, and these guys is 0 and 21, these guys is 0 and 11, and these guys is 0 and 13. So key in on that. And you are asked to, using that data, create a phylogenetic tree using this template. You're just going to write those organisms where you think they belong here, and then provide reasonings for why you place them on the tree, and why the placement on the tree of the species that is least related to the others. So if you're wanting to do this, you can kind of take a picture, do a screenshot, or this will be recorded and you can come back to it later to do your homework. Okay. So one word from the College Board. We all know that not all students have access to the internet or device. We're working on solutions to help students get what they need to show their best work. If you need mobile tools or connectivity or know someone who does, you can reach us directly to let us know, and reach the College Board at cb.org/tech. And this will be especially important since you'll be taking the AP exam on your own device. So you want to get that as soon as possible so you get familiar with it before you have to take your exam. Okay. So again, thanks for watching. I hope you'll join us for all of our lessons. Make sure you complete that practice question for homework, and Ms. Pfizer will review it with you in tomorrow's lesson. Have a great rest of your day. Thanks for watching.
Welcome to AP Biology. Hi, I'm Gail Pfizer from Darnell Cookman Middle High School and Florida State College in Jacksonville, Florida. Today, hold on just a second, let me get rid of a little screen. Today, the topic we're covering is one that colleges have asked that all AP students study, even if it will not be on the exam. Colleges are supporting AP students this year by allowing this more flexible approach to the exam, where it will focus on a lesser amount of content than other years. But in turn, they have asked that AP students learn today's material. To make this relevant to your upcoming exam, we'll also include some exam prep in today's lesson. I'm excited to, I know that you might have seen this information today. This was just released, and our exam date is May 18th. And if you want to see what I'm looking at right now, then you need to go to the College Board website, and it has updates from today there for you. So exam date May the 18th. This is the part that I want to specifically talk about today, is to go and give you a head start as a student to what these 2 questions are going to look like and what you're going to need to do, and how I'm going to try to help you by driving my train to these 2 questions. Question 1 is interpreting and evaluating experimental results, and you're going to have 25 minutes to read and respond, and you're going to have 5 minutes to upload, and that's going to be 65% of the exam score. Now, this is going to be based off of an authentic scenario accompanied by some data in a table or on a graph, and so it has 4 parts to it. And there's going to be, I think there's going to be, let me give a disclaimer. I am not on the test development committee. I am not even an item writer, because that is like the opposite of my skill set, is writing a test question. All right, so A is going to be describe and explain biological concepts, processes, models, and applied context. B, we're going to identify and justify experimental design procedures. C, we're going to analyze data, and D, we're going to make and justify predictions. I know that this is going to be something that we're all going to do very well, because we all work very hard on experimental design in the classroom. All right, the second question is conceptual analysis. There'll be 15 minutes for the response, 5 minutes to upload. It's going to be 35% of your score. This question will be an authentic scenario describing a biological phenomenon with a disruption. There's 4 parts to it as well. Describe biological concepts or processes in applied context, explain biological concepts or processes in applied context, and then predict the causes or effects of a change in a biological system. And then D, you're going to support or justify a claim with reasoning or evidence. And so those are going to be our target task verbs, and those are going to be our target ideas to wrap together with each review topic. All right, now for an overview of our lesson. We're on topic 7.10, and celebration today, because this is wrapping up our evolution unit. On the next lesson, Ms. Kate is going to put together an overview of ecology, and then the lesson after that one is we're going to start reviewing for the exam. So we're really excited about that. This is coming, and it's so close. I need you to make sure you have pen, pencil, paper. I don't think we're doing any math today, no graphing today. It's going to be a pretty easy, fun, I think, topic. All right, so today I'll be introducing you to allopatric and sympatric speciation, the commonly used definition of species. We're going to talk about punctuated equilibrium and gradualism, adaptive radiation. We're going to talk about prezygotic and postzygotic mechanisms. You should already be familiar with natural selection, artificial selection, and then the science skill explained. By the end of this lesson, you will also be able to describe the conditions under which new species arise, describe the rate of evolution and speciation under different ecological conditions, explain the processes and mechanisms that drive speciation, and predict the effects of change in biological systems based on biological processes. All right, so now for our warm-up. Yes, last lesson with Ms. Kate, you had topic 7.9. So here we have a table, and it has the number of amino acid differences in cytochrome C among 5 species. You see the species listed in a row on the left, and on a column on the left, and then a species in the top row, and they're the same 5 species in each place. And so if you did your homework from last night or last session, then I just hold on to it for just a minute, because I want to give the folks that are just tuning in to us for the first time a chance to do this as a warm-up instead of homework. And so we'll be doing that, we'll continue to do that through the review session. Last night's homework is going to be today's warm-up, okay? All right, so A, using the data in the table, create a phylogenetic tree on the template provided to reflect the evolutionary relationships of the organisms. You're going to provide reasoning for the placement on the tree of the species that is least related to the others. So I'll give you just a minute. Let me get the clock going. Okay, about 10 more seconds. Okay, let's take a look at the scoring guideline. All right, so you should have your D polylepsis on the left, the farthest away from the E ferris on the right, and then let me get this going here. And then these 2 right here, Forsteri and Gallus, they are going to, and I'll go back to that so you can see why they're together, but I want to talk about this. There can be free rotation around the node. So this right here is a node. So if you had G Gallus over here and A Forsteri over here, that's still fine. It's good. And the same thing with the E Africanus, and E ferris. All right, so and then we have D polylepsis is the furthest point. It's away from the E ferris, and let's find out why. There are 20 amino differences between D polylepsis and E africanus, which is more differences than others, and E africanus. So it is the farthest away of the tree. The more changes reflect the more distance in the relationship. Okay, I'll go back for just a second. second. Let's see, we have from Ferris, we have 21 amino acid differences. So that was the farthest one away. And then we had one difference very close, and that's why Africanus ended up right next to it. And then these two are very closely in number away as well. So that's why they ended up in the middle. All right, so our course, this is wrapping up the evolution, natural selection unit. Tonight's topic is speciation, and each topic has a learning objective, and that's what we need to be able to do. And we need to know content in order to answer these questions. And that content is called essential knowledge. And each topic has an associated science skill. And this is how we're going to show that we know the answer to the question, which is the learning objective. And the science skill is associated with our topic is explained. So today we're going to have a topic skill pair. It's going to be speciation and explain. And I'm going to go ahead and answer a question of someone that emailed me. And I want to, he asked, where did we get the information of what you need to know? Where I get this information is straight from our curriculum and exam design, the CED. I look at the topic, I look at the learning objectives and all the content that we need to know to be able to do that. And it's right off of the page is how I structure what I'm putting into this lesson. Okay, all right, so what is a species? The biological species concept states that species can be defined as a group capable of interbreeding and exchanging genetic information to produce viable, fertile offspring. Now, let's talk about allopatric speciation, and then we'll talk about sympatric speciation. So allopatric speciation means other country, and sympatric means same country, and both of them are mechanisms that can drive speciation. So allopatric speciation is two geographically isolated populations of one species that become different species over time, and this is due to selection pressures in different environments. So if you look at the diagram at the bottom, you'll see this is the original population, and then we have a geographical barrier that is put into the original population, and then eventually reproduction isolation is going to occur, and it's going to begin occurring right here, and then we're going to actually have two different species. Okay, so reproductive isolation occurs as new environmental pressure selected for those organisms with the adaptations to survive and reproduce in that environment. So that's allopatric, okay, which means other country not in the same place anymore. Okay, all right, sympatric is different, it's not as common, but sympatric speciation is not due to geographical isolation. It has to do with two or more populations of one species, and they become different species over time due to isolation. This isolation is due to a number of things: polyploidy, sexual selection, or habitat differentiation. The model below represents a subpopulation that's exploiting resources in a particular region in this environment. So here's our here's our original species, and here we have a subpopulation of this population that's kind of hanging out in this one area that's exploiting some resources here, and eventually we're going to have species 1 and species 2. Okay, so polyploidy, audiploidy, and alloploidy, and there's some words that are going to be a mouthful here. All right, polyploidy means greater than 2N, and polyploidy plants can only be fertilized by another polyploidy plant. So autopolyploidy is when a 2N plant fertilizes another 2N plant and has a 3N offspring, and this is due to non-disjunction during meiosis, and we'll cover that topic during the review period. Allopolyploidy is reproduction of different but related species. There's hybridization, and it's followed by the doubling of chromosomes. If you'll take a look at the Clarkia genus, there's a particular species that has 2N equals 14. This one 2N equals 10, but their offspring has 2N equals 24. Okay, various pre-zygotic and post-zygotic mechanisms can maintain reproductive isolation and prevent gene flow between populations. And I put in a little bit of vocabulary here too, in case you weren't sure. Zygote, we're talking about a fertilized egg, and so for pre-zygotic that means before fertilization, and for post-zygotic that means after fertilization. Okay, so I'm going to give you some examples of pre-zygotic barriers, and then we'll jump over to post-zygotic barriers. So this is a pre-zygotic barrier. It's a habitat ecological isolation. The organisms have preferences for living or mating in different habitats. We have an aquatic snake has their place they like to live, and a terrestrial snake has their place they like to live, and they're just not hanging out together. Geographical isolation is another pre-zygotic, living or mating in different geographical areas with a physical barrier. That's not going to happen. All right, so organisms are separated from exchanging genetic material with species members, and then an example of that would be Darwin's finches. All right, temporal isolation. Organisms have non-overlapping reproductive periods. They have a different mating time either of the day or the season of the year. An example of the wood frog is most likely to reproduce. High point is in late March. Leopard frog in early May. Behavioral isolation is another pre-zygotic. There's mating rituals that exist within species. The blue-footed boogies is one of the examples. There, the male has to have the right dance moves and moving his feet in the right way to persuade his female partner. All right, mechanical isolation. There's structural differences of the reproductive organs. An example of that would be the shape of flowers. All right, another pre-zygotic barrier, gametic isolation. The gametes themselves are incompatible. We have molecular incompatibilities between sperm and egg, or we have chemical incompatibilities that limit the sperm's viability. An example of this would be the red sea urchin and the purple sea urchin. All right, so that was the pre-zygotic barriers, and now we're going to briefly talk about some post. This is reduced hybrid fertility. An example is mules, and a mule is the offspring of a female horse, which is a mare, and a male donkey, which is a jack. And the mule is a hybrid; it cannot reproduce. If it does, then the offspring are sterile. And then just an FYI, other facts that I went down rabbit hole and found today: hinny is a male stallion and a female donkey. That's a jenny. And horses have 64 chromosomes, and donkeys have 62. All right, here's another post-zygotic barrier: reduced hybrid viability and hybrid breakdown. Okay, so this salamander, he has reduced hybrid viability. He looks very weak and fragile. And the hybrid breakdown example is different species of cotton reproducing. All right, so pre-zygotic and post-zygotic, that part's over. Now we're going to talk about some models of speciation, gradualism, and punctuated equilibrium. If you'll look at the butterflies on the left, here's on the left, and we have a light-colored butterfly, and we have this transitioning into this dark butterfly. Okay, that is gradualism, which is the ecological conditions change gradually over a long period of time. Now on this one we have a light-colored one, and then there we go, all right, straight to black. Okay, and then punctuated equilibrium, the ecological conditions remain unchanged for a long period of time, and then change drastically. All right, adaptive radiation. Species invade several new geographical separate environments. Populations adapt to new environments, and new species evolve from the ancestral species. An example of this is the age of the mammals after the extinction of the dinosaurs, when small animals were able to go into new territory, and then speciation occurred from them. All right, the science practice that we are going to talk about next is 2BA, and then it's explain relationships between different characteristics of biological concepts, processes, or models represented visually in theoretical context. And the task verbs that we're going to be using are explain, identify, and describe. And I think that we saw those in question 1 and question 2 that we have ahead of us in the next 5 or 6 weeks. All right, so here's a simple explain in a multiple choice format, and you just saw this on the previous screen. I know this is going to be easy. So you know the image. So let's see what the question is asking. Which of the following best explains how the ecological conditions are likely to be different in the two models? And we have A. In model A, the ecological conditions remain unchanged for long periods of time and then change drastically. In model B, the ecological conditions change drastically from generation to generation. In model B is in model A, the ecological conditions remain unchanged. In model B, the ecological conditions change gradually over long periods of time. And C, in model A, the ecological conditions are changing drastically from generation to generation. And in model B, the ecological conditions remain unchanged. And D, in model A, the ecological conditions change gradually over a long period of time. And in model B, the ecological conditions remain unchanged for long periods of time and then change drastically. Okay, this is an example of explain. Remember, when we're explaining, we're talking about how or why something is happening. And if we were just describing it, we would just be telling the characteristics of a image, graph, table, something like that. All right, I'll give you just a few seconds to answer that because I don't think you need much more. Okay, and I hope you answered D. In model A, the ecological conditions change gradually over a long period of time. In model B, the ecological conditions remain unchanged, and then unchanged for a long period of time, and then change drastically, right there. Okay, now for a check for understanding. All right, so let's look at this map. It looks like a map, and we're going to have, there's a lake 1 and there's a lake 2, and there is a population A and B live in lake 1, and a population A, D, C, and E live in lake 2. You know, I'd like to go first to the image so I can kind of understand what the text is going to be about before I get to it. All right, so let me see what this says. Researchers are modeling the effects of repeated cycles of isolation with occasional interbreeding among 5 hypothetical fish species A, B, C, D, and E, found in 2 separate lakes, lake 1 and lake 2, that are occasionally joined by flooding, as shown in figure 1. The research team indicated that species A would be considered to be the single common ancestor of species B, C, D, and E. So now the question: which of the following mechanisms could most likely account for the origin of species B in lake 1? Okay, species B in lake 1. Would it be A, allopatric speciation, B, directional selection, C, stabilizing selection, or D, sympatric speciation? I'll give you just a few seconds, about maybe 15. Okay, I hope you answered D, sympatric speciation. All right, here's another question. The fossil record indicates that in some cases, reasonably well-defined species appear suddenly and remain unchanged for a long time before they become extinct. This phenomenon is referred to as A, allopatric speciation, B, sympatric speciation, C, phyletic gradualism, D, punctuated equilibrium, or E, parallel evolution. I know these seem like easy questions tonight, nothing like the past lesson where I've had you like in 3 slides of text to answer a question. This is just an easy topic. All right, so I'm gonna give you a few seconds. All right, and I hope you said D, punctuated equilibrium. All right, so let's take a look at this. We have a table, and we have a habitat. We have 4 habitats, and the first habitat we have sandy soil and no insectivorous birds are present. In habitat 2, we have sandy soil and insectivorous birds present. And in habitat 3, we have loam soil, no insectivorous birds present. And in habitat 4, loam soil, insectivorous birds present. And then I have colors of beetles. I see I have light tan, dark brown in each of those habitats. And then I have a number released, and that's consistent across, and then number recaptured. And there's some low numbers and a couple of them that just caught my attention. Okay, let's see what the text tells us. Individuals of a particular species of ground beetle are either light tan or dark brown. Light tan beetles are predominant in habitats with light colored sandy soils, and dark brown beetles are predominant in habitats with dark colored loam soil. In an experiment designed to determine the survival rates of light tan beetles and dark brown beetles in different habitats, 500 light tan beetles and 500 dark brown beetles were released in each of 4 habitats. Each beetle had marked with a small red spot or red paint on the underside of its abdomen before it was released. 1 week after the beetles had been released, any marked beetles that could be found were recaptured. The results are presented in the table below. It is assumed that the differences in the number of beetles recaptured are directly related to differences in survival rates. So the question we need to answer: which of the following processes best explains why fewer light tan beetles than dark brown beetles were recaptured in habitat 4? So let's look at, we have dark soil, okay, and we have birds that are going to eat us, and we have light tan. Okay, what do you think is happening there? A, immigration, B, genetic drift, C, mutation, D, adaptive radiation, or E, predation? All right, I'll give you 5 seconds for that one. All right, predation. Okay. All right, so we have our next question is some guided, some guided practice. We have 2 short FRQs. And this one, it has some different things that you might not have seen before. So let's go through the image. On figure 1, we have a diagram of a choice chamber, and we have mosquitoes, and we have a guinea pig, and we have a hand. And they're distinguished through the mosquitoes like the guinea pig or the hand. Okay. And in the second diagram, there's a graph, and there's numbers going up, and there's bars going down, and maybe you've never seen anything that looks like that before. But don't let it be intimidating. It's very simple to understand with even this description for the figure. All right, the mean host preference index plus or minus 1 as of the forest and domestic forms of mosquitoes. A positive value indicates preference for a human host, and a negative value indicates preference for a guinea pig host. So we have forest form is in dark, okay, and domestic is in the light gray. All right, and negative is for the guinea pig. So the negative is in the forest, and then the light is the human. Okay. All right. Now the yellow fever mosquito, Aedes aegypti, is a major vector of human disease. In a particular location in Africa, there are 2 forms of the mosquito. The forest form of the mosquito is black and often lays its eggs in tree holes and pools of water in the forest. The domestic form of the mosquito is brown and prefers to lay its eggs in rainwater collected near human dwellings. Researchers used a choice chamber, figure 1, to investigate the host preference A. aegypti. The researchers recorded the number of forest form and domestic form mosquitoes that bit human or guinea pig hosts during the several 10-minute trials. The researchers used this data to collect a host preference index for each form, as shown in figure 2. Researchers also identified a gene in mosquitoes, OR4, that encodes for an olfactory receptor. A volatile odorant sulfotone binds to the OR4 receptor, and humans produce higher levels of sulfotone than do guinea pigs. Now this text in diagrams probably originally went with different more questions than the one. So this particular one only has to do with pre-zygotic and post-zygotic and isolation mechanisms are topic for tonight, speciation. So I'm going to read it to you. A researcher proposes that 2 forms of mosquito are evolving into 2 different species. Identify one potential post-zygotic isolating mechanism and describe how the isolating mechanism would result in the evolution of the 2 forms in the different species. So actually you're not using anything in this table, in that graph, or in that aquarium. Those would have been for other questions that we don't have that are not in this unit that we're talking about. So what you're only going to talk about now is post-zygotic isolating mechanisms, and then describing what that is, and then example. Okay, I'm going to give you a few minutes. No, not a few minutes. 1 minute will be enough for that. Okay, about 10 seconds. Okay, this question is a perfect example of what I was talking about in the previous lesson, that you don't need every diagram or graph to answer a multiple part FRQ question, that you need to just see which ones apply. And in this case, we could have done that particular part of the question without looking at either one of them. It has to do with some biology moving forward from this information. So let's look at the scoring guidelines. All right, so if you said hybrid inviability, hybrid sterility, or reduced hybrid fitness, or hybrid breakdown, then that would have been your post-zygotic mechanism. And then your description, you would have had maintains reproduction isolation and prevents gene flow. Okay, all right, hopefully you scored those points. All right, now this is the one with the examples. Okay, now the short FRQ, 2 of 2, because I had 2 really short ones for you to do today. Discuss 2 pre-zygotic isolating mechanisms that prevent hybridization between 2 species. Now include in your discussion an example of each mechanism. So 2 pre-zygotic, okay, and then stick in there, explain, discuss, let's say that you need to tell what that is, okay, you need to explain it, and then include an example. And you can include an example that I gave you earlier, and we'll take a minute for that one as well. Okay, that's 10 seconds. Okay, all right, so let's look at your options here. And what did you have to? So you could have scored for habitat ecological isolation, preferences for living, mating in different habitats or microenvironments, and geographical isolation, living or mating in different geographic areas with a physical barrier, mechanical isolation, structural differences of reproductive organs, temporal isolation, different mating time of day or season of the year, behavioral isolation, different mating rituals between species, and then gametic isolation, molecular incompatibilities or chemical incompatibilities. And then an example of one of those that I gave you previously, and then there you would have had at least those 2 points, or the 4 points. All right, now we're going to have a little debrief and summary. All right, so I hope today you understand the definition of a species. and that if maybe you've never seen the words punctuated equilibrium and gradualism before, they sound kind of scary alone, but they're not. Pre-zygotic and post-zygotic mechanisms are fun, and you should also be able to describe the conditions under which new species may arise, describe the rate of evolution and speciation under different ecological conditions, explain the processes and mechanisms that drive speciation, and predict the effects of change in biological systems based on biological processes. All right, so I pulled this as our common errors and misunderstandings because I've been teaching this for a long time, and it seems like shading seems to get my kids sometimes. So I saw that could possibly happen in that one question. So it made me think of another question. So what I suggest you do is determine before you're trying to figure out something, where's the land and where is the water? Okay, so this says Lake 1, and that could be, is it the white part? But this makes it very clear. The Lake 2, okay, is the dark, and that little Lake 1 is also dark. Okay, all right, and the same here. We have Connecticut, a state, Long Island, and then New York, and then we have this water around. Okay, so that's just something I thought might be helpful if you figure out which is land and which is water before you get started. Okay, all right, and now Ms. Kate is going to do ecology on the next lesson, and she picked out this fantastic ecological topic question for you to do for homework, so you can have a little bit of extra practice in ecology. So in an attempt to rescue a small isolated population of snakes from decline, a few male snakes from several larger populations of the same species were introduced into the population in 1992. The snakes reproduce sexually, and there are abundant resources in the environment. The figure below shows the results of the study of the snake population both before and after the introduction of the outside males. In the study, the number of captured snakes indicate the overall population size. So here we have a light gray is our total snakes captured, and a darker, almost black juvenile snakes captured. Our y-axis, which is our results, dependent variable, remember, number of snakes captured, and then we have years along the bottom and increasing, and then we have males when they're introduced as a little arrow. Okay, so here we have a perfect example of data to analyze in part C of that question 1 that we're heading into. Okay, so that's what's going to be there. You're not going to be making a graph. You're going to be analyzing a graph and working with a graph that's already there, a table that's already there. All right, so A, describe one characteristic of the original population that may have led to the population's decline in size between 1989 and 1993. B, propose one reason that the introduction of the outside males rescued the snake population from decline. And C, describe how the data support the statement that there are abundant resources in the environment. So that's your homework for this lesson, and it's going to be the warm-up for the next lesson, which will be our ecology night. And I need to get you to do this if you need to by April 24th. Device and internet access. We know that not all students have access to the internet or device. We're working on solutions to help students get what they need to show their best work. If you need mobile tools or connectivity or know someone who does, you can reach us directly to let us know. cb.org/tech. And thanks for watching. Review sessions will start Tuesday, next Tuesday. So Monday, Ms. Tay will have ecology, and then Tuesday, Ms. Tay will begin with biomolecules, and then Wednesday, I'll have proteins and nucleic acids, if that helps you know where we're going. Okay, we're starting at the beginning of the curriculum, and we're going to move through the units 1 through 6. Thanks for watching. We hope you'll join us next time. Be sure to do your homework, and I'll see you next week.
Okay. Hi. Welcome to our. Okay. Sorry, we're having a little bit of a late start today due to some technical difficulties. We seem to be okay. Happy Monday. Welcome to today's AP Biology lesson. Today, our lesson will be on new material in Unit 8. On Friday, April 3rd, an announcement was made providing details about the 2020 AP Biology exam. In case you're just hearing this, I'm going to give you some details about the exam. The 2020 exam will be administered on Monday, May 18th at 2 p.m. Eastern Standard Time. All exams will be taken by students at home. The exam will consist of 2 free response questions. The first question is a long FRQ and will have a 25-minute time limit. The second question is a short FRQ and will have a 15-minute time limit. Beginning tomorrow, we will be presenting lessons that will review the first 6 units in the AP Biology course. All lessons, including these with new material, will include practice with science skills which are relevant with any content. We will also provide practice with free response questions in each lesson. A little bit more about the announcement that was made about the 2020 exam. This is a screenshot from AP Central. One thing I do want you to notice is that the time says 1 p.m. That's standard time, Central Standard Time, excuse me. But the primary date will be Monday, May 18th. If any students have conflicts with that date, there will be one makeup date, and that will be Wednesday, June 3rd. Again, this is posted, so there will be 2 questions for a total of 45 minutes of testing time. 25 minutes will be provided for the first question with 5 minutes to submit your answers, followed by a 15-minute short answer question, and then another 5 minutes to submit your question. At the bottom, it talks about these video reviews that we're doing, and tomorrow we'll begin our review sessions. They're the same time every day from 4 to 4:45 p.m. Eastern Standard Time. The link is there, but if you're here, you probably already found it. And as we get closer to exam date later on in April, there will be some guidance provided about how you're going to be able to submit your responses, and also there'll be some practice simulations so that you'll be ready to submit your answers on exam day. A little bit more information about the actual questions on the AP Biology exam. So again, question 1 is a long free response question, and it is going to be about interpreting and evaluating experimental results, and it will make up 65% of your total exam score. So on a normal testing year, we would tell you to spend about 20 to 22 minutes on a long FRQ. So you're getting a little bit more time, and then you're going to have 5 minutes to upload your response. What will happen in this question is you'll be given a scenario, and it'll be accompanied by data in a data table or graph. And you're going to be asked to do 4 different things. So here's where those task verbs that we've been working on come into play. You're going to be asked to describe and explain biological concepts, processes, or models. You're going to be asked to identify experimental design procedures. You're going to be asked to analyze data. And finally, you're going to be asked to make and justify predictions. And you can see how those points are distributed. The second question is a short FRQ. And again, if this was a year, I always tell my students to plan to spend about 6 minutes per short FRQ, and you have 15. So you have quite a bit of time there. So, read the question carefully and you look at those command terms or task verbs. This second question will make up 35% of your total exam score. So you'll be given a scenario that will describe a biological phenomenon with some type of a disruption. And you'll be asked to describe the biological concepts or processes. You'll be asked to explain biological concepts or processes, or describe characteristics that are represented visually. And the last time we were together, we looked at phylogenetic trees and cladograms, and those would be examples of concepts that are represented visually. You will also be asked to predict the causes or effects of a change in a biological system. That's that disruption, and then justify those predictions. And this is a 4-point short FRQ. So hi, I'm Allison Kate. I'm affiliated with both UC Berkeley and Redmond High School, and today I'm teaching you from my home in El Cerrito, California. So let's do our warm-up. If you're just joining us, the warm-ups are AP exam practice questions that are given for homework from the previous lesson. So our last lesson was on Friday, and it was on topic 7.10, which I believe is speciation. My colleague, Ms. Pfizer from Florida, assigned this homework to you. So if you have it, bring it, get it out. If you don't have it, grab your pencil and paper and try to answer this as we move through this FRQ. So let's read the question together, and then we'll take a look at that visual representation of data, and then we'll look at our task verbs, what we were asked to do with this question. So in an attempt to rescue a small isolated population of snakes from decline, a few male snakes from several larger populations of the same species were introduced into the population in 1992. The snakes reproduce sexually, and there are abundant resources in the environment. The figure below shows the results of a study of the snake population both before and after the introduction of outside males. In the study, the numbers of captured snakes indicate overall population size. So you can see on the y-axis, the dependent variable is the number of snakes captured, and we also have a legend that tells us the total number of snakes captured are represented by the lighter bar, and the juvenile snakes captured are represented by the darker bar. On the y-axis, we have the year, the time, which is the independent variable, and we have an arrow that shows us when the males from the outside population were introduced, and that arrow is right here. So as we look at this, we can get a trend for both the total number of snakes captured and the juvenile snakes captured. So your task verbs, first in part A, are to describe one characteristic of the original population that may have led to the population's decline in size between 1989 and 1993. See those bars get lower, and then propose one reason why the introduction of outside males rescued the snake population from decline. You can see after they were introduced, those bars begin to increase. And then in part C, describe how the data support the statement that there are abundant resources in the environment. So there are 3 tasks that you needed to address in this question. So let's take a look at the scoring guidelines. In part A, each of those bullet points for the description are worth 1 point. You don't need to say them all, but you do need to say one of those 4. So one way to say the description is that the population that may have, or the characteristic that may have led to the population's decline would be that it lacked genetic diversity or variation, or that it was aged, an old population, post-reproductive, there weren't enough young snakes. It could have had an unfavorable sex ratio with too few males, or the population could have had a harmful mutation or a disease. So any one of those points that describe the decline between 1989 and 1993 was worth 1 point. The second part proposed one reason that the introduction of the outside males rescued the snake population. Again, any one of those bullet points would earn the point. So those new males could have increased the genetic diversity in the population, they could have increased reproductive success, they could have established a beneficial sex ratio, or introduced resistance to disease that was affecting the original population. And then the last part asks you to describe how the data support the statement that there are abundant resources in the environment. And if you look at that graph, you can see that the population does increase in number. So you could say that the population can and does grow, or you could have said that the resources were limited, the population would not grow. So this was a 3-point question. Each part A, B, and C was worth 1 point. So let's look at today's lesson. Don't forget your paper and pencil because we do want this to be interactive. So today we'll be introducing you to the concepts of community ecology and disruptions to ecosystems. And we're going to get some practice with science skill 5A, which is calculations of mathematical equations that are in the curriculum. We're going to look at the Simpson's diversity index equation, and we're going to look at both sample, multiple choice, and response questions that combine these contents and skill. As we get started today, there are a few concepts that you should be familiar with: community ecology, ecosystems, and human impact on ecosystems. And then finally, the terms competition, predation, symbiosis, habitat, and invasive species would be important to have in mind. By the end of this lesson, the objectives are that you will be able to understand that communities and ecosystems change on the basis of interactions among populations and disruptions to the environment. And the overarching idea is that we'd like you to understand that evolution is characterized by a change in the genetic makeup of a population over time and is supported by multiple lines of evidence. And you might be thinking, well, that's an evolution objective, and we're talking about ecology. What we see today does provide evidence for that change in the genetic makeup of a population. So just to kind of pinpoint where we are in the overall AP biology course, we're in Unit 8, which is about ecology. The units are broken down into smaller topics. We're going to be looking at 2 topics today: 8.5, which is about community ecology, and 8.7, which is about disruptions to ecosystems. Each topic is broken down into learning objectives, or LOs. This is what students need to be able to do with the content knowledge. Today, we're going to be working on a couple of different LOs from each of these topics. So if you look at community ecology, we would like you to be able to explain how interactions within and among populations influence the community structure, to be able to describe the structure of a community according to its species composition and diversity, and to explain how community structure is related to energy availability in the environment. And then for disruptions to ecosystems, we'd like you to be able to explain how interactions between the environment and random or pre-existing variations in populations. We'd like you to be able to explain how invasive species affect the ecosystem dynamics. We'd like you to be able to describe how human activities lead to changes in the ecosystem structure and/or dynamics. And finally, we'd like you to be able to explain how both geological and meteorological activity leads to changes in ecosystem structure and/or dynamics. The essential knowledge is the knowledge that is required to be able to perform the learning objective, and that will be my teaching part of today's lesson. So today, our topic skill pair is community ecology along with mathematical calculations. So here's what you need to know. Okay, so we're going to start off with community ecology, and we want to know that the structure of a community is measured and described in terms of species diversity. This can be quantified, and we quantify using something called the Simpson's diversity index, and it's a big scary equation. I'm going to walk you through it, so don't be scared. This formula can be found on the AP biology formulas equation sheet, and the Simpson's diversity index is 1 minus the sum of the total number of organisms of one particular species divided by the total number of organisms of all species, and all of that is squared. And I know that looks scary, but let me take you through an example because it's really not that scary. Okay, so first thing, this should make you feel really good. Students are not required to memorize the formula. It is in the formula sheet, but you do need to understand the meaning of each of the symbols. And you'll be happy to know that students will not have to do the calculation, but you do need to understand what the diversity value indicates about species richness. So if you go back to the first slide I shared with you, how we describe a community is based on the diversity of living things in that community. So let's take a look at the symbols. D is the diversity index. Capital N is the total number of organisms of all the species found in that community. The weird looking Z-like thing is the Greek letter sigma, and it means the sum of, you may recall this from the chi-square formula. Lowercase n is the number of individuals of a particular species. So those are the parts of that equation. Since the diversity index, D, is a measure of species richness, a high value of D suggests a stable and established site. A low value of D could suggest pollution, which decreases the diversity, recent colonization, so there's not a lot of different organisms in that environment. Maybe it's undergoing succession, or there could be some type of human interaction with agricultural management. So let me give you an example. In my example, Lake Catay contains 934 brown trout, 733 smallmouth bass, 34 catfish, 2003 carp, 243 steelhead trout, and 32 northern pike. And this diversity index value was calculated. I'm going to show you how I got that number to be 0.9 roughly. Okay, that's high. The closer to 1, the more diverse the community is. So this means that Lake Catay is a stable community since the diversity index value is high. So if you look at this data table, I've got all of the organisms listed in the first column. I've got the total number of organisms of each type, and at the bottom, I added all that up, and I got the total number of organisms, capital N, is 3,970. And then I just did some math. I took the number of species, in this example, it would be the brown trout, and I divided it by the total number of species, which was 3,970, and I squared that, and that's this value. And I did it for each of the types of fish in this community, in this lake, and I added it up. That's the sum of, and I got 0.055. Remembering that D is 1 minus the sum of those values, I simply subtracted that, and that's where that diversity value index number of 0.945 comes from. Again, the take-home lesson is that the higher that number, the closer to 1, the greater the diversity in that particular community. We'll practice more with this when we get to our guided practice section of this lesson. So let's talk about community ecology ecology in terms of how organisms access energy and matter within a community. And I know you guys have studied this since you were, middle school, maybe even elementary school science students. And what we're talking about here is this trophic structure or feeding structure of the community. The food chains and food webs show the interaction. So let's first look at the food chain, which is just one path of the energy transfer. And remember, and this is a common misconception that students often have, the arrow is pointing to the eater. So that means that the energy is going toward that organism. So we look at the food chain. We know that we start at the first trophic or feeding level, and this is with the primary producers. So these are plants or algae that are producing chemical energy from solar energy through the process of photosynthesis. The second trophic level are the primary consumers, and they feed directly or gain their energy directly from the primary producers. So they're the herbivores. The third feeding or trophic level are the secondary consumers. They're the carnivores, and they eat the herbivores. And then you can go on to a fourth or a fifth trophic level with tertiary and quaternary organisms feeding on the previous level. One thing I should point out is probably 5 trophic levels is maximum for any food chain. And I'm illustrating a terrestrial food chain, but please keep in mind that aquatic food chains could be an example as well. And one thing that I'm sure your teachers have taught you is something called the 10% rule. And what that means is only 10% of the energy that is derived from the previous trophic level is actual energy that goes to that organism. The other 90% of that energy goes to the business of doing life. Maybe it's lost in the process of respiration, or it's the energy to move or find a mate, or it's lost as heat. So that in this example, the herbivore, which kind of looks like a grasshopper, will only get 10% of the energy that was produced by that plant. If you look at the food web, it shows all of the possible energy paths. And again, the arrow points toward the organism that is gaining the energy. And so if you look at the bunny is an herbivore. It eats the grass. It is going to be getting its energy from the grass. If you look at the top consumers in this food web, you can see there are 2 raptors. There is an owl and there's a hawk. And those guys let's look at the owl first. The owl can get its energy by eating the bunny. It can get it from eating the squirrel, the bird, or the rodent. So there are multiple ways that top consumer can get its energy. So now let's talk about some relationships among interacting populations in a community. We can characterize these as either positive interactions or negative interactions. And I've kind of classified them and use some shorthand. So let's start off with competition. Competition is illustrated by the shorthand of negative/negative. And that means that for the organisms that are involved in competition, neither of them benefit. Okay. The next kind of column is predation, parasitism, and herbivory. You can notice the shorthand has a plus sign/minus sign. And that means that one of the organisms in this association is going to benefit while the other is harm. And I've showed you an image of parasitism in the lower left, and that where larva does not look happy being parasitized. And then the other group's niche partitioning is plus shorthand, which means that all of the organisms benefit because there's no niche overlap. And so all the organisms kind of get what they need in their own little environment there, micro environment. Mutualism is a situation where both organisms benefit, and I'm showing you a picture in the middle image of ants and aphids that are both benefiting from this association. And then finally, there's commensalism, which the shorthand is plus/0, and that 0 means neither harmed nor benefited. And you can see the right side has an example of commensalism where there's a bird on the back of a cattle, and there are many species that have this type of relationship. The bird benefits because it picks up food from the cattle, and the cattle don't see it being there. All right, so keep in mind both of the type of interaction and that shorthand. All right, 2 other key terms that I want to define for you because they're important in determining how these different populations access energy and matter in a population. So first of all, I have some images of some different organisms obtaining energy. So you can see in the left side there's bee. It is obtaining nectar from the flower. It in turn is pollinating that flower by moving pollen from flower to flower. So that's a mutualistic association. And you can see that that bear on the far right is eating the salmon, and that is predation. The bear is the predator. It's winning. The salmon, unfortunately, is the prey, and it is losing. So that would be a plus/minus, whereas the bee and the flower example are plus/plus. So the 2 terms I want to define for you are keystone species and dominant species, and a common misconception is to mix these up. So let's try to keep these straight. A keystone species is a species whose presence and role within an ecosystem has a disproportionate effect on the other organisms within the system. I can give you 3 examples. The American beaver is one example of a keystone species in North America. You can see the image on the left shows the beaver, which alters its ecosystem by building dams, and it provides little habitats for other organisms in that community. In the middle image, you see the otter, and it is an important keystone species in the marine environment, and it balances the kelp population. So without that otter in there, the kelp population overgrows and disrupts that ecosystem balance. And then finally, on the right, we see an image of some wolves, and a wolf is a keystone species in certain ecosystems, and often the top predator is also a keystone species, which is the case of the wolf. So what's the difference between that and the dominant species? The dominant species is the species that is generally the most populous or comprises the greatest biomass in the ecosystem. It doesn't necessarily have the greatest impact on that particular ecosystem. It's just the most numerous. So let's talk about some disruptions to ecosystems. A term that I pointed out that you should be familiar with is invasive species. Invasive species are species that are often introduced by humans that take hold outside of its native range. And you probably know lots of examples of this in your own areas, but I'll give you a couple of examples. The first one is the kudzu weed. It's an invasive plant species in the United States. And I know the first time I saw this, I was visiting some friends in the South, and we were driving on the highway, and this weed was everywhere. I'm like, what is that? And they were super surprised that I had never seen a kudzu weed. But its introduction has produced devastating environmental consequences. This has earned it the nickname of the vine that ate the South. And another example is a lionfish, and it threatens the well-being of coral reefs and other marine ecosystems, including the commercially and recreational fishes that depend on them. So it can impact humans' enjoyment, recreationally speaking, and then perhaps our food supply because the lionfish outcompetes those species. A little bit more about invasive species is they can outcompete native species, they can kill native plants and trees, destroy habitats, and overpredate the native species within a particular area, and that can throw the entire ecosystem out of balance. The image is just showing what's happening to a freshwater lake as this weed just infiltrates and blocks out the sunlight, so perhaps any other species is prevented from growing there. So one thing that kind of struck me is that these invasive species are considered a greater threat to native biodiversity than pollution, harvest, and disease combined. And they threaten that biodiversity by causing disease. They can act as predators or parasites. They can compete. They can alter the habitat, and they can hybridize with local species. So not just invasive species, but human populations can also impact the physical environment. So the way that we consume food, use energy, change the land, make pollution, these are all driving forces behind global ecosystem change. We do this in many ways, predominantly pollution, burning fossil fuels, and deforestation. And changes like these have triggered climate change, soil erosion, poor air quality, and undrinkable water. I'm showing you an image at the bottom where a human is burning some forest land in what is called slash and burn agriculture, where they're burning down the forest to make way for planting crops or for raising cattle. So humans have converted forest to agricultural and urban uses, exploited species, and fragmented wildlands. This changes the demographic structure of forests. It alters habitats, degrades the environment with both atmospheric and soil pollutants, and introduces exotic pests and competitors. So I just want to point out that humans can have drastic consequences and disrupt ecosystems. There are also geological and meteorological events that affect habitat change and ecosystem distribution. Natural events like earthquakes, volcanic eruptions, landslides, and floods can all be very powerful, and they can change both human and wildlife habitats. And they can change it in damaging ways, sometimes in helpful ways, as well, but most often it is in a damaging manner. Climate change can alter where species live, how they interact, and the timing of biological events such as flowering, which can fundamentally transform the current ecosystems and food webs. Climate change can overwhelm the capacity of ecosystems to mitigate extreme events and disturbance such as wildfires, floods, and drought. And I live in California, and last year we had a very difficult fire season brought on by local drought. And finally, you guys probably all heard of this, that mountain and Arctic ecosystems and the species that live there are particularly sensitive to climate change. And I'm showing you a picture of polar bears because we know that the Arctic, the increase in climate is causing the icebergs to melt and disrupting the habitat for those polar bears. So some key takeaways that I hope you got from this discussion is that the availability of resources can result in uncontrolled population growth and ecological changes. The distribution of local and global ecosystems changes over time. Humans can impact the change at local and global levels and accelerate that. Introduction of new diseases can devastate native species. Habitat change can occur because of human activity, and both geological and meteorological events can affect habitat change. And there are plenty of studies that are called biogeographical studies that illustrate these changes. So let's take a look at some practice with multiple choice and free response. Let's get back to that diversity index, remembering that it gives us an idea of the diversity of that community. And I'm giving you two different communities, a meadow and an African park. And you can see the numbers of the individual trees in the meadow community and the given diversity index of value. And again, if you want to practice this, you could totally apply that formula after this broadcast is done. You can stop it here, get the data, and then practice the math if you are so inclined. But the diversity index is 0.59. When you look at the African park with its lions, giraffes, wildebeest, elephants, and hyenas, when you calculate its diversity index is 0.25. So a question that might come from this is to distinguish the diversity differences between these two different locations. And hopefully you remember that the larger the D value, the diversity index, the greater the species diversity in that particular ecosystem. So the meadow ecosystem has a greater diversity because its D value is 0.59 compared to the African park ecosystem, the D value 0.25. So the take-home message is the higher the D value, the greater the diversity. Multiple choice practice. So let me read you the question and you read along with me. Researchers studied the diversity of macroscopic invertebrates in different areas of watershed ecosystems. They collected samples from 58 sites along 11 streams in the same geographical area. Their data were classified by stream type and can be found in Figure 1. The stream type classifications range from the perennial stream type, which always has water, to the intermittent stream type, which is dry for more than 9 months during the year. I want you to also notice that the diversity of these macroscopic invertebrates in different stream types has error bars that represent a 95% confidence interval, which means that there is plus or minus 2 standard error of the mean. So when you look at this, before we get a look at the question, if we look at the graph, we can see that the dependent variable is Simpson's diversity index, and we've got those values. Remember, the higher the value, the more diverse. And then on the x-axis, we have the independent variable, which are the type of streams, perennial, transitional, seasonal, and intermittent. And you can look at the height of the bars. I also want you to look at the error bars as well. This is going to be important in answering this question. So the question that you're asked is way down there at the bottom. You see that here. Which of the stream types has a statistically lower macroscopic invertebrate species diversity than the seasonal streams? Okay. So the question is which of the stream types, and it is statistically significant. So I want you to call your attention. So if you look at the bars, right, you can see that the transitional and the intermittent have lower values than seasonal and perennial. If you look at the error bars on the transitional overlaps on the seasonal, right? Got that? It also overlaps on the perennial. So let's take a look at the intermittent. With its error bars, it does not overlap on the seasonal or the perennial. So hopefully that will help you with your answer choice. So let's take a look at the answer choice. You can look at A being transitional streams only, B intermittent streams only, C perennial and intermittent streams only, D transitional and intermittent streams only. Let's go back. Okay, so you can see the graph. Hopefully you got your answer. So here we go. Correct answer is B, intermittent streams only. And you might have gotten thrown off if you didn't look at those error bars and choose either C or D. Okay, so let's move on. All right, let's do a check for understanding by looking at a free response question. So this starts off with a graph, and the graph is titled the variation in population density of beetles. We can see our dependent variable is the population density measured in beetles per square meter, and we can see that the y-axis, I did that again, the x-axis or independent variable is the year. We can see a legend that shows 3 species. Species A is indicated by the dashed line, species B by a solid line, and species C are the dots. Let's read the question together. According to fossil records and recent published observations, 2 species of leaf-eating beetles, species A and B, have existed on an isolated island in the Pacific Ocean for over 100,000 years. In 1964, a third species of leaf-eating beetle, species C, was accidentally introduced on the island. The population size of each species has been regularly monitored as shown in the graph above. So here is your question, and look at your task verbs. Describe the effect of the introduction of beetle species C has had on the population density of species A and B. So you want to describe the effect that introducing species C has on both A and B, and then propose an explanation for the patterns of population density that you observe in species A and B. So take a look at the graph and see what's happening. Okay, so as species C gets introduced, what happens to species A? It doesn't really change much, right? Species B, we can see that there is a decline in that line. All right, so let's take a look at the scoring guidelines for this because we're going to describe that and propose an explanation. So first of all, species C had little effect on species A because its graph didn't change. However, as species C increases, B decreases. And in order to have been awarded that point, both of the lines for species A and for species B needed to be addressed. An explanation for species A, for that dash line, there was little or no competition from species C when it came onto the island. Another way to say that is there's no niche overlap. For B declined, so there must have been competition or niche overlap. All right, so I hope that makes sense for a type of FRQ you could get. So here's one more practice with disruptions to ecosystems, a little bit more about human impact. As part of the new suburban development, a sports complex consisting of athletic fields and buildings is constructed in a formerly wooded area. Predict one ecological consequence on the local plant community that is likely to result during the site preparation and construction of the sports complex, and then justify that prediction. So we're talking about a consequence to the plant community and then justifying that. So 2 points for this, and if you see the prediction needed to be linked to the justification. So if you predicted that there would be plant death or loss of plant biomass, you have to justify that because trees or shrubs were removed. There could have been pollution that affected the growth of those plants. There may have been some plants removed that created fewer individuals available for reproduction, and then the habitat being altered could change the amount of sunlight or isolate populations. You could have said that there were reduced numbers of plant species, that there was a loss of biodiversity, again because of the removal of trees or shrubs or reduced habitat pollution. Decreased genetic diversity, the population size is reduced. We talked about population bottlenecks in an earlier lesson. Habitat loss, removal of trees and shrubs or soil loss due to the lack of ground cover. Increased success of sun-tolerant plants because shading trees were removed, or the introduction or immigration of new plant species because the new habitat was created and now we're introducing landscape environments. So let's debrief. You should now be able to explain how interactions within and among populations influence community structure and describe the structure of a community according to its species composition and diversity. You should be able to explain how community structure is related to energy availability in the environment. You should be able to explain the interaction between the environment and random or existing variations in population. Explain how invasive species affect ecosystem dynamics. Describe human activities that lead to changes in an ecosystem. Explain how both geological Biological and neurological activity leads to changes in ecosystem structure and or dynamics and perform mathematical calculations. In this case, it was Simpson's diversity index. And again, your key takeaways: evolution is characterized by a change in the genetic makeup of a population over time and is supported by multiple lines of evidence. Communities and ecosystems change on the basis of interactions among populations and disruptions to the environment. So one thing I want to point out is some common errors and misunderstandings. This was a question on this topic from last year's exam. You can look at the graph. Figure 1 shows the probability of nest success in an environment with predators. It shows warblers only and warblers and cuckoos, and then it's got a probability of nest success as the dependent variable on the y-axis. There's a lot of background information, and I'm going to read it to you, but I'd like you to read along with me. And then you can see the question that they're asking is really straightforward. This was a short FRQ, so the task verb is to describe. And maybe I'll read you the question before we go into all the details. So you can see where we're going. You want to describe the symbiotic relationship that exists between the cuckoo and the warbler in an environment without predators. All right, so let's read the background information on this question. Some birds, including great spotted cuckoos, lay their eggs in the nest of other birds, such as reed warblers. The warbler parents raise the unrelated chicks and provide them with food that would otherwise be given to their biological offspring. A researcher conducted an investigation to determine the type of relationship between the warblers and cuckoos in an environment without predators. The researcher found that the nests containing only the warblers were more likely to be successful than nests containing warblers and cuckoos. That is not the data that's shown. Remember, the data that's shown is with predators. A successful nest is defined as a nest where at least one chick becomes an adult warbler. In some geographical areas, several species of nest predators were present. Researchers have found that cuckoo chicks, while in the nest, produce a smelly substance that deters nest predators. The substance does not remain in the nest if the cuckoo chicks are removed. Figure 1 shows the probability that nests containing only warblers or containing both warblers or cuckoos will be successful in an environment with predators. In a follow-up experiment, the researchers added cuckoos to a nest that contained only warblers, which was group 1, and removed cuckoos from a nest containing warblers and cuckoos, which is group 2. So that is the data that is presented. Again, describe the relationship, the symbiotic relationship that exists between the cuckoo and the warbler in an environment without predators. So the description is that the cuckoos are parasites of the warbler. Another way to say that is that the cuckoo benefits from the relationship and the warbler is harmed by the relationship. So what was the common student misconception or gap in knowledge that was seen when this response was graded by the reader? So this was like a global, what did students miss? And if you look to the left, some responses confuse competition with symbiotic association. So you need to know the difference between competition and the different types of symbiotic relationships. Some student responses confuse predation with parasitism. They are different, so make sure you know the difference between those. Responses that identify the relationship only by the shorthand as plus or minus were not detailed enough to indicate understanding of the relationship. So the question asked for description, and the species needed to be identified based on their role in the relationship by saying something like the cuckoos benefit and the warblers harm. So this is something to keep in mind. The readers can only score you on what you write, not what we think you mean. So we know you understand the situation is that one benefits and one is being harmed, but we don't know if you understand the specific type of interaction. So a response that demonstrated understanding would be something like the symbiotic relationship is parasitic because the cuckoos take the food from the warblers and lower the success rate of the nest overall when there are no predators. So that's just way more information about your understanding than just putting the relationship as plus minus. I hope that makes sense. All right, finally, your homework, AP exam practice. All right, so this is practice for tomorrow, and I'll be with you again tomorrow. So I'll be debriefing this at the beginning of the lesson. You are given a situation where you have invasive species. In this case, they're red fire ants. They're introduced into an ecosystem that often threaten native plants and animals. You are asked to describe 3 different factors that contribute to the success of an invasive species in an ecosystem. Hopefully, this lesson gives you the background to answer this question. So this should be a short and sweet warm up tomorrow. I'd like to thank you for watching. I hope you'll join us for all of our lessons. I'll be here with you tomorrow, so be sure to complete that practice question, and we will review it as our warm up tomorrow. Thanks for watching. Bye-bye. Hi, welcome to today's AP Biology lesson. Today, our lesson will review material in Unit 1. Just to remind you that on Friday, April 3rd, an announcement was made providing the details about the 2020 AP Biology exam. The 2020 exam will be administered on Monday, May 18th at 2 p.m. Eastern Standard Time. All exams will be taken by students at home. The exam will consist of 2 free response questions. The first is a long FRQ, and we'll have a 25-minute time limit. The second question is a short FRQ, and we'll have a 15-minute time limit. Beginning today, we will be presenting lessons that will review the first 6 units in the AP Biology course. All lessons will include practice with science skills, which are relevant with any content. We will also provide practice with free response questions in each lesson. I just wanted to share with you a resource you can find at AP Central. The first thing I want to point out is at the top here where it gives you the time. That is Central Standard Time, and I reported to you that the exam would be administered on Monday, May 18th at 2 p.m. Eastern Standard Time. So you need to figure out what time zone you're in to know what time your exam will begin. There will be a makeup on Wednesday, June 3rd, and that will be for students who are unable to attend the primary test stage. And as I said, there will be 2 questions. I'll talk a little bit more about the specifics in the next slide. And at the bottom here, you can see the link to our daily course reviews from 4 to 4:45 p.m. Monday through Friday. And if you're already here, you don't need a link, but if you want to share that, you can. So just to remind you about the structure of this year's exam, the first question, which is a long FRQ, will be about interpreting and evaluating experimental results. You will have 25 minutes to write your response and an additional 5 minutes to upload it. This will make up 65% of your total exam score. Now, in previous test years, when I'm preparing my students, I usually tell them that they should plan on spending between 20 and 22 minutes on a long FRQ. So we're right in that area with a little bit more time this year to actually read and then record your response. So what will happen in this first long FRQ is you'll be given a scenario accompanied by data that is presented either in a data table or a graph. And then this is where our task verbs, those of you that have been joining us know that we talk about the task verbs in each of our lessons, and that's where these come into play. So you will be asked to describe and explain biological concepts, processes, or model. You will be asked to identify experimental design procedures, and you will be asked to analyze data as well as make and justify predictions. And you can see in the far right column the number of points that you would be awarded for each of those sections. For the second short free response question, in previous exams when I'm preparing my students, I usually tell them that they should spend between 6 and 8 minutes on those short free response questions. This one is a 4-point question, and you see that you have 5 minutes to respond to it, and then the additional 5 minutes to upload your response. This will make up 35% of your total AP exam score. In this question, you'll be given a scenario describing a biological phenomenon with a disruption. And again, here are task verbs. You need to describe biological concepts or processes, and we're going to actually practice that today. You need to explain biological concepts or processes, or describe characteristics that are represented visually. And if you were with us last week, a really good example of a concept that is represented visually is phylogeny, and we talked about how to construct phylogenetic trees and cladograms. You also need to be able to predict the causes or effects of a change in a biological system, and then justify those predictions. And so each one of those sections is equal weight with 1 point each. I just wanted to share with you a slide that indicates what the University of California system, and I suspect many other colleges are saying about this year's exam. The College Board recently announced the changes to the AP exam content and formats for the spring 2020. UC recognizes the effort that students have already applied in these challenging courses and will award UC credit for the 2020 AP exams completed with scores of 3, 4, or 5 consistent with previous years. So this should be reassuring that even though the exam format has been changed, your scores will still be awarded college credit, at least in the University of California system. And as I said, I expect other colleges to put out statements soon. So I'm Allison Katay. I'm affiliated with both UC Berkeley and Redwood High School, and I'm teaching today from my home in El Cerrito, California. So let's do our warm-up. If you were with me yesterday we talked about ecology, and I gave you a homework practice FRQ about invasive species. So if you have that homework response, go ahead and get that out if you'd like to. If you're just joining us and you'd like to try to answer this as we talk about it, then get that pen and paper handy and please do so. So the question says, invasive species such as red fire ants introduced into an ecosystem often threaten native plants and animals. You are asked to describe 3 different factors that contribute to the success of invasive species in an ecosystem. So let's take a look at how this was scored, and this was worth 3 points, and each bullet point is worth 1 point, and you can earn up to 3 points. So any one of those responses would earn a point. So saying things like, since there are no natural predators, parasites, or pathogens, the invasive species will be successful. They may have an effective aggressive mechanism to be able to be successful. They have no limit on their resources. There are no environmental inhibitors such as pollutants. They may be an R-selected species, which means they have an increased reproductive capacity and they grow really rapidly in terms of their growth rate. They may introduce a new phenotype into a large population that can be selected for if it's advantageous. They may be occupying a niche that is not occupied by any other species, so they have no competitors. Prey may lack defense mechanisms because they have encountered this new species, and they may have favorable or appropriate environmental conditions such as rainfall and temperature and sunlight and so forth. So if you selected 3 of those or wrote those 3 factors, then you would have scored a 3 out of 3. Good job. All right, so today what are we going to be working on? As I mentioned, it's our first review session. I'm sure you're excited to get to the review. We're going to take you way back to the chemistry of life. This is in Unit 1, and we're going to be reviewing 4 topics and 1 science practice. So today we're going to look at the structure of water and hydrogen bonding. We're going to look at the elements of life. I'm going to introduce you to biological molecules and properties of biological molecules. And today I'm going to focus on the carbohydrates and lipids, and tomorrow you'll get a lot of instruction on proteins and nucleic acids from Ms. Pfizer. We're also going to be looking at science practice 1A, which is asking you to describe biological concepts and processes. So if you think back to that second FRQ, that is one of the task verbs. Today we'll be looking at several free response practice questions that combine the content of each of these topics and skill. There are some terms that hopefully when you hear them, you'll remember them from prior lessons. They are dehydration, synthesis, hydrolysis, monomer, and polymer. So we're going to talk about those concepts today using those terms. By the end of the lesson, here are your objectives. You should be able to explain how the properties of water that result from its polarity and hydrogen bonding affect its biological function. You should be able to describe the composition of macromolecules that are required by living organisms. And you should be able to describe the properties of monomers and the type of bonds that connect monomers in biological molecules. And I might note that each of the type of bonds is different depending on the class of biological molecules, but they are all created and broken down using the same chemical reactions. So here's what you need to know. Just want to kind of bring you back to where we are in the framework. I told you I'm taking you way back to Unit 1, chemistry of life, and the units are broken down into topics, and each learning objective is what students need to be able to do with the content knowledge for that topic. The essential knowledge is the knowledge that's required for you to perform the learning objective. So we'll be reviewing that on our debrief. So you can see that we have 4 topics in Unit 1, and the rest of those topics will be reviewed tomorrow. So let's go ahead and start with the content. So in topic 1.1, we're talking about the structure of water and hydrogen bonding, and the idea here is that the subcomponents of biological molecules and their sequence determine the properties of that molecule. So I want to start with going way back that everything is made of matter, and matter is made of atoms. And a little cartoon for you. Hopefully that'll give you a little giggle. Since water is made of hydrogen and oxygen, we'll kind of review atomic structure using those 2 atoms. So I've got a hydrogen atom, which contains 1 proton in the nucleus, and protons are positively charged, and 1 electron outside of its nucleus in its orbital. We also have oxygen. Oxygen has 8 protons, which are positively charged, and 8 neutrons, which have no charge, and those are located in the nucleus. And then we have 8 electrons. 2 of the electrons, which are negatively charged, are in the first energy level, and the remaining 6 or valence electrons are in the second energy level. And just to remind you that it is the electrons that determine the type of bonds that are going to be formed. So the number and arrangement of electrons. So we'll take a look at that in the next slide. So when we talk about water, molecular formula is H2O. It's composed of 2 hydrogen atoms and 1 oxygen atom, and they're bonded together with covalent bonds. To remind you, covalent bonds share electrons. I'd also like you to notice that there is a negative sign around the oxygen and positive signs around the 2 hydrogens. And we're going to talk about that in more detail now. So when we see those symbols, and in this image we see this funny D right here, which is the Greek letter delta. So this delta is the symbol for a partial charge, and you can see that in the water molecule it has 2 partial negative charges and 2 partial positive charges. And if you notice that the negative charges are around the oxygen, where the positive charges are around the 2 hydrogen. And the reason for this is oxygen is more electronegative, so the electrons are shared unequally. So the oxygen ends up hugging the electrons, and they spend more time around the oxygen, creating a slightly negative side, and less time around the hydrogens, so they have a slightly positive side. We call this being polar. So water is a polar molecule. As a result of water's polarity, there are special bonds called hydrogen bonds that form. And hydrogen bonds don't just form in water, although that's what we're going to be focusing on today. But hydrogen bonds form in many other biologically important molecules. You'll see tomorrow that hydrogen bonds are found in proteins and are responsible for their secondary structure. And you'll also see tomorrow that in DNA, the 2 strands of the DNA helix are held together by hydrogen bonds. But right now, I just want you to see that what happens is that the negative side of one water molecule is attracted to the positive side of the hydrogen in another water molecule. That is a hydrogen bond. And hydrogen bonds are often represented with those dots, so you can tell that they are connected. The other thing, and you'll see in a later image, one water molecule can form up to 4 hydrogen bonds with other water molecules. So as a result of water being polar and forming hydrogen bonds, there are a number of properties of water that emerge that are very important to life on Earth. I'm going to go into more detail about the first 3 that you see there, and they are cohesion, surface tension, and adhesion. But I'd also like to mention high specific heat, high heat of vaporization. Water is a versatile, in fact, term, a universal solvent. It is excellent at dissolving polar molecules. It has an unusual phase change, which I'll explain in a minute, and water ionizes. So let's go back to high specific heat and high heat of vaporization. It means that because of these hydrogen bonds, it takes a lot of energy to change its state of matter. So it takes a lot of energy to get it to go from a liquid to a gas, and that is the high heat of vaporization. It also means that water resists change, the high specific heat. It takes a lot of energy to change water's temperature. And you're probably familiar with that if you live near a large body of water. I live right by the San Francisco Bay, and the water will moderate our coastal temperatures, whereas inland, farther away from the water, you see a much more wide range of temperatures. It gets hotter and colder. So both high specific heat and high heat of vaporization are due to hydrogen bonding and the fact that it takes a lot of energy to change its state of matter or its heat capacity. Unusual phase change is also due to hydrogen bonds, and what I mean here is that when water freezes, it goes from a liquid to a solid. It actually becomes less dense, and ice floats on water. And this is not true for other solids. Other solids become more dense. So this creates some really advantageous things for living things. For example, if you're a fish and you live in a lake, that lake will not freeze solid. The other thing, it allows seasonal snowmelt. So the fact that water does become less dense when solid is due to the way that those hydrogen bonds form. And finally, a word about ionization. When water ionizes, it produces hydrogen ions and hydroxyl ions. And you may be familiar with the pH scale. It is a measure of how many hydrogen ions are in a solution. And if there are a lot of hydrogen ions, it has a low pH. It's very acidic. acidic, and if it doesn't have as many hydrogen ions but has more hydroxyl ions, we say that its pH is basic. If the amount of ions are exactly equal, we call that neutral pH. And most of the chemical reactions in living things do occur at a neutral pH. There are some exceptions, and I believe when you get into talking about enzymes, pH will be talked about in more detail. So let's talk about those three properties that result from hydrogen bonds in a little bit more detail: cohesion, adhesion, and surface tension. Cohesion means that water molecules like to stick to each other through hydrogen bonding, whereas adhesion means that water molecules like to stick to other molecules through hydrogen bonding. And so if you look at the pictures I have for you, cohesion is responsible for the water beating up. You see that on the leaf. It's also responsible for beating up on that spider web, but it also adheres to the protein in that spider web. So that image shows both cohesion and adhesion. And then finally, surface tension results from hydrogen bonds resisting being stretched. So it creates a nice tight surface. And the image there is showing you a water strider that can actually walk on water. And if you've ever skipped rocks on a lake, you know this feature of water. And just again, reminding you that if you look at the diagram here, it's kind of reminding you why these things happen, that there are hydrogen bonds, and this water molecule is forming hydrogen bonds for other water molecules. And that is kind of the maximum number that the water molecules can account for in hydrogen bonding. So moving on to topic 1.2, we have the elements of life. And all organisms must exchange matter with the environment to grow, reproduce, and maintain organization. Atoms and molecules from the environment are necessary to build new molecules. So we're going to focus on carbon, nitrogen, and phosphorus right now. But if you see, I've got tiles from the periodic table, and you may have learned this as CHOPS: carbon, hydrogen, oxygen, nitrogen, phosphorus, and sulfur. These are the 6 elements that make up 96% of living things. The other 4% is made up of some other elements such as chlorophyll and calcium and potassium. And so they are important, but they are way less prevalent than these 6. So carbon, and we're going to talk more about this, but carbon is used to build all of the biological molecules: carbohydrates, proteins, lipids, and nucleic acids. Carbon is used in storage compounds like starch and glycogen, and also in cell formation in all organisms. It forms the cell wall in the form of cellulose in plant cells. Nitrogen is used to build proteins and nucleic acids, and phosphorus is used to build nucleic acids and certain lipids, the phospholipids. More on all of this is coming up. So I want to introduce those terms. I told you should be familiar with it just in case you forgot. And these are the terms monomer and polymer. Monomer is the subunit that serves as a building block of a polymer. So mono means one, poly means many. So a polymer is a larger molecule that's made up of many similar or identical monomers that are linked together by covalent bonds. And I'm going to give you examples in each of the groups of organic molecules. So the monomers of carbohydrates are monosaccharides, which are also known as simple sugars. And you're probably familiar with glucose, but there are other simple sugars that I'll point out in just a moment. And I'm showing you the ring structure of glucose. The monomers of proteins are amino acids. And I just have a generalized image of an amino acid there. We'll talk more about what those components are in just a minute. And finally, the monomers of nucleic acids are nucleotides. A nucleotide is composed of 3 parts: a phosphate group, which is why we need that phosphorus, a pentose sugar, and a nitrogenous base. And that nitrogenous base is why we need the nitrogen. So let's take a look at carbon. This flowchart kind of shows you why carbon compounds are so important to living things. They make up carbohydrates, which consist of sugars and starches, and as I mentioned, cellulose, which is a structural component made of carbon, hydrogen, and oxygen. They are often called hydrates of carbon. Quite literally, there's twice as much hydrogen as carbon and oxygen. We also have lipids. Lipids consist of fats, waxes, oils, and steroids, and what they all have in common is they are nonpolar, and they are composed of carbon, hydrogen, and oxygen, and in the case of phospholipids, phosphorus. Nucleic acids consist of nucleotides, which contain carbon, hydrogen, oxygen, and phosphorus, and proteins consist of amino acids, which contain carbon, hydrogen, oxygen, and nitrogen. So I want to talk a little bit about the 2 reactions that are really important in building and breaking down our macromolecules, and these 2 reactions are called hydrolysis and dehydration synthesis. And you probably, from those names, hydro and dehydration, understand that they have something to do with water, and you would be correct. Okay, so dehydration synthesis literally means the dehydration or removal of water. And the way this works, if you look at the monomers, you've got a monomer which has a hydroxyl or OH group indicated in blue, and you've got a second monomer which has a hydrogen that is showing you it's an OH group, and the hydrogen is in blue. And so what's going to happen is that we're going to remove the water by combining the hydroxyl from one monomer and the hydrogen from another, and that forms water. And then the resulting monomers are linked together to form a polymer, and they are linked together by a covalent bond. Okay, I'm going to give you specific examples with macromolecules in a minute, but this is kind of a generalized view of dehydration synthesis. I should also tell you that dehydration synthesis is known as condensation. And the way I like to think about it is you're condensing those 2 monomers by removing water to create that polymer. The opposite reaction is called hydrolysis, which literally means using water to lyse or break apart a larger molecule into smaller molecules. So if we've got that polypeptide or polysaccharide or nucleotide that is linked together, if we add water, we hydrolyze it, we're going to break that bond, and we are going to split those monomers into 2 and re-add that water to each monomer. The hydroxyl group will go to the first one, and the hydrogen will go to the second one. So let's take a look at hydrolysis in actual biological molecules. So I've got a disaccharide, which is a 2-unit sugar. So it's composed of 2 monomers. And my example is sucrose, which is probably what you put in your coffee or tea, and it's cane sugar. So we want to break that down so that we can digest it better. By the way, disaccharides are kind of how the sugars travel around. They're the transport forms of sugar. And we want to break it down. It does require enzymes and energy. The enzyme in this case is called sucrase. We add water, we're going to hydrolyze it, and when we do that, we are going to break it down into 2 glucose monomers. Okay, well, excuse me, in the case of sucrose, we would get 1 glucose and 1 fructose. In the case of maltose, which is another disaccharide, you would get 2 glucose monomers. We can also hydrolyze proteins. So what I'm showing you is a dipeptide, and what we're going to do is we're going to add water to bust apart that carbon-nitrogen bond right here, which is called a peptide bond, and I'm going to show you in a later slide how that bond actually gets formed through dehydration synthesis. But we add the water, and we end up with 1 amino acid and another amino acid. If this was a polypeptide, we're only showing you 2 parts. It actually is, because here's another peptide bond. This is going to be the peptide, and this is the amino acid. But you can see that what happens to the water is the hydroxyl group goes on this end, and the hydrogen goes on this end. And I'm going to give you some more information about what these ends are called in a couple of slides. So let's take a look specifically at nucleic acids and proteins in just a minute, because the structure and function of polymers are derived from the way their monomers are assembled. In nucleic acids, biological information is encoded in the sequence of those monomers. In other words, each nucleotide, which in turn has that 5-carbon sugar, a phosphate, and a nitrogenous base, the sequence of those is going to determine the biological information. Just to note that DNA and RNA differ in both structure and function, and as I mentioned, you're going to get a lot more information about this tomorrow. In proteins, the specific order of amino acids in that polypeptide, which is known as the primary structure, the sequence of those amino acids determines the overall shape of the protein. Amino acids have directionality. One end has an amino group, and the other end has a carboxyl group, and I'm going to illustrate that with an image in just a minute. The R groups, which are called the functional groups of the amino acids, can be characterized by their own chemical properties. So they can be classified as hydrophobic, hydrophilic, or ionic, and the interactions of these R groups determines the structure and function of that region of the protein. And again, you'll get much more on this tomorrow. So let's take a look at how we build a nucleic acid. So we are going to do a condensation or a dehydration synthesis reaction here. And so what you've got is 1 nucleotide. It has its phosphate group, and there's our phosphorus. It's got a pentose or 5-carbon sugar, and it's got a nitrogenous base. And we are are going to remove a water molecule. We're going to remove a hydrogen from this nucleotide and a hydroxyl from this nucleotide. That will form water, which you see over here, and those two nucleotides will be linked together through a covalent bond, and this is called a phosphodiester link. This is just showing you that nucleotide is composed of a phosphate, a 5-carbon sugar, and a nitrogenous base, and this just kind of puts it all together for you with the overall structure of a nucleic acid. So since this has 2 strands, it is going to be DNA, and I also know that by the 4 types of nitrogenous bases here. One other thing to point out is you can see those 2 strands of DNA are connected by hydrogen bonds, so hydrogen bonds don't just occur in water. Now let's take a look at how monomers of amino acids are put together to build a protein. This is the generalized structure of an amino acid. You can see that it starts with a central carbon, and remember that carbon has 4 covalent bonds available, and it's got an amino group or terminus, NH2, and it's got a carboxyl group or terminus. Then it's got a hydrogen that takes up its 3rd covalent bond, and the R group is the side group. This is interchangeable. So each of the 20 naturally occurring amino acids has a different R group, and that will determine the characteristics of that amino acid. It can be simple like a hydrogen, which is the case of glycine, or it can be a complicated ring structure, which you might see in an amino acid called phenylalanine. So let's take a look at a condensation or dehydration synthesis reaction that is used to build polypeptides. So here's our first amino acid, and here's our second amino acid. And again, we've got our amino end and our carboxyl end. So what's going to happen, this is kind of funny, but what's going to happen is the hydroxyl from the carboxyl group is going to join with the hydrogen from the amino group to form water. When that happens, notice what gets bonded together, the carbon and the nitrogen. And that carbon-nitrogen covalent bond is found only in proteins, and it is called a peptide bond. That's a really important thing to remember. So let's take a look at some carbohydrates and see how their structure and function is derived from the way that they're assembled. So again, just to kind of give you a flow chart, over here we've got our group, which is the carbohydrates. They can be either simple or complex, and you might know this from your own diet. If you eat simple carbs, you're going to get readily available energy, and that comes from your monosaccharides, which are glucose, fructose, and galactose. And disaccharides are kind of the way that they're transported around your body or a plant body. It's how the organism kind of moves their sugars from one place to another. And there's maltose, lactose, and sucrose. And you may be familiar with lactose. It's milk sugar, and you may be familiar with it because you've heard of something called lactose intolerance. And that means that individuals who are lactose intolerant do not have a functional enzyme lactase to break it down using hydrolysis. So more on that another time. I'm sure that will come up again. So if we look at the image where I've got starch versus cellulose, right? Starch versus cellulose is all in how those monomers are assembled. And what I would like you to see in starch, that is a storage form or a complex polysaccharide. Plant starches are a little bit different than animal starches. Glycogen is an animal starch. And in this case, when this flowchart is talking about fibers, we're mainly talking about cellulose, which is the bottom image. So I'm sure you can notice that there's a difference between starch and cellulose. And if you look kind of when I draw an imaginary plane across these ring structures, the bond is below that plane. And it turns out that when we link our glucose monomers together using dehydration synthesis, if that bond occurs below that plane, we're linking alpha glucose monomers, and we can easily digest those. We have the enzyme that can break those bonds and release energy, and so we can use that for an energy providing molecule. If you look down here at cellulose, if you look at that imaginary plane, the bond is above the plane. And the reason, and the way I remember that is these beta glucose monomers that are linked together, and I remember this as beta above. And it turns out that humans do not have the enzyme that recognizes that what we call a 1,4 beta glucose link. And the reason that we can't digest cellulose is we don't have that enzyme. So cellulose makes up the bulk of our waste products because we can't digest it. Of course, there are organisms that can. All you need to do is think about termites, and they can get their energy from eating wood. So let's take a look at one more example of the carbs and their sugar monomers. In this case, we're showing you another dehydration synthesis or condensation reaction. In this case, we have 2 monosaccharides, glucose and glucose, and we're going to build a disaccharide maltose. We're going to do that by removing a water molecule. When we form this is that 1,4 link, and this is called a glycosidic linkage. Remember that this reaction, dehydration, because we're removing water, requires energy and the correct enzymes to build that disaccharide, and then you would just keep doing that to build your polysaccharide. I want to talk about the last group of biological molecules that I'm going to get into detail about, and those are the lipids. Again, let's start this flowchart here. So lipids are all grouped together because they are non-polar, and you've got fats, sterols, and phospholipids. Fats are composed of triglycerides, and I'm going to show you how you can build a triglyceride using dehydration synthesis. Triglycerides are stored fat or consumed fat. They make up cell membranes. Specifically, phospholipids are important in cell membranes, and I'm going to go into detail about that. They're also extremely important in creating the myelin sheaths that cover your nerve axons. Sterols are very important. Sterols are steroid molecules which are characterized by being nonpolar, which is why they're classified with lipids, and they are 4 linked carbon rings. Probably the most familiar one you know is cholesterol, and cholesterol is important in cell membrane structure. It helps to maintain the fluidity and provide structure for that cell membrane. We tend to think of it in dietary terms, and a lot of people think it is bad. And while it is in fact, there is one type of cholesterol that is not good for you in terms of your blood vessels. Your blood is polar, and these cholesterol molecules are nonpolar, and they tend to get stuck to the inner lining of your blood vessels, and that can cause problems. But cholesterol is a really important molecule. It is the basis of your sex hormones, testosterone and progesterone and estrogen. It's also the basis for vitamin D. And then finally, phospholipids. Phospholipids are super important as part of every lipid bilayer, which makes up cell membranes. So that's kind of the breakdown of the lipid flowchart. They are nonpolar, and that's an important thing to remember as to why they're linked together. There are 2 things I want you to know. Differences in saturation determine the structure and function of lipids. So you've probably heard in terms of diet that there are saturated fatty acids and unsaturated fatty acids. Saturated fatty acids have no double bonds. They tend to be solid at room temperature, and they're more animal fats. In terms of your diet, they're less healthy for you. The unsaturated fatty acids do have double bonds. You can see that is indicated by the green portion of that molecule, which means that they're a little bit more flexible. You can also have polyunsaturated fatty acids, which have more than 1 double bond. These tend to be your plant fats and or oils. They tend to be liquid at room temperature. And then finally, phospholipids. Phospholipids are really important molecules because they contain polar regions due to that phosphate. You can look up here. The phosphate is shown in blue, and it's hydrophilic, meaning that it likes to be near water. Hydro means water, philic means affinity for. But it also, because of the nonpolar fatty acid chains, is hydrophobic at the same time. And this makes a useful molecule in creating that barrier as part of the cell membrane. So I've showed you the cell membrane. It's not a great image, but you can see that this is the phospholipid bilayer. The phosphate heads are at the top, and they point both into the cytoplasm and out to the surface of the cell membrane. And then we've got all of these fatty acid tails, which are pointing toward each other because they're hydrophobic. One other thing, you can see a cholesterol molecule is embedded here, and that gives it a little bit of fluidity. You can see one here. It's not labeled, but that's also a cholesterol molecule. And then one other key component, as long as we're talking about cell membranes, is proteins. And proteins can span the entire cell membrane, or they can be on the surface. And those are called peripheral membrane proteins versus the integral membrane proteins that you see there. Okay. So let's talk about how you build a triglyceride. So I'm showing you a fatty acid. In this case, it's palmitic acid, and you can see that this is a saturated fatty acid because there's no double bonds. You can also see that it's got this carboxyl group at the end, and it's going to give up a hydroxyl group. And then we've got a glycerol molecule, which is a 3-carbon alcohol, and it's going to give up a hydrogen when we use dehydration dehydration synthesis. To build a triglyceride, you need 3 fatty acids. So what's going to happen is we're going to do that. We're actually going to remove 3 water molecules. So you can see that that is a triglyceride. You've got the 1 glycerol molecule, and what happens is this bond here is called an ester link. So where the water was removed in the dehydration synthesis, you get that bond, which is called an ester link, and that is an example of a triglyceride. Let's get some practice. And I have 4 practice FRQs, 1 for each topic. I'm not sure we're going to get to all of them, but if you want to go back and look at these once this is recorded, it would be excellent practice for you. So let's do the first one and the third one, okay? And we'll save the second and the fourth one for you. So topic 1.1 is the structure of water and hydrogen bonding. And you've got 2 pictures. You've got a beautiful picture of ice, and it illustrates those water molecules being bonded by hydrogen bonds, and they're actually separated, which is what makes them less dense, and they float in liquid water. And then you've got an image of liquid water. The question is asking you to describe why hydrogen bonds form between water molecules. So go back. Water is made of 2 hydrogens and 1 oxygen, and because the oxygen is negative and the hydrogens are positive, they are going to be attracted to one another, and that is the hydrogen bond. So your response should indicate that the slightly negative oxygen atom in one water molecule is attracted to the slightly positively charged hydrogen atoms in another water molecule, so that those hydrogen bonds form between the 2 water molecules. I'm going to skip this one for now, but you should go back and practice it. It's about carbs. So first of all, you want to discuss the structural components of the group, then you want to identify 2 examples and briefly describe their function, and then also explain the process that joins these polymers from their monomer subunits. So I'm going to skip that, but the scoring guidelines are here. You can go back to that. Let's take a look at 1.3. This is a question that uses membranes as a vehicle or a way to get you to talk about macromolecules. So you're asked to identify 3 macromolecules that are components of the plasma membrane in a eukaryotic cell and discuss the structure and function of each. And you should be able to do 2 super easily just based on this review. The third one I mentioned, but you may have to rely on prior knowledge for this one. So, and I know this table is not very good, but you can see that definitely we've got our phospholipids, which is a glycerol and 2 fatty acids, and the polar phosphate group is attached. And this is what makes those cell membranes selectively permeable. It restricts some molecules from passing through while allowing others. Cholesterol is another one. It makes the membrane fluid. It's got a 4 carbon ring structure. And then you can mention proteins. And as I just briefly mentioned, they are embedded as integral membrane proteins or they are peripheral membrane proteins. There's a 4th one for you, but I'm going to let you do that on your own. It's talking about how enzymes are constructed. And the key to this one is to remember that enzymes are examples of proteins. Okay, so quick check for understanding is getting practice with the science skill described. And it puts it together again with a cell membrane context. So estrogens are small hydrophobic lipid hormones that promote cell division and the development of reproductive structures in mammals. Estrogens passively diffuse through the plasma membrane to bind to their receptor proteins in the cytoplasm of target cells. Describe 1 characteristic of the plasma membrane that allows estrogens to passively cross the membrane. So you need to understand about the plasma membrane, and you need to understand about estrogens, which are lipids, right? So because estrogens are nonpolar, they slip right through the spaces between the phospholipid fatty acid tails, which are also nonpolar, so they enter easily. And then just to show you a award-winning full credit student answer, let's look at the top one. Plasma membranes are formed of phospholipid bilayers with a middle region made of hydrophobic tails. Because these are hydrophobic, the estrogens can diffuse between the tails into the cell. This allows small and uncharged or hydrophobic molecules to diffuse into the cell passively. So that's a full credit answer. So remember that your task verb describe is to give a picture of something in words. Describe it as if someone could not see it. All right, so really quickly with your debrief, hopefully now you're able to explain the properties of water that result from its polarity and hydrogen bonds. You're able to describe the composition of macromolecules and the properties of monomers and the type of bonds that connect them in biological macromolecules and describe biological concepts and or processes. 2 key takeaways: living systems are organized into a hierarchy of structural levels that interact, and the highly complex organization of living systems requires constant input of energy and the exchange of macromolecules. I'm going to skip this common errors and misunderstandings slide in the interest of time, but please view it if you stop and view the recorded one. I want to give you your homework. Your homework is due tomorrow, and Ms. Beiser will be with you tomorrow to go over it. This is a carbon question. It tells you that carbon is very important in living systems, and the following structures are examples of 2 different categories of macromolecules. You are asked to identify each category, category 1 and category 2, and then describe how each type of macromolecule is important to the structure and function of living things. So I have presented both of these, so hopefully you can easily identify them and then describe their function. All right, so thanks for watching. It was great being with you today. I hope you'll join us again. Make sure you complete that practice question. We'll review the answer in the next lesson. Have a great rest of your day. Bye-bye.
Hi, welcome to AP Biology. I hope you can see me. All right, I'm Gail Pfizer. I'm from Darnell Cook Mendel High School and Florida State College in Jacksonville, Florida. And I'm here to talk about and help prepare you for our May 18, 2020 AP Biology exam that will be at 2 o'clock p.m. Eastern Standard Time. And there's going to be 2 questions that are 2 free response questions. And the first question is going to be about interpreting and evaluating experimental results. And you're going to have 25 minutes to read and respond, and then 5 minutes to upload. This is going to be 65% of your exam score. This is going to be based on an authentic scenario accompanied by data in a table and or graph. And it's going to be built with 4 parts, okay? And it's going to be 1 to 2 task verbs at each of those parts. So let's talk about each one of those briefly. In A, you're going to describe and explain biological concepts, processes, models, and apply context. In B, you're going to identify and justify experimental design procedures. And C, we're going to analyze some data. And D, we're going to make and justify predictions. So let's talk about those task verbs really quickly. Describe, you're just listing the characteristics of whatever it is you're talking about. Explain is going to be the how or the why. Identify is going to be really short, just what is it. And then don't forget justify. You got to make your claim. You got to make prove it with your evidence, and then support it with some reasoning, some biological support there. All right, and then analyzing data, there's going to be a table or a graph that you're going to analyze. All right, in question 2, it's conceptual analysis, and it's going to be 15 minutes for that response. There's going to be 5 minutes to upload, and that's going to be 35% of your score. It's going to be based on an authentic scenario describing a biological phenomenon with a disruption, and it's going to have 4 parts. It's going to be 1 task verb in each of those parts. The first one's going to be describe biological concepts or processes and apply context. B is going to be explain biological concepts or processes and apply context. C is predict the causes or effects of a change in the biological system, and D is going to be support or justify claim with reasoning and or there's that evidence again. So let me talk about our lesson for today. We're going to be just talking about, it's going to be very narrow focus. We're just going to be talking about proteins and nucleic acids, and there's going to be a lot of work with the science practices built in. Excuse me. All right, so here's our warm-up. And our warm-up is from our homework from yesterday, and which was from, if you're just joining us now, it was from our previous lesson, which had to do with properties of water, monomers, polymers, and then carbohydrates and lipids. So here's a question that they have for homework, and if you're just joining us for the first time, then we're going to go ahead and give you a moment between the big reveal of the answer of this question. Okay, carbon is a very important element in living systems. The following structures are examples of 2 different categories of macromolecules. Identify each category and describe how each type of macromolecule is important to the structure and function of living things. So you see category 1, and I think that you can see my cursor move, I am going to count. I'm going to tell you how I can tell what this is. Okay, so right now, let me see here. Okay, so there's the first carbon. Here's 1, 2, 3, 4, 5, 6. Okay, so I know that there's 6 carbons. My H's. There's 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, 11, and 12. So 6 carbons, 12 hydrogen. And now for our oxygens, there's 1, 2, 3, 4, 5, 6. So there's 6 carbons, 12 hydrogens, 6 oxygens. That's a 1-to-1 ratio. I know that's a carbohydrate. Okay, by looking at. All right, and in category 2, I see 2 long long chain fatty acids, and I see a phosphate group here. So I know I must be in the lipid category, and it must be in this must be a phospholipid, right? All right, let's look at the scoring guideline. All right, so category 1 is a carbohydrate. It's a monosaccharide, and then you could be specific and put the word glucose in there. It functions to provide readily available energy to organisms, and also functions as a monomer of polysaccharide. In category 2, it's a lipid, it's specifically a phospholipid, and it's amphipathic, which means it has a polar end and a non-polar end, and then it also functions as a component of the cell membrane. So we have at the bottom, those are out of in my classroom, and my kids built those this year. And this is a very popular assignment, so you may have them in your classroom also. This is a dehydration and with carbohydrates, dehydration synthesis, and then we have some triglycerides hanging over here on the cabinet. Okay, so here's what you need to know. So learning objectives, this is what you need to be able to do, excuse me, to apply to both proteins and nucleic acids. So we need to, I'm so sorry for that noise, but we're afraid about the sound, and so I need to keep my email open. All right, so describe the composition of macromolecules required by living organisms, which that's where you're going to talk about the elements of each, and then describe the properties of monomers and the types of bonds that connect the monomers and biological macromolecules, and that's where we're going to talk about dehydration synthesis and hydrolysis and covalent bonds, and then describe the properties of monomers and types of bonds that connect the monomers and biological macromolecules, and this is where we're going to talk about the structure of nucleotide, the primary structure of amino acid, and those R group properties, and then explain how it changed. See how I highlighted that in red? Where do you think this is going to fit? This is going to fit in 2C, which had to do with predicting a change. Okay, I not only want you to learn well how to describe, explain, but also be ready to talk about what happens if you don't have it or what happens if you change it. Okay, no matter what you're talking about. All right, so how a change in the subunits of a polymer may lead to changes in structure and function of the macromolecules. We're talking about in this section nucleic acids, directionality, the 5' to 3', how DNA is anti-parallel, A goes with T, C goes with G, all that kind of stuff. Amino acids, we'll talk about the carboxyl terminus and how at the end that we add amino acids to develop polypeptides, and then we'll talk about 4 levels of protein structure. And we're also going to end it with describing the structural similarities and differences between RNA and DNA. All right, so first we're going to talk about protein. So these are the molecules that you need to build protein. You need carbon, hydrogen, oxygen, nitrogen, and sulfur. And you'll find the sulfur. I have a diagram of cysteine right here. Cysteine and methionine have the sulfur included in their molecular structure. So here, let's go ahead and talk about how this is built. So right here, this is our central carbon, okay, and then it always has this hydrogen, and then this group right here, this is our R group or our R chain, and all of those will differ from amino acid to amino acid, and that's what gives them their different properties. On the right here, we have our amino functional group, okay, and on this side we have our carboxyl functional group. All right, okay, so let's do dehydration synthesis first, and then we'll do hydrolysis, and we'll do both of them with protein. Okay, so amino acids are monomers that bond together with peptide bond, which is covalent bond to form a polypeptide, which is polymer through the process of dehydration synthesis. So let's look at here's one amino acid, here's another amino acid. We're going to take the hydroxyl group and this H, and in the reaction we're going to pull it out, and we're going to make a molecule of water. Okay, so now we have these 2 amino acids joined together with covalent bond. You see this little gray arrow? That's where it happened, and that's called a peptide bond. All right, the process of hydrolysis is just the reverse. We're going to break apart the polypeptide into amino acids. So we're going to take this. There's 6 amino acids in this diagram. They have a reference R 1 through 6, and so we're going to take it in a reaction with an enzyme. We're going to put water in, and we're going to bust it apart, and you see an OH and H. There's the OH coming back on this carboxyl group, and an H to this amine group, and we just took a 6 amino acid strand and made it into 3 amino acid strands. Okay, so I talked about this, and this is the R group. Remember, this diagram I circled them all. There's 1, 2, 3, 4, 5, 6 of them, and those are different in all the amino acids. The R group of an amino acid can be categorized by its chemical properties: hydrophobic, hydrophilic, or ionic. The interactions of these R groups determine the structure and function of that region of the protein, and we'll talk about those functions a little bit as we move through this unit, but specifically when we get to their own unit in our units 1 through 6. Like we'll talk about cells, and we'll talk about proteins and cells, and we'll talk about the proteins and cell communication and the proteins and enzymes. All right, so I wanted to bring this up so you can understand why these R properties, R chain, the properties are so important. This is glutamic acid, and on the left, this one right here, and see this R chain right here, it's very much different than that one, right? Well, this one has a hydrophilic property, and this one has a hydrophobic property. So this one's hydrophilic. It means that it enjoys being on the outside of the protein and interacting with water. Okay, and if you're hydrophobic, that means you don't want to be on the outside of the protein, you want to be on the inside of the protein, away from water. And so this is a big problem if you have one substituted for the other, and this is in particular in the case with sickle cell. And if you want, I talked a lot about mutations, protein synthesis, and the substitution of these 2 amino acids in our natural selection video that was on March 26th. If you want to go back there for the whole story, there's a lot of good material in those videos that have to do with science practices and other material also. All right, so anyway, so sickle cell, you're supposed to have glutamic acid, and anyway, you end up with valley, and that's how you have the non-functioning cell. Okay, all right, so amino acids have directionality with an amino end and an H2 terminus and a carboxyl COOH terminus, and here is the amine. There's the NH and H there, and here's a C, 2 O's in the H. This is a hydroxyl group. This is a carboxyl group altogether. Okay, so we have if we're adding amino acids, they're going to be joined to this end. Let's say, and they're also in that same video. There's also protein synthesis and transcription and how you read the genetic code from there, all that there. All right, and we'll talk about that again also in protein synthesis when it comes up. All right, so we're going to add amino acids to this end when we are at the A and P site on the ribosome and adding amino acids to make a long polypeptide. All right, so proteins comprise linear chains of amino acid connected by the formation of covalent bonds at the carboxyl terminus of the growing peptide chain. All right, so we're going to talk about the 4 levels of protein structure now. The primary structure is determined by the sequence order of their constituent amino acids, and the specific order of amino acids in a polypeptide, which is the primary structure, determine the overall shape. So here, each one of these dots, okay, those represent amino acids. For example, I just drew these names on. Okay, there's arginine, there's valine, there's glutamic acid. Here's our carboxyl group. Here's our amine group. So this is where it begins, and this is where more amino acids are added to this end. That's our protein structure. It kind of looks like a pearl necklace, somewhat. All right, secondary structure. Secondary structures arrive through local folding of the amino acid chain into elements such as alpha helices and beta sheets. So if you look at here is see this was that strand, but now it's in this alpha helical conformation, and so we have these black dots. They represent hydrogen bonds, and that's what's causing this to take the shape. Same with the beta pleated sheet. The dots there that representing hydrogen bond, and that's causing it to form this particular structure. All right, the tertiary structure. This is the overall 3-dimension shape of the protein, and often minimizes free energy, decreases the free energy to increase the stability. Okay, so we have hydrogen bonds, disulfide linkage. You might see that as disulfide ridge or disulfide bond, depending on what you're reading, and ionic bonds and hydrophobic interactions. So all of those are happening in this particular formation of this structure. All right, here's our quaternary structure. It arises from non-covalent interactions between the multiple polypeptide units. So all of those that we just talked about formed this tertiary structure, and then we have other interactions between this tertiary structure and this tertiary structure for the quaternary structure. And here they are all together. How this particular end result here will determine the function of it. Okay, all right, so we have primary, secondary, and then tertiary, and then quaternary. All right, I talked about this a moment ago. Some major functions of our proteins we'll discuss a little bit now, and much later, catalysis by enzymes, cell structure, and communication between cells. And now for our nucleic acid. Okay, so we'll go through it the same way we just did proteins. Okay, we'll first talk about the molecules that we need to build a nucleic acid. We need carbon, hydrogen, oxygen, nitrogen, and phosphorus. So here's in that phosphate group. That's where we're going to need the phosphate. So let's take a look. We can go ahead and do a little bit of breaking. And this is a nucleotide. Okay, and this is a 5-carbon sugar. This is a nitrogenous base, and this is a phosphate group. And we'll talk about a little bit more in depth. Oh no, let's go back. Let's stay there for a minute. No, we'll go. So let's do for, I'll talk about the structure in a minute, but let's learn how to put them together first. Okay, so dehydration synthesis, we're going to form covalent bonds. We're going to take nucleotides, which are the monomers, and we're going to form a polymer, which is called the nucleic acid, whether it's RNA or DNA. So here's our nucleotide, here's our phosphate group, our 5-carbon sugar, and our nitrogen base. Now that's where I need to talk to you a little bit more about the nucleotide itself. Okay, so if you're looking at this 5-carbon sugar, here's carbon 1, 2, 3, 4, and 5. Okay, that's where the 5' to 3' direction language is coming from. It's talking about the orientation of the carbon on the 5-carbon sugar. So our phosphate end of one nucleotide, this one right here, this will be nucleotide 2, is going to bond with the hydroxyl, the 3' hydroxyl end of the previous nucleotide. So this H and that OH will go through a reaction to form a covalent bond called a phosphodiester bond, and then we'll have the two nucleotides together. And in the same process of dehydration, when we're talking about lipids or carbohydrates or amino acids joining together, that molecule of water is going to be formed. Okay, so that's dehydration synthesis for nucleic acid. All right, so now let's talk about everything we can about this nucleotide. Okay, we see that it has a 5-carbon sugar, and we'll walk through that again. There's carbon 1, 2, 3, 4, and 5. Okay, the phosphate group, which is negatively charged, is on the 5' end, and then the 3' end, that's where we're going to be joining the next nucleotide. Okay, all right, so what else do I want to talk about? The nitrogenous base. There's two types of nitrogenous bases. We have a purine and we have pyrimidine. This is my pyrimidine, and I know that is because it's a single ring. If it was double, then I'd know it's a purine. And my pyrimidines are my thymine, cytosine, and my uracil. Okay, and then my purines are my adenine and my guanine. All right, so when we add a nucleotide to a growing strand, this would be a DNA polymerase. Probably it's not labeled, but here we have a 5' end of this strand, 3' end of this, and then we have our complementary base pairs. We have our A's and our T's and our C's and our G's. So the next one would be, if this was an A, then that would be a T that's coming in. It's going to be joined to this 3' end, and a covalent bond will be made. And I'm going to go through the whole process of that nucleotide being added when we get to DNA replication. Okay, all right, so it's called anti-parallel. So if you will look, I tried to attempt flipping some molecules around. So let's look at, here's a nucleotide, here's our phosphate group, here's our 5-carbon sugar, and here's our nitrogenous base. There's our carbons 1, 2, 3, 4, 5. Now, if you, let's see if I can do this. I do this in my flash. If you took it and flipped it upside down and around, so two turns, up and down and around, then you're going to end up with this orientation. And if you look, there's our 1, our 2, our third carbon, 4, and 5, and everything is on the opposite side and turned and upside down. Okay, that's called anti-parallel, and that is how the strands are oriented to form our double helix. And let's see, we talked about our 5' is on our phosphate end, 3' is on our hydroxyl end. Okay, all right, so now let's talk about the hydrogen bonds between the nucleotide. Cytosine nucleotides pair with guanine, and they have 3 hydrogen bonds between them, and then adenine and thymine, they have 2 hydrogen bonds between them. Remember that in a question that I'm going to ask you in just a little bit. Okay, so these are things similar between DNA and RNA. They both have 3 components. They have 5-carbon sugar, they have phosphate group, and nitrogenous base. Okay, they both have nucleotides that are joined together with a covalent bond, and they're both linear molecules, and they both have 5' and 3' end, and they both have nitrogenous base, which are perpendicular to the sugar phosphate. Okay, all right, now the difference is RNA, here's RNA, single strand, DNA, double strand, 5-carbon sugar is called ribose in RNA because it's RNA is ribose. Okay, and DNA has double strand, and that's deoxyribose, and then your DNA is going to have thymine, and then your RNA will have uracil and not thymine, and then DNA has also that anti-parallel directionality. All right, so that's it for the content. We'll apply it in some questions in just a little bit. Let's talk about what science skills we're going to do. We have 60B. We're going to do a lot, but these are some specific ones that are unique to this presentation. All right, so 60B, we're going to predict the causes or effects of a change in or disruption to one or more components in a biological system based on a visual representation of a biological concept, process, or model. And then 2B, we're going to explain the relationships between different characteristics of biological concepts, process, or models represented visually. So right now you should be thinking, oh, I see this in 1A and B and 2A and B on our test. That's what we're going to be doing this explaining, and then the 60B, that's going to fall nicely into that C of that question too. All right, this is one of my favorite questions, and it's from probably the first AP exam since the revision that happened in 2012. Okay, so which of the following correctly illustrates a dipeptide and an amino acid in the optimal position to form a tripeptide? So let's take a look at A. All right, so here's our dipeptide, and there's our amino acid that we want to add to this, and same situation all the way down. So let's start from the bottom. So we have an amine end here, and there's an amino functional group. This looks like a methyl functional group. Can we bond those together? Is that how we do this? No. And here we have an amino end, will that work? No. And then we have a carboxyl, and carboxyl, that's not gonna work either, but look here, carboxyl and our amine end, and that will bond together. That's the way we add amino acid to the strand that we're creating, is to that carboxyl end. So A would be our answer. Okay, I know the bottom of this is hard to read, and I'll read it for you. Here we have this image that I keep using with a cysteine. So let me read this to you. Researchers compared similar proteins from related organisms in different habitats. They found that the proteins from organisms living in harsh environments had a greater number of cysteine amino acids than did proteins from organisms not living in harsh environments. The structure of cysteine is shown. Bonds can form between the sulfur atoms of different cysteine amino acid, that SS bond, and that is a strong bond, covalent bond. All right, so which of the following best describes the effect of a greater number of cysteine amino acids on the stability of the protein? All right, so listen carefully to the choices. We have A, the change has no effect on the stability of the protein because only one type of amino acid is involved. B, the change leads to increased protein stability because of an increased number of SS bonds in the tertiary structure of the protein. C, the change leads to a decreased protein stability because of an increased number of SS bonds in the tertiary structure of the protein. And then D, the change leads to an increased protein stability only when the added cysteine amino acids are next to the other cysteine amino acid in the primary structure. Okay, so we're going to have increase, so we can go ahead. If I had a pen, I would be crossing out this decreased. Okay, no, C's not it. A, no effect, that's not it. Now, when I do, they have to be right next to each other in line. Like, did this have to join? Did another cysteine have to join right here for that? No. So the choice here that works for us, the change leads to increased protein stability because of an increased number of SS bond in the tertiary structure of the protein. Okay, so that's our answer B. All right, now I'm going to pause on these because I'm going to give you time to answer these. This is another one of my favorite questions. I always like to include this on everything that I give my students. They'll be doing this tomorrow. It was number 56 on I think of the first exam. Also, there were some really rich, good questions on that test. All right, so number 56. A culture of spirogyra and autotrophic alga is maintained in a water solution containing dissolved carbon dioxide and a source of phosphate but lacking nitrogen compound. That's key. A researcher determines the rate of synthesis of several organic compounds found in the spirogyra before and after several weeks in the water solution. Which of the following graphs best illustrates a likely result of the experiment? Okay, I'm going to let you look at this for just a minute. I'm going to turn my timer on. I haven't done that yet. Okay, 10 more seconds to think about it. Okay, all right, so let's look at this. What did I say happened? We are lacking what? Nitrogen. So what element? Where is nitrogen? We're going to find it in proteins. We're going to find it in nucleic acids. So if we're not able to use nitrogen anymore, then the amount of proteins in nucleic acids are going to decline. So is it going to affect the carbohydrates and lipids? No, because they're carbon, hydrogen, oxygen, and then lipids have the phosphate for phospholipids. So let's look for one where everything's fine other than proteins and nucleic acids are declining. All right, so no decline here, and then we have a decline in protein, decline in nucleic acids. Everything else is holding steady. Okay, are we declining? No, we actually went up. Impossible. And then we went up and we went down. So our answer here that is B. Okay, and then one more question from that same time. All right, so we're looking at two boxes. Here's segment 1, and we have, this is what I see. We have a strand of DNA, and in segment 2 we have a strand of DNA. They're normal, 5' to 3', and they're anti-parallel, and they're complementary. complementary nucleotides, all that looks good. And then I see the sequence in segment 1 and the sequence in segment 2. Now the question, the sequences for the two short fragments of DNA are shown above. Which of the following is one way in which these two segments would differ? A, segment 1 would not code for messenger RNA because both strands have T, a base not found in RNA. B, segment 1 would be more soluble in water than segment 2 because it had more phosphate groups. C, segment 1 would become denatured at a lower temperature than with segment 2 because A to T base pairs have 2 hydrogen bonds, whereas G to C base pairs have 3. And D, segment 1 must be from a prokaryote because it has predominantly A to T base pairs. So take about 10 seconds, 15 seconds to answer that question. Okay, so let's look at those. Segment 1 would not code for messenger RNA. Both of them are DNA, so T is supposed to be there. Okay, segment 1 would be more soluble in water than segment 2 because it has more phosphate groups. Now each nucleotide has the same amount of phosphate groups. Segment 1 must be from a prokaryote because it has predominantly A to T. No, so segment 1 would become denatured at a lower temperature than with segment 2 because A to T base pairs have 2 hydrogen bonds, whereas G to C base pairs have 3. I hope that you got that answer, and C is right. Guided practice, we're going to walk through an FRQ and 2 little parts, well maybe 4. It is an A, B, C, and D to this, and this is a cool question. In the box in the table, you see that there's a treatment 1, treatment 2, treatment 3, and treatment 4, and the medium that are growing these strains of yeast. Okay, you can see that from looking at the top line of the reading. And we're going to have all amino acids present in treatment 1, no amino acids present in treatment 2, all amino acids present except methionine in treatment 3, and all amino acids present except leucine in treatment 4. So let's look at our wild type, which is our normal strain, which does not have the mutations that they just put in this experiment. So the wild type can grow with all, it can reproduce with all amino acids present. It can reproduce with no amino acids present because it makes all of them then. Okay, and then all amino acids present except methionine, yes, because it makes its own methionine, and all amino acids present except leucine, it still can make its own leucine. Now, mutant 1, let's take a look at that. Mutant 1, can it grow in all amino acids? Yes. Okay, can it grow with no amino acids? No. That means that, and we'll read about it in a moment, that there's a mutation that it took out the ability to not produce something. Okay, so it can't grow without it. And then in treatment 3, it can't grow all amino acids present except methionine. If it can't grow in this treatment, then it must not be able to produce methionine itself, right? Okay, in treatment 4, it can grow in all amino acids present except leucine. So right here. And so if we can grow without the leucine, that means mutant 1 is making the leucine. Okay, all right, mutant 2, we have all amino acids present. Yes, it can grow. No amino acids present. Nope, it can't grow because there's something it can't produce. And then it can grow in everything present except methionine. Yes, it makes its own methionine. And then everything present except leucine. No. So I hope you see something happening right here. Okay, now I'll read the text table. This table 1, the data shows that growth of a haploid yeast strains on media at different amino acid content. A plus sign indicates that the yeast strains grow, and a negative sign indicates that the strains do not grow. Amino acid synthesis in yeast cells occurs through metabolic pathways, and enzymes in the synthesis pathways are encoded by different genes. The synthesis of a particular amino acid can be prevented by a mutation of a gene encoding an enzyme in the required pathway. A researcher conducted an experiment to determine the ability of yeast to grow on media that differ in amino acid content. Yeast can grow as both haploid and diploid cells. The researcher tested 2 different haploid yeast strains, mutant 1 and mutant 2, each of which has a single recessive mutation and a haploid wild strain. The resulting data are shown in table 1. So you have 2 things to do here. A, you're going to identify the role of treatment 1 in the experiment, okay? And then B, you're going to provide reasoning to explain how in mutant 1, okay, so mutant 1 can grow on treatment 1 but not grow on treatment 3. I kind of gave you that answer a minute ago. All right, so I'm going to give you maybe 45 seconds to jot this down real quickly. Okay, I got another 5 seconds. Okay, I'm not going to show you the answer yet because we have C and D. All right, well, let's see. All right, so let's see. What did they do? They added a column to this table. Diploid cells produced by mating mutant 1 and mutant 2. So in C, yeast mate by fusing 2 haploid cells to make a diploid cell. In a second experiment, the researcher made the 2 mutant haploid strains to produce diploid cells. Using the table provided, predict whether the diploid cells will grow on each of the 4 media. Use a plus sign to indicate growth and a minus sign to indicate non-growth. So just write yourself a 1, 2, 3, 4, and then a plus or minus next to each of those. So you can do that quickly. I'll give you a second. Okay, ready? All right, so A. So the response identifies the role of treatment 1 as one of the following, and it says, the first one, positive control for yeast growth. And the next one, to test the viability of all yeast strains. And then the third one, treatment 1 allows the researcher to be confident that changes in experimental outcome are due to differences. I've been thinking about this today. I've been thinking about this all day. I want you to say all 3 of these things when you're writing about control. I want you to say it's a control. I want you to say that it's making sure everything else will live. And then I want you to also, no matter what the experiment is, to say to see if the results were due to the treatment. Okay, you need to say all 3 of those things to just make sure that you have the right answer that we look for when that question comes up. All right, and then B, the response explains that mutant 1 can use methionine when it's present in the medium, but mutant 1 cannot synthesize methionine. Okay, and then C. All right, I hope that you have a plus in every column because if all amino acids present, they definitely could grow, and no amino acids present, they can grow because why? Because one of them is producing leucine and one of them is producing methionine, so that together as a diploid, they're able to produce everything in every situation. All right, debrief and summary. Our takeaways for today: the elements that make up each macromolecule. Okay, you need to know those. The properties of the molecule determine its relationship with other molecules. The structure of the molecule is related to its function. You should also be able to describe the properties of the monomers and the types of bonds that connect the monomers in biological macromolecules. Explain how a change in the subunits of a polymer may lead to changes in structure and function of macromolecules. Describe the structural similarities and differences between DNA and RNA, and predict the causes or effects of a change in or disruption to one or more components in a biological system based on a visual representation of a biological concept, process, or model. So I want you to describe it, explain it, and predict what you do without it. Okay, in almost everything that you do to prepare for this exam. All right, so common errors and misunderstandings. This isn't really a common error and misunderstanding, but it's a problem. And so I'll just read this to you. And I'm talking to my kids as well as you. This unit is the fundamental foundation for units to follow. Remembering the basic knowledge of the details in this unit will allow you to build your analytical skills as we move forward and enable you to perform the skills needed to do well on the exam day. You will not have time to look up these facts and general knowledge about molecules. I recommend you develop online quizzes with your friends for the basic knowledge covered in Unit 1. You're not going to have time to go look this kind of stuff up even if you can have your notes when you have 45 minutes to do everything that you need to do. All right, and then our homework for tonight, which is due tomorrow on Ms. Katay is going to talk about organelles and cell size. We're going to start Unit 2 in the next lesson. All right, so here's your homework. The stems and fruits of pineapple plants contain a group of protein-digesting enzymes collectively called bromelain, and often used as an anti-browning agent for fruits and vegetables. Fruits and vegetables brown when they are bruised during transport or sliced and exposed to air. This browning is controlled by enzymatic pathways that produce brown pigment. The browning of fruits and vegetables reduces the nutritional value of the food, so anti-browning agents such as bromelain are used. All right, A, identify the type of monomer in which this enzyme is composed. That should be easy breezy. B, bromelain works by breaking the enzymes that cause browning into smaller molecules. Explain how the reaction that breaks up the enzymes occurs. That's like the second thing we talked about. A is the first thing we talked about. B is the second. And the C, we haven't talked about it, but I think that you're going to get this anyway. The pH of a solution determines the charge of certain R groups. The pH of pineapple fruit ranges from 3.5 to 5.2. Predict the effect on the activity of the bromelain if it is used in a product with a pH of 11. Okay, and then provide reasoning to justify your prediction. Okay, all right. So let's see. That's your homework. Device and internet access to this by the 24th. We know that not all students have access to the internet or a device. We're working on student solutions to help students get what they need to show their best work. If you need mobile tools or connectivity or know someone who does, you can reach us directly. Let us know. tv.org/tech. Please see that done by April 24th. And thanks for watching. I hope you'll join us again for the next lesson for the beginning of Unit 2. Be sure that you do your homework question, and Ms. Katay will start that off tomorrow. Bye. 1.
Hi, welcome to today's AP Biology lesson. Today our lesson will review material in Unit 2. On Friday, April 3rd, an announcement was made providing details about the 2020 AP Biology exam. The 2020 exam will be administered on Monday, May 18th at 2 p.m. Eastern Standard Time. All exams will be taken by students at home. The exam will consist of 2 free response questions. The first is a long FRQ and will have a 25-minute time limit. The second question is a short FRQ and will have a 15-minute time limit. All review lessons will include practice with science skills, which are relevant with any content. We will also provide practice with free response questions in each lesson. My name is Allison Kate. I'm affiliated with both UC Berkeley and Redwood High School, and today I'm teaching you from my home in El Cerrito, California. So just a little bit more about the exam format in case you're just joining us for the first time. There will be 2 questions. The first question is a long free response question, and it will have you interpret and evaluate experimental results. You'll be given a 25-minute time limit with an additional 5 minutes to upload your response. This will compose 65% of your exam score. And just a word about the timing. If I were preparing my students for the traditional exams that we've had in the past, I would tell them to plan to spend about 20 to 22 minutes reading the question and preparing a response for a long free response question. So you can see you have a little bit more time than I would normally recommend. In this question, you will be given a scenario that will be accompanied by data in either a table or a graph, and you'll be asked to do 4 different things with this scenario. And this is where the task verbs that we've been talking about in all of our lessons come into play. So in this case, in the first part, you're going to be asked to describe and explain biological concepts, processes, or model. In the second part, you'll be asked to identify experimental design procedures. In the third part, you'll be asked to analyze data. And in the fourth part, you'll be asked to make and justify predictions. And you can see how those points are distributed in the far right column. For the second question, which is conceptual analysis, you'll be given a 15-minute time limit plus an additional 5 minutes to upload your response. And this will make up about 35% of your total exam score. Again, if I were preparing my students, this is a 4-point short FRQ. So I would tell them to plan to spend about 6 to 8 minutes reading the question carefully and then preparing the response. So I think you have lots of time to be able to answer this question thoroughly. Here you'll be given a scenario that will describe a biological phenomenon with a disruption. And you will be asked to do 4 different tasks. Each task is worth 1 point. So in the first part, you'll be asked to describe biological concepts or processes. You'll be asked to explain biological concepts or processes or to describe characteristics that are represented visually. And this is a science skill to look at things that are represented visually, such as models or data tables or graphs or cladograms or other graphic representations of some type of a biological concept or process. In the third part, you'll be asked to predict the causes or effects of a change in a biological system, and then you'll be asked to justify those predictions. Just another reminder that because this year's exams are modified, I want you guys to know that you are still going to be able to earn the full amount of college credit that you would have earned in other years. And this is a statement from the University of California system, and I suspect that most colleges will have their own statements, but I'm just going to read you the part about the UC system recognizing the effort that students have already applied to these in these challenging courses, and will award UC credit for the 2020 AP exams completed with scores of 3, 4, or 5, which is consistent with previous years. So let's go over our warm-up. Our warm-up was given to you as last night's homework, and this was given to you by my colleague, Ms. Pfizer. Let's take a look at the question. It had 4 parts, and you can look at those task verbs. As always, you should have your pencil and paper handy if you would like to follow along with me now, or if you did this homework last night, have it out so you can compare your answer to these scoring guidelines. Okay, so yesterday, Ms. Pfizer went over a lot of information about macromolecules, specifically proteins and nucleic acids, and so this question really is based on the fact that you need to understand that enzymes are proteins. Okay, so the stems and fruits of pineapple plants contain a group of protein-digestive enzymes collectively called bromelain, and often used as an anti-browning agent for fruits and vegetables. Fruits and vegetables brown when they are bruised during transport or sliced and exposed to air. This browning is controlled by enzymatic pathways that produce brown pigments. The browning fruits and vegetables reduce the nutritional value of food, so anti-browning agents such as bromelain are used. In part A, you are asked to identify the type of monomer of which the enzyme is composed. In part B, bromelain works by breaking the enzymes that cause browning into smaller molecules, so explain how the reaction that breaks up the enzyme occurs. Part C, the pH of a solution determines the charge of certain R groups. The pH of pineapple fruit ranges from 3.5 to 5.2. You are asked to predict the effect of the activity of bromelain if it is used in a product with pH 11, and then you're asked to provide reasoning to justify your prediction. So there are 4 separate tasks, and identify and explain, predict, and then justify your prediction. Okay, so if you're just joining us, hopefully you are able to answer these questions as we go. So let's take a look at the scoring guidelines. The first part, remember I told you that you have to know that enzymes are protein. So what make up proteins? What are the monomers of proteins? And of course they are amino acids. So your response needs to indicate that amino acids are the monomers of the enzyme. In part E, you need to be able to understand how that protein is broken down. And 2 days ago, I did a lesson that went over the types of reactions. One of them is hydrolysis, and hydrolysis uses water to break apart the bonds in larger polymers. In this case, we're going to break apart the peptide bonds that hold the amino acids together. You could have also said that this enzyme breaks the bonds between amino acids using water as a reactant. In part C, remember we're taking the enzyme out of its optimal pH. So these enzymes work at a low pH, an acidic pH of pH 3.5 to 5.2. If you take it out of that and try to have this reaction occur at pH 11, the activity will decrease. It's out of its optimal range. And in part D, you need to have the justification for your prediction that the activity of the bromelain enzymes will decrease. And that's because we know that with those amino acids, they have R groups, and those R groups can be affected by pH. It can change the charge of the R groups, which would make the folding of the protein enzyme different. So it's ultimately its shape would be changed, and anything that changes the shape of the enzyme could ultimately affect its function. So there was a lot in this, from the monomer component to the types of reactions that build and break down organic molecules, understanding the optimum range for pH activity in terms of pH, and then how that pH affects the enzyme's activity. So let's move on to today's lesson. Here's our overview. We are reviewing today, and we're going to be reviewing 3 topics in Unit 2. We're going to take a look at cellular organelles, we're going to look at cell structure and function, and we're going to look at cell size. Today we're going to practice with the science skill, making a scientific claim. We're going to do this today by looking at practice for response questions that combine the content of these 3 topics and the scientific, making a scientific claim skill. The learning objectives for today's lesson, these are the LOs from the course exam description. You should be able to describe the structure and or function of cellular components and organelles. You should be able to explain how subcellular components and organelles contribute to the function of the cell. You should be able to describe the structural features of cells that allow organisms to capture, store, and use energy. You should be able to explain the effect of surface area to volume ratios on the exchange of materials between cells and the environment. And you should be able to explain how specialized structures and strategies are used for the efficient exchange of molecules to the with and to the environment. So here's what you need to know. This is the content part of today's lesson. I'm going to start with cellular organelles, but I'm not going to do an exhaustive list of all of the organelles found in cells. I'm going to be talking about the ones that are specifically mentioned in the course and exam description. So these are the ones that we're going to focus on. And the first one I'm going to focus on is the ribosome. And if you look at that third bullet point, the reason that ribosomes are so important is they are found in all living things. Every cell has a ribosome, and this reflects its common ancestry. So if you look at my image on the lower right-hand side, I've got 2 types of cells. I've got eukaryotic cells and prokaryotic cells. Eukaryotic cells have a membrane-bound nucleus, and prokaryotic cells simply have a nucleoid region. But you can see that they both have ribosomes. The other difference between prokaryotic and eukaryotic cells is prokaryotic cells do not have any membrane-bound organelles, and that means is that ribosomes are not considered a membrane-bound organelle. The other things that you should understand about ribosomes is they're composed of ribosomal RNA and protein. You can get really down into the nitty-gritty composition by talking about the types of proteins and the size of the subunits, but for right now, just understand that they are composed of ribosomal RNA and protein, and that ribosomes, their function is to make proteins. Ribosomes synthesize protein according to a messenger RNA sequence that is transcribed from the instructions found in DNA, and we're going to have a whole lesson on that later on. So right now, we just kind of want to know that ribosomes make proteins. They synthesize proteins. The next organelle I want to discuss is the endoplasmic reticulum. It's kind of a mouthful, so we often refer to it as just the ER, and it occurs in two forms, the so-called smooth and rough. The rough ER is associated with membrane-bound ribosomes, and so if you look at the image, you can see all of these smaller ribosomes are attached to this surface of the endoplasmic reticulum. One of the functions of the rough ER is it creates compartments within the cell, and if you take a look at this, there are spaces. They're called cisternae, and there's so what's going on in this space could be a completely different type of chemical reaction than what's going on in this space. So there are multiple ways to divide the cell, which is very small, and you can increase the surface area of that cell by dividing it or compartmentalizing it. When I talk about this with my students in my classroom, I ask them to envision an office with a number of cubicles, and maybe the worker in cubicle 1 is on the phone and talking to somebody on the phone. Maybe the worker in cubicle 2 is working on the computer, and the worker in cubicle 3 maybe just writing something with pen and paper. They're all doing different tasks, but they are separated by those physical barriers, and that's exactly what the endoplasmic reticulum does. It also helps in transporting the products of the ribosomes protein production, so it will synthesize proteins, and then those proteins can actually be transported through the ER to other places in the cell. A note about the smooth ER, you can see a little bit, I'll show you another image later on, but you can see a piece of it over here, and the smooth ER lacks ribosomes, so it's not involved in protein synthesis, but rather it's involved in lipid synthesis, and it also plays a role in detoxification of certain molecules. The next organelle that I want to talk about is the Golgi complex. It goes by various names, the Golgi apparatus, the Golgi complex, Golgi bodies. We're going to refer to it as the Golgi complex, although some of my images do still call it the Golgi apparatus. It was named, here's a little fun fact for you, it was named after an Italian scientist named Camillo Golgi, and students often ask me that because it is such an odd name. So the Golgi complex is made of membranes. It's flattened stacks of membranes, and it also has some vesicles. And one thing I want to point out to you is that it has two sides, the so-called cis phase, and this is where all the incoming proteins that the Golgi apparatus may be transporting and modifying, and then it has the trans phase, which is the shipping phase. So once the materials, the proteins move through this system, they get packaged, they may get refolded, they get modified in some ways, then they are going to be shipped out in a vesicle format. And so oftentimes when we talk about the Golgi, we refer to it as kind of the packaging plant of the cell, the shipping, receiving and shipping part of the cell. We'll talk more about the functions of all of these in just a minute. The next one is the mitochondria, and everybody knows it affectionately as the powerhouse of the cell, and that's because the mitochondria is responsible for producing ATP, which is the main energy form that cells use. Mitochondria have a double membrane, and in this image, it's really nice because it color codes them. So the outer membrane is shown here in red, the outside is this tan color. This is the outer membrane, and then there is another membrane, it's an inner membrane, and it's highly folded, it's convoluted. And again, you might be seeing variations on the theme. When we fold up this inner membrane, it allows us to increase our surface area and compartmentalize what's going on inside of this organelle. So it's got an outer membrane and an inner membrane, and the space between them has a fancy scientific name. It's called the intermembrane space. And then this blue portion that you see here has another name. It is called the matrix. And in a upcoming slide, I'm going to talk about the chemical reactions that are involved in producing ATP that will occur both in the confines of the matrix, and then also some that will occur in the actual cristae, the inner folded membrane of the mitochondria. The next one is the lysosomes. And lysosomes have the name of a suicide sac, and that's because they are responsible for getting rid of any things that have made their way into the cell that need to get broken down. Lysosomes are membrane-enclosed sacs that contain hydrolytic enzymes. We talked about these. Hydro means water, lytic means to bust apart. So hydrolytic enzymes are going to break down any foreign particles or worn out organelles or anything that shouldn't belong in the cell. Another thing that lysosomes do is perhaps the cell has engulfed some food and has it enclosed in a vacuole. The lysosome will fuse with the vacuole, unleash its hydrolytic enzymes, and digest that food vacuole inside the cell. And then the last one I'm going to talk about, not the last one, there's one more after this, is the vacuole. And vacuoles are found in both plant and animal cells, and they have a variety of functions. Let's first look at the plant cell because they play a really important role in plant cells in maintaining the turgor pressure of that cell. You might remember that plants have a cell wall that surrounds them, and so this is an opportunity for the cell to store some water and maybe some solutes to increase the turgor pressure of that cell. The membrane that surrounds the large central vacuole in a plant cell is called a tonoplast. And I have one example of a vacuole in an animal cell, and I'm going to talk much more about this in later lectures when we talk about how organisms maintain their osmotic pressure and potential. But this is an image of an animal cell. This happens to be a paramecium, and parameciums are filled with macromolecules and solutes, so they're hypertonic to the freshwater environment they live in, and that means that water is going to move into their cell bodies. These contractile vacuoles act as pumps, and they pump out the water to maintain that water balance. And if they weren't there, this organism would drown because the water would be flowing in at a rapid rate. Other things that vacuoles do in animal cells is they store food and waste products until they can be either digested or removed from the cell. And chloroplasts. I believe this is our last organelle today. Chloroplasts are specialized organelles that are found in photosynthetic algae and plants. They are the sites of photosynthesis. Chloroplasts have a double outer membrane. I'm going to go into more detail when I start talking about the function, and then of course we have a whole lesson on photosynthesis. So you'll have a lot of practice with this structure. But for right now, I want you to see a similarity with the mitochondria, which is the organelle that I already discussed. And the chloroplast also has a smooth outer membrane. Further, it has a smooth intermembrane. It's got an intermembrane space. It's not labeled. Then it's got another compartment, and this compartment is the stroma, and it's a dense fluid-filled compartment where parts of the photosynthetic reactions occur. And then the chloroplast has a third membrane system. And the third membrane system is called the thylakoid membrane, and inside the thylakoid is yet another space, and it is called the thylakoid space. And that is where the light reactions, the light absorbing reactions of photosynthesis will take place. And over here, you can see chloroplasts are tiny. So we're looking at an actual slide of some plant cells. I believe this is from a water plant, and you can see all of those teeny tiny chloroplasts that are inside that plant cell. So I'm going to go into detail about the functions of each of these organelles, but these are kind of the statements that come from the CED, the course and exam description, about each one. So let's read them together, and then I'll go through them using images to support this content. So the endoplasmic reticulum provides with mechanical support. It carries out protein synthesis on ribosomes that are attached to its membrane, and it plays a role in intracellular transport. The mitochondrial double membrane provides compartments for different metabolic reactions. Lysosomes contain hydrolytic enzymes, which are important in intracellular digestion and the recycling of cells' organic materials, and program cell death, which is called apoptosis. And vacuoles have many roles, including the storage and release of macromolecules and cellular waste products. In plants, it aids in the retention of water to maintain turgor pressure. So now we're going to look at each one of these organelles and function. So I've got an image of the endoplasmic reticulum. On the left side, I've got the rough ER, and on the right side, I've got the smooth ER. And I'm going to focus on the rough ER. The rough ER, as you can see, has all of these ribosomes that are attached to it. So ribosomes synthesize proteins. So the ribosomes that are attached to the ER make proteins that are going to be exported from the cell. They will work together with the Golgi to actually modify, package, and transport these proteins out of the cell. Okay, so it does provide that transport function. It provides the work surface for the ribosomes, and it is connected to the Golgi, which will modify those proteins for export. Just to contrast that, can you see in the cell there's also some proteins that are free? They're not bound to the ER. Those ribosomes. These are ribosomes not bound to the ER. These free ribosomes are actually going to be making proteins that are for use in the cell itself. So there are two proteins that are, there are two ribosomes that are free, and they make proteins that are going to be used within the cell. And then, of course, the ribosomes that are attached to the rough ER are going to make those proteins that are going to be exported from the cell. And then you can see the smooth ER, it lacks those ribosomes, and it is not involved in protein synthesis, but rather in lipid synthesis. So it can make fatty acids, it can make phospholipids, which are used in cell membrane construction, and also steroids. And its detoxification function, it will detoxify molecules that are found in alcohol, drugs, and metabolic waste products. This is the mitochondrion, and I promised you that I would show you the two different places where the reactions that produce ATP are located within the mitochondrion. And again, you're going to have a whole lesson on cellular respiration in a later review session. So this is just a pinpoint where in the mitochondrion these reactions occur. So the Krebs cycle occurs within the matrix of the mitochondrion, and the electron transport chain, which is what really makes the lion's share of the ATP, is a series of proteins that are actually embedded in that inner membrane, in that highly folded membrane that we call the cristae. So again, we see that part of the production of ATP occurs in the Krebs cycle, or the citric acid cycle, that's in the matrix, and the rest of it, ATP production occurs along the inner mitochondrial membrane. And then we can follow up with the chloroplast because it has a very similar structure. And I know that Ms. Pfizer talked to you about the endosymbiotic theory, and now you can really get a look at why chloroplasts and mitochondria are once believed to have been their own free-living prokaryotic organisms that were built by a cell. So these guys, chloroplasts with their membrane system, and if you look at this image, they have the outer membrane, that inner membrane, and a third membrane system called the thylakoid membrane. Thylakoid literally means stack of coins from the Greek, and so when they're stacked up, we call that stack a granum, and you can see within that chloroplast there are multiple grana. Surrounding the thylakoids is the stroma. Now, the light reactions, or where the pigment chlorophyll resides, is in the thylakoid membrane. So the first part of photosynthesis, or the so-called light-dependent reactions, occur within the thylakoid membranes, and the Calvin cycle, or Calvin-Benson cycle, which is where we actually fix our carbon dioxide into our organic carbohydrate molecules, our sugars, occurs in the stroma. So those two reactions take place in different parts of the chloroplast, just like we saw the different parts of cellular respiration reactions that make ATP occurring in different parts of the mitochondria. So I hope you're seeing the similarities between these two organelles. The last topic today is about cell size, and if you've ever wondered why cells are small, this is the lesson for you. So it has to do with the cell surface to volume ratios, and it turns out that the larger the surface to volume ratio is, the more efficient the cell is going to be, and the cell needs to interact with its environment to obtain nutrients, to eliminate waste, to acquire or dissipate thermal energy, or otherwise exchange chemicals and energy with the environment. So this is a lab activity that you may have done while you were in biology class. I know that Ms. Pfizer is going to go over this with you tomorrow in your review lesson, but this is just an image right now to show you that the extent of the diffusion of that white part of this cube is the greatest in the smallest cell, and that's because it has the largest surface area to volume ratio. Oh my gosh, so here's a bunch of mathematical equations and formulas, because when we look at surface area and volume, there is some math involved, and so these equations are lifted straight from your biology equation and formula sheet. And I want you to be familiar with these so that you can see how they're used in the AP biology curriculum. So we're going to focus on the surface area of a sphere and the surface area of a volume, which are the first two equations. Let me again point out the key points, take-home points from the course and exam description. The surface area of the plasma membrane must be large enough to adequately exchange materials, and we're going to see in some later slides some ingenious ways that cells increase their surface area. These limitations can restrict cell size and shape. Smaller cells typically have a higher surface to area volume ratio, so they have a more efficient exchange of materials with the environment. As cells increase in volume, the relative surface area decreases and the demand for internal resources increases. So what you tend to see instead of cells getting really, really big is that we end up making more cells, and so we have multicellularity rather than one great big cell or one great big organism. More complex cellular structures like membrane folds are necessary to adequately exchange materials with the environment. I have several examples coming up. And as organisms increase in size, their surface area to volume ratio decreases, which affects properties like the rate of heat exchange with the environment. So you also see some organisms have specialized structures to dissipate that heat, or in some cases they have behavioral mechanisms to do thermoregulation. So here's some examples of some different strategies to obtain nutrients and eliminate waste that organisms have evolved. If you look over here, this is what your intestinal cells look like. So you know that you absorb all the nutrients from the food that you eat through your small intestine, and your intestine is lined with these projections, and these projections are called villi. And again, those folds increase the surface area so that you can absorb as many nutrients as possible. On top of villi, you have microvilli, which are even more extensions to further increase the surface area for nutrient absorption. If we look at gas exchange organs, we can see that there's external gills and we have lungs. And so I'm going to focus in on the mammalian lungs because this is what your lungs look like. You can come down here and get an enlarged view of an air sac or an alveolus. Your lung is made up of millions of these small air sacs. And what happens when you get down to that level, we increase the surface area for diffusion of oxygen into your lungs and carbon dioxide out. So it greatly increases your surface area. And another fun fact for you, the surface area of your lungs for gas exchange of oxygen and carbon dioxide is roughly equal to the surface area of a tennis court. So that's kind of an amazing fun fact. And then one final thing about fish, and we'll talk about both freshwater fish and saltwater fish in a later lesson, but they kind of go to the extreme. So what happens is they have gills and they swim. This is a freshwater fish, I believe it's a trout, and it's swimming through the water, and its gills have set up something called a countercurrent flow, where the oxygen-rich water will flow into the oxygen-poor blood, whereas the carbon dioxide from the blood flows back out into the water. So they have really super efficient exchange through gills. So there's lots of strategies that different cells and different organisms have adapted to increase the efficiency of the exchange of nutrients and waste. So let's get some practice with these ideas. These are all going to be some former free response questions. This first one is from an exam that was given before 2012, and so it's an older question, so the scoring guidelines are a little bit different than the types of exam questions you'll see on your exam. But this question reads, during an investigation of a freshwater lake, an AP biology student discovers a previously unknown microscopic organism. Further study shows that the unicellular organism is eukaryotic. You are asked, your task verb is to identify 4 organelles that should be present in the eukaryotic organism, and then to describe the function of each organelle. Now I just want to also point out a test tip to you. When you are asked to identify 4, just identify 4. We will only score the first 4 that you identify, so you are wasting time by listing more than those 4. So just give us your 4 best ones. And then you're asked to describe the function of each organelle. So remember that this is eukaryotic, so think about the 4 that you want to talk about. I've given you more than 4 in this lesson. Grab that pen and paper and write them down, and then let's see if we can match the function to each one. Okay? So I know this chart is a little light. And let me tell you how this was scored. So you got 1 point just for identifying and listing your 4 organelles. Okay? And then for each of those organelles you listed, you would earn an additional point if you had had the function correct. And the reason that these organelles are listed in the order they're listed is that's how often they appeared in the responses by students the year that this question was given. So because it was eukaryotic, a lot of students said nucleus, which I did not specifically talk about in this review, but the nucleus is found in the eukaryotic organisms, eukaryotic cells surrounded by a nuclear envelope, and it contains the hereditary information, the DNA, the chromosomes, or the genes. The next one listed is ribosomes. Ribosomes synthesize proteins. And then we have the ER, and you could score a point for just talking about generalized endoplasmic reticulum, or another point for talking about rough and or smooth ER. So we know that the endoplasmic reticulum is an internal transport organelle, and it compartmentalizes the cells. That when we specifically refer to the rough ER, it is important in protein synthesis, and the smooth ER is particularly important in lipid synthesis and also detoxification of certain substances. Mitochondria is responsible for producing ATP. Chloroplasts are responsible for photosynthesis, which produces carbohydrates using sun's energy, solar energy. Then we have vacuoles, vesicles. They are different, but the distinction is kind of a fine nuance, so they could be lumped together here. And they can be for storage or transport. We did not discuss cilia and flagella, which are used in cell motility. We did not discuss basal bodies, which are a support for cilia and flagella. Golgi bodies, which again are the packaging plant. They modify proteins, package and transport these modified proteins. Lysosomes we mentioned to contain hydrolytic enzymes and break down any waste products, metabolites, or pathogens. And then an organelle we did not mention is a peroxisome, which is involved in detoxification of specific reactions that produce hydrogen peroxide, which is toxic to cells. So hopefully you were able to list at least 4 organelles and describe the function of each of those. The next one is going to ask you about the concept of protein production, modification, and export. So this question reads: a molecule of messenger RNA, which is abbreviated mRNA, has just been synthesized in the nucleus of a human cell. So that should give you a hint about what kind of a cell we're dealing with, i.e. eukaryotic. If the cell is a secretory cell, explain how the protein is eventually targeted, packaged, and secreted to the exterior of the cell. So since it's going to be secreted, we're going to talk about that the ribosome is likely attached to the endoplasmic reticulum, and we're going to explain how it gets targeted, packaged, and sent to out of the cell. So this was worth 3 points, 3-point question. So any one of those bullet points would have earned you a point. First of all, there are special proteins called chaperone proteins that help hold that newly synthesized polypeptide into the protein, that it could be modified in the Golgi by the addition of sugars or phosphate groups to somehow get in the response that there is an endo or internal membrane system. In other words, that protein is moving from the ER to the Golgi, where it is packaged up into vesicles, and that those vesicles are moved to the surface, and then they fuse with the cell membrane, and that protein would be released to the outside through the process of exocytosis. One more practice. This is a question about cell size. You are given some data, which you're provided with a radius of 2 spherical structures that are surrounded by a membrane, and you are asked to calculate the volume and surface area of each of these. And again, the equations are given to you, and you're given the radius. So all you have to do is plug that in. So let's take a look. So to calculate the volume, you use the volume of a sphere formula, which is the volume is equal to 4/3 times pi times the radius cubed. And the radius in the first cell is 1. That makes that math super easy. And the radius in the second case is 3. And your units are going to be millimeters cubed. And then to calculate the surface area, it's 4 times pi times r squared. And again, the first one is easy because it's 1. The second one is a little bit more challenging, but then you would get your answer. So in the first case, you have a volume of 1 cubic millimeter and 1 square millimeter for the surface area, which is a 1-to-1 surface area to volume ratio. In the next case, you're going to calculate the volume to be 23 millimeters cubed, and the surface area is 9 millimeters squared. So the surface area to volume ratio is 3-to-1. Remember, the higher the surface area to volume ratio, the more efficient the cell will be. Okay, so let's do a check for understanding by practicing with our skill using the content about cell size and the skill of making a claim. So I'm giving you a data table, one like you just saw, and it's got 3 cubes, a 2-centimeter side with a surface area of 24 square centimeters and a volume of 8 cubic centimeters. We've got an intermediate one that is 1 centimeter with a surface area of 6 and a volume of 1 with a 6-to-1 surface area to volume ratio. And finally, we have a small one with a size of 0.5 centimeters, giving us 1.5 centimeters squared surface area and 0.123 cubic centimeters volume, which works out to be a 12-to-1 surface area to volume ratio. You are asked, using this data and the image below, to make a claim about cell size that could be supported using these observations and calculations. All right, got that paper? Let's make a claim. What is your claim about cell size that you could use these data to support? So did you say smaller cells are more efficient at moving materials in and out of cells than larger cells? So how would you support that claim? So here's your evidence. The smallest cube had the least amount of that red pigment remaining. The largest cube had the most amount of red pigment remaining, and the medium cube had a medium amount in between the other cubes. The reasoning here is that it's clear when you observe these 3 cubes that the smallest cube would be the most efficient at moving materials in and out. The smallest cube had the largest surface area to volume ratio, 12-to-1, while the largest cube had the smallest, 3-to-1, and so it would be the least efficient. Okay, so that's a little practice with making a claim, which is to state, assert, or maintain that something is true. Keeping that claim in mind, you need to also think about the evidence you have to support the claim and the reasoning you have to connect that evidence to that claim. All right, so your learning objectives today. You should be able to describe the structure and function of cellular components and organelles. You should be able to explain how subcellular components and organelles contribute to the function of the cell. You should be able to describe the structural features of cells that allow organisms to capture, store, and use energy. You should be able to explain the effect of surface area to volume ratios on the exchange of materials between cells and the environment. You should be able to explain how specialized structures and strategies are used for the efficient exchange of molecules to the environment. Your key takeaways, these are your enduring understandings, are that living systems are organized in a hierarchy of structural levels that interact, and that highly complex organization of living systems require constant input of energy and the exchange of macromolecules. All right, before I give you your homework, let's take a look at some common errors and misunderstandings. This was an AP exam question about cell structures, and then I want to share with you some global things that students did when they responded to this question that readers noticed when scoring it. So you're given a data table, and the data table has 3 types of cells: cell type X, cell type Y, cell type Z. And then you've got the relative amounts of organelles in the 3 cell types. So let's look at cell type X. It's got a small amount of ER, both rough and smooth. It's got a large amount of mitochondria, and it has cilia present, and a small amount of Golgi bodies. Cell Y has a large amount of both types of ER, smooth and rough. It has a moderate number of mitochondria, no cilia, and a large amount of Golgi. And finally, cell Z doesn't have any of those things. None of those organelles are present in cell Z. Your question is, based on the analysis of this data, identify a likely primary function of each cell type, and then explain how the data support the identification. So you're making a claim. You're making a claim that the function of cell type X is blank, and then you're using the data to support that claim. Okay, so let's take a look at how students answered this. So cell type X, because it had cilia and a large amount of mitochondria, the mitochondria produces the energy, and the cilia move. They move things along surfaces, or they move an organism. So the function of this cell type is likely some type of movement. Okay, don't name the cell. You don't need to name a muscle cell or any specific cell. You are identifying the function. Okay, cell Y had a lot of ER and Golgi, and so it's going to be involved in some type of protein synthesis and then secretion of that protein for export. If you focused in on the smooth ER, then you're going to talk about lipid synthesis. So either one of those, that you're not going to get points for both of those, but either one. And again, notice that we're not naming a specific cell type. We're identifying a function, and we're supporting it based on what we saw in the data table. And cell Z. Cell Z could have had a number of functions. It could have been for transport. transport. It could have been for, let me give you an example because that one is kind of tough. When it says oxygen transport, so we know that red blood cells transport oxygen. So red blood cells don't have any other organelles other than that hemoglobin protein, which actually transports the oxygen. It can transport water in plants, and we'll see in a minute that the cells that do that are xylem cells, and they're dead. They have no living tissue, so they have no organelles present. They could be protection. Your outer epidermal cells are dead. They don't have any organelles. Support like that xylem. Storage like the adipose tissue, which has no organelles, stores fat. And then no function at all. The cell could be dead or is undergoing cell death, which is called apoptosis. So none of those require organelles. So let's see what the reader saw as common errors and misunderstandings. The first thing is that students did not make the connection between the function of the cell and the organelles present. Rather, they identified the special type of cell. And I tried to point that out to you when we talked about cell X. A lot of students just said it's a muscle cell because it needs lots of energy and it has mitochondria, instead of identifying the function of movement. Students often identified cell X as cellular respiration and didn't link the ATP production by the mitochondria, which is providing energy to the cilia for movement. Students were often confused about the function of organelles. They interchanged the functions of cilia with villi, and hopefully now you can see the difference. Cilia is involved in movement or transport along the cell surface, whereas villi increase the surface area of a cell. They often stated that the endoplasmic reticulum makes ribosomes and that the ribosomes get exported from the cell. So clearly the endoplasmic reticulum supports the ribosomes and they make proteins that get exported from the cell. And finally, students had a lot of difficulty when presented with cells Z that lack organelles. And I know when I gave this to my students that they were challenged by that as well. So you just have to remember that there are some cells that do not possess all of those organelles. Think red blood cells, think xylem, think adipose. And then often students name cell types like prokaryotes or stem cells or neurons rather than identifying the function. And so it's really important to read the question carefully. All right, so let's take a look at your homework tonight. So your homework tonight is 2 organelles that we talked about today, mitochondria and chloroplast. And there's a long introduction, but what you're asked to do with this question is to describe the importance of intermembrane separating the different regions of both the mitochondria and the chloroplast. Okay, so I want you to read that question carefully. I want you to look at the diagrams that are presented visually for you, and then I want you to describe the importance of those intermembranes. Tomorrow, Ms. Pfizer will be with you, so she will debrief this homework. I'd like to thank you for watching and hope that you'll join us for all of our lessons. Be sure to complete the practice question because it'll get reviewed tomorrow. I will see you next week, so have a great lesson tomorrow and a great weekend. Thanks for watching.
Hi, welcome to AP Biology. I'm Gail Pfizer from Darnell Cookman Middle High School and Florida State College in Jacksonville, Florida. I made this to be a no-judge room today. I'm going to try out a new platform. I never use PowerPoint in my classroom, maybe one day out of 180, so I've been kind of uncomfortable with it. And this is, so what I'm using is what I use with my interactive board with my kids in my own room. And I'm going to, from this point on, as long as this works, unless something goes terribly wrong today, this is what we're going to do. It allows me to move things around, write on it, things like that, and hopefully engage you and keep you with me the whole time. Speaking of which, I'm going to tell you, I think I pretty much have a routine now. We're going to have a warm-up, then we're going to talk about content. We'll do a short FRQ, very short together, and then we'll do some multiple choice to make sure that you know the content, and then we'll write a moderate to medium to a long FRQ, and then we'll have a homework assignment. And I think I've done it enough now that I think that is probably the standard that we'll go through the rest of the time. I use these in my classroom. I don't, we watch them online together, and then I have them type their answers into a form so that they can get some typing practice while they're at it. So that's how I'm using this in my own classroom. All right, so on May 18th, 2020, you're going to have an AP Biology exam at 2:00 p.m. Eastern Standard Time. This is the last day that I'm going to go over this with you because I've been doing it for a week now, and I think everybody knows this now. Anyway, so question 1 will be interpreting and evaluating experimental results. You'll have 25 minutes to read and respond, 5 minutes to upload, and this will be 65% of your exam score. It'll be based on an authentic scenario accompanied by data in a table and a graph. There's going to be 4 parts, and those parts may have a task verb or 2 in each. So A, describe and explain biological concepts, processes, models, and applied context. And then B, identify and justify experimental design procedures. C, analyze data. And D, make and justify prediction. Remember, in describe, you're just going to list the characteristics of whatever it is they're asking you for. But when it's explain, it's going to be the how or why. Identifying, keep it short, just say whatever it is they're asking you. Now, when it comes to justifying, you may be justifying a claim you're making, or you could be justifying a claim that a researcher is making. And then analyzing data, they're going to give you a table or a graph to fit in there. And then the D, make and justify, we just talked about. All right, question 2, conceptual analysis. That will be 15 minutes for your response, 5 minutes to upload. I've been having my students do a lot of uploading documents to send me to my email and send me on my website and things like that. So just the routine is getting established. All right, so this is question 2 is going to be an authentic scenario describing a biological phenomenon with a disruption. There's 4 parts, but there's only 1 task verb in each. Describe biological concepts or processes in applied context. Explain biological concepts or processes in applied context. C, predict the causes or effects of a change in a biological system. This means that something's going to be removed or something is going to be changed, and you have to figure out what's going to happen from there. All right, and then support or justify, again, make that claim, that evidence, and that reasoning. Okay, and the reasoning is something to be strong biology behind what you're saying, and evidence is usually the data. All right, so our lesson overview. Today we're still in unit 2, and we will be through tomorrow also. I'm going to go through, well, not tomorrow, I'm doing Monday, and then this today is doing Tuesday, so 2 more days. And anyway, membrane transport today, and by the end of this lesson, you'll be able to explain these learning objectives. I put everything into one form to kind of shorten this up. All right, the roles of each of the components of the cell membrane, the fluid mosaic model, how the structure of biological membranes influences selective permeability, the role of the cell wall in maintaining cell structure and function, the mechanisms that organisms use to transport large molecules across the plasma membrane, and the processes that allow ions and other molecules to move across the membrane. All right, so first we're going to start with a warm-up, which if you watched the last lesson, it was about organelles and cell size. So this was the homework from that lesson. So it starts with being the homework of this lesson. And if you've been watching me for a while, let me get the right color. Okay. All right, so if you've been watching me for a while, you know that I like to tackle the diagram first, so I can kind of figure out what's going on before I start reading the text, because that's just my learning style. I like visual, and I can kind of figure things out that way. So I can see that I have a mitochondria and I have a chloroplast, and then it's telling me that I'm going to have this gray shaded areas are going to be high in hydrogen ions. You can also use the word protons. Okay, so here I am in the intermembrane space and also in the lumen of the thylakoids. Okay, so now mitochondria and chloroplasts are the energy processing organelles of eukaryotic cells. Both organelles contain outer and inner membrane. The inner membrane of the mitochondrium contains many folds. The chloroplasts have a third membrane forming the thylakoid. Molecules such as enzymes responsible for the synthesis of ATP are embedded in the inner membrane of the mitochondria and the thylakoid membrane of the chloroplast. Okay, your task verb, describe the importance of the inner membrane separating different regions of the mitochondria and chloroplast. So why, not really why, describe the importance. What is it doing, that inner membrane? I'll give you just a moment and I'll give you time to start too. Okay, just jot down a quick answer if you're just joining us for the first time. Okay, and I'll let you check the scoring guidelines. Here we go. All right, see this red dot here. So this page is very busy, but here's the answer. All right, the response indicates that the inner membranes permit different reactions to occur in different regions of the organelles. Okay, so that was the importance of the inner membrane, and it told you that in up here when it was telling you what's going on in that area. Okay, all right, so now we go to content. This is what you need to know. All right, phospholipids have both hydrophilic and hydrophobic regions. The hydrophobic phosphate regions of the phospholipids are oriented toward the aqueous external or internal environments, while the hydrophobic fatty acid regions face each other within the interior of the membrane. So if you look at this whole thing that we're looking at, okay, so we're looking at a phospholipid bilayer. This area in here's our fatty acid chains. Okay, those are fatty acid chains. They're hydrophobic, and then here is our phosphate head of our phospholipid, and those are hydrophilic. So it's hydrophilic on the inside and the outside. Here's the inside, internal and external. And if you'll notice these tiny little molecules, there's our water molecules. So hydrophilic, water loving. The phosphate head is where the water is on the outside of the cell, and also where the water molecules are in the inside of the cell. Okay. All right, embedded proteins can be hydrophilic with charged and polar side groups, or hydrophobic with non-polar side groups. So let's look at so this looks like an alpha helical structure there, right? And so the R groups that are in the amino acids in this area, they would have to be non-polar to be in this area. That's what this is saying. And then the amino acids that are in this area, okay, those are the polar amino acids to be able to be out here in the polar area where there's water. Okay, and same here. All right. The fluid mosaic model of cell membranes consists of a structural framework of phospholipid molecules that are embedded with proteins, steroids such as cholesterol and eukaryotes, glycoproteins, and glycolipids that can flow around the surface of the cell within the membrane. So this is the cell membrane, and let's get our orientation. So here it's made up of here's all our phospholipids. Okay, it also has cholesterol. So these four, do you remember the four fused rings? Those were cholesterol. Those were lipids. Remember? All right. So now let's look at some other proteins within this membrane. So let's start with this one. Okay. So right here we have a protein that is crossing the membrane. It's on this side and it's on this side. That means it is transmembrane. Okay. Another word for this particular one can be used is integral, which means it is built into the membrane. So that's where you'll find it. Also, this is a peripheral, and it can also be called an integral because it is also stuck in the membrane. Okay. So we have a peripheral, but it's an integral, but it's not a transmembrane. Okay. So not all transmembranes are integral, but not all integrals are transmembrane. All right. So glycolipids, here we can get yellow. Here's our carbohydrate for the glyco part of the word. Okay. See this part of the word glyco. All right. So there is our glycolipid, and it's attached. It's tethered to the fatty acids that are inside of the this fatty acid layer, the non-polar layer. And then we have a glycoprotein. Again, here is our carbohydrate chain that is attached to this protein right here. Okay. So that's where the words glycolipid and glycoprotein come from. All right. So I was talking about the fluid mosaic model and where that comes from. So fluid mosaic model, first of all, fluid mosaic model means that it's fluid. It's moving around. It's a dynamic. This whole thing right here is not static. It's moving. Those proteins are moving. And if you were looking at the surface of a cell, then it would look like a mosaic tile model. Okay. So that's where the name comes from. These pictures are from a lab in my room, and it's more about having fun on that day actually than anything else. But it's a bubble lab, and within the inside of these straws, they have a bubble solution, and you can see the molecules moving around in the frame, and it's kind of moving around like a fluid mosaic model because they all have different colors because they're reflecting light of different ways. So that's the bubble lab. If you have not done that in your classroom, you can easily do it at home. All right. So this is about permeability, and the structure of cell membranes result in selective permeability. Cell membranes separate the internal environment of the cell from the external environment. Selective permeability is a direct consequence of membrane structure as described by the fluid mosaic model. So here we have this is outside cell. Okay. So we have some molecules, and they are able to go through. Okay. The phospholipid bilayer, and so that means that the bilayer was permeable to those molecules. But what if it was another molecule? Would this be able to go through there? No. Okay. So it's only selective in what can go through. So that's where the selective permeability will come from. All right. So small nonpolar molecules, including nitrogen gas, oxygen gas, and carbon dioxide, freely pass across the membrane. Hydrophilic substances such as large fluid molecules and ions move across the membrane through embedded channel and transport proteins. Polar uncharged molecules, including H2O, pass through the membranes in small amounts. Okay. So let's look at this. We have we can go ahead and kill two slides here with one stem. Here we have CO2. It's a waste product. Okay. And you see we have a large quantity here, small here, and we're moving from a high to low concentration gradient. Okay. And we're moving through the membrane. And now this, on the other hand, is moving from the outside. We have oxygen gas coming into the cell. Okay. And it's moving from a high to a low concentration gradient. But these large molecules, they cannot get in. Okay. They're too big to come through the membrane. So they must have a protein carrier molecule to assist them to let them flow through. And then they will move through from a high to a low concentration. And same with these larger molecules over here. They're moving from high to low, and they have to have the protein carrier channel because they're big. All right. Oh, I'm going to talk about H2O. I'm going to talk about aquaporins when we talk on the next lesson. All right. So cell walls provide a structural boundary as well as permeability barrier for some substances to the internal environment. Cell walls of plants, prokaryotes, and fungi are composed of complex carbohydrates. So you can see here. Let me get something. All right. So now let's do that. All right. So right here. So these are plant cells. The plant cells all next to each other. And so what you have here, you have a cell membrane. Okay. And then this layer here, this is our cell wall, and you can see that it is made up of one of the things that's there because there's others is a polysaccharide called cellulose, which gives it its structure. Okay. And that's one of the polysaccharides that you noted as a polysaccharide in the biomolecule chain. Now this is down here. We have bacteria. We have gram-positive and gram-negative bacteria. And when you get to Ecology and get your microbiology class, you're going to be doing something called gram stain. Anyway, you're going to learn about these two types of bacteria. Notice and this is peptidoglycan. Okay. And it's really thick right here. But look here. It's just a thin layer here. So when you put this gram stain on, then this will stain differently than the gram-negative, and that's how they can tell the two apart. But the point was that here is the cell membrane, and it is outside of the cell membrane acting as a barrier. Okay. Now on the fungi cell wall, we have here's our cell membrane right here, and then this red, those are pieces. Those are kind of like little curve. Okay. That's the polysaccharide, which is chitin. All right. Passive transport. So passive transport is the net movement of molecules from high concentration to low concentration without direct input of metabolic energy. Passive transport plays a primary role in the import of materials and the export of waste. Okay. We've already talked about that. Here we're going passively. So now we can add another word. So we're moving from a high to low concentration, and this is passive transport, and this does not. Okay. Without energy. This does not require ATP to do this. Same thing here. We're moving from high to low. This is passive transport. Doesn't require energy. Okay. Active transport, on the other hand, is look at this. Here are 3, and here are a lot more than 3. Okay. So we're going to move from here to there. So we're going against the gradient. So active transport requires the direct input of low energy to move molecules from regions of low concentration to regions of high concentration. So if I had to say 3 things about this, I'm saying it's moving high to low against. Okay. So make sure you say that against the gradient. I'm sorry. I'm going to set up. It's low to high against the gradient, and then we're going to call it active. And then it is yes, we need energy. Okay. So when you talk about passive or let's get that over there as evidence. Okay. We're moving from low to high. All right. So when you're talking about either one of these, I want you to say 3 things. I know I always want you to say 3 things about everything, but we need to go from low to high against the gradient. We need to say it's active transport, and we need to say it requires ATP. Okay. All right. So concentration gradients. This wraps up all of it together. The 3 things I want you to say about passive, and then the 3 things about active. So the selective permeability of membrane allows for the formation of concentration gradients of solids across the membrane. So on this side here we have passive transport. We're going from high to low. We're moving from a high to a low concentration gradient. Okay. All right. Active transport. We're going from a low to a high, and we're going against. You have to say that. Okay. We're going against the gradient, and we must use ATP. So 3 things about each will cover you in any situation if you're talking about it. All right. So now let's talk about endocytosis and exocytosis. We'll talk about endo first, and I'm going to scribble this part out right here because that's not where I want you to be focused. The processes of endocytosis and exocytosis require energy to move large molecules into and out of the cells. In endocytosis, the cell takes in macromolecules and particulate matter by forming new vesicles derived from the plasma membrane. All right. So let's look at here's our food for our cell. Okay. I'm gonna go ahead and color it, and I'm gonna make it smaller. Okay. All right. So our plasma membrane here is our plasma membrane. Look what it does. It engulfs. Okay. It engulfs this food particle, and then it's going to form a vesicle around it. It's going to transport it into the cell, and then it's going to fuse with this lysosome. Okay. And then that lysosome is carrying digestive enzymes. It's a digestive enzyme, and now it's breaking down that food into usable parts for your cell. Okay. All right. And that's endocytosis. There's actually 3 types. Well, the one we just talked about was phagocytosis. Penocytosis has to do with it's like the cell's taking a big gulp in order to grab some small molecules that it needs. Okay. Excuse me. And then receptor mediated. That means we need these purple molecules in the cell. But first they have to bind to this receptor that's on the cell membrane, and then a vesicle is pinched off and then travels to its location inside the cell. So that's those 3 types. All right. we're leaving now. And so before we got here, so this green is protein that needs to be produced by the cell, but it's not going to stay in the cell. It's going to go out somewhere. Anyway, so the vesicle is coming from where? So we were produced, this green protein was produced on the in the endoplasmic reticulum, right? And then it pinched off into a vesicle and moved from the endoplasmic reticulum to the Golgi apparatus. And then there's modifications that happened in different layers. And then it was pinched off, and now that's where we are right here in this diagram. So all of a sudden now this vesicle is fusing with this membrane, and then those contents will be released from the cell. All right, so there's two studies. That's content. We're done with content. So we've done warm up and we've done content. So now we're going to do some FRQ practice. They're really short ones for this. I couldn't find anything really long and medium. No, you're disappointed. All right, so science skill practice 6B, support a claim with evidence from biological principles, concepts, processes, and or data. The task words what we're going to use: describe, explain, identify, support, and predict. All right, so this is it. This is what I got. The relationship of structure to function is one of the major things in biology. For the following structure function pairs, describe. Okay, so there's a task word. Describe the structure and then explain how the function is related to the structure. And you have two choices here. We'll call this will be choice A, and this will be choice B. So A, you're going to describe the structure and explain how it's related to its function, or membrane protein structure and active transport. Or you can do a membrane protein structure and facilitated diffusion. And it's just going to be a little bit of difference between the two anyway. I'll give you a minute to think about that, maybe 45 seconds, and then I'll be back. Okay, about 5 more seconds. Okay, let's see what the scoring guidelines say. All right, so these kind of fit. Everything fits both, other than one statement here. Okay, so the description. Did you say there was a phospholipid bilayer? Did you say there was an integral protein in the membrane, and that the protein's 3D shape allowed it to act as a channel, bind solutes, and or bind ATP? So you would need this piece right here if you had chosen to talk about ATP. About active transport. Okay, now the explanation. Some solvents like ions and larger hydrophobic molecules cannot cross phospholipid membranes unassisted. Integral proteins allow such substances to pass. Hydrophilic channel binding of solute leads to shape change in protein. Hydrolysis of ATP. Okay, so we would need this right here. Causes shape change in the protein, leading to shuffle of material from one side of the membrane to the other. So what we're talking about hydrolysis. What did we do when we did hydrolysis? We did what? So if this was ATP, then we're going to put in a molecule of water, right? And it's going to bust it up, and then you have ADP, and that energy from that one phosphate group bond is what you use to fuel this movement from low to high. All right, we're going to check for understanding. These are multiple choice to make sure that you understand the content. All right, so we've seen this diagram before. Okay, right? So you already know. And this is a model of the plasma membrane showing several biological molecules, including a transmembrane protein. All right, and let's see this is the question. Which statement best explains why correct protein folding is critical in the transmembrane protein shown above? And there's 4 choices. A, interactions of the hydrophobic and hydrophilic amino acids help to anchor the protein in the membrane. Okay, maybe. All right, interactions of the peptide bonds of the protein with the membrane will affect the rate at which substances can cross the membrane. Interactions of the peptide bonds of the protein affect the rate. No. All right, interactions of the protein and phosphate and phospholipids increase membrane fluidity. No. And interactions of the quaternary structure of the protein will increase hydrogen bonding in the membrane and make the membrane less fluid. No, that's not what's happening. All right, so our answer is, and this is how I approach. I know there won't be multiple choice questions on your exam, but this is what I teach my students to do. Find what's wrong in all the other choices, okay, if you need to. All right, next question. Plant cell walls are composed of cellulose, while fungal cell walls are composed of chitin. A group of scientists hypothesized that this difference means that the cell wall has largely different functions in plant cells and fungal cells. Alternatively, another group of scientists hypothesized that despite their biochemical differences, plant and fungal cells serve similar function. Okay, so we're talking about being similar, and that's our alternative. All right, which of the following observations would best support? Okay, we need to support our guy with the alternative hypothesis. All right, so let's look at these. Plant cells are found just outside the plasma membrane, while fungal cell walls are found just beneath. Those two things are opposing each other, so that's not similar. So we gotta get rid of that. And let's jump to C. Some plant cells have secondary walls that confer additional rigidity, while fungal cells do not. As an opposite, not similar. And photosynthesis occurs in plant cells, but does not. Those are opposites too. B. In both plant cells and fungal cells, the cell wall surrounds the outside of the cell membrane. Yes. Okay. Talking about in both, and it's doing the same thing, and that's what our alternative hypothesis said. All right, I'm not going to interfere with this one too much. I'm going to let you do it yourself after I read it to you. All right, a human kidney filters about 200 liters of blood each day. Approximately 2 liters of liquid and nutrient waste are excreted as urine. The remaining fluid and dissolved substances are reabsorbed and continue to circulate throughout the body. Antidiuretic hormone ADH is secreted in response to reduced plasma volume. ADH targets the collecting ducts in the kidney, stimulating the insertion of aquaporins in their plasma membrane and an increased reabsorption of the water. If the ADH secretion is inhibited, which of the following would result? And I will tell you this: if ADH, so if you have a diuretic, that makes you urinate. All right, so antidiuretic means you're not urinating. Okay, all right, so A, the number of aquaporins would increase in response to the inhibition of the ADH. B, the person would decrease oral water intake to compensate for the inhibition of ADH. C, blood filtration would increase to compensate for the lack of aquaporins. And D, the person would produce greater amounts of dilute urine. I'll give you a few seconds, and I'm not going to answer. I'm going to Okay, and I hope you chose D. Okay, so that what was D? D was the person would produce greater amounts of dilute urine, because if your ADH is inhibited, that means that you are urinating a lot. Okay, this is wrong because this one right here, because it said that the ADH stimulates. Where did it say that? Stimulated the insertion of aquaporins, but we're inhibiting, so it wouldn't do that. It would increase. All right, now a team of biologists developed a new drug, and one team member hypothesizes that the drug is incapable of freely passing across the plasma membrane and requires the help of membrane proteins to enter cell. Alternatively, another biologist on the team, here's another alternative. Okay, lots of alternative biologists today. Anyway, on the team hypothesizes that the drug can diffuse passively across the plasma membrane, like O2 and CO2. Which of the following, if true about the drug, best supports the alternative? We're supporting the alternative again. Okay, that this new drug will exhibit simple diffusion across the plasma membrane. Would it be A, the drug is a small non-polar molecule? B, the drug is a small charged molecule? C, the drug is a large polar molecule? And D, the drug is a large charged molecule? Okay, I can't help myself. I'm gonna go ahead. I'm gonna get 2 from you. All right, which one do you think it is? And I hope you chose. It's a small non-polar. Okay, all right. I think this is the last one of our multiple choice question, and then we'll move on to a little small FRQ. All right, which of the following statements best explains the processes of passive and active transport? A, passive transport is the net movement of substances down a concentration gradient that requires metabolic energy. Active transport is the movement of substances up a concentration gradient that does not require energy. B, passive transport is the net movement of substances down a concentration gradient that does not require metabolic energy. Active transport is the movement of substances up a concentration gradient that requires energy. I hope you're keeping up with me. All right, C, passive transport is the net movement of substances up a concentration gradient that requires metabolic energy. Active transport is the movement of substances down a concentration gradient that does not require metabolic energy. And D, passive transport is the net movement of substances up a concentration gradient that does not require metabolic energy. Active transport is the movement of substances down a concentration gradient. So which one of these are you going to pick? Because you know it has to say, it has to go passive. Passive goes from high to low, no energy. Active goes from low to high and needs energy. So one of those said that combination of things. I'll give you about 10 seconds to figure that out. Okay. Oh, and B, let's look at B. Passive transport is the net movement down a concentration gradient, right? And does not require energy. Yes. Active transport, movement of substance up a concentration. What do I want you to say? I guess. Okay. Gradient that requires energy. All right. And this is the last one. Just kidding. All right. So we have a diagram here of cell membrane. We have a protein in that cell membrane. We also are using ATP, and we're moving in this direction. And what's happening happening? We're pushing hydrogen or protons, whichever one you call it, hydrogen ions or protons, against the gradient. Okay, which of the following, excuse me, which of the following best predicts the effect of not having ATP available to supply energy to this process? So where does this fit on question number 2, right? If what happens if we took that out, what's going to happen? So our choices are A, hydrogen ions will stop moving through the protein. B, hydrogen ions will move in the other direction through the protein. C, hydrogen ions will continue to move through the protein in the original direction, but at a slower rate. And D, hydrogen ions will begin to move through the phospholipid portion of the membrane in the original direction. And I hope you chose A. All right, that's the interval choice. I know that we put that there. Now we're going to write A. Let's look at this FRQ. Oh, this looks familiar, doesn't it? Well, I just kind of like this question a lot, and I think that a strong understanding of hydrogen ion concentration gradients are so important for you to take forward as you move to photosynthesis and cellular respiration. So that's why I kind of stayed with it. We did A in the warm up, but now we're just going to jump to B. So same thing. We have hydrogen ion concentration or higher in the shaded area. I'll read the scenario one more time, and then we'll see what B wants us to do. All right, mitochondria and chloroplasts are the energy processing organelles of eukaryotic cells. Both organelles contain outer and inner membranes. The inner membrane of the mitochondria contains many folds. The chloroplasts have a third membrane forming the thylakoid. Molecules such as enzymes responsible for the synthesis of ATP are embedded in the inner membrane of the mitochondria. Okay, so this is where those enzymes are. Okay, and then on this thylakoid membrane also, and embedded in the inner membrane of the mitochondria and the thylakoid membrane of the chloroplast. So B, based on the diagram, describe how the structure of these organelles control the movement across the membrane. Okay, so now we're talking about how are we making those hydrogen ions stay over there where they're at. And B, I'll give you 10 seconds to maybe 15 because this should be easy. All right, so let's check yours against the scoring guidelines. And you could have said any of the following. You could have said, I would say this one, I would have said this one. Selective permeability, and then I probably brought that in. We haven't talked about compartmentalization yet. I'm going to do that on the next lesson. The compartmentalization will fall in at the end of that. And then surface area to volume, just did this yesterday with Ms. Katay. All right, so I hope you said at least one of those things. All right, so here's part C. Complete the table to represent the relationship between the volume and the surface area of a sphere. Okay, here's a sphere again, and surrounded by a membrane. So I changed this from there was a table that had some flipped information, and so I changed it around a little bit. You've seen this before in your topic question in AP Classroom. So here's your sphere radius. Okay, there's 1 and there's 3, and then we're going to figure out what the volume is of the sphere. We're going to figure out the surface area, and then we're going to do a ratio of surface area to volume. Okay, this is what we're going to do. So all we have to do is plug in that radius. So for the volume, this would just be 4/3, okay, times 3.14, and R is what? R is 1, so R to the third is still 1. Okay, so that's how you figure out the volume there. The volume of the other one would just be 3 times 3 times 3, which would be 27, and substitute the same formula. And then the surface area is 4 times 3.14 times R squared on each, would be a 1 squared here, or 4 times 3.14 times 3 squared, or the surface area of the 3 millimeter sphere radius. So I'll let you do that math real quick, and then also look at D and explain how the folding of the mitochondrial inner membrane and the individual chloroplast thylakoids are beneficial to a cell that is carrying out chemical reactions. Okay, so take care of C and D. I'll give you about 45 seconds to do that. So I think this will move pretty quickly. Okay, let's take a check. I'll look at our scoring guidelines. All right, so your table should look like this, and this is going to be 3:1, and for our 1 millimeter ratio, and there's going to be 1:1 for our 3. This is larger. That one's a larger cell, okay, or a larger sphere. So this smaller one absolutely should have a greater ratio of surface area to volume. 3:1. This means that it's going to be more efficient in moving molecules in and out of that cell that it needs to processes it needs to take care of. And then part T, the response indicates that the folding increases surface area so that the membrane can hold more enzymes and more reactions. Okay, you can ATP when it's in parentheses on a rubric. That means it was optional that you said that. More reactions can take place, and if less surface area was present. So this was about increasing the surface area. So I think that you probably were able to do both of these well. All right, so debrief and summary. Takeaways for today. You should be able to describe the role of each component of the cell membrane. You should be able to explain how biological membranes influence selective permeability. You should be able to describe and explain transport mechanisms for moving large molecules across the plasma membrane and the processes that allow ions and other molecules to move across the membrane. You should also be able to describe the fluid mosaic model, explain the role of the cell wall in maintaining cell structure and function. And as always, you need to be able to describe it, explain it, and predict what would happen without it. Common errors and misunderstandings. You have to read what the question is asking you. You need to reread it, okay? So don't just read it once. Make sure you understand exactly what part. So they may give you all of this text, but the question is asking you about this line and not all these others. And you need to make sure you know what the question is asking of you. Now, they should also focus on the bold printed terms, okay? That's going to tell you if you learn your task verbs, you learn how to respond to those. This is kind of like training. You're learning biology, but you're also training to answer these questions. All right. Let's see. Homework. Proteins, large complex molecules, are major building blocks of all living organisms. You need to discuss the following in relation to the proteins. Oh, I'll finish this now. The role of proteins in membrane structure and transport of molecules across the membrane. So what you're going to write, you're going to discuss the roles of protein in membrane structure and transport of molecules across the membrane. So all of this that we've talked about today, that's what you're going to write about. Okay, and there is a lot of information in the rubric scoring guideline your answer will fall into. Okay, there's many things to say. All right, I want to go back. I want to add something. Okay, when you're doing surface area to volume, okay, a lot of kids do this and then they get it wrong. Okay, so make sure you say surface area to volume. I'm going to add that. All right, and device and internet access. You need to do this by the 24th. We know that not all students have access to the internet or device. We're working on solutions to help students get what they need to show their best work. If you need mobile tools or connectivity or know someone who does, you can reach us directly to let us know. cv.org/tech. And thank you for indulging me on using this new tool. I felt so much more comfortable, like I was back in my classroom. I'm able to write on the board, and I'm able to engage. I hope that you were more able to engage. Only thing that didn't happen was the power didn't go off, because that kind of happens in the classroom. All right, so next review session, facilitated diffusion and the diffusion portion of investigation 4, and then that will be Monday live and then recorded after that. And then Tuesday, Ms. Katay will be talking about the rest of the investigation 4, which has to do with osmosis. Okay, thanks for watching, and I'll see you next lesson. It'll be me on Monday. Bye. Welcome to AP Biology. I'm Gail Pfizer from Darnell Cookman Middle High School and Florida State College in Jacksonville, Florida. For an overview of our lesson today, we'll be talking about facilitated diffusion and compartmentalization and its origin. And by the end of this lesson, you should be able to score points for these learning objectives. Go ahead and grab pen and paper if you haven't already. We're going to explain how the structure of a molecule affects its ability to pass through the plasma membrane. We're going to describe the membrane-bound structures of eukaryotic cells. We're going to explain how internal membranes and membrane-bound organelles contribute to compartmentalization of eukaryotic cell functions. We're going to describe similarities and differences in compartmentalization between prokaryotic and eukaryotic cells. And we're going to describe the relationship between the functions of endosymbiotic organelles and their free-living ancestral counterparts. Before we get started, I wanted to review a diagram that I had in this. It was Fridays, and it had to do with cell membranes and proteins in cell membranes. And I had put together an image like 3 different times during the day. At the last minute, actually put the wrong one in. And so I believe it was only this particular word, the integral, that I wanted to. That I think that it said peripheral on Friday on the last lesson. Anyway, so I just wanted to take you through this one more time. If we have a transmembrane, okay, transmembranes means we have a protein that spans the plasma membrane and it's on the exterior and it's located on the interior. So this would be one, and this of course is one, and we see this one. So those are transmembrane. Now this can also be called an integral because it is embedded into the cell membrane itself. So this also could be an integral, but it is not transmembrane. So all transmembrane proteins within the cell membrane are integral, but not all integral are transmembrane. I think I tried to say that in the last lesson. I'm not sure if I did. Let's go ahead and talk about some other things while we're here. Let's do cholesterol. So here we have our 4 fused rings. There's our cholesterol molecules, and then we have our carbohydrate chains. This particular one is a glycolipid, so that's the glyco part, the carbohydrate part, and it's tethered to a lipid within the cell membrane, so that's a glycolipid. And then we have another one here is a excuse me, we have a glycoprotein, and it's attached to this protein. So there's the glyco part, is the carbohydrate part, and then the protein is underneath it. Excuse me. And then I think we covered everything, and the phospholipid bilayer. Anyway, so in the future, well, we'll go backwards because we're going to put all of them on. There's going to be a Google Drive that you can access that are going to have all the slide decks of all of these presentations. And so what I was thinking is that I know that sometimes my students like to fill in the blanks of trying to remember what something looked like. So I think when it's appropriate, then I'll put a blank slide like this one. Excuse me for being so scratchy. I'm trying. Okay, so like you would say, this was cholesterol. Okay, so I'm going to put in. I'll leave this blank for you, and then when you can download the slide deck, or you can look at this again. You can fill it out yourself. Okay, so now for our warm up. Now that you're really ready. Okay, so this was the Friday, the last lesson, which was from membrane transport, and that was the homework then. So it's the warm up now. Proteins, large complex molecules, are major building blocks of all living organisms, and your job is to discuss the following in relation to proteins. First, the roles of proteins in membrane structure and the transport. So there's two things you're going to discuss, and I would treat discuss kind of as a combination of describe and explain. Like the characteristics are important, and how and why, if that's appropriate. I'll give you just a moment to work on that. If you already have that ready, then just hold on tight, and I'll show you the scoring guideline. I'm gonna get my timer out. Okay. Let's take about 10 more seconds. Okay, so let's look at the scoring guideline. So there are a number of things you could have said, but I'm gonna focus on the system role and then about the transport. Now, this you could have said a number of things. Description of integral and peripheral proteins. I probably would have said that. Membrane synthesis defines membrane sidedness. This you could also went into receptors, enzymes, cell-to-cell communication, all of those things that you already know because you already covered all the content and that you're reviewing now. Role in transport proteins may be specific. So I'm going to need ATP. Remember this active transport ones, and description of transport mechanisms, the binding, the conformational change, and the release of a molecule. You could have went on into that, and then how proteins form channels. I probably would talk about channels and moving through them, and then you could have elaborated to specifics. You could have talked about ATP synthase and how that hydrogen ion concentration gradient drives the production of ATP, and you could have also talked about the sodium potassium pump, which we will talk about today. We talked about receptor mediated on Friday. All right, I'm gonna go ahead and move to the next slide. All right, so what you need to know. So membrane proteins are required for facilitated diffusion of charged and large polar molecules through a membrane. So examples that we're going to talk about aquaporins, and we're going to talk about channel proteins. So as we talked about last time we were together, we talked about how very few water molecules can go through the membrane because they're partially charged. So you must have aquaporins enabled to go from a high concentration gradient to a low concentration gradient of water. Okay, or you could say high free water to low free water, or you could say high water potential to low water potential. Let's go through here. So we have water can move through this, and it would move towards this area if we had a higher solute in this particular area, in this cell, inside the cell versus outside the cell. Okay, so now I need to make myself just stay focused on this diagram because I want to go into so much more that I will in the next slide. All right, so charged ions, including sodium and potassium, require channel proteins to move through the membrane. So if you look here, it says sodium's high on the outside of the cell. So we have a high concentration of sodium, and it's going to move to a lower concentration of sodium. So it's going to move from high to low, and it's going to it can only do that through a sodium channel. Now there's a higher concentration of potassium on the inside, and it's going to lead from a high to a low concentration as well. And it also is going to need this channel to do so. Now, once you get the ions on the wrong side of the membrane, when you have the sodium too much is over here, and then potassium too much is out here, then you're going to need your sodium potassium pump to move the sodium back out and the potassium back in. So I'll take that a little bit further on the next image. Okay, membranes may become polarized by movement of ions across the membrane. So I'm going to draw real quickly. All right, so this is a phospholipid bilayer, my version. And let's say we have here's our sodium channel, and here's our potassium channel, and I'll leave it at that. Okay, and then I'll catch the sodium potassium pump on the next slide. So inside the cell, let's say this is a neuron. The inside the cell's membrane potential is negative 70 millivolts. So this is at resting. So this is right here on this graph. Now, if you have a something that happens, say it's a neurotransmitter bind or a voltage, this is a voltage gated, or it could be a ligand rate gated depending on its location. And it's going to, when there's electrical disturbance, this is going to open up. So we have what we did have was a bunch of sodium out here, right? So the sodium is going to move when it opens from high to low. Now that moment right there takes us from a resting membrane potential of negative 70 to a plus 30ish. Okay, so now we're positive on the inside of the cell, and then the next channel, this potassium channel, is voltage gated. And when this change in membrane potential happens, then it's going to open, and then the potassium that was higher on the inside is then going to move out of the cell. So that moves us back to negative. But as we'll talk about, and that was this moment right here heading back. As we on the next slide, we're going to talk about why that just can't be. Okay. All right. So the sodium potassium pump is going to contribute to this. So what I just did was take us through here in step one. We have higher sodium on the outside and low sodium on the inside. We have high potassium on the inside and low potassium on the outside. And this is normal resting potential right here. Okay. So if we now, if this channel was the sodium channel was the open, then that sodium is going to move through, and that's going to change the membrane potential then to positive. And that's where that positive 30 mark is coming in. 20 something 30. Okay. So now we have more sodium and more potassium on the inside. So it's positive. And now the potassium channel is going to open. Or we're doing that. Okay. So we have to do it here. Potassium is going to open, and the potassium will move. Here we go to the outside. So we have had an increase on the inside potassium, and it went through, and now we have potassium on the outside. So we're back to a negative membrane potential, which is fine, right? But another action potential can't happen if you have the wrong ions in the wrong place. You can't have your sodium on the inside, potassium on the outside. So the sodium potassium pump is going to maintain that membrane potential and allow more action potential to occur by getting them in the right location. It's going to pump the potassium back in, and it's going to pump the sodium back out. Okay. All right. I know. I think I've showed this image so much, and it's going to continue on because we're going to be going into photosynthesis and cellular respiration for the next few days. So just at least you know this diagram. All right. Membrane proteins are necessary for active transport. Metabolic energy, such as from ATP, is required for active transport of molecules and or across the membrane and to establish and maintain concentration gradient. So let's look at this gray area. Right. So here's that gray area, and remember this was areas of higher concentration. So we have a higher concentration of hydrogen ions built in this intramembrane space and also in these thylakoid lumens. So why? So it's being compartmentalized in that location to establish this gradient. So it's going to be for a purpose in photosynthesis and cellular respiration. But how does this happen in the first place? It's going, if we're pushing hydrogen across the membrane, we're going to have to go from a low to a high concentration gradient. And to do that, we're going to need to use ATP. So from our last lesson, this was active transport, right? We went from a low to a high concentration, and we needed ATP to do that. Okay, from low to high. Okay, so in my classroom, you probably wonder what blocks had to do with this. Before I allow kids to try to measure Jell-O with a ruler, I have them practice with blocks, and I have them determine the surface area and the volume and they get the ratios and learn all the math. This year we added a cylinder. Okay, and so because that in CED there was the surface area of a cylinder and a volume of a cylinder was added. So after we spend our time and we know what we're doing, then I let them boost on the Jell-O. So investigation 4, part 1, diffusion. To maintain homeostasis, materials move in and out of cells. Diffusion moves these molecules from an area of high concentration to low concentration. This lab uses artificial cells to help students study the surface area to volume relationship in regards to diffusion efficiency. We first learned how to calculate surface area and volume, and then how to present the ratio. The cubes are made with unflavored gelatin, sodium hydroxide, ethanol, and phenolphthalein. Then the cubes are placed in vinegar and timed until they turn clear. Why do the cubes turn clear? The vinegar and acid diffuses into the basic block, decreasing the alkalinity. However, the indicator that turned the blocks pink was in response to an alkalinity, and that's why they're turning back clear. Okay, so here's a sample of my kids. There's their blocks. Okay, those are really huge blocks, and they take a very long time to get some results with blocks that large. All right, so here's a good sample. We have a small sample. These are kind of out of order. You can see that this one is already clear, and this block is almost clear, and this one has quite a ways to go. So here's a sample. Oh, wait, so this I can't show this, but it is linked to the PowerPoint slide that will be available to you if you want to watch it. It's on my YouTube channel. But this was what do you call it? A time lapse. Kids do that in my room a lot, and they'll to show their experimental process along the way. So that's a small block. This was the medium block, and then way back here was the larger block. And so this one clear first, and then second, and then third. Okay. All right. Here's some sample results from doing so. Here's cube 1, cube 2, cube 3, and I recommend that when you're trying to, if you had this as a test question on exam, that you put your chart in the order that you need to say it. Like if you need to at the end, if you need to talk about the surface area to volume, then make sure you have here's the surface area, here's the volume, I divide, and then I get this. Okay. So cube 1, we had surface area to volume of 1.5 to 1, and cube 2, 3.3 to 1, and then in cube 3, 6 to 1. So the smaller cube, here we go, smaller cube right here. This was a medium cube, and this was a large cube. Had a greater surface area to volume ratio, and it went clear the fastest. This meant that the this was a cell, the nutrients can diffuse more efficiently into the smaller cell. All right, compartmentalization. Eukaryotic cells maintain internal membranes that partition the cell into specialized regions. Intracellular metabolic processes and specific enzymatic reactions, I'm going to go ahead and underline that, are compartmentalized within membrane-bound organelles in eukaryotic cells. This compartmentalization contributes to the functions within a eukaryotic cell. Okay, so we have all of this is happening. We're establishing these concentration gradients, and we're compartmentalizing it into intermembrane space here and in the lumen of the thylakoid for a reason, for those enzymatic reactions to be allowed to happen. Okay. All right, prokaryotes generally lack internal membrane-bound organelles, but have internal regions with specialized functions and the strong one. All right, so let's say this is our prokaryote, and it has one continuous long circular chromosome. Okay, it doesn't have a nucleus. It doesn't have a nuclear membrane-bound nucleus. But what it does have is it's called a nucleoid region. Okay, where you'll find that. All right, so one circular chromosome is located in the nucleoid region of a prokaryotic cell. Other internal regions perform respiration and photosynthesis on extensions of their cell membranes that use a proton gradient and enzymes to produce energy. So you may have, let's see, let me do an example. So say this is a prokaryotic cell with respiratory properties. Okay, let's use that word. Then there are other internal membranes that they can have the respiration occur on. And same thing if this was a photosynthetic. There's other internal membranes where this happens. Okay, theory of endosymbiosis. I know I've already talked about this in one of the evolution videos. Membrane-bound organelles evolve from once free-living prokaryotic cells via endosymbiosis. So here we have our prokaryotic cell right here, and here is our circular chromosome in our nucleoid region. And in the next step, we have that cell membrane is invaginating into the cell, and this will eventually be our nucleus. And this will then have your endoplasmic reticulum, and your Golgi, and your vesicles, and your plasma membrane. All that's made of the same materials, and that would be part of what's called the endomembrane system. Different from endosymbiosis, but sounds a lot alike. Okay, so in the next stage, you see that this large cell is engulfing this prokaryote that has respiratory properties. Okay, so now this cell is using this prokaryote to produce ATP. Okay, and the same thing can happen with the chloroplast. This was originally a prokaryotic organism, came in with membranes that had photosynthetic functions, and then it became a chloroplast. Okay, and then we know this because two major reasons we know this is that the double membrane. So say this was a cell that is engulfing this other cell. See the membrane of the cell one engulfing the other cell, and that makes us have our double membrane. Another feature is that this prokaryote came in with its own DNA, and so did this chloroplast. And as mitochondria and chloroplast, well, as prokaryote, as a mitochondria and a chloroplast, they have their own circular DNA also. All right, so the science skills that we're going to focus on 6B: support a claim with evidence from biological principles, concept, processes, and/or data. And then we're going to also predict the causes of effects or of a change in or disruption to one or more components in a biological system based on biological concepts or processes, and some task verbs that we'll use a couple of times today to describe and support. Excuse me. Okay, so we're going to go through about 4 questions. I think some multiple choice a little bit later to see if you understand all the content. But we're going to write this one FRQ together. So what I want you to do first is what I do. You have to do, but look at the diagram first. Here we have a cell, and I can tell we have a eukaryotic cell because we have a nucleus. We have endoplasmic reticulum and some Golgi bodies, right? We have mitochondria, but we have also this apicoplast, and that might be a new word for you, but that doesn't necessarily mean anything. Okay, you just need to look at the characteristics of this. If you can see, it has its own DNA, and then it has another feature. It has what? It has a double membrane. Okay, now let's read the question. Based on figure 1, describe 2 pieces. Okay, so if I was teaching this in classroom, I would say box in your command words, circle your how many pieces of what? Of your evidence a researcher could use to support the claim that apicoplasts evolved from free-living prokaryotic organisms. I'm going to give you just about 20 seconds to jot those down quickly. Okay, let's look at the scoring guideline. So if you said possesses a double membrane and possesses DNA, you have both of those points. All right, so now let's do the multiple choice to make sure you understand everything that we talked about. Number 3, researchers claim that a particular organelle originated from a free-living prokaryotic cell that was engulfed by a larger cell, as shown in figure 1. So here's figure 1. All of this is figure 1. Here's the question where it starts. Which of the following provides evidence to best support the researcher's claim? The organelle has a phospholipid membrane. Remember this, we have to support the researcher's claim that said it was engulfed. Okay, the organelle has protein in the membrane. C, the organelle has a double membrane. Or D, the organelle has internal aqueous environment that is similar to the cytosol of the larger cell. So 2 seconds there, make your decision, and I hope you said C, right? That is the only one. While other answers can be right, this is the one that the answer is what they're asking to support the researcher's claim. And then you have to go, what exactly did he claim? He claimed it was engulfed by a larger cell. All right, so this is a familiar diagram. I'm sorry, this print is so small, so that's I could do to get on here. But at least you're familiar with it already. A cell's membrane potential is maintained by the movement of ions into and out of the cell. A model showing the influence of membrane proteins on the movement of sodium and potassium ions across the plasma membrane is presented in figure 1. Okay, so we're familiar with figure 1 already. All right, based on the model presented in figure 1, which of the following outcomes will most likely result from a loss of protein X function? Oh, here we go. Okay, so if we took out protein X, this looks just like what question 2, right? Part C, where you're going to predict what's going to happen if you take something out or you change it. Okay, this is similar. All right, so based on the model in figure 1, which of the following outcomes will most likely result from loss of protein X function? A, the membrane potential will be disrupted by an increase in sodium concentration inside the cell. B, the membrane potential will be disrupted by an increase in K concentration inside the cell. C, the membrane potential will be maintained by the sodium potassium pump, moving more K ions into the cell. And D, the membrane potential will be maintained by the diffusion of sodium ions in the cell. I'll give you 20 seconds. Okay, let's take a look at the answer. And I hope you chose B. Okay, let's talk about Y. All right, so let's take out protein X. So what can't happen now? K is not going to be able to leak out. So it's going to be still in here. This pump, okay, this only has to do with sodium, and this pump is going to pump potassium back in. So this is going to be disrupted because of increased K inside the cell. All right, which of the following statements best supports the claim that certain organelles within eukaryotic cells evolve from free-living prokaryotic cells? The cytoplasm of both eukaryotic and prokaryotic is surrounded by a plasma membrane. Oh, this kind of looks like something we've already did, just a little bit different. Eukaryotes and prokaryotes both contain ribosomes, but the ribosomes of eukaryotes are more complex in structure than those of prokaryotes. C, eukaryotes exchange segments of internal membranes between the endoplasmic reticulum and Golgi apparatus, but prokaryotes have no such internal membranes. D, some organelles contain their own DNA that is more similar to prokaryotic DNA in structure and function than to eukaryotic DNA. Okay, so just take 5 seconds because I know you know the answer. Okay, and the answer is D. All right, I think that's the end of my multiple choice. No, sorry. All right, this is December 11 in particular test bank questions that I made. All right, the endosymbiont theory proposes a model for the evolution of mitochondria. According to the model, an ancestral eukaryote engulfed a small free-living prokaryotic organism. The engulfed prokaryote then formed an endosymbiotic relationship with the eukaryotic host, which the following observations best supports that model. A, prokaryotes and eukaryotes acquire nutrients from the surrounding environment. B, organelles such as mitochondria and the endoplasmic reticulum have membranes composed of phospholipids. C, mitochondria and some prokaryote shells share similar metabolic reactions that produce ATP. And D, eukaryotes evolved after prokaryotes and have more complex structures. I'll give you just 5 seconds on that one, and I hope you chose C. Okay, now that's the end of. I think that you pretty much have established that you understand the theory of endosymbiosis. All right, so now we're going to do an FRQ together. I love this question. It actually made me go look for the research too, and I actually saw there in the found online that I can't put on here, but I saw these red buds and then the blue flower. The petal color. Let's look at the boxes first. So what do we have on our table? We have vacuole pH, we have flower color, and we have cell volume. So in the bud, we have a 6.6 pH, and it's red and it's small. The cell volume is small. Okay, so and then the vacuole pH in here is 7.7, and then it turns blue, and then it's large. So this more alkaline pH causes it to be blue. All right, so what's going on inside the cell? The cells have a potassium channel, and that one has a potassium channel. They both have proton pumps. There's one, there's another. I can see something different here. The potassium hydrogen transport protein is active in the open flower, whereas not in the bud. Okay, and then we see water moving into the vacuole. So let's read this. The petal of the Mexican morning glory changes from red to blue, and the petal cells swell during flower opening. The pigment heavenly blue anthocyanin is found in the vacuole of petal cells. Petal color is determined by the pH of the vacuole. A model of the morning glory petal cell before and after flower opening is shown in table 1. So the first thing you need to do is identify the cellular component in the model that is responsible for the increase in the pH of the vacuole during flower opening. So we're increasing pH. What is responsible for? Just write the 1-2 words down, whatever it takes. Okay, go ahead and do that. All right, and then you're going to describe the component. What did it do? Okay, so you identified something, and then why did you say it was that? Is what you should answer there in your describe. What's happening at that location? I'll give you just a couple seconds, maybe 10. All right, and then part B. A researcher claims that the activation of the potassium hydrogen transport protein causes the vacuole to swell with water. Provide reasoning to support the researcher's claim. So what happened? Because the researcher claimed that this transport protein caused the vacuole to swell. So he said that this right here caused this to swell. Okay, so you describe why. You provide the reasoning why. And I'll give you 10 seconds for that. Okay, so let's look at the scoring guideline. And part A, this is when I've got you both of those tests first. The potassium hydrogen transport protein is identified as a cellular component and responsible for the increase in pH. You probably wrote more than was needed, but it's better to write a complete thought than to try to figure out what the rubric wants you to do specifically. Like answer the question completely. All right, part B, response includes one of the following reasonings. The concentration of solid K is increasing inside the vacuole, and the solid K is moving into the vacuole, and that's making it hypertonic, hyperosmotic, and lowering the water potential. So you have work tomorrow with Ms. Kate. You're going to be reviewing osmosis, water potential, and tonicity. All that will happen tomorrow. But if you figured it out today, that's good. Because look at all this K. All this K had higher solid here, so that water is going to move right in there. Okay, so your takeaways for today. The structure of a molecule affects its ability to pass through the plasma membrane. Membranes contribute to compartmentalization and contribute to the functioning of eukaryotic cells. You should also be able to describe the membrane-bound structures of the eukaryotic cell. You should be able to describe similarities and differences in compartmentalization between prokaryotic and eukaryotic cells, and describe relationship between the functions of endosymbiotic organelles and their free-living ancestral counterparts. And always, my motto: describe it, explain it, and predict what you would do without it. Okay, some common errors and misunderstandings. Students, okay, so if you write this, it is not the same as this. Okay, if you were in interpretation-wise, if you were to say the same numbers and you were to say 10 to 1, and then you had 10 over here, and you said your surface area to volume ratio is 1 to 10, you have to be very careful in how you write things in what order, and then how you complete your sentence with those terms. All right, and then another place is the location of proton gradients and their role in ATP production. We're moving into photosynthesis instead of respiration after osmosis tomorrow, and that's really exciting. And I think that we probably established a real good handle on proton gradients, and then we can just take it one step further, and you have photosynthesis and cellular respiration down. Then tomorrow you will talk more about osmosis and all that, but the movement of sodium and potassium into the cell and out of the cell, and how it's getting in one place. That sodium potassium pump, you really don't need to know the action potential part of the cell membrane, but it's a good way to learn the role of the sodium potassium pump in the cell. All right, so for your homework, you need to so a difference between prokaryotes and eukaryotes is seen in the organization of their genetic material, and your job is to discuss the organization of the genetic material in prokaryotes and eukaryotes. Now, I do not want you to just write the prokaryotes do not have a nucleus, a nuclear region, and the eukaryotes have a nucleus. It's bigger than that. Go back into the things that you learned in, maybe some hints if I can think of what's in gene regulation. Okay, go into unit 6 and think of things that you could say about the genetic material to be different prokaryotes versus eukaryotes. Okay, all right, and device and internet access, you still have time. Do this by April 24th. We know that not all students have access to the internet or a device. We're working on solutions to help students get what they need to show their best work. If you need mobile tools or connectivity or know someone who does, you can reach us directly. Let us know at cb.org/tech. And I got you out of here in about 35 minutes today. Thanks for watching. We'll hope you will join us again. Be sure to complete your homework question. Be ready for the beginning of the lesson tomorrow on osmosis, which is membranes, osmosis, and the osmosis portion of investigation 4. And that's with Ms. Kate tomorrow. Thank you. Bye.
Hi, welcome to today's AP Biology lesson. My name is Allison Kate, and I'm teaching today from my home in El Cerrito, California. And I thought that I would give you a little geography lesson before the biology lesson today. For those of you who may not be sure where El Cerrito is, it is about 15 miles east of San Francisco and about 5 miles north of Berkeley. So from my table today, I can look out and see the Golden Gate Bridge, and it's a beautiful day here. So thanks for being with us. I usually start, for those of you who have been tuning in, by talking about the 2020 exam details. And today I thought I would skip that and just jump right into our lesson. For those of you who are just joining for the first time today, you can see our recorded lessons, and all of that information is in those lessons. So let's get started with our exam overview. Okay, excuse me, our lesson overview. So today, maybe, awesome. All right, there we go. Today we are going to be reviewing topic 2.8, which is on tonicity and osmoregulation. And these are traditionally very difficult topics. So I'm going to give you lots and lots of practice. We're going to work on science practice 4, which is constructing a graph, and science practice 5, which is performing mathematical calculations. And we're going to be working today with 2 formulas. These are from the AP Biology formula and exam sheet, and they have to do with water potential. So don't forget your pen and paper. And also today, you'd probably be happiest if you had a calculator with you. Before we embark on today's lesson, you should be familiar with the principles of diffusion and osmosis and cell membrane structure. And you should also be familiar with the terms hypertonic, isotonic, and hypotonic. I'm going to give you lots of practice with those today as well. By the end of this lesson, you will be able to explain how concentration gradients affect the movement of molecules across membranes, explain how osmoregulatory mechanisms contribute to the health and survival of organisms, and construct a graph. So we're going to get started with our warm-up. And you know that this is a question that was given from the previous lesson. So my colleague, Ms. Pfizer, gave you a lesson yesterday on membranes and facilitated diffusion. And she asked you for homework to discuss the organization of the genetic material in prokaryotes and eukaryotes. And she gave you a hint. She told you to think about what you know about the nucleus and think outside that area. Okay. So if you didn't do this last night, be thinking about it. Maybe you can jot down some ideas. And if you did do it, grab your homework so you can compare your answer to the scoring guidelines. So first, let me explain how this was scored. This was worth 4 points. You got 1 point for each bullet, but you had to have each pair, right? You had to have a bullet from a prokaryote and a eukaryote in order to earn that point. So I think you'll see what I mean when I go over this first one. So if you talked about no introns being present in prokaryotes, you also had to say that introns are present in eukaryotes. In prokaryotes, as we all know, the DNA is not in the nucleus, but rather in a nucleoid region. In eukaryotes, the DNA is in a nucleus separated from the rest of the cell by a nuclear membrane, but it is also found in both mitochondria and chloroplasts. Prokaryotic DNA is not surrounded by histone proteins, whereas eukaryotes is, so it does contain histone proteins. Prokaryotes generally only have one chromosome, whereas eukaryotes have more than one chromosome. And finally, plasmids, or extracellular pieces of DNA, are common in prokaryotes and they're rare in eukaryotes, although they are found in yeast. All right, so I hope everybody got a 4 out of 4 on that one. So here's what you need to know about today's topic. First, let me tell you what tonicity is, and then we'll talk about how it relates to osmoregulation or maintaining an osmotic balance within your cells and body tissues. So tonicity is a measure of the effective osmotic pressure gradient. It's the water potential of two solutions that are separated by a semi or selectively permeable membrane. Another way to say that is that it's the relative concentration of solutes dissolved in a solution, which determines the extent and direction of diffusion. So if you look at my image, I have a cell membrane, and just to review that cell membrane structure, you can see the phospholipid bilayer. So the phosphate heads are represented by those pink circles, and they are polar, and the fatty acid tails are pointed toward each other, and they are non-polar. So those phosphate heads point out toward the outside of the cell and in toward the cytoplasm. And those two solutions that the cell is suspended in may have different amounts of dissolved solutes. So that is what determines the tonicity, which will then determine the way that materials move across that cell. In the middle of this cell membrane, we have an integral membrane protein, which is called an aquaporin. And I tell my students that this is just like a little water slide, right? So the polar water molecules, which are small, can easily pass through this pore in this integral membrane protein because it is a polar channel for the polar water. So now let's look at the actual statement from the course and exam description. It tells you that external environments can be hypotonic, hypertonic, or isotonic to the internal environments of cells. And we're going to define those terms and give lots of examples throughout this lesson. Then it tells you that water moves by osmosis from areas of, and then you have some slashes. And I honestly think that this is why this topic is confusing to students because there's lots of different ways to say the same thing. So we can say that water moves by osmosis from areas of high water potential to low water potential, or we can say it moves from low osmolarity to high osmolarity, or we can say that it moves from low solute concentration to areas of high solute concentration. All of those things are saying the same thing, but they're saying it in a different way. Okay, so when I teach this to my students, I always tell them that the most important thing to remember is to remember where water is, and water will always move from where there is more water to where there's less water. And we'll see examples of this in relation to water potential, osmotic potential, and solute concentration throughout today's lesson. So for a science scale, we're going to practice doing some calculations. The first relevant equation is the equation for water potential. Water potential is represented by the Greek letter psi, kind of looks like a pitchfork, and it's got two components. Psi sub p is the pressure potential, and psi sub s is the solute potential. And again, we'll be talking more about these when we get into the lab activity that has to do with water potential. So let's take a look at these terms: hypertonic, isotonic, and hypotonic. So hypertonic comes from the Greek hyper meaning over and tonos meaning stretching. Here's the definition: it's any solution that has a higher osmotic pressure than another solution. It refers to a solution that has more solute than another solution. So when I'm teaching this to my students, and I have them imagine a solution that's filled with lots and lots of sugar. Sugar is a solute. And if you have lots and lots of sugar, let's say you drink that solution, you'd be super hyper, right? Because there's more sugar than another solution. Okay, so now keep that in mind because now we're going to talk about a hypotonic solution, which is any solution that has a lower osmotic pressure than another solution. This refers to a solution that has less solute and more water than another solution. So now imagine just drinking a glass of distilled water. It is pure water, so there's no solute. That would be a hypotonic solution. Now notice that I'm always talking about these in relationship to another solution. You can't simply say that a solution is more hypertonic, right? You have to compare it to another solution. Finally, an isotonic solution, iso means same. So it refers to two solutions that have the same osmotic pressure across that membrane. And when you have that, it allows for the free movement of water across the membrane without changing the concentration of solutes on either side. So there's movement, but you're not changing the solute concentration. My image shows you both an animal and a plant cell in the three different kinds of solutions. So let's look at hypertonic first. So if we place an animal cell in a hypertonic solution, the hypertonic solution will cause the animal cells to lose water because they have less solute, more water than the hypertonic solution. So the water will leave the animal cell where there's more water for the hypertonic solution where there's less water, and the cells will shrivel. Another term for that is crenate. In a plant cell, notice that plant cell has a cell wall around it, and if you place a plant cell in a hypertonic solution, it will actually lose water to the environment, and we call that being plasmolized. And that's also, if you think about a plant cell that's growing in the soil, if the soil is too salty, let's say that there's been a drought and there's not enough water, or maybe there's been some agricultural runoff that has a lot of salt in it, then the surrounding area is hypertonic, then that plant is going to lose water and wilt, okay, because its cells become plasmolized. An isotonic solution, as mentioned, the concentration of solute or water is the same across that membrane, so you shouldn't see any change in the size or shape of that cell. And you might, if you watch any medical shows or doctor shows, they talk about using an isotonic saline solution if they're trying to give an IV to a patient, and that is telling you that saline is salt and our actual body fluids are a little bit salty, so then you do need to be isotonic. So if you're getting an IV and they don't put in an isotonic solution, then your blood cells can either shrivel or swell. So that's really important. In a hypotonic solution, your cell actually has more solute, right? There's dissolved solutes and proteins and such. So if you place your cell into a hypotonic solution, water will flow in and the cells will become swollen. They could even burst or lyse. And if you look at a plant cell, again, that cell wall, when water flows into a plant cell, it actually makes that cell turgid or stiff, and that's how the cell maintains its sugar pressure so that it doesn't wilt. A loss of sugar pressure is wilting. Okay, so hopefully that gives you a working understanding of these three terms, but we're going to use these throughout this lesson. All right, so first let's read the statement from the CED. It says that osmoregulation maintains water balance and allows organisms to control their internal solute composition or water potential. The formula, and we're going to get lots of practice with this formula. I know it looks scary. It says psi sub s is equal to negative ICRT. I'm going to go through what those are, but it reminds me that several years ago on an AP Bio FRQ, this question was given. The students were asked to describe what each of those components mean. And you gotta love the students. A lot of them didn't know this, and so they entertained us by saying that it means I can't really tell. So none of you are ever going to be in that position because I stands for the ionization constant. That is the solute in the solution, its tendency to ionize. So an example would be if you were looking at a sodium chloride solution, we all know that sodium chloride will ionize in water into sodium ions and chloride ions. If you put a sugar into water, it does not ionize. So that is a value that tells you whether or not the solution will ionize. C is the molar concentration or molarity of the solute, and you'll see when we get to our lab that there's a way to determine the molar concentration of a particular cell in a particular solution. R, you may recall from chemistry days, it is the pressure constant, and you may know it from the ideal gas laws, PV equals nRT. It's the same R, and it doesn't change, and it's listed there. You would never have to remember this, but it's 0.0831. It has weird units, liter bars per mole degrees K, and the T is for temperature, and the temperature is in degrees Kelvin. And so when we do these activities or experiments in a lab, the thermometer that we use will measure in degrees Celsius. So you always have to remember when you're using this formula to convert the degrees Celsius to degrees K by adding 273. All right, so what do these images represent? I'm showing you some onion cells in a hypotonic solution. So onion cells are going to have some dissolved solutes in them. These cytoplasm have been stained with carbonyl fusion, so they look pink. You pop them in a hypotonic solution, and water is going to move into the cytoplasm and cause them to swell. And then we have a single-celled organism. This is a paramecium, and it lives in a freshwater environment. So you might want to think about the consequences of maintaining osmotic balance for that organism. If it's salty, right, because it's got dissolved solutes and it lives in fresh water, what's going to happen, right? The water's going to move in. So what does that paramecium need to do to prevent itself from blowing up? And I'll give you a little hint. I pointed out structures called contractile vacuoles. Okay, so let's take a look at this slide that from the CED tells us that growth and homeostasis are maintained by the constant movement of molecules across membranes. So in this case, the homeostasis is of the osmotic balance in your fluids and cells. So just to follow up on the paramecium, remember their cytoplasm is hypertonic to the environment they live in because it has solutes dissolved in it. So that means that water's going to rush in by osmosis. So what happens to prevent that paramecium from taking on water and blowing up is that those contractile vacuoles act as pumps, and they pump out that excess water, kind of like if you were in a canoe that was taking on too much water, you would bail it out. So those contractile vacuoles bail out the excess water and allow that paramecium to maintain homeostasis in terms of its osmotic balance. Let's take a look at both a freshwater fish and a saltwater or marine fish. So freshwater fish obviously are kind of like the paramecium, but they're multicellular, so they have solute hypertonic concentration compared to the water that they live in. And they have two adaptations that help them maintain their osmotic balance, which is they have super efficient kidneys, so that excretes the water quickly, kind of like those contractile vacuoles. But they also reabsorb the salt that's in their urine before they excrete their urine, and then that minimizes the losses and actively takes salt from their environment. By bringing in that salt can make them minimize the loss of that salt in their cells. And then finally, in a saltwater or marine fish, it has an internal osmotic concentration lower than the surrounding seawater, right? So it's hypertonic, sorry. So it tends to lose water and gain salt. So this adaptation has a way to actively excrete salt out from its gills. So in both the freshwater fish and the marine fish, you see that there are special cells in the gills that maintain their osmotic balance. So just want to see if you're paying attention and see if I can get you to stay to the end. This is topic AK. Those are my initials, and I have a multiple choice trivia question for you. I don't know if you guys are on Facebook, probably not. It's more for people like me. But there's a thing going around that has you list 5 scientists that you've met. Only one is not true. So I'm going to give you multiple choice, and you have to choose the correct answer. And if you stay tuned to the end, you'll see if you were right. So your first choice is Dr. Michael Nachman. He's of rock pocket mouse fame. If you've been tuning in for a while, this may be a giveaway. James Watson, the co-discoverer of the structure of DNA. Rosalind Franklin, who took the X-ray crystallography photograph of DNA that led Watson to make his discovery about the structure. Dr. Bonnie Bessler does research in quorum sensing and bacteria. And Dr. Arthur Kornberg discovered DNA polymerase from a thermophilic bacteria. So I have met 4 of these 5. Which one didn't I meet? All right. So let's take a look at labs now. And there are 3 parts to the diffusion and osmosis lab, which is investigation 4 in the AP Biology lab manual. Ms. Pfizer went through the diffusion part with you yesterday. Today I'm going to take you through the osmosis lab, which uses dialysis tubing, dialysis bags, and then we'll also take a look at the water potential lab that uses potato cells. So in the first one, we are going to investigate osmosis using dialysis tubing, and we're going to look at the relationship between solute concentration and the movement of water through this selectively permeable membrane by the process of osmosis. And I know that you've probably done this lab or seen this lab, but I want to just tell you a little bit about the dialysis tubing. So dialysis tubing is used in kidney dialysis, and this is where patients who have kidney problems will go, and they will actually have their blood cleaned of any excess toxins and salts through this dialysis tubing. And the way it works is they have microscopic pores in them that prevent the excretion of large particles or the movement of large particles through these pores, but prevent small molecules and water to move across them. So what we do is we take this tubing and we cut it into strips, tie a knot in one end, fill it with our solution, and then tie a knot in the other end, and it creates a bag. And then we can use these as a model membrane system to look and see how materials move across that membrane. So a little bit of review. Remember that we're going to be using isotonic, hypotonic, and hypertonic in this lab. Isotonic means that the solutions have equal concentrations of solutes, and so you're not going to see any net movement in one direction. So there'll be no net change in the amount of water in either solution. If 2 solutions, one has more solute, it's going to be hypertonic to the one with less solute, and if a solution has less solute, it's hypotonic to the one with more solute. Again, these words can only be used to compare solutions. So let's get a little practice with this. And this is a technique that I use with my students. I still do this to this day, even though I've been teaching about this for 3 decades. And that is to draw and figure out from your drawing what solutions are in terms of their tonicity, and then figure out what direction the water will move. So we've got a figure over here. It's a beaker and a bag that is identified as a semi-permeable membrane. And we are asked to label which solution is hypertonic and which is hypotonic, and then use arrows to show the initial net movement of water. Keep in mind, water will always move from when there's more water to where there's less water, or from a hypotonic to a hypertonic solution. Okay, so you figure out which is hyper and which is hypo. So the solution in the beaker is hypertonic to the solution in the dialysis bag, and that's because in the dialysis bag you have pure water, no solute. In the beaker you have some solute in the solution, so it's going to be hypertonic to the water in the bag. And this means that there's more water in the bag, so it will move from the bag into the beaker. And if you measure that, you weight it, the mass of that bag will decrease, and that is the technique that we're going to use in our lab. So again, sucrose is hypertonic, the distilled water in the bag is hypotonic, and because there's more water in the bag, water will leave the bag for the beaker. Okay, so here's the lab. You are going to create your dialysis bags. You're going to create 6 dialysis bags. In the first bag, you're going to put 0 molar sucrose solution, which is just distilled water, and then into separate bags, you're going to put 0.2 molar sucrose, and then 0.4 molar sucrose, 0.6 molar sucrose, 0.8 molar sucrose, and finally in the last bag, you're going to put 1 molar sucrose. And then you're going to mass them and record their initial mass, and then you're going to place each of the tubes into separate containers of distilled water. You're going to let them sit for 30 minutes, and then you're going to take them out and reweigh them, and you're going to calculate percent change in mass. So before we look at some data, some results of this experiment, I want you to make a prediction about what will happen with respect to the different molarity solution and mass mass change in the distilled water dialysis bag. So let's start there. In the distilled water dialysis bag that you're placing into a distilled water beaker, what's going to happen? Okay, now make a prediction about what's going to happen in each of the bags that do contain sucrose, starting with the 0.2 and working your way up to the 0.1. Okay, so because the dialysis bags with sucrose solutions are hypertonic to the distilled water in the beaker, water's going to move across the dialysis bag membrane out of the beaker and into the bag, and that means that the bag will increase in mass. The bag placed in the 0 molar sucrose solution will show no change because those solutions are isotonic. All of the bags containing sucrose will increase in mass, but the one that will increase the most is the one with the highest concentration of sugar because it would be the one that had the largest concentration gradient. Okay, so I want you to think about that because that prediction is what we're going to look at in our data. So I'm showing you some data from my class, and I always project the data to my students once they have entered it in. And in this particular lab, you can really spot trends and anomalies. So if you look at group 1, beautiful job, group 1. In fact, we know that there should be no change in mass when you take distilled water and put it into distilled water, and they got that. They also have that trend that as you increase in sucrose concentration, you increase in the percent change in mass. So group 1 did a great job. Take a look at group 2. They're off to a good start with their 0 molar, and look at 0.2 molar. Yikes! So there's some type of experimental error because they have a huge increase in percent change in mass, way over even the 1 molar sucrose solution. So that is an anomaly. Both group 3 and 4 look pretty good. They follow the trend, but they have a little bit of an increase in their 0 molar. Group 5 is great. Group 6, they have the trend, but they have a large change in the isotonic solution. So I'm going to leave this up here. You guys can look at this later. The next step in this lab would be to calculate the means for each concentration and also calculate plus or minus 2 standard error of the mean. Okay, so let's say we did that and we graphed it. And here's our graph. I want to point out that on the x-axis you've got sucrose molarity, which is the units per moles per liter, and on the y-axis we have the percent change in mass, and this is the mean or average of the class. And oftentimes I'll have my students do their group as well as average just to see the difference. And have them graph their plus or minus 2 standard error of the mean error bars. So I'm going to go ahead and just draw in this best fit line, and you can see that the data shows the overall trend, right? That as you increase in the concentration of sucrose in the bag, you increase the mass of the bag. So they got good results. However, if you're talking about statistical significance, if you look at the error bars, right, we do see that there's error bar overlap here. So we know that 2 of the groups or 3 of the groups were not right at the isotonic point of 0 here, and then we know that there's some overlap on the point. So not great data, but this is actual student-generated results, and we see the trend. So our hypothesis would be confirmed by this data. So moving on to the water potential lab. The water potential lab uses potato cores, but you can use any vegetable. I know some teachers use carrots, or you can use zucchini, or some teachers will even have you look at the difference between different kinds of potatoes. And I'll tell you that the whole point of this exercise is to determine the molar concentration of sucrose in a potato cell. In other words, you're trying to find out how sweet a potato is. So I often like to have my students do a regular like brown potato and a sweet potato and really ask the question, is a sweet potato really sweeter than a regular potato? And you can figure that out by determining the concentration of sugar or sucrose within these potatoes. All right, so before we get rolling, let's just review water potential. Water potential is that term that botanists use when we want to predict the movement into our outer plant cells. Again, this is super important for plant cells because they can't move to find water. So if you are a plant cell, let's say you're a potato plant and your roots are in that soil, you need those cells to be hypertonic or have a low water potential to encourage the water to move in from the soil through the process of osmosis. So this is a really important concept for those plants. All right, so remember that we're looking at water potential being represented by that Greek letter psi, looks like a pitchfork, and it has 2 components as mentioned previously. It has a pressure potential and a solute potential. But I want to point out something to you in this lab. Of course, we know that water will always move from where there's more water, a higher water potential, to where there's less water, a lower water potential. So water potential measures the tendency of water to leave one place for another. One way you can do this is picture that water diffusing down a water potential gradient. By convention, the water potential of pure water at atmospheric pressure is defined as being 0. So the pressure potential is 0. Okay, this means that for this experiment, the water potential is equal to the solute potential. So if you look at that equation and the pressure potential is 0, then water potential is going to be equal to the solute potential. Okay, one of the things that I know is confusing for students after teaching this for a really long time is that solute potential is always negative, right? Since pure water has a water potential of 0, any solutes will make that water potential lower. It'll be more negative. Okay, so keep that in mind as we do our calculations. Okay, so again, I'm using some data from my class, and so what happens is you put your potato cells, your carrot cells, or your zucchini cells into beakers with the different sucrose solutions, and you let them sit overnight. You do an initial mass and a final mass and calculate the percent change in mass. Okay, and in this case, you're going to see some of the potato cells are going to increase in mass and some are going to decrease in mass. You can also, if you want more practice, calculate the plus or minus 2 standard errors of the mean. Remember that it's super important that you take the temperature of the liquid in the beaker because you need that temperature data for your calculation of solute potential. So we can see that the overall mean temperature here is 21.8 degrees C. All right, so let's say we graph that, and let me just point out that we still have the same labels. On the x-axis, it's the sucrose concentration in moles per liter, and the y-axis is the percent change in mass over that 24-hour period. This graph is a little bit different, so we're not really using this to display our data. We are using this to determine the isotonic point, or the point at which our best fit line crosses the 0 axis. Okay, and so that would be right there, okay, because the isotonic point represents the point at which the sucrose concentration inside the potato core equals the sucrose concentration in the liquid. Pretty ingenious, right? So when we find that isotonic point, we just drop down to the x-axis, and we can figure out approximately what that molar concentration of sucrose in that potato core is. And in this case, it's about 0.22 moles per liter. Okay, so now we have to calculate it. And I know that looks scary. That's the scary one with the solute potential. So again, I is the ionization constant. We did this experiment in sucrose, so the ionization constant is just 1 because sucrose does not ionize in water. We could repeat this experiment in salt water, and we would find that the ionization constant would be 2 because there are 2 ions, sodium and chloride ions. The molar concentration is C, which we just determined from the graph of the potato core data, and we determined it to be 0.22 moles per liter. R is our old friend, the pressure constant, 0.0831 liter bars per moles degree K. And our temperature, the only thing we have to remember there is we need to convert our degrees C to degrees K, and we can see we do that by adding 273. And now all we do is plug and chug, right? So all the good thing in that crazy units on the pressure constant, everything is going to cancel, and we're going to be left with units that are bars, which are pressure units. So you can check my math, but you would just go ahead and plug in all those values into your calculator, and you're going to come up with a solute potential, which is always negative, right? 5.39 bars. Okay, so let's do some practice. So my first practice is just to see if you're comfortable with the concept of isotonic, right? So this question is asking you the beakers of which dialysis tube are initially isotonic to the distilled water in the beaker. So I want you to look at the initial mass of each of the dialysis tubes, and there are 6, and then you look at the mass after 90 minutes, which would be the final mass, right? So isotonic means they're the same, so we should not see any change in mass, right? So look over your data, look over your choices, and see if you can spot any that do not change in mass. Got it? Correct answer is E, right? The distilled water did not change. It was 11.2 all the way across. Okay, here's some more practice. This is a prediction. So let's say you've got an animal cell, and you're going to place it into a 0 molar sucrose solution. That's pure distilled water. So an animal cell has solutes, right? So putting it into a 0 molar solution means it is a blank, right? Hypotonic solution. Okay. If you take that same animal cell and you put it into a 1 molar sucrose, there's a lot of sugar in 1 molar sucrose. That solution would be Yes, hypertonic, right? So what's going to happen? So you place that blood cell into a hypotonic solution, it is going to gain water, okay? Because the blood cell has solute, it has less water. So the blood cell water is going to move in, and the opposite is true in the hypertonic solution. This was a prior exam FRQ, and so you can see the scoring guidelines here. So in the 0 molar, we would predict that the cell will gain water or mass, it will swell, burst, or lyse, and that tells you right there the importance of understanding this concept. You don't ever want to have your red blood cells either blow up or shrivel up. So the reason for this prediction is that the cell is hypertonic to the sucrose solution. It's actually a 0 molar, so it's pure water, and water will move in. For the hypertonic solution, the cell will lose water or mass, it will shrivel or crenate, and that's because the cell is hypotonic to the sucrose solution, and water potential is greater inside the cell, so water will move to create equilibrium. All right, one more practice with this, and I know you guys have probably done this with microscope practice, where you take a red onion and you peel off that thin tissue, the epidermal tissue, and you go ahead and put that on the microscope slide. You can suspend it in a little bit of sodium chloride solution, and you can see the original slide in that 1% looks like brick wall, right? So the cytoplasm is in gray, and you can see the cell walls that are shown in white. Now the student added 15% sodium chloride solution and observed the changes. So after adding it, look at that cytoplasm, it's all kind of shriveled up and pulled away from the cell wall. There's a special name for that I told you about earlier, so if you remember that. So which of the following most directly explains the changes in the cells? Is it because the degradation of DNA in the nucleus of the cells? Is it because of the lysis of chloroplast in the cells? Is it because of the movement of water from the central vacuole of the cells into the solution, or because the movement of sodium chloride from the solution into the cytoplasm of the cells? Okay, so get your answer, and the correct answer is C, right? The water is moving from where there's more water, where there is less water. Let me say that again. The water is moving from where there's more water to less water, which in this case, in the 15% sodium chloride solution, is that's hypertonic. So the water inside the cytoplasm leaves for that hypertonic solution, trying to balance it out, and the cytoplasm shrivels up. And that's called plasmolysis. Okay, so long FRQ practice, super fun though. And maybe you have done this experiment on your own. I usually do this as a demo for my students. If you take a chicken egg, which is the largest single egg cell you're ever going to find, I guess you could get an ostrich egg, be the largest, but they're a little bit harder to come by. So you, a student conducted an experiment to determine the molar concentration of solutes in chicken eggs. The eggs were first soaked in vinegar to remove the shell. So the shells are just calcium chloride. The acetic acid in the vinegar just dissolves it. So what you're left with is an intact cell with an intact cell membrane, and now you can use it to watch how water moves across it. So these eggs were then placed in sucrose solutions of different concentrations, and the data are shown in this data table. So you're asked to do 3 different things. The first thing is to select the most appropriate data and construct a labeled graph on the axes provided, and I'll show you what that looks like to best illustrate water movement into and out of the egg. After you've constructed the labeled graph, explain why there is a difference in the percent change of mass in the eggs in the different sucrose solutions. In part B, you're asked to determine the molar concentration of that chicken egg, okay, using the data from the graph like I showed you, and then justify your answer. And then finally, this brings it around to why this water potential concept is so important in living things. Movement of water and nutrients are critical for the survival of plants. Using the concept of water potential, explain the role of positive and negative pressure in the transport of water and minerals. Okay, so this is what your graph grid would look like if you were taking this exam. So notice that you have a heavy dark 0 axis right across the middle, and that you're going to need to plot the points that show a increase in mass as well as a decrease in mass, and then you're going to use that graph to determine the molar concentration of solutes in the chicken egg. So for the graph, you should have a title. Your axes should be labeled. The X must be molar concentration, and Y may be change in mass or percent change in mass. You want to make sure that you have your appropriate data selected, that the axis and data are correctly scaled, and that the line graph correctly plots those points according to the data that you selected. You draw best fit line, and in part B, we're going to determine that concentration. So in part B asks you why the egg would gain mass, and it would gain mass if the solute concentration is lower outside of the egg. In other words, there's more water outside of the egg, water will move into the egg, causing it to increase in mass. And the opposite is true if the egg is in a hypertonic solution, then water will move out of the egg, so it would decrease in mass. And another way to say this is that water would move from high solute to low solute, or you could say high water concentration to low water concentration. All right, in part C, water moves from areas of high water potential to areas of low water potential, and water potential has those 2 components that are described in the formula of water potential. Now here's the important part. It has to do with transpiration, which is the loss of water from leaves to the atmosphere due to the lower water potential of the air. So there's more water in the plant, there's less water in the air. Water's going to want to leave the plant for the air. And then thinking about properties of water, which is a lesson we had previously, water is cohesive, right? It wants to stick together. So when that first water molecule floats off, evaporates off the surface of the leaf, then because of cohesion, it's going to hold that chain of water behind it. The pressure in the xylem is negative, okay? And finally, something called rotation has to do with root pressure, which is a positive pressure, gives a little push to get it into the xylem, and from there, that cohesion of water molecules and the adhesion of the water molecules to the xylem tissue is what brings that water all the way up to the surface of the leaf and out through the process of transpiration. Okay, so quick check for understanding. This is something you can do on your own. I've given you some data. Again, it's my class data, and it's asking you, using the mean class data, construct a graph to determine the molar concentration of the potato cores at 19.6 degrees C, and then determine the solute concentration of those potato cores. So you graph your data, draw your best fit line, right, making sure that you've got all your axes labeled correctly, and then find the isotonic point, the point at which the line crosses the x-axis. You can see it's super close to that 0.4 molar, and that would allow you to determine the concentration of sucrose. Let me go back, because then what you would need to do is plug in your values, and don't forget your temperature value, and then determine the water potential, which is just equal to the solute potential. Okay, and the answer is there down there at the bottom for you to see after you try this on your own. Okay, so our task for today was construct. By now you should be able to construct a graph, and this image is just showing a bunch of kinds of graphs. Today we practiced just a line graph. We did also look at some error bars to represent the error of the mean, but we also used a graph that we were able to use not just to display our data, but use it to determine that molar concentration of sucrose. All right, so our debrief. You should now be able to explain how concentration gradients affect the movement of molecules across membranes. You should be able to explain how osmoregulatory mechanisms contribute to the health and survival of organisms, and you should be able to construct a graph. Okay, your key takeaways: cells have membranes that allow them to establish and maintain internal environments that are different from their external environment. Also, osmoregulation maintains water balance and allows organisms to control their internal solute composition and water potential. Some common errors and misunderstandings, or another way to say ways to avoid osmosis and confusion. Okay, always start with osmosis, and remember that water always moves from a higher water potential to a lower water potential. Draw it, sketch the problem, and use it to determine where there's more water, where the higher water potential is, and then draw arrows to indicate the direction of water movement. Learn and understand the meaning of hypo, iso, and hypertonic solutions. And remember, you can only use these terms when you're comparing solutions. Remember that a hypertonic solution has a lower water potential, whereas a hypotonic solution has a higher water potential. Pure water has a water potential of 0, and water potential is negative for all solutions due to the solutes, right? So as soon as you add solute, it's going to make the water potential more negative. Okay. So if you stayed tuned in for the whole lesson, you would know that the correct answer is the one scientist that I never got to meet was Dr. Franklin. And my students know that she's one of my personal heroes, and sadly she passed away before I was born. And I wish that I would have had a chance to thank her for her work on behalf of all scientists, especially on behalf of women scientists. So here's your homework. All right. So what you have here is more practice with this idea of determining the molar concentration and then calculating the water potential. So I've given you a data table and a graph. You have a temperature there, and you're asked to determine the molar concentration of the cell and then calculate the water potential. Okay. So tomorrow this will be your warm up, and my colleague Ms. Pfizer will take you through that as your warm-up. One note from College Board, and that test exam day is getting closer. So if you need either mobile tools or connectivity, or you know someone who does, maybe one of your friends does, reach out directly to College Board and let us know. collegeboard.org/tech. And please do this by April 24th. Thanks for watching. I hope you join us again. I hope you join us for every lesson. Be sure to complete that practice question. It'll get reviewed in the next lesson, which is on enzymes. So have a great rest of your day, and I will see you again on Thursday.
Hi, welcome to AP Biology. My name is Gail Pfizer. I'm from Darnell Cookman Middle High School and Florida State College in Jacksonville, Florida. A lesson overview for today. Today we're going to be going over topics 3.1, 3.2, and 3.3. These have to do with enzymes, and they're located in Unit 3, which is cellular energetics. If you haven't done so already, grab your paper, pen, calculator, graph paper if you have it, and by the end of this lesson, you'll be able to explain these learning objectives. We're going to describe the properties of enzymes, explain how enzymes affect the rate of biological reactions, explain how changes to the structure of an enzyme may affect its function, and explain how the cellular environment affects enzyme activity. But first, let's have time for our warm-up. Okay, so our warm-up today is actually our homework from the last lesson with Osmosis with Ms. Kate, and we have a table with some sucrose molar concentrations and some percent change in mass. Some are gaining mass, some are losing mass, and then we have this nice graph right here, percent change in mass, and then sucrose molarity on the X. So what do we need to do? We need to determine the molar concentration of the cell, and then we need to calculate the water potential. Okay, so in my classroom, we go through this 2-day event of this osmosis lab with potatoes, and they have these beautiful graphs that look just like this for me. And then my question, I come to their lab station and I say, what is the molarity? What is the solute concentration of the potato? And they go, I don't know. And not everybody, but some. Anyway, so we talk about everything from this point up is gaining mass, and everything from this point is losing mass. So what's happening right here? And then they tell me nothing. And so we discover that if it's at equilibrium right there at that point, that is the molarity of the solution and the potato. Okay. So now that we've established that, I kind of led you to the first point of this FRQ, this very short FRQ. So now I need you to take that information, and I'm going to give you about 30 seconds to pull it together with finding the water potential of this solution, or this potato and the solution. Okay, so let's go ahead and we're just going to, let me get rid of some of this. So we're going to insert the numbers into this equation. So we have, there's our looks like a Titan, King Titan, what's her name? Ariel's dad. Looks like his pitchfork, but his, it's our side. And so we're going to put -1. And so why are we putting -1? Because we're dealing with sucrose. If we had salt, we have 2 ions involved, and that would be -2. So we're going to have -1, and then we're going to have our concentration, and then we're going to have our pressure constant, and running out of room, and then our temperature. So what's our temperature? If this is in Miss Kate's classroom, her classroom must be very cold, because mine runs by 22, and everybody's always freezing. So how are we going to get this? We're going to go 273 plus 18, which was 291. Okay, so now do the math, and let's see what we come up with. And I have, let's see if you do, I have a -9.67. Okay, so let's check against the scoring rubric. All right, so yes, here we go. But we have bars. So if we did this all out nicely and worked it out, we would have moles that would eliminate in liters, and then we would have the K, and we'd be left with bars as our unit. All right, okay, I hope everyone scored well on that. All right, so this is what you need to know. This is a very easy, this is going to be an easy day, and so we're just going to talk about enzymes, and we're going to do a couple of short FRQs and a couple of multiple choice. So let's just start right here. The structure of enzymes includes the active site that specifically interacts with substrate molecules. So here we have, this is our enzyme, and now we have that is our active site. And if you'll notice, it is shaped just like this substrate. And our substrate can be something that one molecule that we're going to use through dehydration synthesis, break down into 2 monomers or something like that, or we can have the 2 monomers and we're going to then make a polymer, that type of situation. But here's our enzyme, and there's where our substrates will still fit right into that active site. All right, for enzyme-mediated chemical reaction to occur, the shape and the charge of the substrate must be compatible with the active site of the enzyme. So not only does this need to be the right shape to fit, it also, the charge of this substrate and the charge of this enzyme must be compatible. Okay. All right, so now let's add activation energy into this whole thing. So we have, here's our substrate, here's our enzyme, and together they're going to make our enzyme substrate complex. So what is this? What's the purpose of this enzyme? It is to speed up this reaction. It's to be a catalyst of this reaction. So how does it do that? It does it by lowering the activation energy. So that's what this E little a is. So enzyme lowers the activation energy, speeds up the reaction, and now we have this would be our reactant, and this would be our product of that particular reaction. Now here would be the reactants, and this is the product, and we're breaking something apart, and now we're putting something together. Okay, so same thing. We're going to lower the activation energy, speed up that reaction. All right, so change to the molecular structure of a component in an enzymatic system may result in a change of the function or efficiency of the system. So I'm not really addressing denaturation yet. Okay, so if this active site is supposed to be the shape for this substrate to fit, if this was to have some conformational change, this substrate is no longer going to fit into this enzyme. Okay, so it's not going to function as it was originally designed to. All right, so now let's talk about, I want to focus on the activation energy that's in this graph. So if we were to, so this is the energy that we need to invest into this reaction. So this line right here, let's get a brighter color. All right, so this line right here would be the energy that we needed. So we're just coming to this point for if we had an enzyme. If we didn't have an enzyme, then we would need this much energy invested before we could get to the product. So here is the enzyme lowering the activation energy. All right, so here we are, denaturation. That seems to be something that kids remember from early biology. And denaturation of an enzyme occurs when the protein structure is disrupted, eliminating the ability to catalyze reaction. So here we have our enzyme, and this is our active site. Okay, it's a very elaborate active site. Our enzyme goes in there. Now, if we were to denature this enzyme, and we'll talk about ways on the next slide. If we were to denature this enzyme, this shape is then no longer there for this enzyme to work on this substrate. All right, so environmental temperatures and pH outside the optimal range for a given enzyme will cause changes to its structure, altering the efficiency with which it catalyzes reaction. So again, here's our enzyme, here's our substrate, so there's our substrate, and so now we have our enzyme substrate complex. Now, if we were to apply too much heat, or if we were to get, there's an optimal pH that enzymes work in. If we were to go too basic or too acidic, then we could denature this enzyme. Okay, so pH and too high temperatures. All right, so speaking of the pH, let's talk about the hydrogen ion concentration that is in pH. Let's start this way. All right, so let's look at our protein structures. Remember, we had 4 structures. We had primary, and then in our secondary, I think I had black dots on the last time we did this. All right, so here's our hydrogen bonds are between these amino acids, and it's giving it this form in this alpha helical shape. And then we also had them in our beta pleated sheet. Both of these are secondary hydrogen bonds make them have this particular shape, and there were hydrogen bonds that held our tertiary together. And if we disrupted the hydrogen ion concentration and made it too acidic, made it too basic, that's going to disrupt these hydrogen bonds that are holding it in this particular shape. So this would no longer be a functional enzyme if we disrupted the shape with pH. And speaking of pH, I know this looks crazy, but so I scribbled out this craziness on my board one day, and it was about how to understand what are you talking about when you're talking about pH. So when I did this with just in pen on the whiteboard one day, and then there was some lights went off in some people, or lights came on in some people, and they oh my gosh, I get it. So let's talk about some facts about pH. pH is based on 1 mole of hydrogen ions, and the pH measures how many hydrogen ions are in a solution. And pH is the negative logarithm of a hydrogen ion concentration. And these brackets right here, okay, that concentration is in there. Now, each 1 unit change in a pH scale is equal to a 10-fold change in hydrogen ion concentration. So let's take, for instance, a pH of 5. A pH of 5 is 10 times more acidic than a pH of 6, but a pH of 5 is 10 times less acidic than a pH of 4. I hope I said that right. All right, so what does this mean? In the pH of 1, there is 1.1 moles of hydrogen ions in the solution in 1 liter. And so this means that there's 1/10 of a mole. And then we have this 10 to the negative 1. In a pH of 2, we had 0.01 moles. So that was 100th of a mole of hydrogen ions is in a pH of 2 solution. And there's our negative 2. Okay, so I'm not going to go through all these, but you get the point of how this all of a sudden helps some students understand what this means. I hope it helps you. I had this a little bit different, and I had to give a shout out to our AP calculus teacher that keeps me in line and tells me, no, I just can't make up my own math. And so she made sure that everything was in the right order here. Thank you, Ms. Tony Hickson. All right, so the relative concentrations of substrates and products determine how efficiently an enzymatic reaction proceeds. Increases in concentrations of substrates and enzymes. So increases of substrates and enzymes can increase the rate of the enzymatic reaction. So we can increase the rate with these two things. All right, so however, as level of product increases, feedback inhibition can result. So if we're using an enzyme and we're making a product, then if we get enough product, there's no reason to continue to make that product. So we need to shut that enzyme down. And that is a negative feedback mechanism to maintain homeostasis that could happen. All right, so higher environmental temperatures increase the speed of movement of molecules in solution, increasing the frequency of collisions between enzymes and substrates, and therefore increasing the rate of reaction. So this is my attempt to show that at low temperature, we have low collisions because they're not moving as fast as they would be. The molecules are as if it was at a higher temperature. So more collisions can happen, so the rate of reaction can then increase. All right, so competitive inhibitor molecules. This is one of my favorite topics. Competitive inhibitor molecules can bind reversibly or irreversibly to the active site of the enzyme. So here is our active site of our enzyme that fits our substrate perfectly. Now, our substrate cannot get to the active site now because now there is another molecule with the same shape that is blocking this substrate. That's called competitive inhibition. It's stopping it. It's competing for the same spot. All right, non-competitive inhibitors can bind allosteric sites, changing the activity of the enzyme. So here we have, again, our substrate, our enzyme, but now we have an allosteric site. So if we have a molecule bind this allosteric site, if you notice, look at the active site normally, okay? When this binds, it changes the shape of our active site. And now our substrate can no longer fit into our active site because the allosteric substrate is an allosteric site. All right, this is some work from my classroom. These are models of competitive and non-competitive inhibition. And so you'll notice, and this little guy, here's his active site, and there's his substrate right here. And then she also, there's a allosteric site. And so when actually they work, because that's part of the requirement, they have to work. So when she puts this allosteric, the substrate for the allosteric site in, it actually clamps down on this little guy's mouth, and then the substrate can't fit into the active site anymore. And then we also do this toothpick lab that a lot of people do in their classroom. And so at one lab station, there'll be one person with a huge pile of toothpicks. Everybody, all groups have the same amount of toothpicks, but the group members are different in each group. So there'll be in your hands, the individual is the enzyme, and so your hand are the active site. So one person and 750 toothpicks to break will be much slower than 6 people with breaking apart 750 toothpicks. So we take the count every 30 seconds or so, and then we come up that the rate with more enzyme is much faster than with a less enzyme. Another lab that we do, this is investigation 13, and this has to do with ternate peroxidase. And what we do is students use ternate peroxidase to break down the hydrogen peroxide. So what happens is the oxygen, so when H2O breaks down, so when H2O2 breaks down, you're going to have an oxygen gas. It's going to be a product of that. So in the presence of oxygen, this indicator, glycol, which smells horrible, actually will bind to the oxygen, and when it does, it turns brown. So when it's brown, then we can tell there's oxygen there. And so the more brown, the more oxygen, which means the more peroxidase, that the more H2O2 that is being broken down. And then after we establish our baseline of what it looks like in a normal condition, then we start changing the pH. And this is a student's graph. You can see that nothing's happening at their pH of 3 and their pH of 10, but it looks like 5 took off and probably ended up to be the fastest rate of reaction that happened at that pH of 5. So that was optimal for ternate peroxidase. All right, and then I know this is a whole week of enzyme labs. We have so much fun with enzymes. So these are yeast balls, and we make them with sodium alginate. And so we put them in this little container. This is like a water bottle. That's that little tiny half water bottle size. Anyways, we put the yeast balls in some hydrogen peroxide as a substrate in there, and there's this little half pipette right here, and then there's this graduated cylinder. And what's going to happen is this yeast, the enzyme within the yeast, is going to break down hydrogen peroxide, and then oxygen gas bubbles will be formed, and they go up, and then there's this displacement of the water at the top of the graduated cylinder. And then measurements are taken every 30 seconds or every minute, and then we see how fast this is working in this condition. You can make the water colder, warmer, change up the amount of substrate, change up the amount of yeast balls that you have in the apparatus, is what I call it. Okay, all right, so that's all for content and for science. We're going to work on some science skills. So a lot of this is going to have to do with leading to, actually, we're going to be leading to experimental design. So 3CD, identify experimental procedures that are aligned to the question. And task words we're going to use today, we're going to see a lot of identify and determine and discuss. So first, let's do one together. And so we're going to identify, and how many? We're going to identify 2, and we're going to identify 2 what? 2 environmental factors that can change the rate of an enzyme-mediated reaction. And then we're going to discuss how that would affect them, as in, is it going to make the rate increase, or is it going to make the rate decrease? So I'll give you about 20 seconds to jot down some words really quickly to see what you think. All right, you have a couple? All right, let's look. So for identification, you could have said temperature, pH. You could have also said salinity. Inhibitors, stirring and mixing, but never came to mind personally. Pressure, you can change the pressure in classrooms by opening the door to the hallway. Oxygen, and like in discussion part of this, you should have said there's something missing on this scoring guideline. If this temperature increases, then this rate should increase. That little arrow is missing there. Okay, so temperature increases, rate increases. That's one discussion point. Temperature decreases, rate decreases. High temperature causes denaturation. Other factors that you could talk about improper folding, change of the active sites, and alter kinetics. That have to do with the temperature altering the rate of collisions. You could have also talked about the pH and salinity and active site changes related to all of these. Okay, all right. So now let's graph. We're going to do 2 things. We're going to graph, and then we're going to determine an optimal temperature for respiration of the yeast. So here we have a table. Here's our temperatures in Celsius 0 through 70, and our number of bubbles of gas produced per minute. And let's see what this says up here. Yeast cells are placed in an apparatus with a solution of sugar, a major nutrient for yeast metabolism. The apparatus detects bubbles of gas released by yeast cells. The rate of respiration varies with the surrounding temperatures, as indicated by the data below. So I'm going to take this to the next page, and I need you to grab whatever you're graphing on, and we're going to kind of have a parallel graphing together moment. I don't want you to copy me, look at me. I just want you to on yours, and then we'll compare both in the end. Okay, I have all this data on the next page as well. All right, so I'm going to get going. First, let me. All right, so. It's pouring rain in my house right now. Okay, I'm finished. I could leave it this way. I could do a couple of different ways. Also, if you could use data points, you can use a line or you can use bars on this particular graph. Now, since we're dealing with enzymes and you often think about enzymatic rates, then I might have drawn a line for this graph, but I know I would have probably just done data points. So let's look at these scoring guidelines. See, that's exactly what it's supposed to look like. So now we don't need a title, but let me say this. You must label your y-axis completely. Okay, it must say what it's supposed to say. And if you title a graph, then if you left something out of here and it was up here, then that would be okay. And then we have our x-axis, we have it properly labeled. We had our scaling. Did you notice me scaling? I knew the graph as I practiced earlier to make sure I could do this with you. And so I already knew that I couldn't skip lines or anything like that. All right, so correctly plotted points. Points may or may not be connected with a line. And a bar graph would have been acceptable. And our optimal temperature was at 30. Now I know that you're not going to be doing a graph on your test this year, but you're going to probably be analyzing one. So if you know how one was built within a topic, then that's going to help you be able to analyze it in that same topic. And then possibly, in a few minutes, we're going to talk about rate, and then we'll talk about possibly rate on the exam. Okay, so let's look at these multiple choice. We have a hydrogen peroxide solution. We have 1%, 5, 10, 15. Looks like we're increasing our hydrogen peroxide solution. Average time in seconds for distillate, 30 seconds, 25, 20, and 10. So looks to me like the higher solution, the faster flow. So let's read. To study the actions of the enzyme catalase on hydrogen peroxide, students performed the following experiment. Catalase was extracted from potatoes by blending raw potatoes in a blender with cold distilled water. The filtrate was stored on ice. The following hydrogen peroxide solutions were made: 1%, 5%, 10%, and 15%. Filter paper discs were soaked in the catalase filtrate and dropped into the beakers containing the various solution. The activity of the enzyme was measured by the amount of time it took for the disc to float to the surface of the solution of the bubbles produced by the reaction. The following data were obtained. So let's talk about what happened. So here's our beaker, and we have some discs soaked in hydrogen peroxide. And when that hydrogen peroxide is then be broken down by that catalase, then there's going to be oxygen gas bubbles. They're going to be underneath these discs, and it's going to lead these discs up to the top of the beaker in the catalase, right? Okay, that's what happened. So let's read the question. If the potato was boiled for 10 minutes and cooked for 10 minutes before and cooled for 10 minutes before tested, the average time for the disc to float to the surface of the hydrogen peroxide solution would be, hydrogen peroxide solution, they're soaked in catalase when they go back down. If I got that backwards just a second. All right, so less than 1 second, 5 seconds, 10 seconds, 30 seconds, or more than 30 seconds. Okay, this was a clue. They were boiled. So what do you think it's going to take? I hope you said E. It's going to take more than 30 seconds because we denatured those enzymes. All right, next question. We have a graph. We have a, excuse me, we have 3 lines. We have a pepsin line, a chymotrypsin, and a trypsin. And so pepsin is in solid, chymotrypsin is in dashed, and bold is the trypsin line. Okay, protein digestion in humans is primarily carried out by 3 enzymes. Pepsin is found in the stomach, which is pH of 2, where it aids in the breakdown of large proteins into smaller peptides. Where trypsin and chymotrypsin are found in the small intestine at pH of 8, excuse me, where they aid in further breakdown of the proteins into amino acids and dipeptides that can be absorbed into the bloodstream. Graph 1 shows the effective pH on the activity level of the 3 enzymes. All right, so what's our question? Which of the following best predicts how the structure and function of pepsin will change as it enters the small intestine? So we're going to go from a 2 to an 8. All right, so pepsin will not change in shape. No. Pepsin will not change. I don't even need to read the rest of it. Pepsin will not change in shape again. Pepsin will change in shape because of basic environment of the small intestine. Therefore, its enzymatic activity will decrease. Okay. Pepsin will change in shape because of the presence of trypsin and chymotrypsin in the small intestine, both of which act as competitive inhibitors. Are they acting as competitive inhibitors? No. So our answer is C. All right, I'm going to stay out of this one as much as possible. All right, the enzyme peroxidase is found in many organisms. It catalyzes the breakdown of hydrogen peroxide into water and oxygen gas. The rate of peroxidase activity at different pH values was assessed by students in the lab. The students' result was shown in the graph. So here we have relative peroxidase activity. We have pHs of 3, 5, 7, 9. And the question is asking you, if the experiment is repeated at pH of 11, the observed activity level of the enzyme will most likely be A, the same as the level at pH 7, the lower than the level at pH 9, greater than the level at pH 9, or between the levels of 5 and 7. All right, I'm not going to answer that one. I'll let you answer that one. And I'll give you 5 more seconds to think about it. Okay, and I hope you said what? B, lower than the level at pH 9. Okay, all right, so here we're going to have some guided practice. We're going to graph again, and we're going to, because that rate is what this is all about, is an enzyme. All right, so we're going to graph the data, and then we're going to calculate the rate. This is something that can easily be a graph that's on your exam. I know nothing about your exam, but I do think about what's something easy, that easy calculation to fit into, and rate is one of those. So this is worth your time. An experiment was conducted to measure the reaction rate of the human salivary enzyme alpha amylase. 10 milliliters of concentrated starch solution and 1 milliliter of alpha amylase solution placed in a test tube. The test tube was inverted several times to mix the solution and then incubated at 25 degrees Celsius. The amount of product maltose was present was measured every 10 minutes for an hour. The result was given in the table below. So now we're going to graph something, but it's right there. We're dealing with time. So I think you know what kind of graph to make with this, and it's going to be an enzymatic rate. So we're going to graph, and then we're going to calculate the rate. We'll look at the rate period between 0 and 30 minutes. So let's go graph this. We can graph it together. Here is our time, and there's our micromolar concentrations of our maltose. Okay. Just has 12. 0, 5.1, 10, 8, 9, 3, 4, 11, 5. All right. Okay, does your graph look something like that? All right, so now let's do the rate. It said between 0 and 30 minutes, right? So if we go 10.4 micromoles over 30 minutes, how much do we have per minute? We go ahead and do that calculation. And we have 0.35 micromoles per minute. Now, this would be an easy skill to be tested on. You can still do a calculation. You can still be involved in a graph. So I'm going to make sure my students are up on their rates and what's going on with enzyme lines. I think it's very important. All right, then this is what it looked like. Our graph did look like that. We had correct orientation of independent was the time, dependent was the maltose, and we have a units and intervals. We scaled it. I'm an obsessive scaler, and correct graphing of all data points, and then our correct setup. And there we have our calculation fell in a range that was acceptable for the rate calculation. All right, so explain why change in the reaction rate was observed after 30 minutes. I'll give you about 15 seconds to think about that and jot something down. Okay, so did you say that the reaction rate slows and levels off, and then why? So because it said explain, so that was how or why. So the rate slows as the substrate concentration declines. If you're using up all the substrate, then there's nothing else to break down or put together. Now, enzyme inactive by about 40 minutes. If your enzyme lost its activity over time, became labile enzyme, or if we had product inhibition come into play with this, that can also be an explanation of why it slowed down. All right, debrief and summary. Today's takeaways: enzymes decrease activation energy, increases in substrate. Enzymes and temperature can increase the rate of reaction, but that temperature and pH, if it's the wrong temperature, if it increases too much and the pH is not in optimal range, then denaturation can occur. You should also be able to describe the properties of enzymes, explain how enzymes affect the rate of biological reactions, explain how changes to the structure of an enzyme may affect its function, explain how the cellular environment affects enzymatic activity. And I always want you to be able to describe it, explain it, and tell me what you would do without it. And now I want you to tell me what would happen if you changed it. So some common errors and misunderstanding, and this will become more clear when we talk about when we get to photosynthesis and cellular respiration. One enzyme is not responsible for the end product in a metabolic pathway. The product of one reaction is going to become the reactant of the next reaction. And then I know that sometimes allosteric inhibition it's always interference, but it's not always a negative thing that's happening. Negative feedback has to happen. It's necessary part of homeostasis. Our bodies can't continue to make a product that we don't need and waste that kind of energy. And then let's see what else. Oh, I know what else I'm going to add. It was about temperature. I forgot to. So cold does not denature. Okay, that's why we put it in a refrigerator to slow the enzymes down, so they're not working before we need them to work in lab. It's also why you put things in a refrigerator. One of the reasons. Okay, so your homework tonight. There's two graphs involved in this. The effects of pH and temperature were studied for an enzyme catalyzed reaction. The following results were obtained. So we have enzyme activity and temperature. Looks like we have an optimal temperature, and enzyme activity and optimal pH. So what you're going to do is you're going to describe a controlled experiment that could have produced the data shown for either temperature or pH, and be sure to state the hypothesis that was tested here. But I'm going to change this up a little bit. There's not going to be one right, exactly the same answer. Okay, so this is what I just want you to have some experience or some practice with some experimental design. This could be something else that could happen. A graph would give to you, and then you're all of a sudden putting all the pieces together. That would be a great effort here. All right, so hence, what are you measuring? Make sure you cover this. What are you measuring and how? You'll need an independent variable, a dependent variable, you need a control. Go ahead and put together a null, okay, and then also your alternative. So have all those pieces together and of what a good experimental design to get to this would look like, okay, and that will be. I'll talk about this second. All right, device and internet access. Do this by April 24th. We know this is for college board. We know that not all students have access to the internet or a device. We're working on solutions to help students get what they need to show their best work. If you need mobile tools or connectivity or know someone who does, you can reach us directly to let us know at cb.org/tech. All right, one more slide. And thanks for watching. And I hope you'll join us again. We'll be sure to complete your homework question for tomorrow. And then Ms. Tay will go over that first and her lesson tomorrow or after this lesson. If you're not watching them in day by day regimen, then you're going to. She's going to talk about photosynthesis. So have a good day and come back and see us. Bye.
Hey, welcome to AP Biology. I'm Allison Kate, and today's lesson is going to be on photosynthesis. We're also going to be looking at Science Practice 3B, which is practicing stating the null and or alternative hypotheses, also being able to predict the results of an experiment. And we're going to apply that to Biology Lab Investigation 5 on photosynthesis. So don't forget your pen and paper, and let's go ahead and get started. Some things that you should be familiar with before we get rolling today are chloroplast structure, and hopefully are tuned in to our lesson on cell structure and function. If not, I'll review a little bit about chloroplast structure. You should also be familiar with the floating leaf disc assay. This is the experiment that we're going to do in Lab Investigation 5. And the terms photosystem, pigment, electron transport chain, and proton gradient. So today, a lot of this material is dense, and I'm going to do my best to take you through it. By the end of today, then you'll be able to describe the photosynthetic processes that allow organisms to capture and store energy. You should also be able to explain how to capture energy from light and transfer or pH of the reaction enough. So if you're doing temperature, maybe you started out at 10 degrees C and you went all the way up to 50 degrees C. We know that for most enzymes, a temperature that is optimum would be body temperature, so around 37 degrees. So that could describe the results we saw. We saw a peak in the middle with low at the beginning. So if that was a low temperature, there wasn't enough kinetic energy to get that reaction going. If it was at the high end, you could have denatured the enzyme with that high temperature, breaking the hydrogen bonds. And same thing with pH. You would want to have a range of pH. Enzymes do have an optimum range pH, although there are some that work best at low pH, like the enzymes in your stomach, or at higher pH. So you need to make sure you have a sufficient range. You need to make sure you held the other experimental factors constant. So for example, if you're focusing in on a temperature variable, then you would hold the pH constant. If you're focusing in on a pH variable, you would want to hold the temperature constant. So that you need to have all of those things so that you're only focusing on your variable that you're testing. And then you need a control, right? The control group is what you compare your experimental results to. And for this particular experimental design, you would either have no enzyme or inactive or denatured enzyme, or you would have no substrate. You need to, in your design, make sure that you have some way of verifying your results, that you have repeated trials, or that the results you get represent your average or mean data. And then finally, that hypothesis must be clearly stated. You can use an if-then format. You can make a prediction. You can say something like the enzyme activity will be greatest at 37 degrees Celsius, or enzyme activity will be greatest at pH 7. Okay, so that's the scoring guidelines, and I hope you guys did well on that one. All right, so today's lesson about photosynthesis, here's what you need to know. And I'm actually going to give you a lot of details about photosynthesis, then we're going to apply it to the lab, and then we are actually going to practice with our cues. So first of all, overview. Organisms capture and store energy for use biological processes. The statement from the CED, that's the course and exam description, is that photosynthesis captures energy from the sun and produces sugars. And so what you are looking at is an overall image, and it is situated in the chloroplast. So we'll get into the details of chloroplast structure in a minute. The top blue, water, and gray carbon dioxide represent the reactants of photosynthesis, and at the bottom, that red oxygen molecule and the green square that says sucrose represents the chemical energy that's produced. You can also see that there are two different types of reactions going on in the chloroplast, and they take place in different places, but they are connected, and that light is involved. Okay, so we're going to go through this in detail in the next several slides. All right, but before we get into the details of the process of photosynthesis, we want to get an evolutionary lens on this process. And the first thing you should understand is that photosynthesis first evolved in prokaryotic organisms. And there's lots of scientific evidence to support the claim that prokaryotic or cyanobacterial photosynthesis was responsible for the production of an oxygenated atmosphere. So we owe the fact that as aerobic organisms, the atmosphere has enough oxygen for us to breathe. So I have an image of cyanobacteria. It's on the left-hand side, and these are the organisms that built up oxygen in Earth's atmosphere over many millions of years. The second thing is that prokaryotic photosynthetic pathways are the foundation of eukaryotic photosynthesis. So eukaryotic photosynthetic pathways evolved from these early prokaryotic photosynthetic pathways. And you can see in the image on the right, we have purple sulfur bacteria, and they have a prokaryotic photosynthetic pathway that uses a different pigment to capture energy. And you may have heard of this. It's called bacteriorhodopsin, as opposed to the chlorophyll pigments that we see in eukaryotic photosynthesis. So a revisit to the chloroplast. They are the site of photosynthesis. It is where solar energy is converted into chemical energy. And just remember that there's several membrane systems that create compartments. So this concept of compartmentalization, which we've talked about in previous lessons, is really important as we talk about this process. So there's an outer membrane and an inner membrane, and in the chloroplast, they're both smooth. There is a space between them called the intramembrane space. Then we have a third membrane system, and it is called a thylakoid membrane. And they are folded membranes. That name actually comes from a stack of coins, and they're stacked up into what is called a grana. And in this image, you can see that there are 4 grana. So there's lots and lots of thylakoid membranes that are folded up. Outside the grana is a dense solution called the stroma. And I've indicated here that this is the site of the light-independent reactions. I may date myself. I learned these as the light and dark reactions. So the light-independent reactions, formerly known as the dark reactions, can take place in the presence or absence of light. The thylakoid membranes is where the light reactions or light-dependent reactions take place. So we have some compartmentalization, so separation of these two parts of the photosynthetic process. So just keep that in mind, that these compartments are really important. And as I talk a little bit more about the different specific steps of this process, you can think about where they take place. Okay, so photo, if you just deconstruct this word, photo, light synthesis means to make. Photosynthesis is actually composed of light reactions, which again take place in the thylakoid membranes, and the Calvin cycle, which is where we actually convert carbon dioxide into organic molecules. So we have photo and synthesis. And again, you're seeing that same image that shows those two reactants. To build one 6-carbon sugar molecule, it is 6 carbon dioxides that are taken from the atmosphere and 6 water molecules that are taken from the environment, along with light energy and a way to capture it. We are able to produce a 6-carbon organic molecule and release those 6 oxygen molecules molecules, and those are the oxygen molecules that go out to the atmosphere and make it possible for aerobic organisms to do cellular respiration. And tomorrow's lesson is on cellular respiration, and so you're going to see how these two energy processes are related. All right, so before we get going, I want to talk a little bit more about light. So we need to talk about the solar energy part of this. Light is just energy, and it's part of the larger electromagnetic spectrum of radiation. Shorter wavelengths, which are represented by that letter lambda, have higher energy, whereas the longer wavelengths have lower energy. Visible light is the light that is able to be detected by the human eye. And as you look at that whole electromagnetic spectrum, you can see way down on the short high energy end we have gamma rays, and then down on the longer wavelength lower energy we have radio waves. Visible light is just a very small part of the entire electromagnetic spectrum. And the visible spectrum you guys may know because you've seen rainbows or you have used prisms, so you can see the different colors of light. The shortest wavelength of visible light is around 380 nanometers, and it's down in the violet range, and then the longest wavelengths is around 750 nanometers, and those are in the red range. And you may have remembered or learned that the colors in this visible light are Roy G. Biv. That's working backwards from long to short. So you have red, orange, yellow, green, blue, indigo, and violet. So those are the colors that are visible to the human eye. Now, when light strikes a chloroplast, three things can happen, right? It can be reflected, which because the pigment is green, it will reflect that green light, and that's why plants appear green to us. Light can be transmitted, which means it just passes straight through, or it can be absorbed. And the absorption of that light is what we need in photosynthesis. So we have special molecules that will absorb that light. By the way, you have molecules in your eyes, in your retina, that actually also absorb light that allows you to see. Same idea here. So let's talk about the photosynthetic pigments. They absorb different wavelengths of light, and the reason that we have multiple pigments is to expand that visible spectrum of light that plants are able to use to drive photosynthesis. So what you're looking at in that image is the absorption spectrum of three different photosynthetic pigments. We have chlorophylls, and there's two kinds. Chlorophyll A, which in that image is the blue-green line, and you can see it has two peaks. It will absorb maximally at about 440 nanometers, and again at 680 nanometers. And chlorophyll B is that green-yellow line, kind of an olive color versus the blue-green, and it absorbs at 470 nanometers, and again at 700 nanometers. Notice, however, that where the two green lines are lowest is in the range of green light, and that tells you that they reflect green light. And then we have a third set of pigments, which are collectively known as the carotenoids. They are actually beta-carotene, which is what gives carrots their orange color, and then xanthophylls, which are yellow pigments. And you can see they have some peaks down in the violet-blue range, and notice that they drop off after that. They provide, again, an extension of the amount of light that can be absorbed, but they also are involved in photoprotection. And it's kind of like if you go out in the sun and you want protection from solar radiation so you don't get a sunburn, you put on sunscreen. And so you can think of the carotenoids as kind of a sunscreen of the photosynthetic pigments. Okay, so now we're going to get into the tough part. So we're going to talk about how the light-dependent reactions of photosynthesis and eukaryotes actually work. And let me just tell you that the whole point of these reactions is to capture that solar radiation, that solar energy, and convert it into some forms that the plants can use. And the forms that we're going to talk about are ATP, which is the universal energy carrier in cells, and you're going to get lots more instruction about that in tomorrow's lesson, and then a molecule called NADPH. And I'm just going to leave it at that. It's got a really long name, but we just know it as NADPH, and the P is for photosynthesis. Tomorrow when you talk cell respiration, you're going to see a related molecule that does not have the P. And so the whole point of this is to produce the ATP and the NADPH that will be used in the Calvin cycle, which is the light-independent part of the photosynthetic reactions that will actually generate our chemical sugar energy. Okay, so let's start off with the image, and I'm going to kind of talk you through it. This is an image of a photosystem. A photosystem has a collection of pigment molecules. In this case, they're the chlorophyll A molecules that were illustrated here, and it's going to have a light harvesting complex and a reaction center, and then an electron transport chain. I want you to see that this is embedded in the thylakoid membrane, which is just a phospholipid bilayer that you should recognize. And I want you to notice that on top of this image, out here is the stroma, and then down here is the thylakoid space. And this directionality, this compartmentalization is important to understand like the how of this process. All right, so we have a photon of light. It's going to strike the thylakoid membrane, and then we have green pigments known as chlorophylls. Now, a little bit of chemistry here. Chlorophyll has at its core a magnesium atom. The magnesium atom is going to actually lose its electrons. So that energy is going to, when it eventually gets transferred, you can see these black arrows. They're transferring all that energy amongst the chlorophyll molecules to a special pair of chlorophyll molecules that will actually, they get so excited they lose their electrons. And those electrons get boosted up to a higher electron acceptor, and this is called the primary electron acceptor, and that's right here. And those electrons are grabbed by that primary electron acceptor, and they are going to be passed down the electron transport chain. Okay, so now let's look at these two photosystems that work together. And my students are always confused when I tell them that the first part of this light harvesting reaction occurs in photosystem 2, and the reason for that is that it was discovered second. Photosystem 1 actually evolved first, but it comes second in the process. So what you see is they're very similar, but they have different reactive chlorophyll A molecules. So in this first image, and I'm going to show you this in more detail in the next slide, but in this image, light is absorbed by chlorophyll, and it gets transferred to that primary electron acceptor, which receives it, and then it's going to pass those electrons down an electron transport chain, which is a series of proteins and molecules that are embedded in that thylakoid membrane. And out as it transfers those electrons, it's also going to pump protons into the thylakoid space. Okay, let's back up. Where do the protons come from? So water, remember, water is part of the reactant. The first part of this reaction, light splits that water molecule, and that is called photolysis. That's actually what produces the oxygen byproduct. So plants want to get rid of that, which is a good deal for other animals and other oxygen-breathing organisms, right? So that also, when we split water, we generate some hydrogen ions, and those hydrogen ions are going to be key in setting up a proton gradient between those two photosystems, and we're going to use that energy to make ATP. The second thing that happens is we're going to pass those electrons, but we're also going to add more energy to a second photosystem, which will pick up that energy and boost that up to a primary electron acceptor, etc. in photosystem 1. This time, rather than generating that proton gradient, these electrons are going to be transferred directly to some proteins in the thylakoid membrane, ultimately activating this enzyme, which is called NADP reductase. And that tells you what it's going to do. It's going to actually reduce NADP to NADPH, and that is how we get ATP and NADPH from the first part of these reactions. Those two molecules, the ATP and the NADPH, are going to be the energy that originated from solar energy that will be used to produce sugars in the next phase. I know that this is tough, so hang in there. So this is really important, and actually today's lesson, I think, is going to set the stage for tomorrow's lesson, because you're going to apply the same concept with a few different details, a few tweaks to the process of what happens in cellular respiration in a similar organelle, the mitochondria. Okay, so both the mitochondria and the chloroplast use this proton gradient to generate ATP. So again, orient yourself. You're looking at the thylakoid membrane. The thylakoid space is in the middle, and you've got the stroma surrounding the thylakoid membrane. So let's go ahead and take a look as I kind of hit the highlights here. So light is absorbed by photosystem 2. Light also splits water. The electrons that come from the water molecule are going to be used to replace the ones that are lost when the chlorophyll A loses its that are ultimately passed down the electron transport chain. The oxygen diffuses out and is a byproduct of the process. The protons are actually going to be stored in the thylakoid space. So now we're going to move those electrons down the electron transport chain, and as we do that, we move more protons into the thylakoid space. Then that proton gradient, so we've got way more protons inside the thylakoid space than outside of it in the stroma, stroma, they are going to be used to generate the energy that powers the production of ATP. So if you look here, there's a very important part of this membrane proteins, and this is a special enzyme called ATP synthase, and this is the site of ATP production. And the way that ATP is produced is that ADP, adenosine triphosphate, gains a phosphate to form ATP, or adenosine triphosphate. And now my students always ask me two things. First of all, what is inorganic phosphate, and where does it come from? So it is always shown as this P for the phosphate, and then I for inorganic, and it is just floating around in the cell. So it may be there from the reverse reaction of ATP being broken down back into ADP and phosphate. So it's there. And again, what is creating the ATP through the ATP synthase enzyme is that proton gradient. So the next thing that happens, or continuation, those electrons from photosystem 1 are used to transfer directly to the enzyme NADP reductase, and that causes the NADP to be reduced to NADPH. So now we're going to take those two energy producing or energy molecules, ATP and NADPH, to the Calvin cycle. Okay, so you can go back and look at these and kind of go slow because that's a lot of information. All right, so now we're finally at the place where we're going to finally make some organic molecule, we're going to make some sugar. And so the energy that was captured in those light reactions that I just went over, how it's transferred to ATP and NADPH, those two energy molecules are going to power the production of carbohydrates from carbon dioxide in a cycle known as the Calvin cycle, and this occurs in the stroma of the chloroplast. And it actually is going to produce a 3-carbon sugar called glyceraldehyde 3-phosphate, or GPP for short. So I know the overall balanced equation of photosynthesis tells us we're going to make glucose, but it actually takes two turns of this Calvin cycle to get you that 6-carbon sugar. It occurs in 3 phases. They are the carbon fixation phase, reduction phase, and regeneration of something called RUBP. So I'm going to take you through those phases. But first I have a shout out. This cycle is named after Melvin Calvin, who elucidated the steps of this cycle using an algae called chlorella, and his work was done at UC Berkeley. So go Bears. All right, so let's take a look at carbon fixation. What the heck does that mean? Carbon dioxide from the atmosphere is brought into the plant and into the Calvin cycle in the stroma, and it needs to be fixed or converted into organic molecules. And there's an enzyme that does it. Its name is called Ribisco. It stands for ribulose bisphosphate carboxylase, and that's a mouthful. I had trouble getting it out, so we just call it Ribisco. And it turns out that Ribisco is the most abundant enzyme in the plant. A full 15% of the plant's protein is this one enzyme. And what it does is that it grabs the CO2 from the atmosphere and combines it with a 5-carbon molecule from the cycle called RUBP. And there is a super short-lived intermediate, and then that breaks down into another chemical called 3-phosphoglycerate. And now we're going to take that energy from ATP and NADPH to reduce it. So we're going to add carbons to make that molecule larger, and we're going to do that until we create a glyceraldehyde 3-P phosphate G3P molecule. So carbon fixation is the first step. It uses the enzyme Ribisco, and it forms a organic molecule. That organic molecule is reduced using ATP and NADPH to form that 3-carbon G3P. And the final phase, since it's a cycle, is we have to regenerate that 5-carbon RUBP molecule so that it's ready to begin the cycle again. And again, it takes 2 cycles to create that 6-carbon carbohydrate. It's really important to note right here that this pathway first creates a 3-carbon sugar, and it's called the C3 pathway. There are variations on this pathway. One of them is called a C4 pathway, and you might know that it's going to start off producing a 4-carbon sugar. It uses a different enzyme. And then there's yet another variation called CAM pathway. Neither the C4 or CAM pathway are going to be discussed here. They're outside the scope of this topic 3.5. Okay? So just a review. During photosynthesis, chlorophylls absorb energy from light, boost electrons to a higher energy level in both photosystems 1 and 2. Photosystems 1 and 2 are embedded in the internal membranes of chloroplast and are connected by the transfer of higher energy electrons through the electron transport chain. When electrons are transferred between molecules in a sequence of reactions as they pass through the ETC, an electrochemical gradient of protons, or those hydrogen ions, is established across that internal membrane. The formation of the proton gradient is linked to the synthesis of ATP from ADP and inorganic phosphate via the enzyme ATP synthase. The energy captured in the light reactions and transferred to ATP and NADPH powers the production of carbohydrates from carbon dioxide in the Calvin cycle, which occurs in the stroma of the chloroplast. So that's your money slide. That is the review of those steps. Hopefully the lesson and taking you through some of those images helps you understand that summary. All right, so now I want to apply this to a lab, and you may have done this lab in your class, but this is the leaf disc assay. And just to note, you may see that word disc spelled differently, and you'll probably see it spelled differently in my presentation. I don't know the correct spelling. It's either DISC or DISK. So anyway, just don't get confused by that. So in this lab, the question we're asking is what vectors affect the rate of photosynthesis in living leaves. In this lab, you have 2 objectives. You want to be able to design and conduct an experiment to explore the effect of different environmental variables on the rate of cellular photosynthesis. And then you're going to connect and apply many concepts, including the relationship between cell structure and function, like we just saw in the chloroplast, and the idea of those membrane systems and compartmentalization. We're also going to try to connect the concepts of strategies for capture, storage, and use of free energy, and then also diffusion of gases across cell membranes. And the gases, of course, are carbon dioxide and oxygen, and the physical laws pertaining to the properties and behaviors of those 2 gases. So I'm starting you off with an image. I've got a cross-section of a leaf, and there are 2 things that are important about this. The first one is that you see that there is a palisade layer, and there's lots of air spaces around it. There's also air spaces in what's called the spongy mesophyll. Those air spaces are going to be important to the design of this experiment. Also, I want you to see that those palisade layer is packed with chloroplasts. Those are the chloroplasts where the photosynthesis reactions are going to take place. This experiment uses light. You can actually do this outside if you have bright sunlight, or you can use a lamp. And syringes. And the syringes are important because we need to start this experiment by removing all of the carbon dioxide and oxygen that exists in those leaf cells. Okay, so let's take a look at this next slide. I'm going to show you, before we get into the data table and the graph, I want to talk a little bit more about how you get this set up. So you can see there are syringes. You can do this at home if you wanted to play with this. If you have never done this before, it's super materials are super easy to get. You just need some leaves. In my class, we use spinach leaves, or you can go on a field trip and just pick leaves from your school or your backyard. And then you just need to have some way to make constant the size of the leaf disc that you're going to cut. So you can use a hole puncher, you can use a straw, and you just want to make sure that you have discs of same size. One other thing, you don't want to cut your leaf disc out of the veins, because that is the vascular tissue and there's no chloroplast. So you cut your disc out. We cut out 10 discs. We will put them in the syringe. The reason that that's important is we're going to fill that up with a solution of water and baking soda, just baking soda like you have to when you make your chocolate chip cookies, and that's going to provide by the carbon dioxide, the carbon source for photosynthesis to take place. And you are going to get rid of all those gases inside the leaf disc by putting your thumb over the top of the syringe and pulling on the plunger. And what happens, you'll feel that pressure and all that you'll see air bubbles, and all those air bubbles you want to get rid of. So you do that by pushing the fluid all the way to the top. If you do this properly, all of your leaf discs will sink. Then what you do is you take them out of your syringe. Some people leave them in the syringe, but you take them out, put them in a beaker of your baking soda solution, and then you turn the light on. And what we're going to do first is just practice this technique by looking at light intensity. So I just threw together some sample data, and so the way that we do light intensity is we measure 0, 5, 10, and 15. The 0 actually is no light, but 5, 10, and 15 centimeters away from the light. So the beaker is that far away from the light source. And what we actually measure is the time it takes for half of those leaf discs to float. And you're probably thinking, well, how do those leaf discs float? Once photosynthesis happens, right, the cells start taking in the carbon dioxide and they start producing oxygen. When they produce oxygen, it infiltrates those air spaces and it causes those leaf discs to become buoyant, and so they begin to float. And so it is a measure of the net photosynthetic rate. And so what you can see here is that the higher the closer the light is to the source, the higher the light intensity, the faster the time is. The faster, but the shorter the time compared to the 10 centimeters takes a little bit longer, and the 15 centimeters takes longest. The no light we have indicated here because photosynthesis will never happen without light. And so in the class we just let it go for 1500 seconds and then call it good if nothing has none of the discs have okay. Okay, so the next thing is to have you guys design your own experiment. And this is the experimental organizer that I use. I have my students decide a possible variable, create a title, make a prediction, indicate what the independent variable is, how many levels of independent variable you're going to have, how many trials you're going to do, how you're going to measure it, and what your experimental constants are. So if you wanted to brainstorm some variables, you can investigate temperature, wavelength of light, C4 versus C3 plants, the size of the leaf disc, the amount of carbon dioxide, sun plants versus shade plants, leaf color, leaf size. Okay, so you go ahead and pick your variable. And this is an example from my class. This is from a group that looked at light color. And the way they did that is using different color filters. So they looks like they looked at red light, yellow light, green light, blue light, and then just regular old white light, unfiltered light. And as you look at their graph, you can see that the one that went the fastest is the lower bar, and that is all wavelengths of light. The one that took the longest is the green light, and that is because green light reflects rather than absorbs light. So there's not enough energy to drive photosynthesis. So my students would show their work by creating a claim that the photosynthetic rate would be lowest when the light color is green and highest when the light color is red. Their evidence is their graph, and their reasoning connects their evidence to the claim. Okay, just one more, and then I'll move on. This is a group that did temperature, and you can see that there is no data for low temperature because there's not enough kinetic energy to actually drive the photosynthetic process. And then at high temperature, the rate is actually lower, and that's because we perhaps denatured our enzymes. So we can see that at 25 degrees Celsius, that had the maximum photosynthetic rate. All right, let's get some practice. So my first practice question is looking at kind of the evolutionary lens of photosynthesis, and I've got fossils of a microscopic organism. They're found in rocks that were determined to be over 3.5 billion years old, and the question asks you to identify two types of evidence that would help answer the question of whether the organism was photosynthetic. Okay, so how would you know looking at these old rocks? So you would know if there was some type of presence in the fossil of photosynthetic membranes or some type of imprinted chloroplast. Another way, probably a more realistic way, is that there would be high levels of oxygen gas or oxides. The rocks would be rusty in the surrounding rock. You might see evidence of photosynthetic pigments, or you might see a chemical analysis of the fossil showing presence of molecules that are associated with photosynthesis. Another way you could do this would do it backwards. If there was a lack of photosynthetic membrane chloroplast or a lack of that oxygen oxidation in the rock, that would suggest an anaerobic environment. All right, let's look at an actual photosynthesis one, not an evolutionary one. And this is a really good question. So there's a lot of information in the stem of the question, and I will read it to you, but I'd like you to read along with me. And then the data table is showing you the photosynthetic rates of 3 groups of shrubs that were grown under varying amounts of illumination. So this is like the light intensity. So we've got shrubs growing in full sun, shrubs growing in partial sun, and shrubs growing in the shade. And you can see all their data with different intensities. So the intensity is the independent variable. So researchers tested the effect of light on the rate of photosynthesis by using a shrub growing under conditions that differ widely in the amount of variable light, but where the availability of water and soil nutrients is fairly constant. So water availability and nutrient availability constant. Under constant temperature, relative humidity, leaf surface area, more constant. The researchers used increasing illumination, which is measured as the photosynthetic photon flux density, the number of photons of wavelengths between 400 and 700 nanometers per unit surface area and unit time, and determine the net photosynthetic rate. And that was measured by the amount of carbon dioxide fixed per unit surface area and unit time at each illumination. Okay, so that's a lot of information. So the first thing you're asked to do is to describe how plants absorb photons of light. So hopefully you could do that by saying that plants contain pigments such as chlorophylls that will absorb photons of light at certain wavelengths, and that's the visible light between that 400 and 700 nanometers. Next, you're going to be asked to construct an appropriately labeled graph to represent that data. And I got us started on the graph grid by indicating the independent variable, and the independent variable is the type of environment, right? So it's grown in full sun, partial sun, and shade. And then I've got my units here, the photosynthetic photon flux density, which is different in each of those conditions. And then my dependent variable was measured, which was how much carbon dioxide was fixed, and that's the net photosynthesis in micromoles per carbon dioxide per meter squared per second. And so the score guidelines would tell you that you would get full points if you had your correct axis labeling, as I just indicated, that you've got your correct scaling and units, and that you have your correctly plotted lines. And in this case, because our independent variable is categories, we're going to use a bar graph. And so if I graph that data, I'm going to get full sun, partial sun, and shade. And you can clearly see from this graph that the full sun has the highest amount of net photosynthesis, the amount of carbon dioxide that was converted into carbon. Okay, so if you look at that top line, this is going to become important in the next part of this question. Okay, so based on the data, identify the shrubs and data point where the greatest flow of electrons through the chloroplast electron transport chain most likely occurs. So we know it's going to be the shrubs in full sun, and that data point that I highlighted in red is at 1500 micromoles per meter squared per second. And finally, predict the effect of growing shrubs growing in one area if they're tested with a compound that blocks electron flow through the electron transport chains of chloroplast, and then provide reasoning to justify that prediction. So we know anything that's going to block the electron flow is going to prevent enough energy being produced to drive the production of sugar, so the plants are going to die. If they can't make their own food, they are not going to fare well. So your prediction is that the shrubs will die, they'll eventually die, they'll not be able to make sugars. And the justification for that is that blocking that electron transport chain is going to prevent the formation of proton gradient, which is needed to produce ATP, and that ATP is needed to produce sugars. So it's a really good practice question. So go back and watch this and take a look at it. So just a quick check for understanding. Remember, our skill was to state the null or alternative hypothesis and predict the results of the experiment. So let's take a look at that first photo leaf disc experiment where we looked at the variable of color or wavelength of light. So a null hypothesis might be the color of light will have no effect on the rate of photosynthesis. An alternative hypothesis might be that the photosynthetic rate will be lowest when the light color is green and highest when it's red. And to predict the results, the photosynthetic rate would be greatest in red. Since chlorophyll absorbs in that red range, it will be lowest in green because chlorophyll reflects green light and will lack the energy necessary to drive the reactions of photosynthesis. One more quick skill practice, this time using that variable of temperature. So the null hypothesis would be that temperature will have no effect on photosynthetic rate. The alternative hypothesis is that photosynthetic rate will be lowest when the temperature is lowest and highest when the temperature is highest. Now that may not be a correct hypothesis because we remember from that data that super high temperature actually denatured the photosynthetic enzymes, and so the rate actually was lower. And that is reflected in the prediction. So the photosynthetic rate will be lowest with lower temperature due to lack of kinetic energy. The rate will be highest at a temperature that is optimum for the enzymes used in photosynthesis. And that's because too high temperatures will stop the reaction by denaturing the enzymes, and the rate will actually fall to zero. All right. Really quickly, just the task verb justify. In this question, students were given a bunch of background information that you can read in that first bullet point, and then you're asked to provide reasoning to justify this claim. So scientists claim that a plant has more than one type of pigment, and it would have a greater fitness than a plant that has only one type of photosynthetic pigment. And you're asked to justify this claim. And the response that you would have to justify this is that if you have more pigments, you would be able to absorb a greater range of wavelengths, and that should increase the plant's ability to perform photosynthesis and then survive and reproduce. Okay, so that gets in that evolutionary piece. Okay, so to debrief, today you should be able to describe the photosynthetic processes that allow organisms to capture and store energy. You should be able to explain how cells capture energy from light and transfer it to biological molecules for storage and use. And the key takeaway today is that highly complex organization of living systems requires a constant input of energy and the exchange of macromolecules. So I'm going to skip the common errors and misunderstandings right now in the interest of time. This is a former section of a free response question that has to do with bacteriorhodopsin, which is that a type of pigment that's found in early photosynthetic systems. I'm going to share with you the scoring guideline, and then there are some common errors and misunderstandings that you can read through when you read this on your own. Because I'd like to move you forward and have you take a look at your homework for tonight. Okay, so go back and look at this and read it on your own. Okay, so So here's your homework tonight, AP exam practice. In a certain prairie community, a dominant prairie grass species has recently been infected with a virus that disrupts one of the electron transport proteins in the chloroplast of affected cells. Describe the most likely effects on cellular processes. Be specific as to which processes and molecules are most likely to be directly affected. So make sure you talk about the cellular processes and then call out the molecules that are going to be affected. Okay. One last word before I leave you today. This is about your device and internet access. We are approaching our exam day. So if you need connectivity or mobile tools, please reach out to College Board and let us know. And the way to do that is through cd.org/tech. Please do this by April 24th, which is next Friday. Thank you so much for watching. I know today was some tough teaching and learning. I hope you'll join us again. Be sure to practice your practice question. It'll get reviewed in the next lesson tomorrow, which is on cellular respiration. And I'll see you guys again next Tuesday. Have a great rest of your day.
Hi, welcome to AP Biology. I'm Gil Pfizer from Darnell Cookman Middle High School and Florida State College in Jacksonville, Florida. Lesson overview for today. We'll be reviewing topic 3.6, cellular respiration. This is in Unit 3. In fact, today finishes up Unit 3, and then the next lesson will be in Unit 4. We're halfway there. So cellular genetics is still Unit 3. Anyway, today we're going to describe the processes that allow organisms to use energy stored in biological macromolecules, and we're going to explain how cells obtain energy from biological macromolecules in order to power cellular function. Okay, so our warm-up today, which was our homework from our last lesson, which was photosynthesis with Ms. Kate. And if you are joining us for the first time, then I'm going to give a moment for you to jot down some thoughts in between after I read this question and before I reveal the scoring guideline. If you have your homework from last night, then go ahead and get that ready, and we'll compare in just a minute. All right, in a certain prairie community, a dominant prairie grass species has recently been infected with a virus that disrupts one of the electron transport proteins in the chloroplast of the infected cell. Describe the most likely effects on cellular processes. Be specific as to which processes and molecules are most likely to be directly affected. So we have our first task verb of the day, and we're going to be describing. So I'll give you just a moment. If you're just starting with us, let me get my timer on. Okay, all right. So let's check your answers against the scoring guideline. So some effects that you may have included. You might have said that there was less ATP produced, less NADPH produced, inability to fix carbon via via Calvin cycle without products of electron transport chain, and decrease the O2 production. All right, so let's get started with what we need to know about cellular respiration. I will go through this in detail, and I'm kind of going to change up for the day of my usual routine, and I'll let you know when that change is about to happen. All right, so cells obtain energy from biological macromolecules in order to power cellular function. So lipids, proteins, carbohydrates, all of those can be used in the metabolic pathways to make ATP. They just come in at different times, perhaps, or in different ways. But carbohydrates is usually what our first thought about when we're thinking about making ATP, and that's the example we'll be using today. All right, so glycolysis is a biochemical pathway that releases energy in glucose to form ATP from ADP and inorganic phosphate. It also forms NADH from NAD+ and pyruvate. So glycolysis is happening in the cytoplasm of the cell. Okay, it's not in the mitochondria, it's in the cytoplasm. So here is our 6-carbon glucose molecule, and we're going to, our products will be 2 ATP molecules and 2 pyruvate molecules, and then 2 NADH. So how did we get that NADH? We had NAD+ come into the reaction, and then combined with a proton and 2 electrons, we're going to make NADH. And that NADH is going to carry the electrons to the electron transport chain and further down the line in this process. Okay, all right, so pyruvate is transported from the cytosol to the mitochondrion where further oxidation occurs. So these are 2 molecules of pyruvate that we just made in glycolysis. Now they can't get into the mitochondria without being actively transported in, but they are going to be, the reaction is going to occur, and the product's going to be 2 CO2s are going to be released. Again, we're making 2 NADH, and now we have acetyl-CoA that can go into the Krebs cycle. And here again, we have 2 NADHs. We have NAD+ plus the H, those 2 electrons, and NADH. And again, along with the NADH from glycolysis, the NADH from pyruvate oxidation they're going to go to the electron transport chain to drop off those electrons. All right, so now we're up to the Krebs cycle. Carbon dioxide is released from organic intermediates, and ATP is synthesized from ADP and inorganic phosphate, and electrons are transferred to the coenzymes NADH and FADH2. So if you'll take a look, you can see that here is our 2-carbon acetyl-CoA molecule, and they're going to combine with a 4-carbon oxaloacetate, and that's going to form the 6-carbon citrate. That's where the citric acid cycle name comes from. So in the cycle, we're going to produce 2 here and 2 here, so 4 molecules of carbon dioxide, and then we're also going to put electrons on NAD+. There's 2 NADHs, 2 more and 2 more. So there's our 6. So there's those electrons ready to go to electron transport chain, and then we're also going to use FADH2, okay, to carry electrons. It was built the same way. You had FAD and 2 hydrogen, and the 2 electrons are added. So now we have electrons on FADH2 and on NADH. Both of those have gained electrons, so both of those have been reduced. Okay, and then we can't forget our 2 molecules of ATP that are made also in the citric acid cycle. All right, electrons extracted in glycolysis and Krebs cycle reactions are transferred by NADH and FADH2 to the electron transport chain in the inner mitochondrial membrane. So here we're going. We have our glycolysis happen, okay, and we have these 2 NADH. We have our 2 NADH from our pyruvate oxidation, and then we have our NADH and our FADH2 from our Krebs cycle. And all of those are going to go to the electron transport chain. I know I have it very tiny right now. I'm about to make it larger so you can see what's going on. And so now that's on the inner membrane of the mitochondria. So that's where the reaction is going to occur. Excuse me. When electrons are transferred, here's a larger version so you don't have to try to figure out what's going on in that little one. All right, when electrons are transferred between molecules in a sequence of reactions as they pass through the electron transport chain, an electrochemical gradient of protons or hydrogen ions across the inner mitochondrial membrane is established. So here we have NADH. So where are we at right now? We're on the inner membrane of the mitochondria. Okay, and this shaded area, this is why I've been obsessing over this diagram and the chloroplast one for the last 2 lessons, is because now we're going to use it. All right, so now we have NADH is going to, let me get the color I can see. All right, it's going to lose those 2 electrons, so it will be oxidized. Okay, and then we have our NAD+ that can now go back to work in glycolysis or Krebs cycle. And then we also have all these protein complexes, and we don't need to know names or anything like that. This is more about what's happening, what the process is about, and what the process is for. All right, so we have these 2 electrons that are going to be carried down the chain, and we'll talk about their future in just a little bit. But what's going on right now is this is going to go, there's hydrogen ions now, and they're going to go against the gradient to the other side. Okay, so this hydrogen ion concentration gradient is being built in this intramembrane space. Okay, so remember that was compartmentalized for an enzymatic reaction to occur. So that's how they're getting there. The NADH is coming in, and it's leaving as NAD+. Hydrogen ions are getting pushed across the gradient, and the electrons are now on the membrane chain. Okay, all right, so when electrons are transferred between molecules in a sequence of reactions as they pass through the electron transport chain, an electrochemical gradient of protons, hydrogen ion, across the inner mitochondrial membrane is established. So let's take a look at this. There's more to this than just repeating what I just said. All right, so here's our inner membrane, and there is our reaction. Our hydrogen ions are building up within the intramembrane space, and they're doing this also over here. So why all of this? Why all of these loops and convolutions? Because we need what? We need increased surface area, because it's not just going on one place or 2 places here. This is all over this inner membrane. So this entire area that has been compartmentalized is now building up hydrogen ions. Okay. Okay, all right. Now, cellular respiration in eukaryotes involves a series of coordinated enzyme catalyzed reactions that capture energy from biological macromolecules. So now we can put it all together. We had glycolysis, and then we had pyruvate oxidation, then we had the Krebs cycle, and then we have our electron transport chain. So now we can finally get to the point of why we're establishing that hydrogen ion concentration gradient. Okay, the electron transport chain transfers energy from electrons in a series of coupled reactions that establish an electrochemical gradient across the membrane. So let's talk about the word coupled. So we have this redox going on. So in case you need brushing up on your chemistry, so when you are reduced, okay, you're gaining electrons. When you're oxidized, you are losing electrons. So let's look at what's happening to NADH. Those electrons are getting off, right? So your NADH is being oxidized. And then look at this other end. There's many other reactions that go along in the middle here. But look at our oxygen is our final electron. You can't see that. Our oxygen is our final electron acceptor in cellular respiration. So it'll be gaining electrons. So that would be reduction. So redox reactions are coupled. There's some gaining electrons, there's some losing electrons. All right. Now, electron transport chain reactions occur in chloroplast, mitochondria, and prokaryotic plasma membranes. So if you'll take a look at this, here we are. This is this gray area. That's the gray area here. So this same electron transport chain is here on this inner membrane. And then if we look at the chloroplast, we have an electron. See this gray area? That's inside of the chloroplast, in the inside of the thylakoid, in the thylakoid lumen or the thylakoid space, whichever one you're calling it. So there's inside that's where the hydrogen ion concentration gradient is forming. And then look in this bacteria. In bacteria, not a one size fits all of how things work. So let's just look at this model. And we have a cell membrane in orange. I just put it, and we have a cell wall in pink. And then this hydrogen ion concentration gradient will develop in between those two. And the same thing, that gray area would be with where the purple is. All right. So in cellular respiration, now we can get to why we're building up this hydrogen ion concentration. So in cellular respiration, electrons delivered by NADH and FADH2 are passed to a series of electron acceptors as they move toward the terminal electron acceptor, oxygen. So let's follow their path. So here was our NADH, and those two electrons NADH lost its two electrons. They're on the electron transport chain, and now NAD plus can go back and pick up more electrons and continue working. And the same thing will happen with FADH2, and then FAD can go do the same and pick up some electrons at the Krebs cycle. And in the process, on each one, we have hydrogen ions. They're going to be pushed against the gradient, and they're going to accumulate in this intermembrane space. Now, the electrons will eventually get off of this chain, and then they're going to bind with oxygen and then to hydrogen. And so their final electron acceptor is oxygen. Remember that later for an answer in the FRQ. Okay, so together, then that will form water. Now, why are we forming this hydrogen ion concentration gradient? What purpose is all of this that's happening in all of these organisms? That's because this amazing, let me get what color do I want to write in? Okay, this amazing enzyme called ATP synthase is in all of these organisms that have these hydrogen ion concentration gradients. So the hydrogen ions, and this is, you need to look on the video that you can see how this works, because they kind of go in and they go through this little elaborate tunnel as they go from high to low. So they'll be moving this way, and the energy created from the concentration gradient is then the bottom part of the enzyme, it turns the bottom part of the enzyme, and as it turns it, it opens up the active sites for ADP and P, and then ATP is formed with that reaction that will happen on those active sites. So it's pretty amazing. Now, we'll get to bigger diagrams, but in photosynthesis, the terminal electron acceptor is NADP plus. It's right here, okay? And notice that NADP is in photosynthesis, it starts with a P, and then NAD, no P, is in the mitochondria. And then other molecules are used in bacteria, and there's a variety, but we still have basically the same. There's some protein complexes, and there's establishment of hydrogen ion, and then there's the ATP synthase also. So I'll talk about photosynthesis. Let's do it on the next page, it's bigger. All right, the transfer of electrons is accompanied by the formation of a proton gradient across the inner mitochondrial membrane or the internal membrane of chloroplast, with the membrane separating a region of high proton concentration from a region of low proton concentration. So let's talk about what happens in photosynthesis. I'll be a review from yesterday, and then you can see the similarity of how this is. It's almost the same thing. All right, so we have sunlight, there's sunlight, there's sunlight. Let me give you your, let me orient you to everything. Here is photosystem 2, photosystem 1, ATP synthase. There's an NAD reductase, NADP reductase right here. You can barely see them. There are some mobile electron carriers that move in between these complexes. And so let's get started. So some photon of light excites the chlorophyll pigment that's in this elaborate photosystem 2, and there's going to be some excitation, and those outer electrons are going to be resonance, and that will make their neighbors respond, and they have electrons that get excited. So everyone gets excited, and then there'll be two electrons that will leave the chlorophyll molecules and go to the electron transport chain. Well, in the meantime, there is a part of this elaborate complex, is this enzymatic site, where water will be split in the process when the sun hits this complex. So water is split, and we have our two hydrogen ions, and then we have two electrons, and then half of the oxygen gas. So and it will bond with another oxygen gas and then leave. So and diffuse out through the membrane. Anyway, so when this carrier, this purple that you can really see right here, when it left this photosystem with these two electrons, it also picked up two hydrogen from the stroma and carried them to the next complex as well. And at the same time, it moved those two hydrogen. There were two more hydrogen that were moved through the protein complex to end up with four entering the intermembrane space. I'm sorry, yes. And now I'm getting all mixed up. All right, so they entered the thylakoid space at that time. So now we have these two electrons still on the chain. Now the same thing will happen. Photosystem 1 will release. There will be two electrons that escape, and now there's no splitting of water at photosystem 1. And now that the two electrons replace those two electrons ends up to their electron acceptor, their final electron acceptor, NADP plus, and then they become NADPH, and then that's going to head off to the Calvin cycle to run the dark reaction or the light independent reaction. Okay, so hydrogen ion concentration is building up again, and then we have the same thing. So we go from high to low, open up that active site with that turn, and then form ATP, which will also go to the Calvin cycle. All right, so that's photosynthesis, and it looks just like almost just like what happened on the mitochondria. We have electrons moving, we have the hydrogen ion concentration gradient building, and then we have the synthesis of ATP from that hydrogen ion concentration gradient moving through that enzyme. And that's the pieces that you need to understand what's happening. Not the name of photosystem 2 or photosystem 1, or you might need to know ATP synthase. I would know that one. But all these other members, you do not need to know their names. You just know how this all happens. In prokaryotes, the passage of electrons is accompanied by the movement of protons across the plasma membrane. So same concept. We're building a hydrogen ion concentration, and a hydrogen ion concentration gradient is going to drive the production of ATP. So all three examples that I just showed you do the same exact method. All right, so now there's some vocabulary that kind of goes with this process. The flow of protons back through the membrane bound ATP synthase by chemiosmosis, okay, drives the formation of ATP from ADP and inorganic phosphate. This is known as oxidative phosphorylation in cellular respiration and photophosphorylation in photosynthesis. So let's break apart what some of these words mean. Actually, the osmosis part of this is happening because this hydrogen ion concentration gradient is moving through. Okay, so that's the chemical part, and it's moving through from high to low, the osmosis, and that's forming the ATP. So that's called chemiosmosis. Okay, oxidative. What do we say oxidative meant? Oh, oxidative meant you were losing electrons. And what does phosphorylation mean? That something is gaining a phosphate group, right? What gained a phosphate group? ADP plus P. So that was the phosphorylation part, and now we have a oxidative. We lost our electrons, and there's many other reactions that happen along this line. That's called oxidative phosphorylation. The difference in photosynthesis phosphorylation has to do with the photons of light, with the energy coming from that first. All right, so now we have been to cellular respiration that has to do with having oxygen. Now we're going to talk about fermentation, and fermentation allows glycolysis to proceed in the absence of oxygen, okay, and produces organic molecules, including alcohol and lactic acid as waste products. So if you look at everything in this area, okay, well, I'm sure about that. That's glycolysis. In glycolysis, we make 2 ATPs and 2 pyruvates and 2 NADHs. So same thing. Glycolysis is part of fermentation of each part, but in alcohol fermentation, now we can't go into the mitochondria, can we? No, because we don't have oxygen. So what's going to happen is that this pyruvate is going to end up taking back those electrons. So it will be reduced. First, it'll lose 2 carbon dioxide molecules and become acetaldehyde, and it gains back those 2 electrons. So it's reduced. NAD can go back to work to make 2 more ATP because that's where we're getting them from in fermentation. That's the only place we have to get ATP from. And then our end product here is a molecule of alcohol, ethanol. So what are some examples of this? Bread, and we're making bread, and you can make wine and beer, and we actually made bread in my classroom this year, and the smell, it goes down the hall, and it smells so good. All right, and now the other type is lactic acid fermentation, and look at this. See, we're still the same. Glycolysis is still the same. We have 2 ATPs, we have our pyruvate, we have no O2. So so we're not going into mitochondria now, or well, this would be in, well, we can do this in eukaryotic cells. So anyway, so no oxygen going into the mitochondria. Now I'll give you an example in a second. First, is there CO2 with this one? No CO2 is a waste product of this. So what happens is this pyruvate gets back those 2 electrons, and then is going to form lactate. So our NAD can go back to work and make more ATP. So this can run for a little while. And speaking of running, if you remember any time that you were running and you had pain in you got out of breath and you had pain in your liver area, for example, and you had to stop and breathe for a while, that was a lactic acid. It's a toxin and it's building up, and that means that you're not running on ATP that was from your mitochondria. Now you are into getting the ATP from your lactic acid fermentation from glycolysis, and then you're going to only be producing this other toxin, and that's what's causing the pain. So you have to stop, you have to breathe some more, you have to breathe enough to remove the toxin from your cells, and then you have to breathe back enough to get a storage again of the oxygen before you get going again. Now respiration and fermentation are characteristic of all forms of life. All right, so the conversion of ATP to ADP releases energy, which is used to power many metabolic processes. So this is very simplified, but here's our ATP molecule, and there's our phosphate groups. I lost my bond somewhere along the way. All right, so there's some high energy bonds between these. Now when this is ADP and this is P, so when this bond is broken, the energy is released, and that energy will fuel a cellular process. Okay, it's a very small molecule with a small amount of energy, nothing like the big glucose molecule. We'll talk about that. All right, so now let's check for understanding, and we're going to talk about that right now. All right, so we're not really going to need these diagrams, but you see we have, here is just for review, there is our glycolysis, then we have pyruvate oxidation, Krebs cycle, and our electron transport chain. So the question, a researcher estimates that in a certain organism, the complete metabolism of glucose produces 30 molecules of ATP for each molecule of glucose. The energy released from the total oxidation of glucose under standard conditions is 686 kilocalories per mole. Yes. All right, so the energy released from the hydrolysis of ATP to ADP and inorganic phosphate under standard conditions is 7.3 kilocalories. Now calculate, so now this is our first calculate for the day. The amount of energy available from the hydrolysis of 30 moles of ATP, so what is this, 30 times 7.3, and that's 219, and so we did that. Now calculate the efficiency. This is where the tricky part comes in, because sometimes kids don't know what that means for them to do. The efficiency, what it means is if we had this much available, if we had 686 available, and we only used 219, that part is the efficient part. So what you do is you take the 219 that we were able to use and divide it by 686, and so my calculator says 0.319, so we will say it has a 32% efficiency. Now the next part of this, we're just describe. So we've had calculate, and describe. What happens to the excess energy that is released from the metabolism of glucose? So we started off with this much energy, and we only have this much we can use. Where'd the rest go? I'll let you take a moment to think about that. Jot down an answer, and then in about 4 seconds we will I'll reveal the scoring guideline for this. Okay, so did we say 219? Yes, we did. 32%? Yes. And did you jot down released as heat? You could have said increases entropy, but this would probably been my go-to if I was answering that question. Okay, now instead of more multiple choice practice and things like that, I had a request from, and here's a shout out to Patrick from Massachusetts, and Patrick wanted to start practicing in a large FRQ, and he asked if we could make that happen this week, and the timing was perfect because if you have seen this practice icon, these are practice questions that are modeling what's going to happen on the AP exam. So in your AP classroom, your teacher does not have to assign this to you. They're there for you to, you can, but they are there for you to practice on your own if you wanted to. So that's where I got this question, and so let's start. And I want to do something. I want to write down that this is what my timer says right now. Okay, so I'm looking at my diagram. I see I have glycolysis, and we're producing NADH, and I use it in electron transport chain when I have oxygen. I use it in fermentation when I don't have oxygen, and the end result of using the electrons from the NADH to carry was I get NAD plus to go back to glycolysis. So electrons from the oxidation of glucose and other nutrients by glycolysis and the Krebs cycle are added to the electron carrying compound NAD plus to form NADH. To meet the continuous requirement for NAD plus by cells, NADH is reduced back to NAD plus through the electron transport chain in the presence of oxygen or through fermentation in the absence of oxygen. All right, so this is the question. All right, so I'm going to write after I read. All right, mitochondrial ND5 is a mitochondrial gene that encodes a subunit of NADH dehydrogenase, the enzyme that catalyzes the initial oxidation of its substrate NADH to NAD plus NH in the electron transport chain of mitochondria. Let's stop right there so I can write myself a note if I was answering this question. Okay, there's this, and that's a gene, and it makes what? It's an enzyme that's going to take apart NADH to NAD plus NH. Okay, because I got to remember that, and then I'm going to go back and read it. Okay, so a mutation in mitochondrial ND5 is associated with a rare genetic disorder that results in a buildup of lactic acid in the body. So we're going to have, because we have this mutation, we're going to have an increase in lactic acid. A researcher hypothesizes that a mutated NADH dehydrogenase has decreased activity, but it's not completely non-functioning, and that by increasing the pool of NADH in cells, the activity of NADH dehydrogenase will increase. To test this idea, the researcher tested a group of individuals with this disorder with a vitamin that is similar to NADH and measured the concentration of NAD plus and lactic acid in the blood over the course of 20 weeks. The results from a represented individual are shown in figure 2. Let's go to figure 2. All right, so I have a graph, and I have concentration of NAD, and I have a graph with concentration of lactic acid. Both are over 20 weeks. Over weeks. Okay, and I see a peak at week 5, and what about week 5 here? It's a low point, but overall the trend, I see a decrease going down of the trend, and this trend went up and then down. Okay, so describe, we're going to do A, describe the pattern of inheritance that is most likely associated with mutation in the MTND5 gene. Okay, so that's what we need to do. Explain why individuals are not typically heterozygous with respect to mitochondrial genes. So all of a sudden, there's this information that we need to have in this question that had nothing to do with what we thought we were going to be talking about. So a long FRQ is not going to just talk about one unit. So like we're in cellular energetics and that's 3, but this just brought in unit 6 material, and that's the way a large FRQ is going to work. It's going to be a combination of unit concepts plus also science practices. So let's answer this. If you jot that down, let's answer that in just a moment. I'm going to tell you what I think. I'm going think that the mitochondrial gene has something to do with maternal. Okay, that's where we got it. If we get into mitochondrial maternal inheritance, and why individuals have not heterozygous, that's because there's only one copy of the DNA. So hold that thought and hope that you and I score at the end of this. All right. So B, identify a dependent variable. So we have to identify. So we're just going to write down the words measured in the researcher's experiment. So let's go ahead and do that. What can we say? The dependent variable. We can have concentration of NAD. We can also have concentration of what? Lactic acid. So let's write that down. NAD and lactic. Okay. Identify. Here we have another identify. One control that the researcher could use to improve the validity of the experiment. Justify the researcher analyzing blood samples at many intermediate time points instead of only at the beginning and the end of the 20 weeks. So we have two things to do. We've got to identify a control and then we've got to justify. So controls. I always write three things about a control. I say the control has no treatment. I say that the control shows what's normally happening without the treatment. And then the control also shows that the treatment is responsible. Okay. Those are three things. If I'm writing about a control, I'm always saying those things. Whether it's needed in the rubric or not, I'm always saying those things just in case. Now justify. Remember we have a claim and evidence and reasoning when we're justifying. So let's take a look. And why would we need to take this? What are we getting from taking these readings instead of just here and just here and just here and here? We're getting what? We're getting a trend, right? We're getting a trend. All right. So we can claim that we're getting a trend, and that would be, we could use our numbers if we wanted to, and then we could reason it out. But this would give us more data to examine or something like that. All right. I'm just in the planning stages. All of this is something I do to get started with writing, because the writing is the easiest part. The planning is the hard part. Okay. Now C, describe the relationship between the concentration of NAD in the blood and the concentration of lactic acid in the blood during the first 5 weeks of treatment with the vitamin. Okay. So what's happening with these two? I need to get rid of some of these lines. So we could say that did it say at 5 weeks to look at it? Yes. Okay. So we could say at 5 weeks that as NAD is increasing, what's happening at the lactic acid? We have decreasing, right? Okay. We can say that. And then we also need to calculate. Here we are calculating again. The average rate of change in blood NAD plus concentrations from week 5 to 17. We're in the graph. All right. So let's look at week 5. Here's week 5. Here's week 5. And we have 300. Oh, we don't need that one. Never mind. We have 300. And then it said week 17. So that's right here. I have the ability to have a little cheating device right here to help me, so I can see where this is going to be level. Okay. So it's not at 50. It's above 50. It's below 75. So let's say it is at 60. Okay. If this is a 300 and this is at 60, then how much did it change? It decreased by 240. Now over what period of time? 5 weeks to 17 weeks. So that's 12 weeks. So what's 240 divided by 12? It's 20. And then we go micromoles per liter and then per, what are these? These are weeks. Okay. So that's our answer. Now, what did we just do here? We did simple calculations that did not require a calculator. So I would get, this could be something that would be easily testable, is finding this kind of rates are really easy to do without a calculator. Let's just keep that in mind and get really good at it. All right. So now let's go to D. The researcher performed a follow-up experiment to measure the rate of oxygen consumption by muscle and brain cells. Predict the effect of the mitochondrial ND5 mutation on the rate of oxygen consumption in muscle and brain cells. So let's think about what happened. So we have this gene and there's a mutation in it. And so what we're not doing, we're not making that enzyme that took NADH to NAD. So what didn't happen? So in our electron transport chain, electrons did not happen. So they didn't travel down and they didn't get off on oxygen as a final electron acceptor. And so then oxygen wasn't needed. So what do we think is going to happen with oxygen consumption? It's going to decrease. And then all those things I just said by drawing it out and thinking it out would be my justification. The researcher had hypothesized that the addition of the vitamin that is similar in structure to the NADH, I'll give you a hint, okay, would increase the activity of the mutated NADH dehydrogenase enzyme in individuals with the disorder. Explain how the vitamin most likely increase the activity of the enzyme. I'll give you just a moment to think about that, and I'll let you do at least one part all by yourself. I'm actually just modeling this so you can see how to plan, because that's going to be critical. Okay, so now let's look at the scoring guidelines. So the score, this is for A description that a mutant allele is only inherited from maternal. Did I get that point? Yes. And then is a mitochondrial gene, and mitochondria only carry a single unpaired chromosome. Okay. And then on part B, an acceptable dependent variable. Acceptable dependent variables include the concentration of lactic acid in the blood and the concentration of NAD. We got this straight off of the dependent variable on the graph on the y-axis. And then identification of the control as measuring NAD plus and lactic acid. All right, in a group of other affected individuals treated with placebo. What's a placebo? A placebo means they're not getting the treatment. I personally, I wouldn't have thought of saying that. I didn't read enough into that question, so I'm not sure if I would gain that point with all my control talk, but maybe so. All right, hold on just a minute. Let me get rid of something. The justification that by collecting more data, the researchers will see a more accurate trend. All right, now C, a description that the concentration of NAD increased and the concentration of lactic acid decreased. Yes, a calculation of 20. We got that. Now the prediction that mutation will decrease the rate of oxygen consumption by the electron transport chain. The justification that mutation likely inhibits the rate of oxygen consumption because the electron transport chain will be less efficient, so less oxygen will be required as a terminal electron acceptor. And then this part, and I hope you said this. The explanation that the enzyme had decreased activity but still function, increasing the amount of substrate should increase the amount of product produced. Because the vitamin was similar to NADH, it could bind to the active site and increase the substrate concentration. How about that? All right, so let's look at this time. 42:53. All right, 29:31. All right, so 29:31 is when I started, and I started reading and I started planning, and that was 13 minutes. So now I have 12 minutes. It's all figured out to put this into some decent sentences that make sense and then upload. So this is so doable. If you're prepared and you have a plan and you do your planning, this moment of being 25 minutes, you should know you're gonna have plenty of time to get that job done. All right, so debrief and summary. Students, you have access to optional free response questioning questions to practice demonstrating your knowledge and skills. For the exam, look for the practice icon. This is my new best friend. These do not have to be assigned to you by your teacher for you to be able to practice, and they're similar in design to the questions on the exam. They won't be the same content or any same topic, but they will be that they're modeling what it's going to look like. And you can, one important thing for you to do is model what your time frame is going to take. Okay, start practicing with that timer. Get one running on the computer in front of you to go ahead and make you nervous as the clock is ticking, because I just did this planning with you in very few minutes and live on YouTube in a very stressful condition. So you can do it too. This bottom blurb is for your teachers. Be sure to use AP Classroom to help you. I use AP Classroom topic questions to warm up. I have my kids. I see my kids online every day. We start with AP Classroom topic questions to warm up. Then I talk through a topic with them, and then I have them watch parts of the videos. They have to have me making them watch me, and then we pull it all together, and then they answer FRQs that are within the video that we watch. So the people at AP Central at College Board are working so hard to give all these tools to these kids to help them. I've never seen a group of people so determined to make that happen. All right, so your homework tonight is a question. This is kind of like a lab that you might have had in Southern Respiration unit. I had peas and corn and beans and all of those things in my room, and snails and roaches and worms. Anyway, let me read this to you so I can get you out of here. An agricultural biologist was evaluating 2 newly developed varieties of wheat as potential crops. In an experiment, seedlings were germinating on moist paper towels at 20 degrees for 48 hours. Oxygen consumption of the 2-day-old seedlings was measured at different temperatures. The data are shown. Okay, so here's your graph. You have 2 varieties, 2 temperatures, and your job is, your homework, explain the relationship between metabolism and oxygen consumption. Discuss the effect of temperature on metabolism for each variety. So 2 task verbs are included in there. All right, so that's your homework. Have that ready for Monday or the next lesson. All right, so if you don't have internet or device, please contact cd.org/tech to let them help you. And you need to do this by April 24th. Also, big on your list right now, check your AP account email, make sure it's right, because they're going to send you a test ticket. All that information is going to roll out to you the week before the test, and we'll be here to help you with that as well. And thank you for watching. Next session will be with me also. It'll be cell communication. Yay, we're on to Unit 4, and so that means we're halfway done. All right, thank you. Bye. Welcome to AP Biology. Hi, I'm Gail Pfizer from Darnell Cookman Middle High School and Florida State College in Jacksonville, Florida. A lesson overview for today. Today we'll be talking about cell communication, signal transduction pathways, feedback mechanisms, positive and negative feedback. The topics as far as the CED will be 4.1, 2, 3, 4, and 5. We're going to talk about how cells communicate with one another. We're going to talk about short and long distances, signal transduction pathways, how the environment can elicit a response. Let's see, how can we change the structure and affect the signaling pathway, and then positive and negative feedback systems. So let's get started with today's warm-up, which was last lesson's homework from cellular respiration. We have, if you're just new to us, okay, if you're new to us tonight, I'm going to give you a moment to answer this question. And if you did this for homework on the cellular respiration day, then just hold tight and I'll show you the scoring guideline. So let's take a look at what's going on. Let me get some ink here. All right, so we have a graph, and we have oxygen consumption in milliliters, and we have two varieties. We have a variety A, and we have a variety B, and it looks like we're taking readings at two different temperatures of how much oxygen they're using. Okay, so an agricultural biologist was evaluating two newly developed varieties of wheat as potential crops. In an experiment, seedlings were germinated on moist paper towels at 20 degrees Celsius for 48 hours. Oxygen consumption of the 2-day-old seedlings was measured at different temperatures. The data are shown in the graph below. And then so what we need to do, explain. So our first task verb is explain the relationship between metabolism and oxygen consumption. And then we have another one, discuss the effects of temperature on metabolism for each variety of seedling. If you take a moment, and I'm going to put my timer on for today. Okay, I forgot to tell you also, and I'll forget if I don't do it now, that if you stay through the end, I'm going to give you a hint as to the location of the slide deck, the PowerPoint presentation slide deck for all of the previous presentations that Ali and I have done so far. Okay, let's take a look at the scoring guideline. Okay, so this part right here, this would be the explain part, and you could have said an increase in metabolism. As metabolism increases, oxygen consumption increases, or you could have said as metabolism decreases, oxygen consumption decreases. You could have said one of those things for the explain. And then on the discuss part, which is all over here, there are three different parts. So this right here is going to be this or that. So we'll start there. General statement that increasing temperature increases metabolic rate, oxygen consumption, with no specific mention of A or B. Just a general statement. And then, or you could have said variety A, rate of metabolism, oxygen consumption increases with increase in temperature, and or you could have said B, rate, well, and you had to say both of them. And rate of metabolism, oxygen consumption increases with temperature. Okay, you could have compared them. Variety B has a higher metabolism, oxygen consumption than A at either temperature, or vitamin. Variety B has better metabolism, oxygen consumption at lower temperatures than variety A. I'm going to back up just a moment to this word. I'm not sure how this snuck in there, but I'm going to put that under a list of possibly not measurable words that I want you to stay away from. You could also have said kinetic energy increases with temperature, enzyme reaction rates increase with temperature, effects on electron transport chain system. So all of those would have been point scoring comments. All right, so what you need to know about cell communication. All right, so ways that cells communicate with one another. Cells communicate by generating, transmitting, receiving, and responding to chemical signals. Cells communicate with one another through direct contact with other cells or from a distance via chemical signal. So cell to cell, they can communicate over short distances and over long distances, and we'll talk about examples of each of those. Okay, so let's start with cell to cell. So this is going to be an immune system example. So let's start with this guy right here. Okay, so this is, we can see that this is some phagocytosis going on, right? And so we have, here's our bacteria, and our bacteria is being broken up into pieces. Okay, and then there's going to be a piece of this bacteria that is an antigen, and the bacteria is not supposed to be in your body, so that's an antigen. And then we're going to present this macrophage is going to present this antigen on the outside of the surface with his major histocompatibility complex, and to show all of his immune partners that this is in our body. So it's a perfect example of you are what you eat, because he's wearing it. All right, so what happens then? There's this antigen will fit into this receptor of this T cell. So this is our helper T cell, and then this helper T cell is going to release cytokines, which are signaling molecule to send a message, and they're sending a message to our B cell for one, and that is going to then produce antibodies. So right here at this moment, okay, this was close cell to cell contact, but then we actually got into either short distance or long distance communication on the other part of the immune system. So this was an example of an antigen presenting cell. That's what APC is, and then the helper T cell. This part right here is our focus. Okay, so another example of cell to cell, it has to do with the plasma desmata, and the plasma desmata it has this opening between plant cells, and molecules can go from one plant cell to another plant cell. So let's talk about some short distance communication examples now. This has to do with embryo development, and if we look at cell 3, so here is cell 3, and these are going to be daughter cells of cell 3, and this particular cell is going to be to the anterior, and this particular cell is going to be to the posterior. But and they're going to have certain jobs. So how does this cell know what its job is going to be? There's molecules released from the anterior portion of cell 3, which are going to connect with receptors on this cell that are going to give instructions that turn transcription factors on, that make this start being differentiated into the beginning of muscle endomass. And then on the other hand, the cells to the posterior part are going to then connect with receptors and then give instruction to this cell to be adult intestine. So again, and this is called induction. So you divide your cells when you're developing, you have to know this is the front, this is the back, and then what's going to be what. So another example of short distance has to do with a synapse. So here we have neurotransmitters that are coming out of the presynaptic neuron, and they're being released into the synaptic cleft, and they're going to bind to this receptor. Now, when these receptors open, something else is going to happen that I'm going to talk about in a little bit. But this is their way, this is their short area of communicating the signal. All right, so let's talk about some long distance communication. Signals released by one cell can travel long distances to target cells of another type. So I want to specifically talk about the two types of hormones. So let's talk about steroid hormones. So here we have that 4 fused rings that it's a lipid, remember? And since it's non-polar, it can go through the plasma membrane, and it can also go through the nuclear membrane, and then it's going to bind directly with transcription factors, etc., that are going to then have this gene transcribed, and then eventually we'll have a new protein produced. Okay, so that's the hormone. And then the other one, hold on just a moment. And then the other cell, we have insulin, which is a protein hormone. It can't go in the cell because it is polar, so it has to have a receptor that it's going to bind with. So here's the receptor, and then there are other reactions that are going to occur in the middle between the actual response from this receptor. And so what we have here are vesicles that contain protein transport molecules. They're going to go to the plasma membrane, so it's going to fuse with the plasma membrane, and then these transport molecules, these protein transport, will then allow this glucose to go into the cell. So that is long distance, but two different types of long distance. One that's going to need a receptor, and one that can go straight through. Okay, so now let's put some names on the general categories of things we're going to be talking about. So we have a ligand right here, and this ligand is binding with its receptor protein. Okay, receptor protein. I'm going to call that part reception. And then here in the middle, in step 2, we have molecules communicating with each other, and they're changing each other and sending a signal. And this is going to be called a signal transduction pathway, or we can just call it signal transduction for here. And then there's going to be at the end a response. There's going to be transcription from the DNA. It's So we make some type of protein that we need. So this is the cellular response. So the signal transduction pathway has reception, the signal transduction, and then a cellular response. All right. So this diagram looks very busy, I know, and I'm going to break it down into sections so it's not that intimidating. So you will never have to know that this is the next step, and then this protein kinase A, and then it's never have to know all that. But what you need to know are some main points and the concept and the process that happens. So here we have our hormone, or our ligand is going to be epinephrine, and it's going to bind with our receptor. Okay, so it's going to bind with this receptor, and this receptor is part of a G protein coupled receptor because it has some G proteins that are coupled with this receptor. So when this ligand binds, this is actually going to change its shape, and when it changes its shape, then this alpha subunit of this G protein is going to bind to this receptor. Let me go ahead and color this one. Now, when it binds to this receptor, GDP is released, and GTP then will bind. Okay, now once that happens, this is going to activate an enzyme that's called adenylyl cyclase. Okay, and when that happens, then this whole next part happens. So I'm going to stop right there and talk about it on another slide. So G protein coupled receptors are an example of a receptor protein in eukaryotes. The ligand binding domain of a receptor recognizes a specific chemical messenger, which can be a peptide, a small chemical, or protein in a specific one-to-one relationship. So this is what happened, and this is the response of our G protein. Now, after the ligand binds, the intracellular domain of a receptor protein changes shape, initiating transduction of the signal. I just went over that changing of shape and initiating transduction. So second messengers such as cyclic A and P. So this is a second messenger, and our epinephrine then was our first messenger. Our molecules are going to relay and amplify the intracellular signal. So let's keep moving. So many signal transduction pathways include protein modification and phosphorylation cascade. So what's going to happen next in this? So this part right here, well, let's include it. No, it's right here. This is going to be our transduction part, and this was our reception. Okay. All right. So now the cyclic A and P, figure that. The cyclic A and P is then going to communicate the signal, and this is I'll talk about that in a second. So we're going to take this kinase, and a kinase is an enzyme, and it's going to phosphorylate molecules. So it's going this protein kinase will then phosphorylate the kinase to be a phosphorylase kinase, and then a glycogen phosphorylase, and then it actually will take glycogen, and then it will be broken down into glucose. Okay. So cyclic A and P, second messenger, epinephrine and receptor, reception, G protein changes, etc. until we get to second messenger, and that's just G protein is not every signal pathway if you thought that. Anyway, second messenger, and then this modification and phosphorylation is going to happen. All right. Signaling begins with recognition of chemical messenger, a ligand by a receptor protein in a cell. Okay, we got that. Signaling cascades relay signals from receptors to cell targets, often amplifying the incoming signals, resulting in appropriate responses by the cell, which would include cell growth, secretion of the molecules, or gene expression. So look at this. This is just not initiating this with one GTP. Look at this number. That's 100 molecules with one epinephrine. This is, what is that? That's 10,000, and that's 10,000, and then that's 100, and then we have a million molecules signaled and about to break down glycogen into glucose. Okay, all right, well, so let's stay there. So I'll talk about what happens with this glucose going to glycolysis because that's what we need it for. And on a later slide, I'm going to go review all this together. So here's another example of a ligand binding, but it's going to do a ligand gated channel. And how this happens, this is that synapse that I was talking about just a moment ago. So here's our neurotransmitter released from a presynaptic neuron, and it's going to be in the synapse, and it's going to bind a receptor that is a ligand gated channel. Okay, so if it's ligand gated, that means ligands will bind it, and then what happens? It's going to open. And so let's do a common error that I don't have listed. I'm thinking about right now. This is not going to move through there. Okay, what moves through there is we have a concentration gradient of sodium that's higher out here than in here. So sodium is going to move in. So that's what this ligand gated channel. So ligand bind bound to. I don't know how to say that. All right, so there's a ligand binding to this receptor, and then it opens. Okay, so that channel open. All right, so the environment can elicit a cellular response. Signal transduction pathways influence how the cell responds to its environment. Microbes communicate with other nearby cells with the use of chemical messengers to regulate specific pathways in response to population density. This is called quorum sensing. So here is a bacteria, and here's a bacteria, and this is two, which is a whole view. Okay, so all of those are SRS, and all of them are producing a protein and releasing it. And then when there's enough, then that protein is going to bind this particular receptor. And when it binds this receptor, then there's going to be this production of this toxin, and then this toxin is released into the host that it is in. Like if it was in our body, then it would be launching its virulence all at one time. Is what this is doing. All right, and that is quorum sensing. All right, so here's the whole story. So why are we breaking down this glycogen? So let's see. Let me start here. So here's our epinephrine. Okay, so our epinephrine is going to bind our what was that? It was our G protein coupled receptor, right? And when it binds, it's going to change the shape of our G protein. It'll change the shape, which will then make our alpha subunit. There's gamma and a beta there, but we don't need to go into that one right now. All right, so our alpha subunit is then going to release our GDP. After that happens, then our GTP is going to bind to our alpha subunit, and when that happens, we're going to be able to go activate our adenylyl cyclase, ATP. So here's our adenylyl cyclase, and our ATP will be converted to cyclic A and P, and then our phosphorylation protein modification cascade starts to happen, and it's going to be what by the time the signal gets to glycogen, there are 1 million signals coming into glycogen. So now we're going to take this glycogen phosphorylase, and what were they doing here? They were adding phosphate groups, right? And the glycogen is going to be broken down into glucose. Now, why do we have epinephrine? Why do we need glucose at this moment? Well, when I was in nursing school, I had this wonderful professor, and she told this great story about running from the bear. And we were talking. The topic was about the sympathetic nervous system. And when the sympathetic nervous system, you release epinephrine. So let's say that all of a sudden you're running from a bear. Epinephrine is going to be released, and it's responsible for the increased heart rate and their increased blood pressure, and your pupils get real big because you're running from the bear. And then what do you need to fuel all this muscle movement and everything else that's happening while you're running from the bear? You need glucose to turn into what? ATP. Okay, so we need glycolysis to kick in. So we have the energy for all the cellular processes for us to run faster than the bear. Okay, all right. Now, cellular responses, at least elicited by a signal transduction pathway. The one I chose to talk about are mating pheromones in yeast. Signal transduction may result in changes in gene expression and cell function, which may alter phenotype or result in programmed cell death, apoptosis. I put a long list here. I'm not going to read them to you, but they'll be in the slide deck. Other things that you could think about with this, but I want to use the mating pheromones in the yeast. This is a picture of a slide in my room a few years ago because I took this picture and I let the slide sit there all day. And so you could, after hours, you could see that I think that I have what's called a shmoo. So a shmoo is like this cartoon character from, I don't know, 30s, 20s, somewhere else, some other time frame that looks like in this shape. And it comes from a cell that starts in this shape. So I think that I have some in here. I know this right here looks like budding, but I think I do have some shmoos that are happening. So how does this happen? I'm going to draw you a very simplified version of this with no names. Okay, just the process itself. So let's have this is our cell membrane, by the way, and this will be the cell membrane of an alpha type of yeast. And this is an A type of yeast over here. Okay, A type is going to release mating pheromones, and the A type mating pheromones will then bind to a receptor on the alpha type. Now there will be layers of different molecules that will activate other molecules, okay, and phosphorylate them. So let's do this. And then we're doing this, and we're the signal's going, and we're adding phosphates, and then we have this one. I put these words down there to give me a cue of what I wanted to do. I don't want to give you names because I want you to start memorizing names. And then these are all activating each other. And then we have this last one, okay, this foramen that's going to get activated, and then the P on it. Okay, so now we're here. Now, what's the job of this phosphorylated foramen? The job is, so you have actin filaments for cellular structure in your cells. So this is a bad version of an active molecule. Now, this was assembled together with little active molecules, okay, this active filament. So let's say that this phosphorylated formin is responsible for adding active molecules to this filament. What would happen with the cell membrane is something that's responsible for its shape started doing this, right? It's going to start taking that shape that we just talked about, and it actually would happen like heading towards, the direction would be towards the A yeast, okay? All right, now, a change in the structure of any signaling molecule affects the activity of the signaling pathway and can alter the cellular response. Mutations in any domain of the receptor protein or in any component of the signaling pathway may affect the downstream components by altering the subsequent transduction of the cell. Chemicals that interfere with any component of the signaling pathway may activate or inhibit the pathway. So this is a hedgehog protein. We do this case study, and I think there's something about silencing the hedgehog in my room. Anyway, it's about this hedgehog protein that can be associated with cell division, etc., but it can be associated with cancer. And so this right here is a view with the hedgehog protein present. And then we had reception, and we had transduction happening, and then we have a cellular response. And here we have without the hedgehog protein, then the transduction is not happening, and there's no cellular response. But what if we did have here? And then what if there was something that was a mutation in this receptor? Would this happen? No, that wouldn't happen. If there was something wrong here, then the rest of this would then not happen. That would be inhibited. All right, so now negative feedback and homeostasis. Negative feedback and positive feedback does work. Give kids problems sometimes, and we'll talk about how to try to figure that out. Organisms use feedback mechanisms to maintain their internal environment and respond to internal and external environmental changes. Negative feedback mechanisms maintain homeostasis for a particular condition by regulating physiological processes. If a system is perturbed, negative feedback mechanisms return the system back to its target set point. These processes operate at the molecular and cellular level. So I'm going to talk about blood calcium levels. So this right here is our set point. Okay, normal blood to normal is our set point. Now, if our blood calcium level was to fall, then parathyroid hormone would be released. You have a thyroid, have these 4 parathyroid glands, and they release this parathyroid hormone, and then that is going to take the calcium out of the bone and put it into the blood, and that will bring the blood calcium level back to its set point. All right. If your blood calcium was to rise, then we need to move calcium out of your blood, and that's where the thyroid will release calcitonin, and then calcium is deposited into your bones, taken out of the blood, and then your blood calcium levels fall. Some other negative feedback systems you can think about is blood sugar regulation by insulin and glucagon. If your blood sugar is too high, then insulin is released. If your blood sugar is too low, then glucagon, and then you also have the thyroid stimulating hormone, which involves your thyroid and your pituitary gland, etc. So those are some other systems. All right, here's positive. So negative, we were returning. So something was out of whack. Okay, it was away from the set point, and we had to bring it back to the set point. So that's negative feedback. It had been away, and we need to bring it back. Positive, we're just going to go away from set point because there is no set point until we have a system change. Okay, and then things will be back to normal. So an example of this would be if we have an injury to a cell. So we have an injury to a cell, and then these molecules are released from that cell, and they activate inactive clotting factors that now are activated. Now these activated clotting factors are going to activate prothrombin, and then we're going to have thrombin, and then thrombin will turn fibrinogen into fibrin, and then that fibrin, which is exactly what it sounds like, fiber, is going to be helping to stop that blood clot formation. All right, so this right here keeps happening until this happens. So that's positive feedback. We keep going and going and going until we get response we need, like the onset of labor and childbirth. You can't turn back when that's going. Okay, you go until the system has changed. You deliver the baby. So this fibrin, think about why when you use gauze, that why it is in this form now. All right, ripening of fruit, lactation, those are other positive feedback loops too. All right, so check for understanding. Our science skill today, among others. Okay, there's a lot of science skills in here. 6E, predict the causes or effects of a change in or disruption to one or more components in a biological system based on A, biological concepts or processes, B, visual representation of a biological concept, process, or model. Some task verbs we're going to use, describe, and explain. When I learn something, this is what I do in my notes. I read words and I make them have a picture, and it just kind of sticks. It's not for everybody, but a lot of people can do that. All right, so now let's look at this multiple choice question. And I want to say that you're not responsible for knowing the immune system. It could be anything that is not in the curriculum. If it's used in a question, if it's out of curriculum framework, then they give you all the upfront necessary information you need to be able to answer that question. But this is just a little quiz on cell communication and who is talking to whom. All right, the vertebrate immune system consists of multiple types of cells that work together to protect the body from infections as well as from damaged cells. In the immune response represented above, antibodies are synthesized and secreted into the blood and the lymph. The diagram shows the interaction of macrophages, B cells, and helper T cells. Which of the following correctly labels the cells depicted in the diagram? Okay, so I don't believe that you would have to know this type of information. It's just out of the framework. But I wanted, since we went over it, you should have learned something about the immune system if you didn't already. All right, so let's look at cell X, Y, and Z. And so here we have our engulfing. Now we kind of did that with endocytosis, right? Okay, so now it's engulfing what? It's engulfing our bacteria, and that bacteria fragment's going to be presented. All right, so cell X was what? Was it a B cell? No. Helper T cell? No. Now cell Y was what? What was our cell Y that was in the middle? It was our helper T. All right, so our answer there is C. Okay, did you remember? All right, and then let's take it one more step. This part was our cell-to-cell communication, and this part was our short or long distance. Okay. All right, so we just went over this diagram as well, so you know it. The model shown in the figure represents the role of two hormones, calcitonin and parathyroid hormone (PTH), in maintaining normal blood calcium levels in humans. If a dietary change results in an increase in blood calcium concentration above normal limits, okay, so we have too much. Which of the following is the most likely effect on calcium homeostasis? All right, so what's going to happen if it's too high? Where were we? Blood calcium levels rise. So what's going to happen? Thyroid. So calcitonin levels will rise. Yes, that's promoting the deposit of calcium into bone. So they gave us. We didn't need to know about calcitonin and parathyroid. They gave us everything we needed to know in a diagram to answer that question. So that's what you kind of can expect if it's out of the framework. All right, here's something else we're familiar with. Let's see. Argatroban is a competitive inhibitor. Okay, we know what that is. Which of the following effects on the coagulation of the cascade is most likely to result from inhibiting thrombin activity? So if we stop it here, what's going to happen? All right, the activation of clotting factors will be blocked. No, because it's downstream from that. All right, the rate of fibrin formation will decrease. Yes, thrombin will be converted to thrombin. Prothrombin, no, that'll be backwards, and the rate of blood clot formation will increase. Definitely not. So our answer there is B. Okay, so let's do a short FRQ together. This one is just really short, and I like this one a lot. Another diagram. So we have, look at what we have. We have a cellular response to infection by pathogenic bacteria. We have at number 1, this is where the bacterial infection is occurring. So what's happening there? We have inactive caspase, and now it's active caspase. Okay, so that's the first thing that happened. And then what happened at stage 2? We have inactive interleukin, and now we have actin. And what's it active? And what's it doing? It's able to leave the cell. That's part 3. Then we have this protein. I can tell because it's telling me this N-terminal is being released. So we have this N-terminal, this gas terminal is now like making a hole. Okay, let's make it 4 in. Okay, make it 4 in our cell membrane. All right, so now let's read. And I put the diagram so you can follow along. Okay, some pathogenic bacteria enter cells, replicate, and spread to other cells, causing illness in the host organism. Host cells respond to these infections in a number of ways, one of which involves activating particular enzymatic pathways, which are in figure 1. That's figure 1. Cells normally produce a steady supply of inactive caspase 1 protein. In response to intracellular pathogens, the inactive caspase 1 is cleaved and forms an active caspase 1. Active caspase 1 can cleave 2 other proteins. When caspase 1 cleaves an inactive interleukin in step 2, the active portion of interleukin is released released from the cell. Okay, we saw that happening. And interleukin is a signaling molecule that activates the immune response. So there, I told you what it does. When caspase 1 cleaves gasdermin, the internal portions of several gasdermin proteins associate in the cell membrane to form a large, non-specific core. Okay, researchers created the model in Figure 1 using data from cell fractionation studies. In the experiments, various parts of the cell were separated into fractions by mechanical and chemical methods. Specific proteins known to be located in different parts of the cell were used as markers to determine the location of other proteins. The table below, which is on the next slide, shows the presence of known proteins in specific cellular fractions. Okay, here is the table that I was referring to. But I'm going to jump to the task verbs. We have explain, we have describe, explain how gasdermin pore formation, and so there's two explains here. Okay, well, hang on, hold on. Explain how gasdermin pore formation and interleukin release. Yes, there's two. So you're going to explain two things. Release contribute to an organism's defense against a bacterial pathogen, and then describe the effect of inhibiting step 3 on the formation of pores and on the release of interleukin from the cell. So I'm going to give you just a moment, and then I'll talk about what I would say, and then we'll look at the scoring guidelines. Just jot down some quick notes. Oh, I also want to say neither one of those questions had to do with that, did it? Now that's exactly the thing that you're going to do on your 4-part FRQ. Not all 4 parts have to do with every graph, every table, every diagram. You have to find the one that's relevant to the question that you're answering, or the 1 or 2 that's relevant. Okay, so explain how gasdermin pore formation and interleukin release contribute to an organism's defense against bacterial pathogens. So the gasdermin pore formation. So it made, what did it make in this cell? Okay, there's my phospholipid bilayer. What did it make? It made a hole, right? So what's going to happen to the inside of the contents? The inside of the contents are going to spill out. So what's going to happen? The cell's going to die. And then the interleukin release. What did the text told us what it was going to do? It was going to go and it was going to call out the rest of the immune system, right? So it's going to bring in, try to immune system to kill this bacteria. Now, describe the effect of inhibiting step 3 on the formation of pores. So, well, that what's going to do to those pores? There won't be any, right? And then on the release of the interleukin, will that have anything to do with step 2? No, interleukin will still be released. So let's check my answer and your answer against the scoring guideline. All right, cell lysis destroys infected cells. Oh wait, I had it going out, but guess what? It's coming in. So then the cell, so if fluid's moving in, then the cell would burst. Okay, so cell lysis destroyed infected cells, or cell lysis prevents bacteria from replicating. Interleukin signaling will stimulate immune cells, components of the immune system, to destroy the infected cells. 2 points. You have a description. The pores will not form, and the interleukin release will not be affected. Interleukin release continues. Okay. All right. So I hope you got both of those points, and I hope you thought about everything coming into the cell with those pores open. All right. So today's takeaway: cells communicate by generating, transmitting, receiving, and responding to chemical signals. You should be able to do all these things. Okay. Talk about how cells can communicate with each other, short and long distances can be involved, or cell to cell, the parts of the signal transduction pathway, and how that pathway produces a cell response. Don't worry about knowing all of those names of anything. Okay. Just be able to look at something and tell what's going on, and then be able to associate it with what you know happens at those stages. Describe the role of the environment in eliciting a cellular response. Explain how change in structure can affect activity. You can inhibit it. Describe positive and negative feedback mechanism. All right. Common errors and misunderstandings. Compare your data. Okay. Let's look at that first. Our homework. Okay. Our warm-up. I highly recommend this. Always compare specific evidence. Okay. Like your numbers. Like if the reading was it had lost 10 milliliters, and this one had used 10, and this one had used 15. I advise you to use those figures to compare. Always compare to control if it's there, or always specifically compare to others. Okay. When you're using this evidence, always make sure you use measurable words. Okay. I would not use better. I would use this is make sure that increase or decrease. Don't use the word just it changed. Okay. Don't do that. All right. Negative feedback is moving back to the set point, not away. I'm not sure what else I can tell you to not get confused with what negative feedback and what's positive feedback. I don't know. Maybe we need some type of acronym or something to remove. Positive is moving away. Not sure, but maybe you can think about that. All right. So your homework tonight for tomorrow's lesson, you're going to have. I'm going to tell you this is your homework. All right. The figure above represents a generalized hormone signaling pathway. Briefly explain the role of each numbered step in regulating target gene expression. We went over each one of these, so this should be easy breezy. Okay. So that's your homework, and let's see what else. Oh, things to do. This is your list: internet and device. You got to get this done by the 24th. If you want, if you need the College Board to help you, contact cb.org/tech. They can help you with device or internet, but they just need to know how to help you. Something that's come up in my classroom and others. Okay. Do this. Make sure your email account is correct in your College Board AP account. It's critical that email is correct. And then also, so they can relax, because I found out today, I asked everyone personally, tell your teacher if you plan to test. Okay. I told mine, this is a no judge zone. People's lives are different than mine right now. Everybody's lives are different. This is strictly up to you. Okay. All right. And thanks for watching. We hope you'll join us again. Be sure to complete the practice question. Tomorrow, the next lesson is today. We'll review the cell cycle and special guest star, Mitosis. Yay. And then my hints to find the PowerPoint deck slide. I'm just going to say this. You'll find them in our videos, but nothing in biology makes sense without one. All right. Thank you. Bye.
Hi, welcome to AP Biology. I'm Allison Katay, and today's lesson is going to be on the cell cycle and the regulation of the cell cycle. We're also going to talk about Lab Investigation 7, just the first part about mitosis, and the cell cycle. We're going to look at 2 science practices today. We're going to perform a chi-square analysis to test a null hypothesis. And we're also going to look at predicting the causes or effects of a change or disruption to one or more components of a biological system based on biological concepts or processes. And as you might guess, these are going to relate to the cell cycle and its regulation. By the end of this lesson, you'll be able to describe the events that occur in the cell cycle, explain how mitosis results in the transmission of chromosomes from one generation to the next, describe the role of checkpoints in regulating the cell cycle, and describe the effects of disruptions to the cell cycle on the cell or on the organism. All right, so let's start with our warm-up, which is from yesterday's lesson on cell communication. Yesterday was kind of the nuts and bolts and overview of cell communication, and today we're going to take a look at some pathways that actually regulate the cell cycle. So let's take a look at the question. You're looking at a model or a diagram that represents a generalized hormone signaling pathway. You are asked to briefly explain the role of each numbered step in regulating target gene expression. So you can see you've got step 1, and step 1 is labeled on the plasma membrane, and you're given direction. You can see that on one side you've got the extracellular fluid, and then on the other side you've got the cytoplasm. And so step 2 takes place in the cytoplasm, and it has multiple steps. And then finally we see step 3 is occurring inside the nucleus, and this must be a eukaryotic cell because we see a nuclear membrane with nuclear pores. And step 3 does something with the DNA. All right, so if you did your homework last night, good for you. Take it out so you can compare your answer with the scoring guidelines. And if you're just joining us now, think about how you would answer this question. Okay, so 3 steps. What are they, and what is their role in regulating this response? Okay, so first of all, step 1. Step 1 is called reception, and reception happens when a ligand, in this case it's a hormone, binds to a receptor that is on the surface of the plasma membrane. Once that ligand or hormone binds to the receptor, we see a shape change in that receptor molecule. Okay, so that kind of marks the end of reception with a shape change once we've got the signal molecule bound to the receptor. The second step, again occurring in the cytoplasm, is called transduction, and this is a multi-step pathway that amplifies the signal. So you can have one signal molecule that is going to generate a lot of other molecules that are going to bring about a coordinated response. Step 3 is called the response, and in the response, in this case, we are going to target the expression of a gene. It could either be stimulating gene expression, expression, or it could be stopping or repressing gene expression. So those are the 3 steps: reception, transduction, response, and they're characteristic of any cell signaling pathway. And as I said, today we're going to apply this to cell cycle regulation. All right, so let's get started. Here's what you need to know. In eukaryotes, there are really 2 processes that we see in the transmission of genetic information, and you probably heard both of them before: mitosis and meiosis. In eukaryotic cell division consists of mitosis, which is the division of the genetic material in the nucleus, followed by cytokinesis, which is the division of the cytoplasm. Okay, so that's mitosis. Gametes are produced by a variation of cell division called meiosis. Meiosis yields non-identical daughter cells that have only one set of chromosomes, which is half as many as the parent cell. And tune in tomorrow, because tomorrow we're going to focus on meiosis, whereas today we're focusing on mitosis and the rest of the cell cycle. All right, so let's take a look at this diagram. The cell cycle is a highly regulated series of events that is used for the growth and reproduction of cells. It consists of sequential stages of interphase, which is in turn made up of 3 parts called G1, S, and G2, followed by mitosis, which is the division of the nucleus, and cytokinesis, which is the division of the cytoplasm. And as you look at this diagram, you can see that the majority of the time is spent in interphase, and that only a small portion of the cell cycle is the actual nuclear and cytoplasmic division. Okay, so briefly, the G1 is the general growth phase of the cell. The S phase stands for synthesis, and this is where the chromosomes are replicated or duplicated. And then G2 is the last part of interphase, and this is where the growth in the cell is specifically about manufacturing molecules and creating energy molecules to use through the process of cell division. And then finally, we have the M phase or mitosis, where the chromosomes are distributed to the new cells, and then the cytokinesis divides, creating 2 new identical daughter cells. We're going to talk about how that process happens next. Okay, so let's just take a look at how the chromosomes are actually passed on to the daughter cells. And I tell my students when I'm teaching in the classroom that if you don't remember anything else about mitosis, the outcome of mitosis is to create 2 genetically identical cells. And you're going to see why that's important in some of my examples, but we want to create 2 genetically identical daughter cells or cells. Okay, so first of all, just to look at this diagram, we're only looking at 1 chromosome, although we know that particularly in a human cell we have 46 chromosomes. We're just looking at 1 to simplify it, and it's represented here in purple. It has not yet duplicated. Okay, you can also see that there's an area that is a constricted part, and that's called the centromere. And then you can see we call these the arms of the chromosome. So then the chromosome is going to duplicate, and that occurs in the S phase of interphase of the cell cycle. Once duplicated, a chromosome consists of 2 chromatids. They are connected along their entire length, and each chromatid contains a copy of the DNA molecule. Finally, you can see that through molecular and mechanical processes that we'll go over in a minute, the sister chromatids are separated into 2 chromosomes and distributed into 2 daughter cells, and these are genetically identical. The DNA copy is identical, and so those 2 new cells are exact copies. So mitosis plays a role in growth of a cell or an organism, if it's a single-celled organism, tissue repair, and asexual reproduction in single-celled organisms. So let's take a look at either cell reproduction, that's the first image we see, or if it's a new organism that reproduces asexually, I have an image of binary fission, which is used in bacteria. This example is an E. coli, and the end result, after the genetic material is copied and the cell splits, we have 2 identical E. coli bacterial cells. In B, you can see that we also use mitosis in growth and development. So just after an embryo is formed, a sperm cell fertilizes an egg, you get a zygote, and then it starts to develop into an embryo. All of those divisions are mitotic divisions, so we want identical cells up until a point where the cells begin to specialize. So you might see this in an animal cell as a blastula stage. And then finally, mitosis happens to renew tissue that is damaged or worn out. And so, for example, one of the reasons you want genetically identical cells here is, let's say you scrape your knee and your tissue needs to get repaired and replaced. You want the tissue that is replacing those damaged cells to be exactly the same. So again, we use mitosis for just cell growth, organism growth if you're single cell, tissue repair, and asexual reproduction. So now we're going to talk about mitosis, which is the actual nuclear division. And we don't really need to know all the details, but I want you to be able to recognize all of the stages. In my class, I do something called handy model, and I'm not sure if you can see it here. I can see my hands, but you can follow along with me. So interphase, make a fist, right? Interphase, the cell is not dividing. And then in prophase, the chromosomes condense and they shorten and thicken. So wiggle your chromosomes. In metaphase, the chromosomes are going to line up in the middle of the cell. In anaphase, they're going to hold to opposite sides. And then in telophase, we get our cytoplasm division, and you have 2 genetically identical cells. So that's the handy model. But let's just kind of take a look at some of the important things. So G2 is the last part of interphase, and it's characterized by the fact that you can still see an intact nuclear envelope and 1 or more nucleoli. You can also see the chromosomes are unwound. They are duplicated, but they are not condensed. So they're kind of fine threads that we refer to as chromatin. I'm not going to read this to you, but it will be here if you go back and look at these slide decks later on. In prophase, what you start to see is the nuclear envelope begins to disappear, and the chromosomes begin to shorten and thicken and condense. You can see that they're held together at that centromere. And then you can also see the formation of the mitotic spindle, which is made of microtubules, and it's super important in actually moving the chromosomes during the process of cell division or nuclear division. You can also see these little asters, which are radiating out from centrioles, and they seem to play a role in kind of the physics or mechanics of bracing those microtubules or spindle during this process of moving chromosomes. One note, centrosomes are only found in animal cells. Plant cells do have centrioles, but they don't arise from centrosomes. Then we get metaphase. Prometaphase comes just before metaphase, and this is where you can really see the spindle fibers beginning to form. And there are 2 kinds of microtubules. There are microtubules that attach to the kinetochores, which are the little protein discs that are at the center of those sister chromatids. So you can see that they are actually attaching to the kinetochores. And then you have non-kinetochore microtubules, which do not connect to those chromosomes. Okay. And then what you can see in metaphase is meta means middle, so those chromosomes are maneuvered to line up right in the middle of the cell at the equatorial plate. And you can see the spindle is composed of aster fibers, non-kinetochore fibers, and kinetochore fibers. And the fibers are actually microtubules. Okay. So then we see that the anaphase is next. In anaphase, you see the chromatids are being pulled to opposite sides of the cell by those kinetochore spindle fibers. And what's happening to move them is there's actually motor proteins that actually will pull toward opposite sides of the cell. And then finally, we get telophase and cytokinesis. And you can see that the non-kinetochore microtubules are important in this stage, pushing them apart to opposite sides. You can also see a cleavage furrow beginning that is going to actually separate those into 2 new cells, identical sister cells. And you can see that in telophase, you kind of get a reverse prophase where you start to see the nucleolysis reforming, the chromosomes are unwinding into the chromatin form, and you start to see that nuclear envelope reforming. And then you can also see that the microtubules in the spindle apparatus has been dismantled. Okay, so that's mitosis. That's the cell cycle. Before I talk about regulation, I want to talk about a special stage of the cell cycle that is called G0. And this is kind of a suspended animation stage where it no longer divides. So it can exit the cycle into the G0, but it can re-enter the cell cycle in response to appropriate cues. And I'm going to give you some examples of what that might be. So non-dividing cells may exit the cell cycle or be held at a certain stage in the cell cycle rather than continuing through to divide and create 2 new cells. So what we're looking at, and I'm going to go into detail in the next slide about checkpoints, but you can think of them as stop and go signals. So what you see here is a G1 checkpoint that has a red light. And what that means is it's not getting the appropriate cell signal to pass through to the next portion of the cell cycle into the synthesis phase in this case. And so it will actually exit the cycle and go into this non-dividing state called G0. So let me give you a couple of examples. Mature neurons or nerve cells are an example of a cell that is in G0. 0. So that means that once a nerve cell matures, it will not divide. It will no longer go through its cell cycle. In contrast, epithelial cells, like your skin cells or the cells that line your digestive tract, they're constantly dividing. They're replacing the layers of cells. So the outer layer of your skin is actually dead epithelial cells, and below that you have the cells that are dividing to replace those. And then finally, we have liver cells, which are an example of a cell that enters the G0 stage, but it can be induced to re-enter the cycle and divide. And that's usually done through a physical means. So, for example, if you wanted to donate a portion of your liver for a liver transplant, they will actually surgically remove a lobe of your liver, and then that incision that is done surgically will stimulate your liver to regrow. Okay, so those are 3 examples: ones that don't divide, ones that constantly divide, and the ones that don't divide unless they have a special cue to cause them to be back, re-enter the cell cycle. All right, so let's talk about checkpoints. Okay, so you can see in my first image that there are 3 checkpoints. They're kind of these red blockades that you see popping up. So they are called the G1, the G2, and the M checkpoint. So in the G1, for many cells, the G1 checkpoint seems to be the most important. Sometimes it's referred to as the restriction point. And what's checked at this point is the size of the cell is adequate, does it have growth factors, and the environment. And the environment is important in that not only do we have internal controls, there are external controls of the cell cycle as well. And my last slide will talk about external or environmental factors that can affect the cell cycle regulation. Okay, so the G1 again is the first checkpoint. It turns out that if cells pass that G1 checkpoint, they do tend to go on, they synthesize their DNA, they prepare to divide, and they divide successfully. But it's also the place that cells can drop out into that G0. Then the G2 checkpoint, and this gets a little complicated, so kind of hang in there with me because there's a lot of acronyms. So in the G2 checkpoint, there is a chemical that is called MPF, which stands for mitosis promoting factor, and it's actually a combination of 2 other molecules, an enzyme called cyclin-dependent kinase, or CDK for short. And you might recall from yesterday's lesson that a kinase is an enzyme that phosphorylates a molecule. So it adds a phosphate group to a molecule. So anytime you see a kinase, that's the job that enzyme is doing. And another protein called a cyclin. And in a later slide, I'm going to show you the relationship in terms of what's going on in the concentrations of CDKs, the cyclin-dependent kinases, and the cyclin proteins, because they are related. When they join together, they create this mitosis promoting factor, or MPF for short. And it controls the movement of G2 into mitosis. And the way it does that is by phosphorylating and activating proteins that are involved with 4 different aspects: chromosome condensation, which is necessary for mitosis, right? You need chromosomes; nuclear envelope breakdown, which happens in prophase, because you need to be able to move your chromosomes without that nuclear envelope being in the way; spindle assembly, how do you move the chromosomes? You need to have your microtubules; and then it actually creates an enzyme that will break down that mitosis promoting factor itself to signal the end of that mitotic phase. So this one's a little complicated, but it is the checkpoint that is between the second growth phase and the actual division of the nuclear material. And then finally, there's a checkpoint called the M checkpoint that happens in mitosis. It happens between metaphase and anaphase. So I want you to think about what happens between metaphase and anaphase, right? In metaphase, the chromosomes line up and the kinetochore spindle fibers attach to them. Okay, so if those fibers do not attach to the chromosomes, anaphase won't begin because there's no way to move those chromosomes. So the M checkpoint is to make sure everybody's attached. All right, a little bit more about that M checkpoint that I think this picture really illustrates it nicely. On the left side, you've got a red flag. You are not going past that, and that's because if you look carefully at the image down here, those spindle fibers are not attached to all of those chromosomes. If you look at the green light, the signal to go, you can see at metaphase, if you look carefully, all of the kinetochore fibers are attached to those chromosomes, and so during anaphase, they can be pulled to opposite sides of the cell. This ensures that you get equal distribution of chromosomes with all of the genetic complement in each of the new cells. So let's take a look at the interaction between cyclins and CDKs. Let's look first here, because this is a really good image to start with. The purple line is the MPF activity, and notice that it's low and it peaks at mitosis, and then it drops off again, and then it's low and it peaks at mitosis, and it drops off again, and then it peaks at mitosis and drops off again. So you notice that MPF is active during mitosis. Okay. Now if you look at the cyclin line, cyclin cycles, right? So we can see that it builds up prior to mitosis, and then it's at its peak, and then it declines rapidly. Then it builds up in the next cycle until it reaches a peak in mitosis, and then declines right after. Same thing again. So now let's look at it in this cycle form. Okay, just to orient you, here's your G1, your S, your G2, and your M. Remember that MPF is working at that G2 checkpoint, which is going to give you the signal to go ahead on into mitosis and divide. So cyclin, its name gets its name because it cycles, right? So here it is fully built up. But then, let me come back to how it degrades over here. So you get your cyclin building up. CDK is around the whole time, but when cyclin builds up to a critical level, they join, they combine together and form mitosis promoting factor, which is the signal that says, okay, go ahead on into mitosis and divide. Then what happens is that the cyclin is degraded and CDK continues to be around, and it will pick up the cyclin in the next cycle. And so that is how we pass from the G2 to the M. So remember that those are all the internal signals that happen to regulate the cell cycle. They're also external factors. And I'm going to give you an example of a growth factor. This growth factor is called platelet-derived growth factor. Platelet-derived growth factor, right? PDGF. That's another acronym. And it will stimulate these fibroblasts, which are cells, to divide in culture. So you can see those culture fibroblasts. You add that growth factor, then they will divide. Okay. There are 2 other things that I want to show you, and these are external signals. One of them is called density-dependent inhibition, which happens when cells that are really crowded, there's not enough resources for all of the cells, they usually will stop dividing. So that gives the cells that are already there the best chance to survive. So when cells get super crowded, they stop dividing. Also true of most animal cells is they need a substrate to grow on and be able to divide. And this is called anchorage dependent. So the cells are actually anchored to a substrate. In the petri dish, you can see they're not crowded, they're anchored to a substrate, and they're dividing. In this case, this is a case of a cancer cell. And what's happening is these cells don't care that it's crowded, and they don't care that they're not actually dividing on a substrate. So cancer cells will exhibit neither density-dependent inhibition nor anchorage dependence. So those are external factors, growth factors, and density, the density that the cells find themselves in. Okay, so there are 2 types of disruptions I want to talk about. Disruptions to the cell cycle. So if you miss an internal signal or an external signal, it can result in cancer or in something called apoptosis, which is programmed cell death. And this is not necessarily a bad thing. I'm going to give you an example that had happened when your fingers and toes were forming. And so apoptosis is a type of programmed cell death in which the cell components are disposed of in an orderly fashion without damage to neighboring cells. So when your digits are forming, your fingers and toes, you can see that they form with a whole wedge of tissue. And then apoptosis happens specifically in the interdigit tissue so that you get nice fine digits. If anybody has web fingers or web toes, you know that apoptotic pathway kind of stops short. Okay. The second thing I want to talk about are disruptions to the cell cycle that may result in cancer. And I want to talk specifically about a transcription factor that's called P53. So first I'd like to talk about this pathway with that transcription factor present. And then in the image below, I want to talk about what happens without that P53 transcription factor. So we all know that our DNA can be damaged by the environment, especially if you're out sunbathing too much. You know that there is a chance that your cells can be damaged in your skin and you can develop skin cancer. So we've got UV light coming in, causes damage to the DNA, and we see a transduction pathway. So that's the signal. We see a transduction pathway that includes some protein kinases that are going to eventually activate P53. P53 then goes to the nucleus and creates a protein that inhibits the cell cycle. Okay, so we got our signal, we've got our transduction, and we have a response, which is to produce protein that inhibits the cell cycle. So that looks like the response is that the cell cycle is inhibited. So the damaged DNA is not replicated, so it stops in that cell. The P53 is stimulating the expression of that protein, which is a CDK inhibitor. So it's going to actually inhibit those CDKs or cyclin-dependent kinases and it prevents the progression of the cell cycle, which means no cell division. All right, so now let's look at what happens without that p53 transcription factor regulating the cell cycle. We see the same signal, light is damaging the DNA. We see the same transduction. We get our protein kinase cascade going. However, p53 is not activated. So there's some type of missing or defective transcription factor, which means that we will not be able to make an inhibitory protein, and the cell cycle is not inhibited. So that means that the mutation that happens prevents the activation of p53, so that CDK cannot be, the inhibitor cannot be stimulated, and the cell cycle progresses with the damaged DNA. So that damaged DNA will then be passed along to the next cell in that cell lineage, and you see increased cell division. Okay, so now that you're armed with information about the cell cycle and how it's regulated, we can investigate this in the laboratory, and hopefully some of you have done this in the classroom. Different instructors do different types of versions of this lab. I'm going to share with you the one that I do with my students. The objective of this lab is to root onions in different environments and then determine the number of cells that are dividing. So you're going to look and see how many of them are undergoing mitosis, and then compare them to cells that remain in interphase, that remain undivided. In this lab, the onions are rooted in 3 different environments. Caffeine, which is thought to slow the cell cycle. And if you think about the old wives' tale about caffeine stunts your growth, that you kind of remember that. And lectin, which is a natural growth accelerator that's found associated with the roots of some plants. So it should increase the cell cycle, and then distilled water is used as a control. So the image there is showing you some different what it looks like when you root those onions in different cell environments. And so we start this with a null hypothesis and an alternative hypothesis, because ultimately we're going to test to see if our data is statistically significant using a chi-square analysis or fitness of it test. So our null hypothesis is that there's no difference in the number of cells in mitosis in any treatment. In other words, caffeine, lectin, and distilled water affect the cell cycle in the same way. An alternative, this is just one alternative, you could have several alternative hypotheses here, but we're going to say that the treatment of lectin will have more cells in mitosis than the control. So we're going to compare lectin to the control, and we're saying that we think there should be more cells that are dividing, which is measured by mitosis, than the control. I wanted to share with you that we really are just going to look at the tip of these onions, and that's because if you look at this diagram, we're looking in this rapidly growing region of this root. This is the root cap, so there's not going to be any cells dividing here. And then up here, these cells are already mature, and so they don't divide anymore. So we're looking here, what is called this zone of elongation and zone of cell division. Okay, really in this area. And so what's going to happen is students will root these onions, and then we will take a tip of the root and do a squash preparation of the onion root tip and look at it under the microscope and try to identify as many cells as we can that are dividing and not dividing. Okay, got that? All right, so this is what it looks like. And of course, there's lots of practice before this with identifying these cells. And really, I don't care if the students are able to identify the different stages of mitosis, only that the cells are dividing. But if you can identify the different stages, then that's awesome. So first of all, cells that are not dividing are in interphase, and they look like this. And the telltale sign is that they've got, nucleoli. This is probably a better example of that, and an intact cell membrane. And you can't really see the chromosomes, right? They're still in that diffuse chromatin form. And then mitosis. And this one, we see a cell in anaphase, and you can clearly see those chromosomes are moving apart from one another. There's a bunch of other examples. You can see a cell here, and you can see a cell here that are dividing. Here's one. This is a prophase. So you can see. So basically, what happens is students make their slides, and then they count up the number of cells they see dividing and not dividing. Okay? So this is some sample data, and this is actually the treatment group. This should say lectin versus the control group. Okay. So I'm going to talk about what we're going to do this data in just a sec. First, I want to introduce you to the formula that is used to calculate chi-square, and this is straight from the AP Biology formulas and equation sheet. X is the Greek letter X. Chi is the Greek letter X. And then we've got sigma, which is also the Greek letter for S. So that's some Greek symbols here. So chi-square is equal to the sum of, which is what sigma stands for, the observed data minus our expected data squared over the expected data. And it is very important that you follow the order of operations here. Okay. When calculating a chi-square value, you need to be aware of something called a critical values table. It has 2 components to it. The first one is called the degrees of freedom, and you can see there we've got 1, 2, 3, 4, 5 degrees of freedom in this chart. They go all the way up to 50. But we're only going to be working with 2 categories, and the degrees of freedom is the number of categories minus 1. And I'm sure that the stat people will have a much better explanation for this, but it restricts the number of choices that you have, the number of categories that you have. Let me give you an example. So when I teach students chi-square, I do an activity with M&Ms, and I have all the different colors. I believe there's 6 colors. And I'll go to the first student, I'll say choose an M&M, and they may choose a green one. Go to the next student, choose another one, they choose a blue one, and so forth. And if I come down to the last student, there's only 1 color left, and they don't have a choice. So that's the minus 1 part of it. Okay, so the degree of freedom is we have 2 categories, interphase and mitosis. Minus 1 gives us a degree of freedom of 1. Now the probability that we're going to use is 0.05, and we use this because our kind of bar of goodness of fit is 95%. So we're going to be 95% certain of the whether or not we can accept our null hypothesis. So over here is a table for working out your chi-square. What we're going to do is we get our observed data straight from our data table, and then we have to calculate our expected cells. We don't really know how many to expect, so we have to base that on our control group data. I'm going to show you how to do that in a minute. And we go ahead and calculate our chi-square, and we compare it to our critical value, and that will allow us to either reject or fail to reject our null hypothesis. And the reason that we do this is, or how we decide this, is that the chi-square value, if it is greater than the critical value, we have to reject our null hypothesis. There is a difference. If we fail to reject our null, it's because our chi-square value is less than our critical value, and then we are going to basically fail to reject is accept our null. There is no difference in our observed and expected data. All right, so let's go through that. So first of all, students have counted in their control group, which is rooted in distilled water, 186 cells in interphase and 14 in mitosis for a total of 200 cells. And in the lectin treatment group, they have counted 146 cells in interphase and 54 in mitosis out of those 200 cells. So how do we figure out our expected results? So to do that, you're going to calculate the percentage of cells in interphase and in mitosis. So first of all, how do you do that? You would take 186 divided by 200, and then that percentage or frequency is then multiplied by the percentages of the total number of cells in the treated group. So when we look at that, we see that we've got 186 cells out of 200, which gives us a frequency of 0.93, and we multiply that by the total number of cells in the lectin group in interphase, and we can see that we get an expected value of 136. Let's do that again for the mitotic cells, right? So we had in the control 14 out of 200, which gives us 0.07, and then we need to multiply that by the total number of cells in the treated group, which is 54, and then we get 26. So then we have to do our O minus E squared. So we see 186 minus 136 squared divided by 136, and that gives us a value of 18.3. We repeat that for the mitotic cells, and we get a value of 30.1. So remember that the chi-square value is the sum of both of those, or all of those. You could have more categories, but in this case we only have 2, and so our chi-square value is 48.4. Now we need to compare that to the critical value. So the critical value is 3.85, and we look for the intersection of that 1 degree of freedom and a probability of 0.05 when we get our critical value. So now we can decide what to do. We are going to reject our null hypothesis because this chi-square value is greater than the critical value. What that means is the lectin treatment has more cells in mitosis than we expected. What it means for the cell or the experiment is that lectin has an effect on cell division. It increases it. Okay, so go back and try the calculation on your own. You can stop it here and work out those details. All right, so let's get some practice with this. A student collected samples of cells from a woody plant during the spring and produced the drawings below. Sample 1 was collected from the woody stem of the plant, and sample 2 was collected from the root tip of the plant. Cells in different steps of mitosis are labeled with letters A, B, C, D, and E in sample 2. This is a 4-part question. So in part 1, you're asked to describe the role of mitosis in the growth of a plant. So why do that plants divide via mitosis. Hopefully, a description would be one that included plant growth occurs when new cells are produced, or that mitosis is a process that produces two genetically identical daughter cells, which can develop into different tissues or divide further, creating that growth. Okay. Part B, you're asked to explain why there are more cells observed in mitosis in the root tip than in the woody stem. And if you were paying attention to that diagram before, we know that only growth of root tips occurs in the tips, and that very little new mitosis or new cell development or new growth occurs in those woody stems once they are formed. Part C is asking you, and it's kind of a weird way of saying it, but it's asking you to refine the model in the space provided for your response by using the letters from 4 of the cell labels of sample 2 to represent the sequential steps of mitosis. All right, so you've got a model. And the model is telling you we go from prophase to metaphase, anaphase to telophase, and now you're asked to refine it by putting in pictures that represent those different stages. So think about prophase. In prophase, the chromosomes are condensing. So notice that you've got cell A, cell B, cell C, cell D, and cell E. Which one looks like prophase to you? So hopefully you recognize that it's E. Metaphase, remember that chromosomes line up in the middle of the cell. That looks like D. In anaphase, the chromosomes are pulled apart to opposite sides of the cell, and that looks like B. And finally, telophase, where you start to see that, in this case, in a plant cell, you start to see the beginning of a cell plate, which would develop into a cell wall, and that looks like cell C. So now we've refined that model. And the last part of this is to explain how the arrangement of cellular components during the step of mitosis portrayed by cell D in sample 2 facilitates the proper distribution of chromosomes in the 2 daughter cells. So let's go back and look at cell D. Cell D is metaphase. So they are asking you to describe what is going on in metaphase to make sure you get the correct distribution of chromosomes in those new daughter cells. So hopefully remember our conversation about the spindle microtubules. And what happens is that we need to make sure we discuss the fact that those chromosomes are lining up across the equator and that the spindle fibers enable the 2 chromatids of each chromosome to move to opposite ends of the cell before the cell divides. And that ensures the correct complement of chromosomes in each new cell. Let's do one more practice. This has to do with regulation. So we're looking at a diagram of the role of P53 in regulating the cell cycle progression in response to DNA damage. And this looks a lot like the example I gave you. So it says that DNA is damaged that stimulates those protein kinases, and it looks like it's going to P53, and P53 leaves the nucleus. Well, P53 doesn't leave the nucleus. That it activates something on the DNA to produce that cyclin-dependent kinase, the CDK inhibitor. Okay. So you are asked first. This is a two-part question. That a skin cell completes 1 round of the cell cycle. Describe the products. Okay. So it doesn't really have to do with the figure yet. So 1 round, what's going to happen? The cell divides, and you get 2 diploid cells that are identical, 2 genetically identical sets of DNA in each of those 2 diploid cells. In part B, you're asked to explain how P53 regulates the cell cycle in the presence of damaged DNA. So if there's damage and you have P53, what's going to happen? Hopefully, you know that the cell cycle is stopped, and that's because P53 stimulates the expression of that CDK inhibitor, and that halts the cell cycle so that you don't continue to make cells that have damaged DNA. Oh, this actually has more parts. Sorry, I forgot about this part. So now you're asked to draw an X on the template in the space provided for your response to indicate the phase during which replication of the damaged DNA would occur. So which phase does DNA replication or duplication take place? S is correct. All right, and then finally, in part D, based on figure 1, explain how mutation to the P53 may lead to an increased risk of cancer. So now you don't have P53. So what's going to happen? It can't make that CDK inhibitor, and the cell cycle will continue, and it will progress through the cycle with that damaged or mutated DNA, and that is going to continue without breaks. And basically, cancer is mitosis without breaks. All right, so quick check for understanding. Again, this has to do with predicting the cause or effect of change or disruption to a component of a biological system, and this is based on the concept of cell signaling and regulation of the cell cycle. So you've got figure 1, MDM2 and P53 signaling in response to DNA damage controls cell cycle progression. The P53 and MDM2 proteins are involved in DNA damage response pathway that's shown above. Oh, and by the way, as you're looking at that, these mean a negative or inhibit. This is positive or stimulates. Okay, so in the absence of DNA damage, the MDM2 mediates the degradation of P53. In the presence of DNA damage, P53 is not broken down and forms active tetramers that stimulate the expression of genes that block cell cycle progression. Red blood cells are produced by the division and differentiation of multipotent stem cells. Researchers have observed that mice with a partial loss of function mutation in the MDM2 gene have a lower concentration of red blood cells in the blood than do mice that express the wild type MDM2 allele. 2 task verbs here. Predict how a loss of function mutation in the P53 gene is most likely to affect the cell cycle in response to DNA damage. Make that prediction and then justify your prediction. So again, we don't have P53. What's going to happen to the cell cycle? Make a prediction and then justify that. Without P53, what happens? Okay, so the cell cycle will proceed. We just said that without P53, we can't make the inhibitor. Cell cycle goes. And the justification is that it cannot regulate the cell cycle. You could be that blanket statement, or you could be more detailed. All right, so your task verb in that last part was justify. So remember that justification means give a good reason for something or show it to be right. All right, so here's your debrief. You should now be able to describe the events that occur in the cell cycle. You should be able to explain how mitosis results in the transmission of chromosomes from one generation to the next. You should be able to describe the role of checkpoints in regulating the cell cycle and describe the effects of disruptions to the cell cycle on a cell or organism. You should also be able to do a chi-square analysis and to do science skill B, which is to predict the cause or effect of a disruption to a event that's happening within the cell or organism. Your key takeaway for this topic is that heritable information provides the continuity of life. All right, let's take a look at some common errors and misunderstandings, and then I'm going to give you your homework. So first of all, I'm going to give you an FRQ, and this was given years ago at on an AP exam, so you may have seen this by looking at practice questions. Here's the question. It asks you to give a statement that the cell cycle is fundamental to the reproduction of eukaryotic cells, and then it asks you to describe how the cell cycle is regulated and then discuss one consequence of abnormal regulation. Okay, so the scoring guidelines are divided into regulation and consequence. So for regulation, there needed to be a description of the checkpoints and make sure that all 3 of those checkpoints were discussed. In your description, the action of MPF and CDKs needs to be discussed in relation to checkpoint regulation. You also needed to talk about contact inhibition. Those are external factors, that's independent inhibition, and then hormones or growth factors. I gave you the example of the platelet-derived growth factor. So all of those things needed to be discussed. In terms of the consequence, there would be a couple of different things to key in on. One, of course, is cancer. That there would be, if the cell cycle is not regulated, that the mitosis cannot stop and the cells continually divide, as seen in cancer. Apoptosis, which is programmed cell death, is another result of abnormal cell cycle control. And then the third thing to talk about would be anything that happens as a result of chromosomes not getting to the right place, right? So you can have too many chromosomes or too few chromosomes. So you have polyploidy or aneuploidy or non-disjunction where the chromosomes fail to separate. And we'll talk more about that tomorrow when we talk about meiosis, right? So here's what happened. Here's the common errors. Students have difficulty explaining the action of MPF and what CDKs do. So go back and look at both of those things. Students often omitted discussion of contact inhibition. So you need to talk about those external factors as well as internal factors. And also students fail to discuss growth factors in the regulation of cell cycle. So those last 2 are external factors. The first one are the internal checkpoint systems. Okay. All right. So some practice. This is homework tonight. It's pretty easy breezy because I don't want to spend a whole lot of time with the warm up tomorrow because tomorrow we're going to be doing meiosis and we have a lot to talk about there. So I'm just asking you to identify the 3 stages, major stages of interphase, describe the sequence in which they occur, and you should be thinking about what happens in each of those. And if you want to get fancy, you can even talk about where those checkpoints occur in interphase. Okay. So that's homework. Do tomorrow. I have a couple of reminders for you. So I have an exam prep checklist for you. The first thing is device and connectivity. So you need to have a device to take your exam on. The exam again is on Monday, May 18th at 2 p.m. Eastern Standard Time. So you need to make sure you have a device by that time. You also need to make sure you have reliable internet. Both of these things you can be helped by the College Board. If you need help with either a device or connectivity, please reach out directly at cb.org/tech, and you need to do that by this Friday, April 24th. Also, make sure that the College Board has your correct email address, and you can do this by checking at your profile on AP Central. And then finally, let your AP Bio teacher in on your plans, whether or not you plan to take the test or not take the test, just so that gives them an idea of how many students are actually going to be sitting for the exam, and also if you need any additional review for the exam. Okay, so thanks for watching today. I hope you'll join us again. Be sure to complete that practice question. It's an easy one tonight, and we'll review the answer in tomorrow's lesson, which will be on meiosis. So I'll see you guys tomorrow. Thanks for watching. Hi, welcome to today's AP Biology lesson. I'm Allison Kate, and today's lesson is on meiosis and how it generates genetic diversity. So we will be looking at topic 5.1 and 5.2 from the course and exam description, and also 2 science skills, Science Practice 1B, which is explain biological concepts and or processes, and Science Practice 3A, which is to identify or pose a testable question based on observation, data, or a model. And we're also going to be looking at the meiosis part of Lab Investigation 7 from the AP Biology Lab Manual. You should be familiar with concepts of cell division. If you tuned in yesterday, we focused on mitosis and the cell cycle. You should also be familiar with the terms gametes, haploid, diploid, and variation. By the end of today's lesson, you will be able to explain how meiosis results in the transmission of chromosomes from one generation to the next. You should be able to describe similarities and differences between the phases and outcomes of mitosis and meiosis, and explain how the process of meiosis generates genetic diversity. So go ahead and grab a pen and paper. We're going to start with our warm-up, which hang on, let me tell you that before I get into the warm-up, I do need to make a correction from yesterday's lesson on mitosis. And first of all, I want to give a shout out to a student who emailed me from Florida. Thank you very much for pointing out there is an inconsistency in the math. And then also thank you to all my colleagues in the AP Biology community who pointed out some misunderstandings. And so what I'm going to do is I'm going to do this in 2 phases. The first phase is I want to correct my method for calculating the observed and expected values. Now, I put this together in haste, so hopefully everything is okay here. But what I did is I went to the AP Biology Lab Manual and I screenshotted the how to do the observed and expected values, and then how to go ahead and apply this to your chi-square calculation. And so you can see that this is from the same onion data, and we've got a control and treated data and then total number of cells. And I actually think the best thing for you guys to do would be to actually go through this and look at it. But I want to just point out how, for one of them, then you can practice doing the other ones, how you get this expected value for the cells that are in the control or distilled water treatment and the cells that are not divided. So if you look at how to do that, it tells you here that we're going to take a value in cell A, which if our sample data is here, so that's 148. We're going to add that to the value in cell B, which is here, and then we're going to multiply that by cell A, 148, added to cell C, which is 161, just below it. So you've got A, B, C, and D, and these are your total number of cells. Okay, so if you go through and put all that into your calculator, add it up, divide by the total number of cells, you're going to come out with a value of 126.6, which is rounded up, use whole numbers when doing your chi-square, and you're going to get 127. And so that is how that expected value is obtained. Go ahead and practice with control mitosis, the treated interphase, and the treated mitosis. Okay. On Friday, when I'm with you again, I'm going to actually have the math set up for you and apply it to the data that I gave you in the lab portion of yesterday's presentation. I was madly working on it this morning, but could not pull it off in time for today's lesson. So stay tuned. All right, so let's go ahead with our warm-up. And if you have been following along with us, our warm-up is always the homework from the previous lesson. So yesterday's lesson was on the cell cycle and mitosis, and I simply asked you to identify the 3 major stages of interphase and then describe the sequence in which they occur. So go ahead and jot these down. If you have your homework, go ahead and get it so we can compare to the scoring guidelines. So this seems a little bit repetitive, but if you're going to identify those 3 stages of interphase, you simply need to say G1, S, and G2. Those are the 3 phases of interphase, and that was worth 1 point each. So you can see this was a pretty easy question. The second part is to describe. And by the way, that identification needed to be in order, so you had to go G1, S, G2. And then the description was also worth 1 point, which is that the cell undergoes generalized growth. Then in the S phase, we synthesize or replicate the DNA, and then the second growth phase is specifically to prepare for mitosis. So that's the description. So that total scoring guideline was worth 4 points. I hope you guys got all 4. So what do you need to know today about meiosis? And keep in mind that one of the outcomes or the things that we want you guys to understand about this is, first of all, what the process of meiosis is, how it contributes to genetic diversity, and then how it is similar yet different from mitosis. So mitosis is a process that ensures the formation of haploid gamete cells in sexually reproducing diploid organisms. It results in daughter cells that have half the number of chromosomes of the parent cells. Meiosis involves 2 rounds of sequential steps. They're called meiosis 1 and meiosis 2. But there are some similarities that we want you to notice with mitosis. So like mitosis, meiosis is preceded by replication of chromosomes. So again, that occurs in the S phase of the cell cycle. So that is representing our duplicated pair of chromosomes. So we've got pairs from our diploid parents, a maternal pair and a paternal pair, and we can say that these are from the paternal side and these from the maternal side. Meiosis takes place in 2 sets of cell divisions called meiosis 1 and meiosis 2, and we're going to go into the events of these in just a moment. The 2 cell divisions result in 4 daughter cells rather than 2 daughter cells of mitosis. So the end result of mitosis is 2 genetically identical daughter cells. In meiosis, you end up with 4 daughter cells, and they are haploid. They have half the number of chromosomes as the parent cell. So here's a side-by-side comparison. If you look at mitosis, and we're looking at a cell that is diploid, and it has a diploid number of chromosomes as 6. So we recall from yesterday that in prophase, the chromosomes will condense into their familiar chromosome form. These are the duplicated chromosomes. Remember, they duplicated in the S phase. And then those chromosomes line up in metaphase, and then they are separated in anaphase. So you have identical diploid daughter cells at the end of mitosis. If we look at meiosis, there are some other things that we're going to talk about in just a moment. But in prophase, what you're going to see here is homologous chromosomes are pairing up, and they line up in metaphase 1. And then at the end of metaphase 1, we have 2 cells with homologous chromosomes. In meiosis 2, those 2 cells divide again, so we end up with 4 cells when the sister chromatids separate. The other thing you should notice, if you look at the colors of the haploid chromosomes that are in each of those, the number of chromosomes that are in each of those haploid cells, they are different. They are different from each other, and they're different from the parent cells that they arose from. So those are some of the key side-by-side comparisons of mitosis and meiosis. Here's a review chart that kind of just puts it all in one place. So if you want to just do a compare contrast, the parent cells are the same chromosome number in both types of cell division. We begin with diploid cells in mitosis and diploid cells in meiosis. The number of DNA replications is the same. DNA only is going to replicate once in interphase and the S phase in both types of cell division. In mitosis, there's a single round of cell division, whereas in meiosis, there are 2 rounds of cell division, meiosis 1 and meiosis 2. In mitosis, there are 2 daughter cells that are produced, whereas in meiosis, there are 4. The chromosome number of the new cells that are produced is the same as the parent. In mitosis, remember, we want genetically identical cells, and then in meiosis, we reduce the chromosome number by half in those 4 cells that are produced. The purpose, as we talked about yesterday, mitosis is for generalized growth, cell repair, and cell reproduction. For meiosis, we want to produce gametes, and in doing so, we want to ensure there's genetic variation in those gametes. So I'm going to take a quick trip through the stages of meiosis. Again, it's really not crucial that you know the exact events that occur in each stage, but there are some key differences in meiosis that I want to point out. So the first thing that I want to point out in meiosis 1 is we're going to separate homologous chromosomes, and that prophase 1 is very different in meiosis than it is in mitosis. So it's typically the longest stage. It takes up about 90% of the time required for meiosis, and just like in mitosis, the chromosomes do begin to condense. However, these homologous chromosomes, one set from mom, one set from dad, right? We've got the maternal and paternal homologous chromosome pairs. They tend to pair up and twist around one another in a process known as synapsis. And there are 4. So this is called a tetrad, and this will be illustrated in a slide that's coming up. So that's super important, and we're going to talk about the significance of this process in just a moment. Then in metaphase, what happens is those homologous chromosomes line up along the metaphase plate, and just like in mitosis, we have the same apparatus moving those chromosome pairs. We've got our microtubules, which are our kinetochore spindle fibers that are attached to the chromosomes through the kinetochore, and then we have the aster at the end, which is kind of the bracing part of this process, and the non-kinetochore fibers that are not attached to the chromosomes. In anaphase 1, the homologous pairs move to opposite sides of the cell, and then in telophase 1 in cytokinesis, we get those 2 cells, each containing homologous chromosomes. In meiosis 2, it's very similar to what happens in mitosis. Prophase 2 is not like prophase 1. There is no synapsis, there is no exchange of material, and then what happens in metaphase 2, the homologous chromosomes line up in the middle of the cell, and now in anaphase, the chromatids are separated, and so the chromatids move to opposite sides, and you can follow either one of these. It happens in both of the cells that are the result of meiosis 1, and at the end of meiosis 2, you have 4 haploid cells with chromatids or chromosomes that are different from each other and from the parent cells that they came from. Again, it's really important to understand that a similarity is in the mechanism, the spindle mechanism that moves those chromosomes. Okay, so now let's go back and take a look at what happens in prophase 1. So in prophase 1, we've got our non-sister chromatids. The 2 homologous pairs come together in a process known as synapsis, and pieces of that genetic material or gene on a chromosome actually cross over, breaks off, and recombines. The crossover points are known as chiasma. So what you see when you cross over is that the non-sister chromosomes actually exchange segments of DNA. So this is more than one of these crossover points is called a chiasmata, and it just is an X shape is what that chiasma means. So now in anaphase 1, we've got those homologous pairs, and you can see we've got different colors, which represents the exchange of genes or segments of DNA on that chromosome. And then what we'll see in anaphase 2, when those sister chromatids separate, we're going to get different recombinations of those chromosomes. And just a nomenclature, naming thing. The ones that look like the parent, so this one looks like the paternal chromosome, that's called a parental. This one looks like the mother, that's a parental. And then the ones that have different pieces of DNA are the recombinants, because they have pieces of DNA from both of those chromosomes. So that is the major event that occurs that makes meiosis 1 different from mitosis. Prophase 1 is where the action is. It's where that extra genetic variation is introduced from the process of crossing over. So now here's the key idea with the genetic diversity. Sexual reproduction in eukaryotes, which involves gamete formation. It includes crossing over, random assortment of chromosomes during meiosis, and then also random fertilization of gametes. All 3 of these things increase variation, which is the raw material for natural selection. So there are 3 mechanisms that contribute to genetic variation. The first is that independent assortments of chromosomes, and I'll show you that in the next image. Crossing over, which we just talked about, occurs in prophase 1 of meiosis 1, and random fertilization, which sperm cell fertilizes, which A is a completely random process. So here's the possibilities of how these chromosomes can line up. If you look at possibility 1, again, I can call out these colors. We've got 2 homologous pairs. If we say the blue ones are paternal and the red ones are maternal, we've got 2 different sets of chromosomes. So they can line up in one of 2 ways. You can see in the first possibility, we have both of the blue ending up in one of the cells at the end of meiosis 1, and both of the red in the other. So that means that our daughter cells are going to have either all blue or all red. If you look at possibility 2, those cells line up. You've got a blue and a red in one cell and a red and blue in the other cell. And so when those daughter cells are formed, they're going to have a different complement of those chromosomes. So it's important to note that the way that those pairs line up is completely random in metaphase 1 of meiosis. And then in independent assortment, each pair of chromosomes sorts maternal and paternal homologs into daughter cells independently of the other pairs. So it is completely random the way they line up, and they assort independently of other chromosomes. So these numbers are meant to awe and amaze you. So the number of possible combinations when chromosomes assort independently into gametes is 2 to the nth power, where n is a haploid number. So for humans like us, we have 46 chromosomes in a diploid cell, which means we have 23 in a haploid cell or gamete. So that means if you go 2 to the 23rd power, there are over 8 million possible combinations of chromosomes. Then random fertilization adds to genetic variation because any sperm can fuse with any egg. So the fusion of those 2 gametes, each with about 8.4 million possible chromosome combinations from independent assortment, produces a zygote with any of about 70 trillion diploid combinations. Crossing over adds even more variation, and that is why no other human looks exactly like you. Each zygote has a unique identity. All right, so now hopefully armed with that information about meiosis and how meiosis contributes to genetic diversity, we're going to take a look at the second part of Lab 7, which investigates meiosis. And the objective of this lab is to observe the arrangement of ascospores in the fungus Sordaria fimicola. The spore arrangements in this particular fungus, which is an ascomycete fungus, which means that it produces its spores in sacs or asci, corresponds to the crossing over events that occurred during meiosis when the spores were formed. And hopefully, as we go through this, that will make more sense. So one of our skills is to be able to pose a testable question. So for this particular investigation, a testable question might be, how does the arrangement of spores show if crossing over has occurred during spore formation in Sordaria fimicola? So what happens in this lab is that students will gather evidence to support the claim that the farther the gene for spore color is from the centromere, the greater the number of crossovers will be observed. And now I have a couple of images for you. So what happens in this lab is we propagate this fungus on a petri dish. This fungus turns out comes in 2 varieties. There's a wild type variety that produces black spores, and then there's a mutant variety that produces tan spores. And we culture them together so that we allow the black spore filaments to reach out to the tan spore filaments, and they actually get together, they exchange their genetic material and reproduce spores. So you get a diploid zygote. I know this is small. I'll show you another image in just a moment. And then they reproduce their spores by meiosis. And then what happens? So these are the 4 products of meiosis. Okay. Then these spores actually undergo 1 round of mitosis. So they create genetically identical copies of these spores. So let me just point out that if I've got this black spore, it is going to produce another copy of a black spore. So in this case, I have 2 products of meiosis, 2 spores that are black. They're going to each divide. So I'm going to end up with 4 black spores. And the same thing will be true of the tan spores. In this arrangement, I have 2 tan spores. They're going to divide. So I'm going to end up with 4 tan spores. So the overall result of that reproductive event is that I'm going to get 8 total spores. They're haploid, and 4 of them are black and 4 of them are tan. So this is just a drawing showing you how that the ascus, this is the sac, and the ascospores line up. And this whole area here is the fruiting body or the perithekium. And in the lab, the students are challenged to open up this perithekium in a way that keeps those ascospores in groups of 8. So in other words, you have to use really a gentle technique. You don't want to spread them out like that. So we want to keep them. I always say they kind of look like strands of pearls. We want to keep them in that intact pattern. So here's a little bit more about the process. So you can see that we've got our zygote, and we've got the genes for the spore color on those homologous chromosomes. DNA replicates, and now we've got a duplicated copy of both that tan chromosome and black chromosome. We're going to look at two possibilities, one where no crossing over occurs, and then one where crossing over occurs. And over here you can see that this is going to be meiosis 1 and meiosis 2, and then the products of mitosis. So if no crossing over occurs, those tan chromosomes and black chromosomes end up in different cells at the end of meiosis 1, and then they will separate into unique gametes at the end of meiosis 2. So you can see we've got gametes that produce tan spores and black spores. When they divide by mitosis, they create a spore pattern of 4 tan and 4 black. Okay? If we look at crossing over, we can see in this image that pieces of the black spore gene are actually going to cross over onto that tan spore chromosome, and so that's the result. Same thing happens with a tan chromosome. We get a little bit of the gene for tan spore color on that black chromosome. And then when they do their first round of division, we can see that they create two different cells. That when they divide by meiosis 2, we end up with an all tan, a tan with a black tip, which is going to produce a black spore because that's where the gene for spore color is. This is tan. This one has a tan gene, and this one has a black gene. So now we have a different pattern, and it looks like this. We get 2 after mitosis, 2 tan, 2 black, 2 tan, 2 black. And because of independent assortment, it could also be 2 black, 2 tan, 2 black, 2 tan. So the way to remember this is that a 4 to 4 arrangement is no crossing over, and anything else shows that crossing over occurred. And this is just to show you in real life what those two different phenotypes look like. All right, so now let's take a look at the analysis. So we prepare these spore slides by easing those ascospores out of the perithekium, and in no crossing over, we're going to get a 4 to 4 pattern. And if you look, I have an example here, you can see that there are 4 black and 4 tan. So that's no crossover. Any other possibility is crossing over. And so I gave you the 2, 2, 2, 2, but you could also have 2 tan, 4 black, 2 tan, or vice versa. So anything other than 4, 4 is going to give you a crossover pattern. And you can see here's an example of this. So 2, 4, 2. And so students go about counting and recording their results. And this is a data table from the lab manual, and you record the number of ASCII. It's a really good idea to count up to 50. More is better because you're going to get closer to the expected value. And so in this case, we can see that students have counted 24 non-crossovers and then 26 crossovers. So then what you can see is we've got, we want to calculate the recombination frequency. So that's how many actually crossed over out of the total number of ASCII. And so that's 26 out of 50, or 52%. Now remember that to figure out where that gene to centromere distance is, and remember that we are trying to find data to support the claim that the farther away that gene is from the centromere, the more crossovers will occur. We need to divide this by 2. And the reason we need to divide it by 2 is that remember that there is a mitotic division that occurs. So map units, which are kind of just arbitrary units to show you where that gene for spore color is on that chromosome, we need to divide that by 2. So we can see that the calculated gene to centromere distance is about 26 map units. Okay, so you can also remember that the recombination frequency tells us how far this should be far the gene for spore color is from the centromere. Data that is close to 26 map units will support the claim that farther the gene for spore color is from the centromere, the greater the crossover frequency. You can also do a chi-square goodness of fit test for this data. This one is a little bit different because we know that our expected value, if we count 50, should be 50% crossover, no crossover. So you can just go ahead and do your O minus E calculation for chi-square that we went over yesterday. From the critical value table, we know that we use probability of 0.5. We still have a degree of freedom of 1 because there are 2 categories, crossover, no crossover. Degree of freedom is the number of categories minus 1. And so our critical value from that critical value table is 3.85. Now we remember that if our chi-square value is less than our critical value, we're going to accept the null hypothesis. And in this case, we're going to fail to reject the null, which means accept. And that means that there's no difference between the observed and our expected results. And we were spot on with map units of 26 map units. Okay. So now let's look at some FRQs. So this first FRQ is a meiosis FRQ. And I'm going to go over how we get this answer in the scoring guidelines because this is one of those common misunderstandings that students have. So we've got a cell, we've got chromosome 1 and chromosome 2, and it tells us that in a certain species of plant, the diploid number of chromosomes is 4. So we've got 2n equals 4. Flower color is controlled by a single gene in which the green allele, which is represented, it's dominant, right? So it's got the capital G, it's dominant to the purple allele, which is going to be lowercase g. Plant height is controlled by a different gene in which the dwarf allele, capital D, is dominant to the tall allele, little d. Individuals of the parental generation, which is abbreviated P generation, with the genotypes big G, big G, big D, big D, and little g, little g, little d, little d, were crossed to produce F1 progeny. Construct a diagram below in the template to depict the 4 possible normal products of meiosis that would be produced by the F1 progeny. Show the chromosomes and the alleles they carry. Assume the genes are located on different chromosomes and that the gene for flower color is on chromosome 1. So going back up to chromosome 1, this is going to be the color. So it's either going to be big G or little g, and this is for height. So it's either going to be big D or little d. Now remember, you have to figure out the first cross. So you got your F1 progeny, then you have to figure out their gametes. And this is always a challenge for students. So I want to give you a minute to think about it, and then let's go ahead and look at the scoring guidelines. So if you look at the scoring guidelines, I've labeled these gametes. We've got gamete 1, gamete 2, gamete 3, and gamete 4. And you need to have those particular gamete combinations. And I'm going to show you how you get it. So the diagram must include unduplicated chromosomes, so a single chromosome with either a capital G or lowercase g, and one unduplicated chromosome for the height allele. So how do we figure this out, right? So if the parental generation is crossed to get the F1 generation, they are heterozygous at each trait. And that's because, let's call this mom, she only has the dominant allele for both color. So this is going to be green and dwarf. And then dad is going to be purple and tall. And because they only have those alleles, they're going to be heterozygous at each trait for each trait. And so this would be a green dwarf F1. But now we need to figure out what the F1 gametes are going to be. And you're going to borrow something that you probably first learned in math class. You're going to use FOIL. So the first 2 are going to be big G, big D, and that's gamete 1. Your second 2 or outer 2 is going to be big G, little d, and that's gamete 3. Inner 2 is going to be big G, little g, big D, and the last 2 is little g, little d, and that's gamete 4. So make sure you follow that. That's the only way you can determine how those gametes are going to end up. Okay, another practice with diversity. So a research team has genetically engineered a strain of fruit flies to eliminate errors during DNA replication. The team claims that this will eliminate genetic variation in the engineered flies. A second research team claims that eliminating errors during DNA replication will not entirely eliminate genetic variation in the engineered flies. Describe one mechanism that could lead to genetic variation in the engineered strain of flies. Okay, so I gave you the top 3, but there's actually more. So you only need to pick one mechanism. So think about that, and let's look at the scoring guidelines. Any one of these bullet points would earn you that point. So sexual reproduction produces offspring with new combinations of alleles or traits. Fruit flies have fewer chromosomes than humans, so it's not 70 trillion possible combinations, but it's still many, many new combinations. Crossing over produces even more new combinations of alleles and traits. Independent assortment produces new combinations of alleles or traits. Random fertilization produces new combinations of alleles or traits. So those are the big take-home lessons from today's presentation. But you could also imagine that immigration or gene flow could introduce new alleles into this genetically engineered fly population. There could be a viral infection that inserts DNA into the fly genome. Non-disjunction, which is the failure of chromosomes to separate properly, could cause an anomaly in chromosome number. In other words, you end up with too many or too few. Chromosomal rearrangements could inactivate genes or result in multiple copies of genes, so things like duplications or deletions. And then there could also be some type of a mutation that could be introduced into the DNA. So any one of those mechanisms would introduce genetic variation. And remember, this is a key idea for meiosis, that it introduces genetic variation, which is the raw material for natural selection to act on. All right, so let's do a check for understanding with skill 1B, which is to explain biological concepts and or processes. So this is a former FRQ from a former exam, and it is going to actually give you a compare contrast type thing between the two types of cell division that we've discussed yesterday and today. Both mitosis and meiosis are forms of cell division that produce daughter cells containing genetic information from the parent cell. You are first asked to describe two events that are common to both mitosis and meiosis that ensure the resulting daughter cells inherit the appropriate number of chromosomes. Think about how they get moved around. And then in B, the genetic composition of daughter cells produced by mitosis differs from that of the daughter cells that are produced by meiosis. And then describe two features of cell division that leads to these differences. I want to remind you something before you try to answer this. Remember this? So I showed you this before. This is kind of the nuts and bolts or the compare contrast slide. So I want you to think about that as you begin to answer this question. Okay. So scoring guidelines for part A, you need two events. So each bullet point is 1 point and it's 2 points maximum. So remember, how do the chromosomes move? Is that spindle apparatus. The microtubules will attach to the chromosomes and move them in both meiosis and mitosis. In both cases, in prophase 1 of meiosis and in prophase of mitosis, the chromatin condenses into chromosomes. We have in both types of division the alignment of chromosomes across the center of the cell in metaphase before they separate in anaphase, and the separation of those chromosomes and chromatids into the daughter cells occurs both in mitosis and in meiosis too. We regulate the cell cycle the same in both types of cell division. So that checkpoint regulation occurs. I explained it in mitosis yesterday, but it occurs in meiosis as well. The replication or synthesis of DNA occurs in the S phase in both types of division, and cytokinesis occurs to separate those daughter cells after either mitosis or meiosis. And then in part B, there's that table, right? So two features of the cell division process that lead to these differences. For mitosis, there's a single round of division with 2 cells resulting. For meiosis, there's 2 rounds of division with 4 cells resulting. For mitosis, the chromosome number is the same as the parent cell. Remember, genetically identical cells, so it's diploid to diploid. And then in meiosis, we have that reduction division. So we reduce the chromosome number by half. So in your gametes, you have 23 chromosomes compared to the precursor cells that they arise from, which is 46. Chromatids separate in mitosis, and they only separate in mitosis 2 because it's the homologous chromosomes that are separating in meiosis 1. Crossing over only occurs in meiosis, and crossing and homologous chromosomes assort independently in meiosis. Okay, so hopefully you got all 4 points on that one. Okay, so here's a sample answer that you can take a look at. That was a stellar answer. The separation of sister chromatids in both mitosis and meiosis 2 ensure that each daughter cell receives the appropriate number of chromosomes. Also, the lining up of chromosomes along the middle of the cell ensures the chromosomes will separate properly. And then in 4B, unlike mitosis, meiosis results in 4 haploid cells rather than 2 diploid cells because cell division occurs twice to produce gametes. Also, the chromosomes in the cells that have undergone meiosis are recombinants of each other because of the synapsing that occurs in prophase 1. And this happens unlike the synapsing, or synapsis does not occur in mitosis. So you can see we don't take points off for a new use of a new form of the word synapse. Okay, and so that was a perfect 4 score. All right, so your task verb this time was describe, and so you want to paint a picture using words. You want to describe it as if someone cannot see it. So here's your debrief. Your learning objectives. You should now be able to explain how meiosis results in the transmission of chromosomes from one generation to the next. You should be able to describe similarities and differences between the phases and outcomes of mitosis and meiosis. You should be able to explain how the process of meiosis generates genetic diversity. Those big 3. You should be able to explain biological concepts and processes, and you should be able to identify or pose a testable question based on an observation, data, or a model. And again, just like yesterday's key takeaway, heritable information provides the continuity of life. So just some common errors and misunderstandings. I told you about how students have a difficult time finding the gametes of the F1 progeny. Remember FOIL, okay? So the good news is that students were able to articulate correct information about both meiosis and mitosis. The bad news is that students equated mitosis and meiosis with the entire cell cycle, not just nuclear division. So keep that in mind, that is just a very small portion of the cell cycle. Many students were unclear about the events that occur specifically during the nuclear divisions. Many identify chromosome replication or DNA synthesis is happening during mitosis or meiosis, and I hope that you've noticed that I have stressed both yesterday and today that synthesis or replication of chromosomes happens prior to nuclear division. Many students fail to identify spindle fiber formation as a distinct event in mitosis or meiosis, and remember that spindle fiber apparatus is how the chromosomes are moved during the process of nuclear division. While many students were aware that checkpoints exist, they were not clear as to their purpose or why they occur. Many students erroneously identified homologous chromosomes as lining up during mitosis or mitosis 2 rather than meiosis 1, as is the case. And students often confuse haploid cells and diploid cells. So try to keep all of those things straight when thinking about these cell division processes. And I know that they're similar. I try to teach them completely separate. So first you learn mitosis, go on to some other things, and then come back to meiosis so that you're not confusing them. So that may be a strategy to keep them straight. All right, so here's your homework, and this will be debriefed tomorrow as your warm up. And tomorrow you'll be with Ms. Pfizer. She's going to be talking about Mendelian Genetics. So that will kind of solidify some of these events of meiosis for you. Meiosis occurs in many organisms as part of sexual reproduction. Describe 3 differences between mitosis and meiosis. And remember, if you need to, you can go back and look at that review slide. So I also want to end by reminding you that you have a to-do list, your exam prep checklist. The exam is going to be on Monday, May 18th at 2 p.m. Eastern Standard Time. At that time, you're going to need to have a device to take the exam on. You're also going to need to have internet connectivity. If you need help either of these things, please reach out to collegeboard@cb.org/tech. And you need to do this by this Friday, April 24th. You also need to be certain to check that the College Board has your correct email address. They're going to be emailing you your exam ticket. You will also be able to access your exam ticket from your student side of AP Central. So there'll be a couple of ways to get it. If for some reason they miss your email address, they send it to the wrong email, or you have a proxy wall that prevents you from receiving that email. And then finally, please let your AP Bio teacher in on your plan whether you're going to test or not test. No judgment. Just want to know who's going to be testing. And finally, we will be doing our last live session will be on Friday, May 1st. The exam isn't until May 18th, but AP exams do start the week of May 11th. So in that gap week, we are going to be doing some exam prep things. We're going to have some practice questions for you. We'll debrief those practice questions with you, and also we'll make sure that you have some practice understanding how to upload your responses. So we hope you'll join us for that week as well. So thank you so much for watching. Make sure you do your practice question. That will be your warm up tomorrow. And as I said, your next lesson will be with Ms. Pfizer, looking at Mendelian genetics. Have a great rest of your day. And one more thing, I will have a corrected chi-square analysis for you from the lab investigation 7 when I see you again on Friday. All right, have a great day. Thanks for joining.
Welcome to AP Biology. I'm Gail Pfizer from Darnell Cookman Middle High School and Florida State College in Jacksonville, Florida. Lesson overview for today. We'll be talking about topics 5.3, which is Mendelian genetics, and unit 5, heredity. And go ahead and grab your paper, pen, and calculator because you're going to need it today. By the end of this lesson, you'll be able to explain how shared, conserved, fundamental processes and features support the concept of common ancestry for all organisms. Explain the inheritance of genes and traits as described by Mendel's laws. Perform chi-square hypothesis testing and use data to evaluate a hypothesis or prediction, including rejecting or failing to reject the null hypothesis. All right, for our warm up. So this is homework from yesterday with Ms. Kate from Meiosis. If you are just joining us for the first time, go ahead and I'll give you a few moments to jot down your answers. answers. Meiosis occurs in many organisms as part of sexual reproduction. Describe 3 differences. They're going to describe 3 differences between mitosis and meiosis. I'm going to get the clock running. Okay. All right. I think you had time to jot down a few ideas. Okay. So there were many ways to approach this. If your description was about the parent cell to the daughter cell, then you possibly said a 2N to 2N in mitosis and 2N to 1N in meiosis. Or perhaps you approached it from genetic results. Daughter cells genetically identical to the parent cell. Daughter cells not genetically identical to the parent cell. Or a number of daughter cells, 2 in mitosis, 4 in meiosis. Number of divisions, 1 in mitosis, 2 in meiosis. Or you could talk about the mechanics. Homologs don't pair. Homologs pair. No synapsis. Chromatid split at anaphase. Chromatid split at anaphase 2. No crossing over. Or maybe you talked about the purpose. Repair, growth, asexual reproduction, or meiosis forming gametes. But whatever you did, you had to describe the process in vogue. If you said something about mitosis, you had to say something about meiosis. Okay. If you had just went into a description of prophase and metaphase and anaphase and telophase, that would not have earned any points. Okay. So what you need to know. All right. So, let me just go ahead and do this part and then I'll tell you the other story. All right. The following 4 features support common ancestry. DNA and RNA are carriers of genetic information. Okay. There's our first one. Ribosomes are found in all forms of life. Major features of the genetic code are shared, by all modern living systems, and core metabolic pathways are conserved across all currently recognized domains. So if you remember, we had glycolysis and fermentation, and so it didn't matter if you had oxygen or not, that there was fermentation going on. We didn't need the mitochondria to do so. So these share conserved fundamental processes and features support the concept of common ancestry for all organisms. Okay, so years ago when I was in a genetics class, I had to take the 1866 paper of Gregor Mendel and rewrite it. Take his paper, it was translated in English, but take his paper and then write a modern day research journal article in proper form. So I thought, what would I ever do with that again? And then look, here we go. I have it out today. So in his paper, and I'm just talking about 3 of the 7 traits here. We had flower color, flower position, stem length. There were dominant traits, which was a violet color, axial flower position, tall stems. White was recessive, terminal was recessive, so was a dwarf stem. But notice that this was his F1 cross. So in his F1 cross, there are 100% of each were dominant. So the parents of this F1 cross, so our parental group, was true breeding. Okay, so this would make true breeding organisms. Okay, so we're going to do this. We have P's and a little line under that for lowercase. Anyway, so when he crossed a true breeding dominant with a true breeding recessive, then he got his F1 cross. And if you notice, you know what's going to happen, right? That every single offspring will be dominant. Okay, now the next table is referring to his F2 crosses. So now he took the offspring of this F1, okay, which we're now going to breed those, and then that's going to give us our F2. So you know where this is going also. Okay, so what we had was, well, let's look at his numbers first. Okay, so with dominant and with these hybrids breeding, he had a 3-to-1 ratio in each of these 3 traits. He did all 7 traits, but here we had the dominant Here we had the dominant phenotype and the recessive phenotype. So there's that 3-to-1. Now this next table has to do with he only had offspring from the dominant phenotype. Okay, he didn't put any recessive into the group, and then he had a 2-to-1 in his ratio. And if you'll look, here's 2 and here's 1. Okay, all right. So enough of my old tables. So as a quick vocabulary, he used the words characters and heritable features. We now call that gene, and then different traits. So variant traits are now alleles. So a gene can have different alleles. Okay, which means different variations of the trait, like the white flower or the purple flower. But specifically today, we're just going to talk about dominant and recessive. Okay, now dominant determines the appearance. Recessive is not going to have any noticeable effect if dominant is present. We had talked about hybrid already. That's cross of 2 true breeding, and that true breeding produces only offspring the same as the parent. Okay, all right. So you're very familiar with this. I'm sure we have a homozygous dominant. I'm going to change to D's. Okay, because the P's are a little bit more difficult to keep up with. All right, so now we have 2 alleles that are dominant, and then we have a heterozygous, and then we have a homozygous recessive. I know you know that already. Okay, so now let's talk about Mendel's law of segregation. So I want you to imagine that. Okay, I'm going to draw this cell. We're in a cell, and right this moment we're at we're at metaphase in meiosis 1. So the homologous pairs are lined up together in the metaphasial plate. That's where we are, and we have a dominant and a recessive trait. Okay, now when we have anaphase, what's going to happen? The D and the big D and the little d are then going to separate, or they're going to do what? They're going to segregate. So now they're separating, and this ends up in a cell, and this ends up in a cell. And in the future, if there is a, I don't have any water sitting here with me. I forgot about it. All right, so here we have if we are fertilized with another one that has a little d, then we're going to have this little d is going to be able to be expressed, and so that's the match right here. Oops, sorry, that's going to match this guy right here. Okay, if we are able to get away from the big D. So that's the law of segregation. They're separated from each other. Okay, I know that you're familiar with a monohybrid cross. We have a hybrid here. We have a dominant and a recessive, and now we have our homozygous dominant, our 2 heterozygous, and then our recessive. So there is our 3-to-1, and this is what allows that 3-to-1 ratio to happen. The law of segregation. It allows this moment to happen. Okay, all right. So rules of probability can be applied to analyze passage of single gene traits from parent to offspring. Mendel in his paper, he actually had this uppercase A, let's say big A plus 2 big A and little a plus little a represents all the characters of this organism. And so as we talk about the laws of probability, we're going to talk about how we figure out the probability of having what, besides just having this 3-to-1 number in our heads. So if we have a situation where we're talking, if we have A or B, we're going to use plus. Okay, and if we have A and B, we're going to use multiply. And the best way for me to teach you to do probability and heredity math is to do math. Okay, so we're going to start off with an easy one. We have 5 children, all sons. If the woman gives birth to a 6th child, what is the probability that the 6th child will be a son? So if we started off with this is the possibility of being a girl, and this is the possibility of being a boy. So then what's the probability of being a boy? It's half of these, right? Okay, so that's just, I know, easy breezy, just getting us started. Okay, so now probability that the genotype CCDD will be produced by parents big C, little d, little d, 2 of those, 2 dihybrid, 2 dihybrids. Okay, so here I took this right here, and now I have a C and a C and a D and a D. So if you look at it like this, what's the probability of getting a little c from this combination? It's half, right? 50%. And what's the probability of getting a little d from this combination? Is a half. And this c from this would be a half. And this d from this combination would be a half. So if you multiply all these together, you have 2 times 4 times 8 times 2, 16. So the probability is 1/16 that you could get this from these 2 parents. Okay, one more. All right, achondroplastic dwarfism is a dominant genetic trait that causes severe malformation of skeleton. Homozygotes for this condition are spontaneously aborted. Hence, the homozygous condition is lethal. But heterozygotes will develop to be dwarfed. So I already put this in to save some time. We have, if you have homozygous dominant, you're dead. Okay, if you have big A little a, then dwarf. If you have a little a, then you're not going to express any of the traits. Okay, all right. So Matthew has a family history of the condition, although he does not express the trait. So Matthew, here's Matthew right here. This is what it looks like if you don't have a trait. Jane is an achondroplastic dwarf. Okay, here's Jane. So she's going to look like this, but she can't look like that. All right, Matthew and Jane are planning a family of several children and want to know the chances of producing a child with achondroplastic dwarfism. So if 3 children are born to Matthew and Jane, so here's child 1, child 2, child 3. What are the chances that the first 2 children will not express the trait, but the 3rd child will be achondroplastic dwarf? So if we have a little a, okay, so what was the chance of getting getting a little a from Matthew? It was 100%, right? What was the chance of getting a little a from Jane? It was 1/2. A little a, so the same combination. And then we'll have to get this big A from Jane. So what's the possibility, probability? 1/2. And then we have a little a that we have to get from Matthew, 100. So wait a minute, that's up. Here's those two, and then that one. Okay, 2 times 2 is 4, and 4 times 2 is what? Is 8. I think I did that right. Let's see. I did it right? Yes, I did. Okay, good. All right, so I got confused on how many A's. I have A's everywhere. All right, so you would keep this one because it's totally 100% if you had a question about that. So if we wanted these 3 kids, that's and. That's when they have to be multiplied. All right. Okay, so now Mendel's law of independent assortment can be applied to genes that are on different chromosomes. So now I want you to imagine again, we're in a cell, and we are lined up in the middle at the metaphase 1 of meiosis 1, and we're at the metaphasial plate, and we're going to then pull to the side and split into 2 cells. So this would end up in a cell together, and this would end up in a cell together. So this dominant and this dominant would be together, and this recessive and this recessive would be together. Would this have to be like this? No, this does not have to be like this. This dominant R could be over here. Okay, if this is a different cell that we have and a different option. Okay, so now this is totally random. This is the independent assortment. These chromosomes can assort independently of each other. They do not have to stay together. So now I have the dominant trait of this one with the recessive trait of the other gene. So now this would be in a new cell, and then this would be in a new cell at the end of meiosis. So they would be totally different options, independently assorted. Okay, so let's do a dihybrid cross, and then I'll tie it together with independent assortment. So if we have two traits and 2 hybrids, okay, for each trait. So let's find out what our possible genotypes are going to be. So we can have a big D, big A, right? And we can have a big D, little a. We can have a little d, big A, and we can have a little d, little a. And since those were the same, okay, we can just repeat that. Okay, so now we have to just check. So this was, so I make sure that you know how this works. So here's a D, here's a big D, there's big D, little a, little a, little a, little a. Okay, so I already did this so we could for the sake of time. And now let's count. There's 1, 2, 3, 4, 5, 6, 7, 8, 9. There's 9 that have dominant trait. There's 1, 2, 3 that have a dominant and then a recessive trait. And then there's 1, 2, 3 that have a recessive and then dominant trait, and then 1 that has recessive. So this dihybrid cross, if you have a dihybrid cross, you will always have a 9-3-3-1 combination, and it will be because of this law of independent assortment will allow this to happen. Okay. Now, and fertilization involves the fusion of 2 haploid gametes, restoring the diploid number of chromosomes and increasing genetic variation in populations by creating new combinations of alleles in the zygote. So this is a meiosis drawing that I did for my class, and I use it every year. But let's start off talking about this right here. Oops, I gotta move something. I was practicing earlier. Okay. All right. So if we start off with this diploid, we have 3 homologous pairs of chromosomes. We got one from mom, one from dad for each of these. Now, and we're gonna go through meiosis. So when we have interphase, here's one from mom, here's one from dad, and replication occurs. Still 3 pairs of chromosomes. Now, if you'll look closely, now there's some variation that has happened on each one. So that was from crossing over. So we can say this variation happened here, and then they're going to align in the middle. And then we have, does the yellow one have to be there? No, it does not. It could be on this side, or this red could be on this side, and the orange over there. So independent assortment happens. So there's more variation. So 2 places genetic variation occurs. Anaphase happens, 2 cells, and here we are at our next 2 cells. About to go through the next phase. So here's our 3 cells, and then they're going to line up in the middle, and then they're going to be separated during anaphase 2. The sister chromatids are, and then we have 4 genetically different. If you look at them, each one of them is totally different than another. So if you had, when fertilization occurs, if you had another very diverse, genetically diverse from their parents' offspring, think about these 2 starting off at this location and then going through this process. So genetic variation just continues to increase and increase through each generation. All right, so I'm going to run over this just a little bit. I think I'll be doing some more of it tomorrow, but so let's just get some groundwork done. So you have sex-linked inheritance, okay, and we'll work with this a little bit in the some questions. So let's do hemophilia, okay, all right, and that is sex-linked, and that means that it's going to come from the mom's X, okay. So let's do, there's female, and let's use here's a male, and let's use a uppercase H for normal, okay, a normal allele, and then we'll use a little h for the disease allele, so a mutated allele. So let's do this, and then we have, all right, so our 2 girls, we have both 2 normal girls, okay, they don't express, this one doesn't express any of this trait because she has a gene that will produce cells of proteins correctly, okay, and so but she is a carrier, okay, as in this mom was a carrier. Now on the boys we have a normal allele, and then now we have the mutated allele, okay, and the only proteins that can be made from this gene are the one with the mutation. So there's not any other normal proteins to be made because we don't have that uppercase H here, okay. So that's how it's sex linked, and that's how it affects males. Does it have to be 100% just males? No, we could spend some time and go through different scenarios and come up with situations where we could end up with a female. Okay, so now let's talk about pedigrees, and I'm going to do pedigrees just like I did math. We're going to learn by doing, okay. So the pattern of inheritance can often be predicted from data, including pedigree, that give the parent genotype phenotype and the offspring genotype phenotype. So let's read what it says first. In the pedigree above, squares represent males, so this is going to be for male, and circles represent females. Individuals who express a particular trait are represented with shaded, okay, so this will be they have the trait. Now, which of the following patterns of inheritance best explains the transmission of the trait? So let's go through some questions that you would ask yourself about this diagram, and then you'll be able to answer it. First, are males and females equally getting this trait? And we see that yes is the answer to that. So we can automatically say this is not sex linked, okay, and we have total, you either have the trait or you don't, so we're not going to do incomplete dominant. And now we have an autosomal recessive and an autosomal dominant. So next question is, do we see it in each generation? Yes, we do. And do we see it from any carrier parents? Okay, so do we have anything that looks like that? No, we don't. Okay, so that makes our conclusion it's going to be not recessive, it's going to be dominant. And the word autosomal is referring to body chromosome, like pair. In us, it would be pair 1 through 22, and then we would have our sex chromosomes, which would be pair 23. So that's where this word autosomal is, means body chromosome. All right, so let's do another one. So let's take a look at the pedigree above. Circles represent females, squares represent males, and shaded figures represent individuals expressing a specific trait. So consistent with that generally. All right, the expression of this trait is most likely due to which of the following. So we have, all right, so let's look at our boys and girls. Do we have boys and girls getting this? Yes, we do. So let's get rid of those 2. All right, do we, let's see, let's either you do or you don't. So we'll get rid of that. All right, and then do we have carrier parents having children with the trait? Yes, we do. There's a situation right here. There's one right there. Here is one right here. So now this is autosomal recessive inheritance. Okay, here is a question about calico cats. This is my calico cat. Her name's Callie, and a student of mine, I'll give a shout out to Amber, gave me this little precious thing when she was 4 weeks old. All right, a gene that influences coat color in domestic cats is located on the X chromosome. A female cat that is heterozygous for the gene, so this is what a calico looks like, has a calico colored coat. In a genetics experiment, researchers made a calico colored female with an orange colored male. Okay, so this is an orange male. To produce an F1 generation, the researchers record observations for the cats in F1 generation and plan to use the data to form a chi-square goodness of fit test for a model of X-linked inheritance. The data for the chi-square goodness of fit test are expressed in the table. Okay, so let's look at this. He expected an equal number of each. Let's go ahead and determine why he expected that. Okay, but I just don't want to take it for granted. So there's a calico option. There's a calico option. There's an orange girl. So calico girl, black male, orange male. Everybody's different, right? So you have one here, one equal distribution. So that's where this 10 number in each of these came from. Now the researchers calculate a chi-square value of 4.6 and choose a significance level of p equals 0.05. Which of the following statements best completes the chi-square goodness of fit test. So that gave us, we didn't have to do the math, although I did the math on the next page so you can see it. All right, so here's our, what do we have? We have 4 categories. So our degree of freedom, remember, is our categories minus 1, so our category is going to be 3. And then we have a 4.6 is our chi-square value, and it is less than, so see we have 11.34, 7.81, so 4.6 is less than our critical value right here. And here's our, so it's heading in this direction. And so our p-value, there's p.01, there's p-value 0.05, and so this is going to be greater than 0.05. So now we know about our probability. It's greater than 0.05. So what we could go ahead and write when we know that p is greater than 0.5, we can say that we are not going to reject our null. We can say this is not statistically significant, and we can say that any variation is due to chance. When I'm writing about a null, or I'm writing about the null and a p-value, I write all of those things to make sure that I have everything I need. So now let's compare it to these answers. The null hypothesis can be rejected. Did we say it can be rejected? No, we said do not reject. The null hypothesis can be rejected. No, the null hypothesis cannot be rejected. Okay, I'm good with that, but it's there too. Because the chi-square value is greater than the critical value. Is it greater? Is it this way? No. So D, the null hypothesis cannot be rejected because the chi-square value is less than critical value. Okay, all right. And so in case you were interested, I went ahead and did this math because the first time I practiced this lesson, it was 2 hours long, and I know my director person would have had this, she would have counts. Anyway, I went ahead and worked out this math, and so here are the expected values. Here's the formula. So expected values I inserted, and then the observed values, and I did the math, came up with the 4.6. So I agree with this, what this question came up with. Okay, and there we go. And D was the answer. Okay, so among others, the sum of science skills that we're going over today: perform chi-square hypothesis testing, use data to evaluate a hypothesis or prediction, including rejecting or failing to reject the null hypothesis. Some task groups we're going to use today: construct, predict, determine, and explain. All right, so I know you've seen this question, but not in this particular context. This question has like 4 different parts to it, and so we're between this container we're getting our bang out of this buck. Okay, so in certain species of plant, the diploid number of chromosomes is 4. So 2 is, so I said diploid 4, 2 equals 4. Flower color is controlled by a single gene in which the green is dominant and the purple allele is recessive. Okay, plant height is controlled by a different gene in which a dwarf allele D is dominant and a tall allele is little d. All right, individuals of the parental P generation with genotypes, here we have a true breeding, okay, and true breeding for 2 traits were crossed to produce an F1. So what's going to happen if we cross those? We already know that if we cross those, that we're going to have this. Okay, we're going to have a excuse me, a big G has to be given from this parent, and a little G has to be given from that parent. A big D has to be given from that parent, and a little D has to be given from that parent. So our F1s will look like this. Now predict the possible phenotypes and the ratios in the offspring of a test cross between F1 individual and a recessive individual. I did this part of this question because I wanted to go over test cross with you. Okay, so if we have these are our parents, we're going to have this as one parent, and then we're going to test cross it with this. So I'm going to build a little box, not 16 squares this time because I don't need them. So what's the only option for offspring I have here? It's going to be a little g, right? And so here I have big G, big D, big G, little d, big D, and little g, little d. Okay, so when they are when they're crossed, we have this. I think I did that right. Okay, so we end up with a green and tall, green and not tall, and purple and tall, and purple and not tall. Okay, now we have 1 of each, so we have a 1 to 1 to 1 to 1 ratio. So when you have a hybrid, and you're test crossing it, this is always going to be your ratio when you have test cross from this with this with this. Okay, all right, blank page with the So here's our score guideline. We had 1 green dwarf, 1 green tall, 1 purple dwarf, and 1 purple tall. All right, so we're going to go through. I did a lot of the parts in advance on this because it's quite lengthy. So in fruit flies, the phenotype for eye color is determined by a certain locus. E indicates the dominant allele, and little e indicates the recessive allele. The cross between a male wild type fruit fly, wild type means normal, and a female white-eyed fruit fly produce the following offspring. So we ended up with 45 wild type females and 55 white-eyed males, and some odd thing happened there. Okay, the wild type and the white-eyed individuals from the F1 generation were then cross to produce the following offspring. So we started off with a parental group that we don't know. Okay, and then we have our F1, and now we have F2. So the first thing that it wants us to do is determine the genotypes of the original parents. Okay, so to do this, this is what I had to do. I had to kind of work this backwards. So if I have let's go ahead and put males and female, and I'm following this up here. Okay, I got this. All right, so now this was a wild type male, so let's use a big E. It's easier for us to keep up with. And we have a wild type female, but we don't really know something, do we? We don't know if she is this or if she is that, right? A white-eyed male, so we know he has a little e, and then a white-eyed female, but she would have to have both. So if we worked this backwards, then we could put our boys in, and let's get our other boy. All right, so if we know we have a Y here, what would have to be here? This would have to be here. This would have to be here. Okay, now let's put our girls in. If this is here, okay, then this would have to be here. Okay, so these are those offspring. So their parents, these right here. Okay, so now we're moving these. Wild type female has to be this, and this white-eyed male has to be this. Okay, now we need to know their parents. What had to happen to make that happen? We could work it backwards, and let's put in both boys look the same, right? There's half and half. So here's our girl. All right, so if we have a Y here, we have to have a what? We have to have an X little e, and to have an X little e there, to have an X little e here, then we would have and here we have to have this. So this is our parents, and there may have been 3 different easier ways to do what I just did, but I want you to see where it's coming from. Now the rest of the question will be, you'll be glad that I did all that, and explain your reasoning. You may use Punnett squares to enhance your description, but the results from the Punnett squares must be discussed in your answer. So this alone would not suffice as an answer. Okay, you would have to go through all those words that I just said to get to this point. Those would have to be in your explanation of how this happened. Now, what's our next part? Use a chi-square test on the F2 generation data to analyze your prediction of the parental genotypes. So we're going to use F2 information. Okay, and all these are different. All these different. Yes, they are. Okay, and then we show all our work. So listen, I'm going to flip to the next page, and that's where we're going to do our chi-square. I have all of this there also. Okay, so if we now remember we had in the Punnett square we had equal probability of getting each. So if we had 100 of the flies, then we can expect 25 of each. So here we have 25 in all of our expected locations, right? And did the math, and it ends up to be our chi-square value is 2. Now, what else would we say? Now that we know our chi-square value is 2, we have how many categories? 1, 2, 3, 4 categories. So our degree of freedom is 3. We have a chi-square value that is less than our 7.81 critical value. Okay, so it's heading that way. Our p-value is greater than 0.05. So we can say the same thing again. Our p is greater than 0.05. We're not going to reject the null. This is not statistically significant. This is any variation is due to chance. Okay, I sat in on a thesis defense today of a student that was mine, and I heard her saying most of all these words that have stuck along with it, been included in college and master's program. All right, so now we have, did we say all these things? Did we come up with the boy being X, big E, Y, and that being the girl? Is that what we said? Yes. Okay, right. Now, did you discuss? I told you to discuss. Like all these words I was saying out loud, those would need to be written out. You can support it with your Punnett square, but you can't just have a Punnett square in that situation. Now, did we talk about sex-linked, X-linked genes? No, we didn't talk about that. And we did it, but we didn't say words out loud. And we talked about what was dominant. And then in the explain portion, we came up with a 1 to 1 to 1. We did the math, which was a 2. And then we talked about all the right things to say. Do not reject the null. You could also say the null hypothesis is supported. It's not my go-to language. This is, and then what else? Anything else we could have said? All right, and do the chance. Okay. All right. Now, debrief and summary. So take away for today. I hope you feel comfortable with what the law of segregation, independent assortment, probability, with what those mean. Okay, and how to use probability and how to use the math of probability. I hope you feel comfortable with reading a pedigree, asking yourself those elimination questions to try to figure out what you're looking at. Does it affect boys and girls? Is it each generation because of parents that are carriers? And if it's sex linked, is only boys being affected? You should also be able to explain how shared, conserved, fundamental processes and features support the concept of common ancestry for all organisms. Explain the inheritance of genes and traits as described by Mendel's laws. Perform chi-square hypothesis testing. Use data to evaluate a hypothesis or prediction, including rejecting or failing to reject the null hypothesis. And this right here, and I've seen it happen in my classroom, that's why I grabbed this. This is not the end all be all of all ratios. Okay, it's not the answer to everything. So if you're talking about a test cross, remember with our scenario, then it is there's a test cross. And why would we need to do a test cross? To get to what kind of which organisms are homozygous dominant and which are heterozygous dominant, because they both have the dominant trait. Now in probability, remember if it says or, you're going to add them. I could find a good example and a horrible test question writer. You would want that to happen. And so here's and so we're going to multiply them. All right. So for homework tonight, this will be sort of as your warm up on tomorrow night's lesson on non-Mendelian inheritance. Blue flowered African violet of unknown ancestry, self-pollinated and produce 50 seeds. The seeds germinate and grow into flowering plants. Of these plants, 36 produce blue flowers and 14 produce pink flowers. What is the best explanation for the pink flowered offspring? Now you have, I think this is an easy question. A, blue flowers are incompletely dominant to pink. B, pink flower color is recessive to blue flower color. C, pink flower color is a result of somatic mutations in the flower color gene. And D, a previous generation of the blue flower parent I think this should be must have included 50% pink flower plants. Now use these numbers, okay, to figure out the answer to this question. All right, and then we have a few more nights of saying this. You're gonna do this by the 24th. That's tomorrow. Device and internet access. If you don't have a device, if you don't have internet, please reach out to cb.org/check your to-do list. I'm gonna add as we go right here. Okay, so device and internet. Okay, check. You should do this. Make sure your email is correct in your College Board account. And you might have put it there when you're in the 9th grade, and you might not be in the 9th grade right now. All right, and tell your teacher you're testing. I've asked all of mine. I have been able to get a hold of a couple of them. Check your phone number also. That was probably put in when you were very young. Also, so check that. Now, if your test ticket is going to be emailed to you, but it's also going to be in your AP Classroom account. Okay, so everybody can breathe again about that. It'll be there. If it's not, it'll be both places. Okay, all right. So thanks for watching. I hope you'll join us again. Tomorrow will be Miss Katay, and she'll be reviewing non-Mendelian inheritance patterns. And that's it. Not 2 hours. What we do. 45 minutes. Bye.
Hi, welcome to today's AP Biology lesson. I'm Allison Katay, and before we get going with today's topic, I want to go back, and I promised you the last time we were together that I would give you a correction for some techniques and math errors when I originally presented this chi-square analysis that is used with the Mitosis and onion root tips lab during the presentation on Tuesday. So first of all, I presented this slide to you on Wednesday, and this is just the screenshots of the AP Biology lab manual in lab 7 that shows you the technique of how to calculate the expected data from your observed data. And I originally used a different technique, but I decided I wanted to go back and do it according to the instructions in the lab manual so that you guys can actually go back and have a resource. So what I'd like to do today is apply this to the lab data that I gave you in that original presentation. So you might recall that this was student-generated data, which is observed data for the treatment group of lectin. And what you're looking at is the control group in interphase and mitosis, and I have gone back and labeled those cells A and B, and then you're also looking at interphase and mitosis for the lectin-treated group, and I've labeled those cells C and D. And this way you can go back and look at the formulas that are presented in the lab manual. I should also point out that it doesn't affect the end result and the evaluation of the chi-square value. So that's it. So this is how you calculate your expected results based on the instructions in the lab manual. So the formula, I have TI, which is the treatment group for interphase, and TM, which is the treatment group for mitosis. So for the treatment group in interphase, you're going to take C plus D, and then you're going to multiply that to A plus C, and then you're going to divide that by the total number of cells, which would be A plus D plus A plus D plus C plus D. And then you're going to, for the mitosis treatment group, use cells C plus D and multiply that to B plus D, and again, divided by the total number of cells. So let's just take our data and plug in the numbers. So for the treatment group in interphase, we're going to take 146 and add that to 54. We're going to multiply that to the sum of 186 plus 146 and divide that by 400. And then for the mitosis treatment, it's 146 plus 54 multiplied by 14 plus 54 over 400, or the total number of cells. And again, we get those numbers, and we end up for the number of cells in the lectin-treated group that are in interphase is 166, and the number of cells that are in the in mitosis in the lectin-treated group is 34. Okay, so now we're going to go ahead and look at the chi-square analysis table. So our observed values in interphase from the lectin-treated group is 146, and for mitosis it's 54. Our expected cells are what we just calculated, so 166 for interphase and 34 for mitosis. And then we just go ahead and plug those into our chi-square formula: observed minus expected squared over over expected, and the same thing for mitosis: 54 minus 34 squared over 34. If we add the sum of those two results, we're going to get a chi-square value of 11.7. Then we have to recall that we need to compare that to a critical value that we derive from a critical value table in order to evaluate. In other words, to say what that value tells us. So from the critical values table, we're going to be using a p-value of 0.05, which means that we're going to be 95% of our result, and that it is not attributed to chance alone. Our degrees of freedom are the number of categories minus 1. So we have two categories, interphase and mitosis, minus 1. So our degree of freedom is 1. And if we look at our critical value tables, it tells us that our critical value is 3.85. So now we're going to compare our chi-square value, which is greater than our critical value. And that means that we are going to reject the null hypothesis. In other words, our null hypothesis is that there is no difference between the cells in mitosis in any group, right? So there is a difference, so we're going to reject that. And what it actually means for the experiment is that the lectin treatment has more cells in mitosis than we expected. Lectin has an effect on cell division. It increases it, okay? And that was our alternative hypothesis, which can be confirmed with an accuracy of 95%. So I hope this clears that up. Please feel free to go back. And again, thank you to the students and teachers who pointed this out. All right, so today's lesson is on non-Mendelian genetics. We're going to look at two science practices. We're going to practice with concept explanation and also performing mathematical calculations. And today, they're mostly doing probability calculations and recombinant frequencies to determine map units. So from yesterday, you should be familiar with Mendelian inheritance patterns. You should also be familiar with autosomes versus sex chromosomes. Today, I'm going to use some terms: pleiotropy, epistasis, polygenic inheritance, and multiple alleles. And these all have to do with the non-Mendelian genetic inheritance patterns. By the end of today, you should be able to explain. I just made up a new task for. You should be able to explain deviations from Mendel's model of the inheritance traits. So first, we're going to go over our warm-up, which is just a multiple choice question, but it really gives you some practice thinking about how Mendelian inheritance patterns work. So we have a blue-flowered African violet of unknown ancestry. So we don't know if it is going to be homozygous, dominant, heterozygous, or homozygous recessive. And it's self-pollinated and produces 50 seeds. The seeds germinate and grow into flowering plants. Of these plants, 36 produce blue flowers and 14 produce pink flower. What is the best explanation for the pink flowered offspring? So this was your homework from last night from Ms. Pfizer, which is Mendelian inheritance. And I want you to think about the Mendelian ratios that you learned about yesterday, and then think about that math, because what Mendel was really helpful in doing is providing that mathematical analysis to his observation. So your choices for this are blue flowers are incompletely dominant to pink flowers, and we're going to talk about incomplete dominance today. Pink flower color is recessive to blue flower color. Pink flower color is the result of somatic mutations in the flower color gene, and a previous generation of blue flower parent, I should say must have included 50% pink flowers. Okay, so think about what the ratio of 36 to 14 is in Mendelian speak. Okay, so here are the scoring guidelines. The correct answer is of course B. Pink flower color is recessive to blue flower color, and sometimes it's a little bit easier to see this if you actually draw out the Punnett square. And so in this case, that blue flower was heterozygous, and so when it combined to produce offspring, it produced 3 to 1, 3 blue to 1 pink, and that's a rough 3 to 1 ratio. Okay, so more practice with this as we go through today. All right, so here's what you need to know. And I know that Ms. Pfizer talked about sex-linked inheritance yesterday, but I want to go over a few other things with regard to sex-linked inheritance patterns, which technically could be placed either in Mendelian or non-Mendelian, but in our course and exam description, they are included in section 5.4, which are non-Mendelian traits. So some traits are determined by genes on sex chromosomes and are known as sex-linked traits. The pattern of inheritance of sex-linked traits can be predicted from data, including pedigrees, which I'm going to share with you in just a moment, which indicate the parent genotype and phenotypes, as well as the offspring's genotypes and phenotypes. So the image that you're looking at on the left is an X chromosome and a Y chromosome, and I really want you to see the difference in size. The X chromosome is much larger than the Y chromosome, and so now if you look at the image on the right, you can see that this is not exhaustive, but there are many traits that are carried on the human X chromosome. Compared to the human Y chromosome, there are really only a few traits that are carried on the human Y chromosome. Excuse me. And that is probably the most important one on the Y chromosome is the SRY gene, and that determines during embryonic development, it will determine the male sex characteristics. Excuse me. So most of the sex-linked traits are going to be carried on the X chromosome, and so when we talk about sex-linked traits, we're really talking about X-linked traits. So I want to share with you some ways to visually represent these inheritance patterns, and I'm first going to show you Punnett square, and then I'm going to show you a pedigree, which are both visual representations of these concepts. So I have 3 different Punnett squares for you, and remember that for an X-linked trait to be expressed, that is for you to actually see the phenotype, a female needs 2 copies, and we call that being homozygous, where a male only needs 1. We call it being hemizygous, which is kind of the new terminology because really they only have 1 X chromosome, so the gene that's carried on that X chromosome is called hemizygous. And again, you're going to see why X-linked recessive disorders are much more common in males than females, and that's because males only have that 1 X chromosome. So females, because they have 2 X chromosomes, they can, if they have a dominant allele on the other X chromosome, it will actually not be, the trait will not be expressed in the heterozygous condition. So in Punnett square number 1, we see the female, which has 2 X chromosomes. By the way, in terms of nomenclature, we usually do these crosses with the X chromosomes, and then we show the trait as a superscript up here. So in the first Punnett square, the first example, we see a female who is not affected. She has 2 X chromosomes with the dominant alleles, whereas the male is affected. He has 1 X chromosome and it has the recessive allele. So that shaded dark shade means they have a condition. Then we can see as the gametes are put across the top, and there's the male gametes and the eggs along the side. When we combine those, we can see that we're going to get all the females, the XX females, are going to be carriers. They're shaded lightly. That means they have 1 dominant allele and 1 recessive allele. And then we have the males, and since they only have 1 X chromosome, they got it from their mom, and they have the normal allele. In the second Punnett square, we see, excuse me again, allergies here in California. We see that the female is a carrier and the male has a normal allele. And in this case, we are going to see that there is a 50-50 chance of the boys getting the X chromosome that has the recessive allele as well as the dominant allele. And then finally, in the third case, if the female is a carrier and the male is afflicted, then we can get that's the only way we can get a daughter that would have the condition, because she would inherit the X with the recessive allele from her dad, and then the X, she has a 50-50 chance of getting that from her mom. Okay, so those are the 3 different possibilities for what happens if you're going to see an X-linked trait being passed down. There are some human disorders that are X-linked traits. Color blindness, there are some other ways to get color blindness, but it's mostly through this X-linked inheritance pattern. Also a certain type of muscular dystrophy and hemophilia, which is a blood clotting disorder, and I'm going to use that as my pedigree example as well. So I'm going to do this both ways. I'm going to do it as a Punnett square and a pedigree so you can see the difference. So hemophilia is a sex-linked recessive trait. A female is the carrier for hemophilia and marries a normal male. So we've got our male gametes, he's normal, so he's got the dominant H. Our female's a carrier, so she's got a dominant H and recessive H. So when we do the cross, we're going to get a normal female, a carrier female, a normal male, and an affected male. And so what's going to happen now is we're going to get some questions, and then I'm going to bring this back to the pedigree. So you might be asked what fraction of the female children will have hemophilia, and it's really important to read that question carefully because they're asking about the female children, not the male children, not all the children, but the female children. So how much fraction of the female children? None. 0% will actually have the disease. What fraction of the female children will be carrier? You look, 50% will be carriers and 50 will be normal. What fraction of the male children will have normal blood? 50%. And what fraction will have hemophilia? 50%. Okay, so again, you can do the Punnett square as usual. I always say draw it because it really makes it clear. So now if we're going to look at a pedigree, which is a really common visual representation of these genetic inheritance patterns, we can see, and this is from the royal family, this is exactly like my Punnett square. Queen Victoria was a carrier, so she's half shaded. By the way, let me do a key first. Males are shown as squares, females are shown as circles, and affected male is shaded, and affected female is shaded. They're not showing you here a carrier would be half shaded. A line between the male and female represents mating, and then you can see the offspring with the birth order going from left to right. So Queen Victoria was a carrier, and Albert was unaffected. In their F1, their daughter is unaffected, but their son has hemophilia, and then their next born son does not have it. Remember, 50-50 chance, and their daughter is a carrier like their mom. So again, just really wanted to point out the different ways that you can see these types of problems or questions. You could get it as a Punnett square or a pedigree. All right, so now I'm going to talk about some other inheritance patterns. We just talked about linked genes. Those are sex-linked genes, but genes can also be linked together on the same chromosome. So genes that are adjacent or close to one another on the same chromosome are linked genes. The probability that genetically linked genes will segregate as a unit can be used to calculate the map distance between them. And I did share this with you when we went over the Meiosis lab, but if you didn't get it then, this will be a kind of a review. So what we're looking at is a linkage map of some genes in fruit flies, and we're going to do a lot of practice questions with fruit flies, so I thought this would be a good way to start. You can see the wild type phenotypes are on the bottom, and mostly I want to focus on gray body, red eyes, and normal wings, and then along the top, the mutant phenotypes would be black body, cinnabar, and vestigial wings. And you can see that the distances between the genes are what we call map units, and one map unit represents a 1% recombination frequency. So I'm going to show you how we determine that recombination frequency. So the first thing, and this kind of goes back to the Meiosis lab, there's evidence that supports the claim that the farther apart 2 genes are, the higher the probability that a crossover will occur between them, and therefore have a higher recombination frequency. So the farther apart those genes are on the chromosome, the larger that number. So this is some data from a test cross in flies, and we can see that we have 965 wild type, 944 black vestigial. So we cross the 2 homozygous recessives. But then we see some new phenotypes. We get a gray vestigial, 206, and 185 black black normal. Now the first two we call parentals because they look like the parents in the cross. The second two we call the recombinants because they do not look like the parents that were used in the cross. So to determine the recombination frequency, we take the total number of recombinants, which is 206 plus 185 or 391 recombinants, and we divide that by the total number of offspring, which in this case is 2300. We multiply it by 100 to get a percent, and you can see that the recombination frequency is going to be 17%. That means that the genes for body color and wing length are 17 map units apart. Okay, so let's take a look at some other ways that traits are inherited that are not typical Mendelian inheritance patterns. And I want to talk about a few of them. So this is the overview, and I will end with a review slide. So we're going to talk about the levels of dominance, incomplete dominance, and my examples are going to be in roan cattle and flower color. And I'm showing it another way for nomenclature purposes. And then we've got pleiotropy, where one gene has multiple effects on the phenotype. Sex-linked inheritance, which we just discussed. Non-nuclear inheritance, which means that there are some organelles that have their own DNA, and they can be inherited separately from genomic DNA in the nucleus. Polygenic inheritance means that there are many genes that influence the phenotype. And epistasis, which is when one gene will have an effect on the expression of another gene. Okay? So let's talk about the degrees of dominance that we're going to see in this example. So first of all, complete dominance is the Mendelian inheritance pattern, and it occurs when the phenotypes of the heterozygote and dominant homozygote are identical. So if this was a cross between a red and a white flower, and we would see a heterozygous genotype, it would still be a red phenotype. In incomplete dominance, the phenotype of the F1 hybrids are somewhere in between the two phenotypes of the parental varietals. So in my example, I'm crossing a red snapdragon with a white snapdragon, and in the F1 we get a pink flower. And this is not blending. And the reason that it is not blending is that if you then cross those two F1 pink flowers, we can see that the original phenotypes do appear. So we will get a 1 red to 2 pink to 1 white. And this is going to be different than the 3-to-1 Mendelian pattern. Some examples of this, of course, flower color, roan cattle, and blue chicken. So if you see a question about any of those kinds of examples, I want you to think incomplete dominance. Okay? Remember that the ratio is 1 to 2 to 1 in the F2, not the 3 to 1 that you learned in the Mendelian ratios. And then the last degree of dominance is codominance, where two dominant alleles affect the phenotype in separate and distinguishable ways. And I'm going to give you an example in blood type in just a minute, but just to tell you the difference in flowers, right? If you did snapdragon flowers, you would see equal numbers of red and white flowers. They would be expressed the same on the same plant rather than seeing that pink flower. So I'm going to use blood groups for 2 examples. I'm going to use it for multiple alleles and codominance. And you may or may not know this, but that in human blood type there are actually 3 alleles that code for the different phenotypes that we see in blood typing. So the alleles are IA, IB, and then lowercase I, which will represent the recessive. The IA and IB are co-dominant, which means they will be expressed equally and create a new phenotype. So what's really going on when we talk about these blood types is that on the surface of your red blood cell, you have a gene that will code for a carbohydrate marker. And if you have IA, you are going to have the carbohydrate A on the surface of your red blood cell. If you have the gene IB, you're going to have the carbohydrate B on the surface of your blood cell. If you have both of them, IA and IB, then you'll have a new phenotype, AB, where both of those carbohydrates are expressed equally. And if you're recessive, you don't have either of those carbohydrates. So it's a lack of carbohydrates. I should also go back and tell you that to get blood type A, you can either be homozygous for A or you can be heterozygous AI. Same is true with B. You can be homozygous for B, you can be heterozygous EI. Now, I'm not going to go into detail right now about blood compatibility. That's kind of a different lesson, but you can be thinking about how that might work. So just to review, most genes exist in populations in more than 2 allelic forms. In this case, we have 3 alleles, IA, IB, and lowercase I. And again, the I gene is coding for an enzyme that adds a specific carbohydrate to the surface of the blood cells. And so you're either going to get A, B, both A and B because it's codominant, or none, which means you have blood type O. The next non-Mendelian inheritance pattern I'd like to talk about is called epistasis. That word literally means to stand upon. So in this case, epistasis is going to have 2 different alleles that are going to, one set of alleles is actually going to change the way the phenotype is expressed. The common example that's used is in labs. And if you have a lab, you know that they come in different varieties. We have black labs, chocolate labs, which are brown labs, and then we have yellow labs. And yellow labs can actually be 2 varieties. Their fur color is yellow, but then the other areas that are pigmented, like their nose and their eyes and their paws, may vary in the amount of pigment that they get. So in epistasis, a gene at one locus will alter the phenotypic expression of a gene at a second locus. And we say that one of the genes is epistatic to the other one. And so the 2 genes that we're talking about in labs is that there's a gene that determines the pigment color, which is going to be dominant for black, recessive for brown, and then another gene that we're calling the C gene, which is for color or no color, and that determines how much pigment will actually be deposited in their fur or hair. And so you can see we've got 2. This is a dihybrid cross, and one of the ways that you can tell epistasis is that it is not a 9:3 to 3:1 ratio, which is what we would expect if we were doing a Mendelian dihybrid cross, right? So in this case, it's modified to a 9:7 or a 9:3:4 ratio. So if you see that on an exam question, you'll immediately want to think epistasis. And so you can see that there are going to be in this dihybrid cross 9 dogs that are going to have black fur, and it's either because they're dominant for the black allele, right, like here and here, or they're homozygous they're heterozygous for the black allele. In order to be a chocolate lab, you have to be homozygous recessive for the color allele, for the pigment color allele, not the amount of color. And then in these 3 cases, what is happening? Actually, let me start here. So this is just our recessive, our homozygous recessive, and that's the one that we would expect to be light fur, right? So that would be our 9:3 to 3:1. Now these 3, what is happening is that the amount of pigment, the amount of color, is the homozygous recessive. And even though we would expect it to be brown because we've got the homozygous dominant and heterozygous genes for color, because we get this homozygous recessive in the amount of color, we are going to say that it is epistatic to that brown pigment, and so what we don't see that being expressed. So it actually stands upon the color, and instead of getting a chocolate lab, we are going to get a yellow lab. I hope that makes sense. Okay, so pleiotropy. Pleiotropy is a funny word. Basically, it means that one gene has many different phenotypic effects, and there are many hereditary diseases in humans, such as cystic fibrosis or sickle cell disease, that have this. There's also a weird trait in chickens called the frizzle feather trait, I think, and it affects the defect in the gene affects cartilage. So anywhere that the cartilage is in the chicken, such as in their feathers or in where their bones connect, the ends of bones, the cartilage is affected. So they can't fly. The feathers don't provide any thermoregulation, and they're deformed. And so, that's important in the pecking order chicken. So having this one gene defect affects a lot of the phenotypes for that chicken. So again, pleiotropy is one gene, has many phenotypic effects. And then I think this is my last non-Mendelian inheritance pattern. It is polygenic inheritance. Poly means many, genic is gene. So it is an example of the additive effect of 2 or more genes on a single phenotype. And there's lots of examples of this. It's anything that is not along a trait that's along a continuum, like height or skin color. And in other organisms, it might be things like the seed, number of seeds or seed weight, that kind of stuff. So in this case, many genes, there are 6 alleles that are going to actually be responsible for skin color in humans. And the easiest way for me to explain it is if you were to receive all 6 recessive alleles in this first square, you would have the lightest possible skin color. If you were to receive all 6 of those dominant alleles, then you would have the darkest possible skin color. And then to get this range that we see in human skin color, it could be any of these different possible 64 possible combinations that would give you a different phenotype. And this is just the genetic side. And just to throw in the effective environment. So let's say that you're here and you have all of 6 recessive alleles, you have very light skin color. If you are out in the sun, let's say you take a summer job where you're out, a lifeguard, or you're working on trail restoration, you might be out in the sun and exposed to that ultraviolet radiation, which is going to stimulate your melanin production. And even though genetically you have light skin, the environment can change that color. So genetics is complicated, and it also has an interplay with the environment. So just a quick review of all of the non-Mendelian inheritance patterns that we just talked about. So just to get the range of dominance, complete dominance of one allele over another is the Mendelian inheritance pattern. You have incomplete dominance, which the classic example is seen in flower color. Don't forget your roan cattle or your blue chickens, which are actually gray because the 2 phenotypes are black and white. They just look blue. Co-dominance, where you see both phenotype types expressed equally. Type A, B is our example. Multiple alleles, where we also use blood types as our example, and those alleles are A, B, and I, lowercase I, the 2 dominants and the recessive. And then pleiotropy, where one gene is able to affect many phenotypic characteristics. You can think of sickle cell or that frizzle trait chickens. Epistasis is where the phenotypic expression of one gene affects the other gene. And polygenic inheritance, where a single phenotypic character is affected by 2 or more genes. All right, let's get some information about the last type of non-Mendelian. And this is traits that result from non-nuclear inheritance. And you might recall from previous lessons that both chloroplasts and mitochondria have their own DNA. You might recall that it is very similar to prokaryotic DNA in that it's circular, like the plasmids that are seen in bacteria. And you might want to think about the evolutionary reasoning behind this and the endosymbiotic theory. But basically what happens in the way that mitochondrial DNA in animals and chloroplast and mitochondrial DNA in plants, the way that it is assorted to gametes and daughter cells is through the maternal lineage. And I'm going to give you some examples. But if you look at this picture, you can see that the nuclear, there are 2 copies, and you inherit it from each parent. So the red X maybe came from dad, and the black X came from mom. The mitochondrial DNA, which is cytoplasmic, only comes from the mother. And the reason for this is that when a sperm cell fertilizes an egg, really all it's providing is its nucleus. It doesn't provide any of the cytoplasm. All of the cytoplasm comes from the maternal, the ovum. Okay? So in animals, mitochondria are transmitted by the egg and not the sperm. And so what you see is over here in my pedigree, it's a different way to show a pedigree. If the mom has some type of a trait that is passed down through mitochondrial DNA, and she has that trait, all of her children will get it, because whether you're a girl or a boy, you're going to get an X chromosome from mom, right? Dad doesn't matter because there's no DNA coming from the dad's nucleus. On the other hand, if the dad is not affected and the mom is unaffected, did I say that wrong? The dad is affected and the mom is unaffected, all of her children will also be unaffected. And again, this image shows you a circular piece of DNA from the mitochondria, and most of those genes really have to do with providing proteins that are used in cellular respiration. Same thing is true in chloroplasts, but this is in plants only, right? So they get both mitochondria and chloroplast DNA, which is transmitted in the ovule, which is the female gamete, and not in the pollen, which is the male gamete. So again, this is a nice illustration. The pollen does not have the chloroplast DNA or the mitochondrial DNA, so when it fertilizes, it only brings the genes that it has in its nucleus, whereas the ovule has both chloroplast and mitochondrial DNA in addition to the genes that are in its nucleus. Okay, so let's get some practice with these concepts. The first one, and these I know take a lot of reading. I'm going to go through this, and then I'm going to give you the answer in words, and then I'm going to show you in some diagrams as well. So what you're looking at is a data table of the phenotype of some F2 flies. So let's look at who was crossed. Each student in the class performed a parental cross between a fly that is true breeding for ebony wings and vestigial wings, and a fly that is true breeding for gray and long wings. And I hope you remember that when I showed you those traits, both gray and long are dominant, and ebony and vestigial are recessive. So we've got parentals that are homozygous dominant and homozygous recessive, and that means that our F1 flies are going to be heterozygous for both of those genes. Then the student crossed several pairs of F1 flies and determined the phenotypes of 500 of the resulting F2 flies with respect to body color and wing shape. The students in the class averaged their data for the frequencies of the 4 possible genotypes. And just when I'm going through this, I want you to be thinking about why it's important to have those numbers, and think about Mendel and what his great contribution to the study of genetics was. It was being able to apply mathematics and statistics to the observations that he was seeing in his crosses with plants. So that number 500, those numbers that you see in the data table, ebony body and long wings is 98, ebony body and vestigial wings is 28, gray body and long wings is 293, and gray bodies and vestigial wings is 81. So based on this data, describe why the dominant alleles for body color and wing shape are the alleles that produce gray body and long wings. Okay, and this is 3 parts. And then based on that data, describe why the 2 genes are most likely on different chromosomes or why they are on the same chromosome. So in other words, are they linked or not linked? And then you're asked to calculate the probability of producing flies that have gray bodies and vestigial wings if a cross is performed between one of the F1 flies, okay, so the hybrid, the dihybrid, and the first analysis, and then a fly that is homozygous, which is homozygous dominant for gray body and gray body and vestigial wings is the recessive. So that would be homozygous for the dominant trait and then homozygous for the recessive. Okay, so you're gonna have to work that out, but I'm gonna show you. All right, and then of course you've got our plus and minus 2 standard errors with the means. Okay, so first of all, if we were to do this, we know that if it was a Mendelian inheritance pattern of chromosomes that or of genes that are on separate chromosomes, we should see a 9:3 to 3:1 ratio. And hopefully you looked at those numbers. We have 500, which is a lot, right? And you can kind of see the 9:3 to 3:1 ratios. Okay, and then probability, the probability is going to be 0.5, and the reason for that is the probability of having gray bodies with the parentals in that cross is 1, and the probability of having vestigial wings is 0.5. So if we multiply 1 by 0.5, we're going to get 0.5. Let me show you that in pictures. So here's the cross, right? So we started off with parents that were homozygous recessive for both traits, crossed with homozygous dominant for both traits. So our F1 is a dihybrid. If we go ahead and do that Punnett square and we work out, you can see that the boxed genotypes represent the 293, and that's our 9. The circle genotypes represent our big E little v 81, which is our 3. The triangles represent our little e big V, which is our 98, and then finally our little e little v, which was 28, is 1. So the 9:3 to 3:1 ratio of the F2 phenotypes supports the claim that the gene for body color and wing length are on separate chromosomes. To do the probability calculation, the cross was our F1, right, which was the dihybrid big E little e big V little v, and then the dominant for the gray body type big E, and the vestigial for the wing little v. So if you do them separately, you can obviously do a shortcut, but if you do them separately, I just want to show you how it came out. For the body color, they're all gray, so that probability is 1. For the vestigial, you get half long, half vestigial, so that probability is 0.5, and then the overall probability is 1 times 0.5. Oops, that should be 0.5 right here, sorry, but the correct answer is 0.5. Okay, all right, let's get some practice in plants because plants are important as well. So in plants, genes involved in chlorophyll synthesis are located in both the nucleus and chloroplast. Some variegated plants with both green and white leaves and shoots have mutations that prevent them from synthesizing chlorophyll throughout their leaves. In an effort to understand the genetics underlying chlorophyll synthesis, scientists perform the crosses shown in figure 1. So what I want you to see, this is the outcome of the leaf, right? So you can have green, you can have white, and you can have a combination of green and white, which is called variegated. So in this case, when the ovule is green, the progeny is green. It doesn't matter what the pollen that fertilized that ovule is. It could be green, white, or variegated. The progeny is green. Ovule is white, same deal. We get the progeny is white regardless of the pollen. In this case, when the ovule is variegated, again, it doesn't matter. We're going to get variegated. Okay, so 4-part question. Describe how genetic information is carried in chloroplast and mitochondria. Okay, got that? We know that chloroplast and mitochondria contain their own genetic material. B, explain how the inheritance pattern that's shown in figure 1 supports the role for chloroplast supports a role for chloroplast genes in the production of chlorophyll in the variegated plants, right? So we know that the chloroplasts are only transmitted to the offspring, the progeny, from the female parent, which is the ovule, and that the progeny produced always matched or can be produced from the ovule used in the cross. So you just need to, I don't know if I can go back. Oops. You can just see that it doesn't matter what the pollen is, whatever is in the ovule, the maternal parent, that's what the offspring are going to look like. Okay, so now ovules produced by an entirely green plant contain a rare mutation, keyword here, in a nuclear gene that is involved in chlorophyll synthesis. The gene that encodes a non-functional protein. Predict the most likely color of leaves on plants that are produced when these ovules are fertilized by pollen from a wild-type green plant. Okay, so now you're asked to shift gears and think about a mutation in a nuclear gene. What color are the offspring going to be if the pollen was from a green plant? Okay, and the leaves are going to be green. This is a nuclear gene that has a mutation, not a chloroplast DNA gene. And the reason for that is we need to justify is that the plants will inherit the functional gene from their dad, from the pollen, in the wild-type green plant. So depending on the amount of the gene's encoded protein that's needed for chlorophyll production, the leaves might be paler green, but they will all be green because they got that gene from the pollen. All right, quick check for understanding. I'm trying to do this quickly, and then you can practice with this later on. Okay, so again, these take a long time, and they really require that you do some careful reading. Fruit flies, Drosophila melanogaster, with a wild-type phenotype, have gray bodies and red eyes. Certain mutations can cause changes to these traits. Mutant flies have a black body and are cinnabar eyes. To study the genetics of these traits, a researcher crossed a true breeding wild-type male, which would be gray body and red eyes, with a true breeding female fly with black body and cinnabar eyes. So again, we've got a homozygous dominant with a homozygous recessive. We get our F1, which are all heterozygous for each trait, so they're dihybrid. Female flies from the F1 generation were crossed with true breeding male flies with black bodies and cinnabar eyes. Now we're crossing the F1 hybrids with a homozygous recessive male for both traits. The data on the next page, it actually is not on the next page, it's right there in front of you, represents the predicted outcomes and the data obtained from the cross. You are asked to explain the difference between the expected data and the actual numbers observed. So think, Mendel, if these genes were on the different chromosome, right, we would expect 1 to 1 to 1 to 1. So we get a same number, 244 for each of the F2 phenotypes, but that's not what happened. We got an observed number of the parents. So we have a gray and red, right? So that was one of the parentals. We had a black and cinnabar, which was the other parent in that first cross. And then we have two new phenotypes, gray and cinnabar and black and red. So we call those the recombinants. So now you need to explain that difference, okay? So your explanation needs to include the idea that we would predict a 1-to-1 ratio if it was Mendelian inheritance pattern. You could do a Punnett square that shows that cross, the dihybrid parent with the homozygous recessive parent. A correct application of the chi-square analysis to show that the observed results do not conform to the expected frequencies. In other words, these are not 9 to 3 to 3 to 1. You could say that because of those things, the body color and eye color are linked genes. You could show ratios to show linkage and independent assortment of wing type versus link traits. You could identify the bottom two phenotypes as the products of crossing over, which are those recombinant phenotypes. And then you can also do the crossover rate. You can do the recombination frequency, which is between 9 and 10%. So again, let me show you working it out. You can see the F1 dihybrid crossed with the homozygous recessive. We should get a 1 to 1 to 1, but we did not. So why? The why is that the eye color and body color genes must be linked. And you can actually show that with math by calculating the recombination frequency by adding 42 and 47 to get the 89 recombinants out of the total number of offspring, and that's about a 9% recombination frequency. All right, so to debrief, you should now be able to explain deviations from Mendel's model of the inheritance of traits. You should be able to explain biological concepts and perform some types of mathematical calculations. Your key takeaway, as it's been all week, is that heritable information provides for the continuity of life. I want to leave you with some common errors and misunderstandings. This question was given, might have even been on last 2018 AP biology exam, and this also shows the effect of the environment on the expression of the genotype as phenotype. So this is kind of a funny one. In the tongue soulfish, its name is cynoglossus. Glossus means tongue. Sex is determined by the combination of genetics and environmental temperature. Genetically male fish have 2 Z chromosomes, ZZ, and genetically female fish have 1 Z and 1 W. They're ZW. When fish are raised at 22 degrees C, ZZ fish develop into phenotypic males, and ZW fish develop into phenotypic females. However, if these fish are raised at a higher temperature, 28 degrees C, the Z chromosome is modified. We denote it as Z star. Z star W individuals develop as phenotypic males that are fertile and can pass on that Z star chromosome to their offspring, even when the offspring are raised at 22 degrees. They're showing a cross between a ZW female and a Z star Z male in that Punnett square. You are asked to predict the percent of phenotypic males among the F1 offspring of this cross if the offspring are raised at 22 degrees C. Then you are given another task, which is that if at least 1 Z or Z star chromosome is necessary for the survival of the fish, a researcher crossed 2 fish and observed a 2 to 1 ratio of males to females among the offspring. Based on the information, identify the genotype of the male parent in the cross, and then finally describe one fitness cost to the female of mating with this particular male. Okay, so here's the scoring guidelines. The prediction is that there would be 75% males, and that's part A, and then in part B, the 2 to 1 ratio of males to females means that you would have to have the male parent being Z star W. And the reason that there's a fitness cost, as you'll see when I show you the Punnett square, fewer offspring will survive. The individuals that get the WW genotype will die. Okay, and another thing is that all of those male offspring will have the Z star chromosome, and that doesn't bode well for the next generation. So if you look at this cross, in the first part for your prediction, you've got your ZW female and your Z star Z male. 1, 2, 3 males, or 3 of those offspring are males, so 3 out of 4 is 75%. The only way to get a female is ZW. To make that prediction, here's the Punnett square. Remember, we get a 2 to 1 ratio, so one of the offspring actually died, and that is that WW genotype. So the parent is Z star W. So I'm just going to look at these results. So the good news is that most students were able to use the Punnett square to correctly predict that 75% of the offspring would be phenotypically male. In part B, there was a drop-off. Most students could identify the likely genotype of the male parent as Z star W. However, only some students were able to discuss the fitness cost to the female, in that she would have fewer offspring that survive, or that some of her offspring would have that Z star chromosome. Okay, so here's your homework. We've seen this one before, but it's a different part of this question. You have a cell, and there are 2 chromosomes. Chromosome 1 is for the color of the flower, either green dominant or purple recessive, and chromosome 2 is for plant height, either dwarf, which is dominant, or tall, which is recessive. So the individuals of the parental generation with the genotypes of big G, big G, big G, big G, and little g, little g, little d, little g were crossed to produce F1 progeny. Now here's your new question. If the 2 genes were genetically linked, describe how the proportions of the phenotypes of the resulting offspring would most likely differ from those of the test cross between an F1 individual and a GG, DD individual homozygous recessive. Okay, so work that out. Basically, you're wanting to answer how can you tell the difference between if genes are linked and not linked. All right, that is due on Monday because it's the weekend, and Ms. Pfizer will be with you on Monday to go over the answer to that. Just want to remind you of your exam checklist. Today is the 24th of April. It's the last day to reach out to College Board. If you need a device or internet connectivity, please reach out to them at cb.org/tech. Also, check to make sure that the College Board has your correct email email address. They'll be sending you your exam ticket via email, although you will be able to access it through AP Central if you do not receive the email. And then finally, let your AP Bio teacher in on your plan to test or not to test. Okay. So thanks for watching. We hope that you'll join us again. Make sure you complete that practice question for Monday. And Monday's lesson will review chromosomal inheritance and the environmental effects on phenotype. Thanks for watching. Have a great weekend. See you guys next week.
Welcome to AP Biology. I am Gail Pfizer from Darnell Cook Middle High School and Florida State College in Jacksonville, Florida. A lesson overview for today. Today I'll be reviewing Unit 5, Heredity. In fact, it'll be the last two sections of the unit, topics 5.5 and 5.6, environmental effects on phenotype and chromosomal inheritance. By the end of this lesson, you'll be able to explain how the same genotype can result in multiple phenotypes under different environmental conditions, explain how chromosomal inheritance generates genetic variation in sexual reproduction, and predict the causes or effects of a change in or disruption to one or more components in a biological system based on a visual representation of a biological concept, process, or model. And you won't need a calculator today, just pen and paper. All right, now for our warm-up, which was yesterday's homework from our, for Friday, it was Friday, Non-Mendelian inheritance. And I went ahead and filled out some things to save a little bit of time. We have our green is going to be dominant, okay, and purple is recessive with little g, D dominant for dwarf, and D recessive for tall. And I know you've seen this question probably 3 or 4 times already, but there's so many parts to it that we need to cover. It covers everything. All right, so here's chromosome 1 and chromosome 2. The original questions had a G on one and D on the other chromosome, okay? And a certain species of plant, the diploid number of chromosomes is 4. 2 n equals 4. Flower color is controlled by a single gene in which the green allele G is dominant to the purple allele little g. Plant height is controlled by a different gene in which the dwarf D is dominant to the tall allele D. Individuals of parental P generation with genotypes uppercase G, G, D, D, and lowercase G, G, D, D cross to produce an F1 progeny. I'm gonna go ahead and get those off back off of there so that doesn't confuse you anyway. Okay, so we have our parental groups. We have big G, big G, big D, big D, and little g, little g, little g, little d is your parental group. And then so their offspring would be this dihybrid big G, little g, big D, little d. Now it's asking us to, so that was our cross here. Now our question is, if the two genes were genetically linked, so now we need to not have them on the same chromosome. I mean, on different chromosomes, we need to have them on the same chromosome. Describe how the proportion of phenotypes of the resulting offspring would most likely differ from those of the test cross between an F1 individual and a little g, little g. So let's set up our test cross like we did the other day. And so our options were, here was our F1 cross. So we'll have big G, D, we'll have big G, little d, we'll have little g, D, and little g, little d. I think I have it right. And the only option we had from the test cross group would be little g, little d. Okay, so here we go. Let me do this really quickly. Okay, so which one would have both dominant phenotypes? Those would look like our parental group. So that would be this one right here, right? And this one would look like parental group, little g, yes. Okay, now anything in between, this would be dominant of our green, and then it would be tall, and this would be purple and dwarf. So these are what called the recombinants, okay? All right, so at the test cross, we had 25% of each. So that was a 1 to 1 to 1 to 1 ratio. So now the question is asking us, describe how the proportions of the phenotypes of the resulting offspring would most likely differ from those of the test cross between F1 individual and a GGDD individual recessive. Okay, so this is what the test cross results would be. So how would genetically linked, what would that look like? So I'll give you just a moment to think about it before I tell you, and then we look at the scoring guideline. And I'll give you a moment to get the clock together. Okay, all right, so when you have linked genes, you're going to have your recombinants are going to be less than 50%. So if these were 25 and 25, they're both going to have to be reduced, okay? So they're going to have to be less than 25% each because we've got to get both of those recombinations together less than 50%. And then that would make our parental groups have to be greater than 25% each, so they have to be greater than 50% each together. Okay, so let's look at the scoring guideline. All right, so the majority greater than 50% would have the parental plant phenotypes, okay? We got that. Greater than 25% would be green dwarf, and greater than 25% would be purple tall. Is that right? Okay, green dwarf and purple tall. Yes, okay, we got that. And then less than 25% would be green tall, and less than 25% would be purple dwarf. Okay, and there's our green tall, and there's our purple dwarf. Okay, all right, I hope you got this right. But remember, less than 50% in recombinants, and we'll talk about it at the very end too. I'll give you some hints on what to look for. All right, so what you need to know. All right, naturally occurring diversity among and between components within biological systems affects interactions with the environment. The same genotype can result in multiple phenotypes under different environmental conditions. Environmental factors influence gene expression and can lead to phenotypic plasticity. Phenotypic plasticity occurs when individuals with the same genotype exhibit different phenotypes in different environments. So plasticity just means a change in gene expression as a result of an environmental experience. So I'm going to take you on a little journey through environmental experiences first. This first one is a seasonal, and it's the white Arctic fox. And the white Arctic fox has a seasonal color code. Melanocyte stimulating hormone receptors are receptors for melanocyte stimulating hormone. And melanocyte stimulating hormone controls the level of tyrosinase within melanocytes. Tyrosinase is required to make melanin. So high levels of tyrosinase are needed to make eumelanin, and low levels are needed to make light-colored pheomelanin. So in the winter, the melanin-producing pathway is interrupted. Melatonin is released during dark hours, so it's a type of photoperiodism that's going on, and inhibits the tyrosine, which is responsible for the production of the melanin. I went down a rabbit hole on this little white Arctic fox, and they actually are they're heavier than this in the winter to help keep warm. All right, so assessments are always just not assessment. Assessments are, you can learn so much from assessment because not only do you have the stimulus to learn from, you have each of the choices to learn something from. And so I put a lot of questions in here to just give us a variety of things to talk about that we wouldn't have talked about probably in class. So this one is actually what we just talked about. Arctic foxes typically have a white coat in the winter. In summer, when there's no snow on the ground, the foxes typically have a darker coat. Which of the following is most likely responsible for the seasonal change in coat color? This was one of the first questions I think after the revision in 2012, and I've loved this question ever since. All right, so our choices are: the decrease in the amount of daylight in winter causes a change in gene expression, which results in the foxes growing a lighter appearing coat. That sounds pretty much what we just talked about. The diet of the foxes in the summer lacks a particular nutrient, which causes foxes to lose their white color and grow a darker colored coat. The stimulus did not tell us anything about diet, so let's eliminate that. Competition for mates in the spring causes each fox to increase its camouflage. There was nothing about competition. Now this is the one, if my kids are going to miss it, they're going to choose this. The lower temperatures in winter denature the pigment molecules. Okay, it's lower temperatures, and this is the pigment. This isn't an enzyme with the active site. It was a trick, and a lot of kids when they see this answer, they choose that, but it's actually about the photoperiodism because we had the snow on the ground, so it'll be longer nights. So our answer was A. All right, our next environmental experience: flower color based on soil pH. My favorite flower is a hydrangea, and a soil pH of 5.5, you'll have blue, and a soil pH of 6.5, you'll have pink. But if you have a mix, then you have purple. Here's another environmental experience example: low population density, probably. Sex determination in reptiles: A, uniparents, asexually reproduce. They reproduce through parthenogenesis, which means the egg develops without being fertilized. It could be a rare response to low population density. All the lizards are females. There are no males. Ovulation is stimulated by courtship mating behaviors, and they alternate 2 to 3 times per season per male and female roles. When estradiol is high, they act as female. When progesterone is high, they act as male. All right, another, well, I think it's our first temperature one. Environmental experience temperature. We have African violets in the form of a question. All right, so an African violet grower observes that genetically identical African violet plants grow near the walls of the greenhouse that have white flowers. I'm going to go ahead and do this. This is our greenhouse. Okay, the plants. Okay, let's do. What did it say? The walls had white flowers. Let me go ahead and do that. All right, the plants growing farther away from the walls have pale blue. That's not very pale blue. Okay, and the plants growing nearest the center of the greenhouse have dark blue. Okay, all right. Which of the following best explains the differences of flower color of the African violets in the greenhouse? So, warmer temperatures result in genotypic alterations, which result in flower color differences. I'm not going to choose this one because this temperature doesn't make the genotype change right there on the spot. The plants along the walls of the greenhouse are homozygous recessive and therefore have white flowers. Well, if we have recessive, then we have like dominant, and we have more than one. So I'm not going to. And there was no mention of planting them in any particular way. An enzyme responsible for flower color does not fold correctly in cooler temperatures, and the greenhouse is warmest in the center. Okay, let's hold that thought. And more light is available along the walls of the greenhouse, so the flowers need less pigment to absorb sunlight for photosynthesis. So if it's greenhouse, the whole thing is green. Every wall is green, and the roof is green. Or if it's clear, it's all going to be the same. So, and less pigment for photosynthesis, none of that makes any sense. So this one right here, and I actually went on a little look for enzymes not folding correctly in colder temperatures, and I did find some research that included those statements this morning. All right, environmental experience, example temperature again, sex determination in reptiles. Australian dragon lizards have a ZW sex determination system. The male genotype is homogametic, ZZ. So here we have our male is ZZ, and a female genotype is heterogametic, which is female, and she is a ZW. However, all eggs incubated at temperatures above 32 are female. Okay, which of the following best explains how the development of a phenotypic female Australian dragon lizard with a ZZ genotype occurs when incubation temperatures are at above 32. All right, lizard embryos with a ZZ genotype cannot develop. Well, we just said that they could, okay, because they're going to become female. So not an incubation temperature above 32, Z's do not turn into W's, okay, just because the gene doesn't change on a chromosome because of that. At incubation temperatures above 32, causing overtransfers genes from the W to the Z, producing females. I think that we already have all eggs are incubated. It's already an egg. That's already happened. And incubation temperatures above 32 inhibit the genes on the Z, okay, on the Z, and that produce proteins necessary for male. And so that is our choice. Okay, all right, and here's another one. I have several of these, but it's fun to talk about these things that we need to talk about. Environmental experience, example, light color, sex determination in reptiles. So a scientist studying phenotypic variation in a species of butterfly observed that genetically identical caterpillars run in similar cages but exposed to different colored lights developed in the butterflies with differences in wing color and body size. So here we have a white wing color, darker when it's red. Okay, and I'm going to grab the red. There's red, and we have smaller when red, and then with blue we have a lighter wing color and we have a larger size. Which of the following best explains the cause of the phenotypic variation observed in butterflies? A, different mutations occurred in the caterpillars that were exposed to different colors of light. No, if they had mutations, there's no guarantee that all are going to have the same mutation, and then that would be before them. Okay, that would be in a previous generation. The energy used to grow a larger body results in butterflies with lighter colored wings. No, individual caterpillars evolved adaptations. Now this sounds to Lamarckian. There were different gene expression of wing color and body size in response to the colors of light the caterpillars were exposed to. There we go with gene expression. Okay, and yes, we have D. All right, effect of increased UV on melanin production in mammals. Humans produce melanin and melanocytes. There are two different types of melanin, and I spoke about these earlier. Eumelanin and pheomelanin. Eumelanin is very dark, and pheomelanin is fair or red. And melanin protects the DNA in the skin from UV rays, which would cause mutation. The melanin forms clusters over the nucleus to protect the DNA. So when I talked about this when I used to teach anatomy and physiology. So let's do this. Here's our nucleus, and here's our DNA. Okay, here's our DNA, and the melanin pigment. So here's the sun. The melanin pigment will cluster to protect the DNA in the nucleus and the cells. Okay, so UV rays are mapped, and higher UV rays are in areas of the world where darker melanin can be found. There is a correlation between the intensity of UV radiation and skin color. Where UV is strong, skin is dark. Pheomelanin is found where UV is not strong because sunlight needs to be absorbed to produce vitamin D because the sun is farther away from this area. So less pigment is necessary to get more sunlight because there's less UV. So I absolutely think that you should watch the biology of skin color from HHMI. It's just an amazing little short video. Make sure you do that sometime. All right, so I know I drew this before, and on I think it was signal transduction pathway. So let's do it again, and we have let's see, what do we have? We have this one over here. Here is our A. So example, presence of opposite mating type on pheromone production is what we're doing now. So that's going to have that's going to turn on our gene. Okay, for yeast. All right, so here we are, and we are. Let's do this. This is the alpha cell membrane. It's not bigger, it just gives me room to work. And here's a receptor, and this A type yeast is going to release its mating pheromone. Okay, and it's going to bind to that receptor, and then I'm going to make this a little bit bigger. And then we're going to have this event, and I'm going to phosphorylate some of these in a moment. Okay, so you know how this went. Let me get this to 5. All right, so we have this signal. This was reception. Signal transduction is going to happen. Phosphorylation is going to happen, and it just continues. And then we have this phosphorylated molecule at the end. Well, it's going to. Here's a actin filament, which is responsible for the structure of holding the shape of the cell. And this particular molecule is going to put actin molecules on the actin filament. So when this is reception, and then the signal, and then this is going to go to work and start making these actin molecules. Genes will go to work. Proteins will be made. Actin molecules will be made. And there we have our shape that we're going to call a shmoo. All right, so it will then reach this A. This alpha wants to reach this A because it's of this mating pheromone. Okay. All right. Now off of the environment. So this was environment. This was an environmental experience because the mating pheromone was in the area. So that's the end of the environmental experiences. All right. Next we're going into chromosomal inheritance, non-disjunction, and then some other patterns that are a little different. Okay. So these are going to be reviewed. Just review these pictures are bigger. If you look in the Mendelian Genetics slide deck, you'll find them there. Anyway, so chromosomal inheritance generates genetic variation in sexual reproduction. Segregation, independent assortment of chromosomes, and fertilization result in genetic variation of population. The chromosomal basis of inheritance provides an understanding of the pattern of transmission of genes from the parent to the offspring. So if you remember, we talked about law of segregation, and we had a big D here and a little d here, and this would never be, this little d would never be expressed as long as it's with that big D. But during anaphase of meiosis 1, we have segregation. We have the separation of the big D from the little d, and then this is often a cell by itself, and this is in a cell by itself. And then if we have a cross with another of the same little d, then we're going to be able to be expressed as in that 3-to-1 ratio of our monohybrid. Okay, as far as the law of independent assortment, when we had our meiosis 1 meiosis 1, metaphase 1, anaphase 1, then we were going to split down the middle, right? It was a bad down the middle. And anyway, so this originally had big D and big R. Do they have to go that way? And little d with little r? No, they do not. All right, so we can have big D with little r. So this could have been over here, that could have been over there, and vice versa. There is no order that it has to go in. It's random, as long as their homologous pairs are next to each other. And that's the law of independent assortment. And we can see that in our dihybrid cross of 9331. And then we have our meiosis. We have fertilization. There's a whole slide deck on meiosis. And if you have, here's 3 pairs of chromosomes. They go through interphase and they replicate. And they go through crossing over. Remember, that was a step of genetic variation happening right there. Independent assortment. We had variation going on. Anaphase. Now we have 2 cells. And then we have anaphase again. And now here, these 4 daughter cells are so different than this original 3 pair. And when they cross with another diverse offspring, someone else's other offspring, then we start to see this all over again. And genetic diversity just increases generation after generation. Okay, certain human genetic disorders can be attributed to the inherited inheritance of a single affected or mutated allele or specific chromosomal changes such as non-disjunction. So first we'll talk about single mutations, okay? So if this is messenger RNA, I can say that it's going to look exactly other than the U. It's going to look like our coding strand of DNA. So I'm just going to pretend that this is our coding strand, it would be GAG, and this would be GTG. Okay, if we had a coding strand with GAG, then we're going to code for glutamic acid and have normal hemoglobin. If we code for GTG, then we're going to get valine and we're going to have a mutated hemoglobin. So now let's take this, the next slide will actually take the messenger RNA to this. This was the mutation right there. Should be an A, but was a T. So let's take these messenger RNA to the genetic code. All right, so we have GAG, so working with normal, and that would be our GAG is right here. There's glutamic acid. So what's going to happen is this GAG will have the anticodon for CUC and bring in glutamic acid. Glutamic acid will then be added to this polypeptide chain, and that's what's supposed to happen at the normal hemoglobin. On the other hand, if we have a GUG, then it's going to code for CAC. There's GUG, there's valine, and it's going to code for valine, and valine is going to be added to that polypeptide chain, and that is a problem. Okay, it's not supposed to be valine. It's supposed to be glutamic acid. And if you can see, glutamic acid is hydrophilic and valine is hydrophobic. If we have a protein and you are hydrophilic, that means that you can be found on the outside of the protein because these are amino acids because you are polar. And then hydrophobic would want to be on the interior away from that water. So that makes a big difference whether you're on the outside or the inside. That means it's going to fold improperly and the resultant function is not going to be the same. So what happens is you have this sticking together of these molecules that make this single shape right here. All right, Huntington's disease is another single affected allele. A genetic counselor is consulted by a young man who is worried about developing Huntington's disease, an inherited disorder caused by a dominant allele of a single gene. The young man explains that his cousin was recently diagnosed with Huntington's disease. Let's go ahead. Inherited, and we're talking about a cousin. And the news has caused him to consider his own risk of developing the disorder. Which of the following questions will help the genetic counselor to evaluate the risk of the young man developing Huntington's disease and transmitting it to his children? So this is a question about questions, is what I call it in my classroom. Okay, were you and your cousin born in the same geographical area? Okay, this was an inherited disorder. It wasn't due to an environmental experience. Okay, were your parents or grandparents ever diagnosed with Huntington's disease? This would be a good question. Were you in physical contact? No, it's not contagious. And were you ever exposed to substances that are suspected of being mutagen? No, this is not about getting this as a cancer. Okay, so Huntington's disease, and let's see. And the what? So I put this slide in because I'm going to mention some things about this. So it said it was autosomal dominant, and it's lethal though. It's later in life after 40, and it's a degenerative disease of the nervous system. Okay, so now that was our single allele, mutated allele, and now we're going to talk about non-disjunction. So this is in anaphase of meiosis 1. And so what should happen is that these homologous chromosomes would be pulled apart, and these two would go to that pole, those two would go to that pole, but instead they stay together. Okay, they're still joined. And what happens is there's only two chromosomes. There's one on this side and one on this side when we get to anaphase 2, and then there's 3. Then we have one of each of these, 1, 2, 3. So at the end in our 4 cells, we have N+1, which is 1 too many, N+1, 1 too many, and N-1, 1 too less, N-1, 2 less. So all of these have a chromosomal abnormality. Okay. Now in the second type of non-disjunction, we're going to have this happen in anaphase 2. And if you'll notice this right here, so let's say so far so good. So we have our two homologous pairs, they separate, that's good. And now these two sister chromatids need to separate, but yet this one didn't. Okay, it did not separate. So we have normal, everything's normal here. So we have at the end a normal cell with those two, We're missing one, so it's N-1, and we have 1 too many, and we have N+1. Okay, example of non-disjunction is trisomy 21, which is Down syndrome. It's a aneuploid condition and has characteristic facial features, short stature, correctable, heart defects, developmental delays, risk increases with mother's age, and most are from meiosis 1. And this is what it looks like. So this is a karyotype. So on a karyotype, you have all of your autosomes for human being, 1 through 22, all pairs. There's 2 of each, and they're organized according to their length, and there's some bands and things like that come into play. But this one right here has 3 chromosomes instead of just 2. So this is why it's called trisomy 21, and this is Down syndrome. And here we have, that's a girl, that's an X would be a girl, but a Y makes this a boy, just an FYI. So this is a boy with Down syndrome. All right, non-disjunction, Klinefelter syndrome. Let's look at this karyotype before we read the question and see if we can tell what's wrong. So we have everything is looking normal. We actually do this karyotype activity in my classroom with this magnetic board, and it's great. We have different scenarios in little bowls, and they switch around between groups of which chromosomal problem they're doing. Everything looks good until we get right here, right? So we have 2 X's, but we have a Y, so that makes us a boy. Okay? The diagram above depicts a karyotype of an individual human. Which of the following statements concerning the karyotype in the diagram is true? A. The diagram illustrates a condition found in females. Is that right? Nope. The diagram indicates a mechanism for increasing genetic diversity. This is not a good mechanism for increasing genetic diversity. Okay? The diagram illustrates results of non-disjunction during gamete formation. Yes, that's right there. And it indicates Down syndrome. No, we know that Down syndrome would have to do with 21. So C would be our answer. Okay, and I also wanted to tell you about another chromosomal non-disjunction problem. It has to do with Turner syndrome, and it's a girl, but she only has 1 X, and so there's nothing here. Okay, and it's also from non-disjunction. Okay, so this was a lesson to myself to practice what I preach today. So look at this diagram and then read. So what we have here, we have an X and a Y, and so they separate in meiosis 1. That's good, and then they're into 4 daughter cells at the end with a haploid. Okay, so figure 1 illustrates the X and Y chromosomes during meiosis 1 and meiosis 2 of normal spermatogenesis. That's what I didn't read. I didn't read this first part. I just jumped in here, and then I spent 5 minutes trying to figure out what was wrong with this picture. Anyway, if the normal spermatogenesis is disrupted, the gametes can have different chromosomes than expected. Which of the following is the most likely cause of 1 of the 4 gametes having 2 X chromosomes and 1 having neither an X nor a Y? So let's do this. Okay, so there's our 1X. Let's add this 1X to it, and then let's take out this Y. I mean, this other X. I think that's what it asks us to do, and then answer the question. So which of the following is most likely cause of 1 of the 4 gametes having 2 X's? There's 2. Okay, so just the meiosis 2 shouldn't have that, and 1 having neither an X nor a Y. Okay, so we have empty. Non-disjunction of the chromosomes during meiosis 1. Let's see, something's wrong here. Oh, I'm trying to write in Y. Okay, all right. Did anything happen wrong here? No, we're still good. That's what we should look like. So that's not it. Non-disjunction of both the X and the Y, not affected Ys. Not it. Non-disjunction of the Y, not it. Non-disjunction of the X chromosome during meiosis 2. This one still looks like this one. Okay, so that is the answer. Okay, and don't be like me and not read before I answer the question. Okay, learn a little lesson today. Oops, let's go back. Okay, last one of these scenarios. Okay, red-green color blindness in humans is caused by a recessive allele located on the X chromosome. Figure 1 shows the potential offspring of a female who is red-green colorblind and a male with full color vision. All the possible male offspring would be colorblind, and all the possible female would have full color vision. If during the production of male gametes an error, so we're to this paragraph right here, was referring to this. If during the production of male gametes an error in meiosis occurred, sperm containing both an X and a Y chromosome could be produced. So it took me a minute to figure out what this was, but this is I swear they have here. Okay, all right. So possible offspring. Now, what does the question ask? How would the extra chromosome? So we need to look for where's their extra chromosome? It's right here. Okay, and let's look at that. If we have Y, what do we already know we are? A boy, and we have a big B, so we know we have normal vision. So that is what our trait is going to be. So how would the extra chromosome affect the male offspring produced by the gamete? None of the potential offspring would be male. I think that's wrong. The male offspring would all be red-green colorblind. Now we already figured it out that they're going to be normal. The male offspring would have full color vision because of the presence of the extra X. Absolutely, there it is. There would be no change. Yes, there is. Okay, of the possible offspring because the extra X chromosome would not be active. That's not true. So our answer here would then be C. Okay. All right. So now on to a new topic, chromosomal changes, and this is deletion. We have an A, B, C, D, E, F, and G. And if we were to take out the C, if it was a gene or a segment with several genes on it, then we're going to lose the gene expression that's happening right here. Okay. Deletion is a problem. The next one would be a duplication. If you see, we have A, B, C, D, E. Here's our C, but now we have a segment repeated. And we'll talk a little bit about that at the end. And then an inversion. So this ED, the CDE, just does a total flip, and now it's EDC. So it's reversed, and there's going to be an imbalance in its gene expression. And then we have a translocation. Here's our translocation. We have our A, B, C, D, E, F, G, our H, I, J, K, L, M. Now look after homologous chromosomes have separated. Here is our H, I, J, and here is our K, L, M on two totally different chromosomes. Okay, and this will also cause some differences in gene regulation. Okay, so guided practice. We're going to write just a very short FRQ today. All right, science skill practice, predict the causes or effects of change and disruption 2. We've been doing that through several multiple choice questions. One or more components in a biological system based on a visual representation of a biological concept, process, or model. And the task verbs that we're going to do today are identify, make a claim, provide reasoning, calculate. I think I took out a lot of these, especially calculate. I built this presentation way too big, but I did leave some great questions in the slide deck if you use it. Okay, and that's under natural selection 7.1, our very first presentation. There's a link to the slide deck. Okay, so writing FRQ responses. Meiosis occurs in many organisms as part of sexual reproduction. So we're going to discuss, and 2 of them, examples of mitotic errors and their effects on offspring. I'm going to give you 30 seconds or so, maybe a minute to think about this and jot down some answers. We're doing great on time, so go ahead and think about this. Okay, did you jot something down? All right, so you should have said, so your 2, you should have said non-disjunction and then the chromosomal changes. So non-disjunction, spontaneous abortion can actually occur because of this. Monosomy, that means the one that we're talking about, like Turner syndrome. Trisomy, we talked about was Down syndrome, and anything that's not your normal N number, it's called polyploidy. Okay, chromosomal changes. Here's the 4 that we talked about: duplication, deletion, inversion, and translocation. But this scoring guideline seems kind of like out of whack a little bit, so I did my own. It looked like the right answers are there, but it's not in the right order. So deletion would be loss of critical gene expression. An example of that was Priyushek, and I learned about that in nursing school actually. And it is a male that has this cry of a cat. And duplication, fragile X, and there's some other repeats. There are problems when there's duplication. And inversion, there's imbalance of the gene expression. There's a 180. We had CDE, and then all of a sudden it went EDC. Okay, and I read also that there were some inversions that they can possibly connect with differences between us and chimpanzees. I read that this morning. All right, translocation, change in gene regulation. There's a condition called Philadelphia chromosome. I actually had my student go on a little website search about this, and it's about chromosome 9 and 22, and it causes chronic myeloid leukemia. All right, so debrief and summary. Today's takeaway: naturally occurring diversity among and between components within biological systems affects interactions with the environment. You should be able to explain that the same genotype can result in multiple phenotypes because under different environmental conditions, and some chromosomal inheritance generates some genetic variation also. Remember, this was the different environments, and this we talked about non-disjunction, we talked about mutations, and we talked about what else? And some chromosomal changes. Okay, all right. Predict the causes or effects of a change in or disruption 2. One or more component in a biological system based on a visual representation of biological concept, process, or model. And we predicted what would happen if it was different almost the whole day. Do this. Stay tuned, because I have some practice exam information for you at the end. So it's a common error. So where I get my common errors from is my classroom, what I see happening, and then for myself, because I do everything I have my kids do. I have every struggle they have. Things are not always easy for me, and so I feel their pain sometimes. But I want to give you one thing here. When you see these, now let's just say this for example: 433, 433, 55, 55. I don't want you to think about it. You already should have linked genes in your mind, okay? Because you can see that this is less than 50%. Okay, 50% recombination. All right, then a dominant trait is not necessarily a favorable trait. Remember the green and the dwarf and the tall? I immediately, without read, because I didn't read, I had D going with tall, okay, because it just seemed like the normal version would be the dominant trait. That wasn't necessarily so. Okay, read questions carefully. The diagram, this was the other one I spent a long time on because I didn't read that was the normal version. I kept trying to find what was wrong with it. So do as I do, and do as I say, and not as I do again. All right, hold on. So here we have homework for tonight. AP exam practice homework. You've seen this before. A spermatocyte produces a polyform from cells. These cells result of non-disjunction during which the following phases. Real easy homework. I would just go jump back to where this is in the presentation. I would take and draw out meiosis yourself and follow this chromosome and then figure out how to get to here. I put this at the bottom and then work backwards. All right, so your to-do list. Check your device and your internet connection. Check your email address and your phone number and AP account. Tell your teacher you were testing and breathe, because your test ticket will be in your account. Also, it'll be in your email and in your account. Next review session, we're actually going to start Unit 6, which is our last unit, and we hope you'll join us again. Stay tuned though, because I'm about to go over some optional practice FRQ information. So let's do that really quickly. We're good on time. All right, so this is for everybody to know. So you need to go to your AP Classroom and log in. So that's your first step, is log in. And then you're going to click AP Classroom up here in the right. And then you're going to click here for optional student practice. And look, you have access to all these videos all the time without going to troll through YouTube looking, okay? They're all a link right here. Now click on this, practice answering 3 response exam questions. And then look for this sign, this green button, okay? It says optional student practice. These questions marked with this symbol mean that you can take the question, you can answer these questions on your own. Your teacher does not have to assign them, that you can do this on your own for your own practice. So if you click on one, and actually this is biology that you're looking at, but the same method goes for all of your classes if you have other AP classes. So this doesn't tell you what the content is about, but it tells you about science practice here. Okay, so we're going to click on one, and then it's going to open up, and we see some graphs. This is biology. They did biology as the example for everyone. And then we're going to type in our answer into the box, okay? And then we're going to hit this small button here that says submit. And then are you sure you want to submit your assessment? You're going to still say yes. And then this is where I want you to stop. Okay, see how this question is scored on the AP exam. Before you submit, would you like to review how this question might be scored on an AP exam? You can review and optionally revise your response. So this is what I want you to do. Yes, review, okay? Because this is going to bring up the scoring guidelines, and you know what you wrote, and you know what you should have written. And so now you can actually go back and you can have the opportunity to revise your answer, and then that gives you the experience of writing out the sentence in the proper format and include all of the content that you need to include. So after you revise it, then you can hit the final, I'm one ahead of myself actually here. Then you can hit the final yes review, okay? Well, that was the yes review. Now you're going to hit submit because you've already reviewed on the last slide because I was ahead of my time. Okay, and I think that's it. All right, so I hope that helps. Go find those questions and then come back tomorrow and we start Unit 6. Okay, thank you. Good night.
Today's AP Biology lesson. I'm Allison Kate, and oh no, that's not what's supposed to happen. So we're just going to get right to our warm-up today. So yesterday's lesson was on chromosome inheritance. inheritance, and you were given a homework assignment that is a multiple choice question, and you can see you have a diagram, and it says a spermatocyte produces the following 4 sperm cells. These cells are the result of non-disjunction during which of the following phases? And so you can see that N represents haploid, so you've got 2 sperm cells that are N+1 and 2 sperm cells that are N-1. So your choices are A, interphase, either the G1 or G2, B, interphase, the S phase specifically, C, mitosis, D, meiosis 1, and E, meiosis 2. And since you have had many lessons on cell cycle and mitosis and also the process of meiosis, you can easily eliminate A, B, and C because we know that there is no haploid gamete production in any of those processes. So now we have to figure out, is it meiosis 1 or is it meiosis 2? So think about the events that happen and when either the homologous chromosomes are being separated or those chromatids are being separated. Okay, got your answer? All right, the correct answer is D, meiosis 1 would produce this non-disjunction or the failure of those homologous chromosomes to separate. And as you can see, what results is an incorrect number of chromosomes in those gametes, and that is called an aneuploidy. You either have too many or too few in the resulting gametes. All right, so today's lesson is going to be on DNA and RNA structure, and so some of it will be a review from when we discussed nucleic acids earlier in the context of macromolecules, and then we're going to talk about DNA replication. We're also going to look at science practice 2B, which is explaining relationships between different characteristics of biological concepts, processes, or models represented visually in applied context. So you can guess that we're going to look at the process of DNA replication, and we're going to look at some models that represent that process and being able to explain the relationships between the different characteristics involved in this process. You should be familiar with kind of the concept or understanding of nucleotide structure, which was presented earlier, hydrogen bonding, which was also presented in an early lesson, and then the idea of 5' and 3' strands, which I'll review because that's very important to DNA replication. The terms double helix, leading strand, lagging strand, and Okazaki fragment will be helpful today. By the end of today's lesson, you'll be able to describe the structures involved in passing hereditary information from one generation to the next. You should be able to describe the characteristics of DNA that allow it to be used as the hereditary material, and you should be able to describe the mechanisms by which genetic information is copied for transmission between generations. So here's what you need to know. So first of all, just reviewing DNA and RNA structure, and understanding that DNA, and in some cases RNA, is the primary source of heritable information. So in general, DNA is going to be double-stranded compared to single-stranded RNA. Now there are many different types of RNA, and there are some that are double-stranded. There is only one type of DNA. When looking at the 5-carbon sugar in DNA, it's deoxyribose, and if you look carefully, remember how we count our carbons. That orange highlight is showing you the functional group carbon 2, and there's just a hydrogen atom, so it's without oxygen, which is where it gets its name, deoxy. And then if you look at the RNA sugar, at that same position, at the second carbon, there's a hydroxyl functional group, so it has that O present, so it's the ribose, and that's where DNA and RNA actually get their names. And then there are some nitrogenous bases that differ in DNA and RNA. So in DNA, the nitrogenous bases are thymine, which is abbreviated T, cytosine, which is abbreviated C, adenine, abbreviated A, and guanine, which is abbreviated G. In RNA, thymine is not present, but a related nitrogenous base called uracil is present. So we have a U in RNA, and then we still have our C, our A, and our G present in RNA. So let's just review what a nucleotide is, and I'm showing you a DNA nucleotide in this case. There are 3 components. The first one is a phosphate group. The second one is a sugar. In the case of DNA, it's deoxyribose, and we have a nitrogenous base. And you can see that the phosphate group is bonded by a covalent bond. It's a phosphodiester covalent bond. And again, if you're looking at your carbons, here's the first carbon, the second carbon that I just talked about. There's our hydrogen making it deoxy. Here's our third carbon, our fourth carbon, and the fifth carbon is actually above the ring of this pentose sugar. And so this is important to note that this phosphate group will either bond by a covalent bond to the 5' carbon or the 3' carbon. I'll explain this again when we get to the replication part. And then our nitrogenous base bonds covalently to the carbon at the first position. So remember, a DNA nucleotide consists of a nitrogenous base, a 5-carbon deoxyribose sugar, and a phosphate group. Okay, so the 4 DNA nitrogenous bases are adenine and thymine and guanine and cytosine. I'll talk a little bit more about them in a minute, but in this image, I'd like you to notice that the nitrogenous bases are held together by hydrogen bonds. And in the adenine-thymine pairing, there are 2 hydrogen bonds, and in the guanine-cytosine pairing, there are 3 hydrogen bonds holding those 2 nitrogenous bases together. Okay, so DNA and RNA exhibit specific nucleotide base pairing that is conserved through evolution. Adenine pairs with thymine, so in DNA, you're always going to see that AT pairing, and then in RNA, you'll see the AU pairing, and then cytosine will always pair with guanine in both RNA and DNA. And again, there's a special name for guanine and adenine because they have a double-ringed nitrogenous structure, and they're called purines, and cytosine, thymine, and uracil in RNA have a single-ring structure, and they are called pyrimidines. Okay, that's important to understand that you have a purine and a pyrimidine are always going to be pairing up. Okay, so adenine and guanine are the purines, thymine and cytosine are the pyrimidines in DNA, and then in RNA, uracil replaces thymine, so uracil is also a pyrimidine. Okay, so this image kind of gets back to that how that phosphate group will bond to the 5-carbon sugar, and the way it bonds will exert a directionality on the DNA molecule. The 2 strands run in opposite directions, and we call this being anti-parallel, and it has really important implications for how the DNA molecule is replicated. So I'd like you to look at the left-handed strand, and you can see that this is, we call this the 5' end because this phosphate group is first bound to the carbon at that position, and then it alternates, so then 3, 5, 3, 5, 3, and so we'll end with an open 3' end. Same thing is true in the opposite or anti-parallel strand. In this case, the phosphate group is first bound to the open end is 3, then we go 5, 3, 5, 3, 5, 3, 5' end. Okay, so we'll get back to that when we talk about DNA replication. So one difference between prokaryotic organisms and eukaryotic organisms, and I know you know this because we've been talking about the difference, many differences in these organisms throughout our review lessons, but prokaryotic organisms typically have circular chromosomes, while eukaryotic organisms typically have multiple linear chromosomes. So I have an image for you. You can see the prokaryotic DNA, and then you can see the eukaryotic DNA. And then both prokaryotes and some eukaryotes contain plasmids, which are small extrachromosomal double-stranded circular DNA molecules. So you can see this would be the main genomic DNA, and then you get your plasmids, which again are double-stranded circular DNA molecules. I want to talk for a minute about the packing of eukaryotic DNA, and I'm going to come back and talk about this in detail on Thursday when we get into the regulation of gene expression, and I'll probably show you this slide again. But just to understand how all that DNA gets packed into our cells in the nucleus of eukaryotic DNA, it gets packed so that it's largely inaccessible to the machinery that's responsible for transcribing genetic information. So this is one of the ways that we regulate gene expression. Chromosomes are dynamic in structure, meaning that they're always changing, and you know that a little bit from the cell cycle and mitosis, where chromosomes are condensing and they're unwinding. So we see a condensed region, and it may be loosened or modified as needed for various cell processes. In eukaryotes, the chromatin is packed using histone proteins, and those histone proteins, as we'll see on Thursday, can undergo chemical modifications that result in changes to how the chromatin is organized. So you can see we've got our double-stranded DNA molecule, okay, and then the histone proteins, they have a tail and a head, and they kind of wrap around that to form what's called a nucleosome, and then those nucleosomes form loops, and then we can see that we have a chromatid that's got that chromatin all densely packed, and this is a replicated chromosome with 2 chromatids, and this is probably what you're most likely to imagine when you think about chromosomes, is that kind of X-shaped structure. Okay, this is the overview overview of DNA replication. These are kind of the important things that you can come back to and think about that are important to know about DNA replication. So first of all, DNA replication ensures the continuity of hereditary information. DNA is synthesized in the 5' to 3' direction, and I'm going to go over that in the next several slides. The replication is said to be semi-conservative. What that means is one of the strand of DNA serves as the template for a new strand, so that each strand is produced, one new strand is produced, and one is the original strand. In blue, I have the names of some enzymes that actually direct the process of DNA replication. So these are all various proteins that play a critical role in the steps of DNA replication. The first one is called helicase, and as its name implies, it will unwind the helix. So it unwinds the DNA strands. We next have an enzyme called topoisomerase, which relaxes the supercoiling of that DNA helix just in front of the replication fork. And I'll show you a picture of this in the next slide. DNA polymerase is the main enzyme, and the reason it's the main enzyme is it is what is going to add DNA nucleotides to synthesize the new strand. However, it requires a primer made out of RNA to initiate this DNA synthesis. There is an enzyme called primase that facilitates the construction of RNA primers. Then that DNA polymerase enzyme will come along and synthesize the new strands of DNA, and it will do it continuously in one strand on what is termed the leading strand, and then in segments or discontinuously on the lagging strand. Because the lagging strand is made in pieces, it takes longer, which is why it's termed the lagging strand. DNA ligase joins the fragments on the lagging strand, and the lagging strand fragments are called Okazaki fragments. So let's take a look first at the model of semi-conservative replication. And I will tell you, I'm not going to go into it now, but there are elegant experiments that were done to show support for this model that is semi-conservative compared to completely conservative, or a model known as the dispersive model. So you can look at those experiments on your own. But we're going to start off with the parent molecule, and it's shown in dark blue, and then what we see, and what we're not being shown here, but the enzyme helicase is going to separate by breaking those hydrogen bonds between the nitrogenous base pairs, and it will separate those parental strands into templates. So they are the old strands that are serving as the templates for the synthesis of the new strands. So then we get the formation of those complementary new strands. So what that means is, on this side, if the parent strand is T, then the new strand will be an A. If this is C, then we get G, and T, and A, and T, and A, and so forth. So what you're seeing here is two identical strands result at the end of the replication process. So this is a model that predicts that when a double helix replicates, each daughter cell, which are shown in light blue here, will have one old strand, which is shown in dark blue, and one new strand, shown in light blue, that is derived from that parent molecule. Okay, so now I'd like to talk about the process of DNA replication. And I'm going to start with the leading strand. It's a little bit more straightforward. And what I'm looking at in the square is just kind of the large overview of how this process occurs. And we're actually going to get into the leading strand, which you can see here is shown in a continuous light blue arrow, and it's also shown in this way. So this is our replication fork, and these are the origins of replication. And so the overall direction of replication is, you can see the red arrows are indicating that. So first I want to focus on the leading strand. And again, here's our directionality. The template is this 5', 3', and so the new strand will always be synthesized in the 5' to 3' direction. You can think of this one as going toward that replication fork. Okay, so let's look at the details. First of all, the anti-parallel structure of the double helix affects replication. The DNA polymerases add nucleotides only to the free 3' end of the growing strand. So here's our growing strand. This is where we're going to be adding our new nucleotides. Okay, so a new strand can only elongate in the 5' to 3' direction. So pay attention to that arrow. DNA polymerases cannot initiate the synthesis of a polynucleotide. They can only add nucleotides to an already existing chain that is base paired with the template. When we say base paired, we mean that they are connected by hydrogen bonds to the nucleotide, or nitrogenous base in that parental strand. So how do we get started then? The initial nucleotide strand is a short RNA primer. There's an enzyme called primase that starts the RNA chain from a single RNA nucleotide and then adds those RNA nucleotides one at a time using the parental DNA as a template. So the red that you're seeing is actually the RNA primer. It's laid down by RNA primase, and it is going to bond those nucleotides in a complementary fashion. The primer is short. It's only between 5 and 10 nucleotides long. In case you're worried about it, it gets taken out later, and I'll show you how that happens. The new DNA strand will start from the 5' end of the RNA primer, right? So now our DNA polymerase can add to that existing RNA primer strand. And enzymes that are called DNA polymerases are what are actually going to catalyze the elongation of that new DNA at that replication fork. The rate of elongation is about 500 nucleotides per second in bacteria and about 50 nucleotides per second in human cells. Okay, so let's take a look at the lagging strand, and then we'll review the whole process. So again, here's kind of where we are. You are here, and now we're looking at these lagging strands. So you can see them here, and you can see them here coming toward the origin of replication and away from the replication fork in both of those cases. So again, in this little picture, the light blue is going to be the newly replicated DNA, and the little red is our RNA. All right, I'd also like you to notice in this image that we have different pieces of DNA labeled 1 and 2, and these are the Okazaki fragments, which are the pieces that are going to be put together in discontinuous steps and pieces that will later be joined by an enzyme called ligase. Okay, so looking at how the lagging strand is actually put together, along one template strand of DNA, the DNA polymerase synthesizes a leading strand, continuously moving toward the replication fork. Okay, so that's what we talked about last time. Here it is, there's the fork, and here it is on the other strand, here's the fork. Okay, so to elongate the other strand, which we call the lagging strand, the DNA polymerase must work in a direction away from the replication fork. So can you see how it's moving away from the replication fork and toward the origin of replication? The lagging strand is synthesized in a series of segments that are called Okazaki fragments. After formation of Okazaki fragments, an enzyme called DNA polymerase 1, rather than the DNA polymerase 3, that is the main polymerase adding those nucleotides, DNA polymerase 1 will remove those RNA primers and replace the nucleotides with DNA, and the remaining gaps are joined by DNA ligase. So if you look carefully at the image, first in the first one we see the RNA primase is this pink enzyme, and it is making the RNA primer, okay, in the 5' to 3' direction. Then we see DNA polymerase 3 will begin at the end, the 3' end of fragment 1, and it will synthesize the new DNA, and that is the Okazaki fragment. Then the DNA polymerase 3 will detach, and over here, DNA polymerase 3 is going to go back and start working on Okazaki fragment 2, okay? But RNA polymerase 1 is back here, and it will actually replace that RNA primer with DNA, and finally DNA ligase will form that bond between Okazaki fragment 1 and 2. So it's lagging strand because it takes a little bit longer to get all those steps done. So this is a summary, okay? Again, this kind of shows you where we are in the process. Here's our replication fork. It's not showing the other side, okay? So again, call your attention. We've got Okazaki fragment 1, 2, 3, 4, and 5. We've got our helicase, which is the enzyme that unwinds. We have our single-strand binding proteins, which I haven't talked about yet, but these are some additional proteins that once that replication fork is split, will kind of hold these together so that they're not kind of moving around too much. The topoisomerase will work up here as well. We have DNA polymerase 3, DNA polymerase 1, and DNA ligase. So a good thing to do is just kind of make sure you understand the actions, all of the enzymes involved in DNA replication. Okay, so what does helicase do? It unwinds the double helix. It unwinds that parental double helix. These single-stranded binding proteins stabilize the unwound templates strands so that those enzymes can hop on and do their work. The leading strand is synthesized continuously in the 5' to 3' direction by DNA polymerase. Primase begins the synthesis of the RNA primer for that 5th Okazaki fragment. DNA polymerase 3 is completing the synthesis of fragment 4. So I've got those arrows for you to show you that. Where's my cursor? So I can show you that. When it reaches the primer on fragment 3, 3, so here's the primer on Okazaki fragment 3, it will detach and begin adding DNA nucleotides to the 3' end of fragment 5, primer in the replication form. DNA polymerase 1 will remove the primer from the 5' end of fragment 2, which you're seeing here, and it will replace it with those DNA nucleotides added one by one to the 3' end of fragment 3. After the last addition, the backbone is left with a free 3' end. DNA ligase finishes the job by joining the 3' end of fragment 2 with the 5' end of fragment 1. Okay, so this is actually, let me go back because that was pretty quick. So these are the enzymes on the steps, and this is kind of the money slide. So if you want to go back and study, this is the one to study. I left this image in here. This is the summary of DNA replication, but I have no words. And so what I thought you guys could do is to look at this and you tell the story. Okay, so you've got a lot of information in this model, and so you can paint a picture. Your task verb would be describe the process of DNA replication based on this model. Okay, so one other thing, DNA replication is not without error, right? And so the DNA polymerase 3 does some proofreading and repair, and there are also some other enzymes that are involved in that proofreading and repair process. So DNA polymerases proofread the newly made DNA, and they replace any incorrect nucleotides. In a mismatched pair of DNA, other enzymes correct errors in that base pairing. One of those we'll see will be an enzyme called nuclease. DNA can be damaged by exposure to harmful chemical or physical agents such as cigarette smoke or X-rays. These are environmental, physical, or chemical agents, and it can also just undergo spontaneous changes or mutations. In nucleotide excision repair, a nuclease will cut out and replace the damaged stretches of DNA. So the error rate after proofreading repair is low, but it doesn't go to zero. So the DNA may have or likely will have some changes in their nucleotide sequence. The sequence change may become permanent and can be passed on to the next generation. But these changes, these mutations, are the source of the genetic variation upon which natural selection operates. So they're actually quite important. So let's get some practice with these concepts. The first one is a multiple choice practice, and it kind of is going to get at the idea of DNA base pairing. So there's another famous experiment that was done by a man named Erwin Chargaff that leads to Chargaff's rules, which are that A will pair with T and C will pair with is that right? A will pair with T and C will pair with G. So what he did is he investigated the nucleotide composition of DNA, and he analyzed DNA from various organisms and measured the relative amounts of adenine, guanine, cytosine, and thymine present in the DNA of each organism. And he did many more than the organisms that you see here. And so in this table 1, we see the relative amounts of bases by percent in an octopus, a chicken, a rat, a grasshopper, and a wheat plant. Okay, so you can study those percentages. Then we also have some percentages of C plus G and A plus T to give you some additional information. The question you're asked is, which of the following statements best explains the data set? Okay, so you're interpreting this data. A, since the percent of A and G add up to approximately 50% in each sample, adenine and guanine molecules must pair up in a double-stranded DNA molecule. Think about that. Do A and G pair up? Since A and T are approximately the same in each sample, adenine and thymine molecules must pair up in a double-stranded DNA molecule. So think about that. Do A and T pair up? C, since the percentage of A plus T is greater than the percentage of C plus G in each sample, DNA molecules must have a poly A tail at one end. We haven't talked about poly A tails. I will talk about them on Thursday. So at this point, if you don't know what a poly A tail is, that might be a difficult one to either select or omit. And then finally in D, since the percent of C and T add up to approximately 50% in each sample, cytosine and thymine molecules must both contain a single ring. So that's getting at purine pyrimidine. So hopefully you have your answer, and hopefully you selected B. So A and T pair up, A and G do not pair up, C and T do not pair up, and poly A tail is a distractor in this case. Okay, so one more, and this is related to prokaryotic DNA. So which of these following statements best explains the structure and importance of plasmids to prokaryotes? Plasmids are circular, single-stranded RNA molecules that transfer information from prokaryotic chromosome to ribosomes during protein synthesis. Are plasmids made of single-stranded RNA? Ask yourself. B, plasmids are circular, double-stranded DNA molecules that provide genes that may aid in survival of prokaryotic cells. So are plasmids double-stranded DNA? C, plasmids are single-stranded DNA molecules which are replicated from the prokaryotic chromosome that prevent viral reproduction within the prokaryotic cell. And D, plasmids are double-stranded RNA molecules that are transmitted by conjugation that enable other prokaryotic cells to acquire useful genes. So hopefully you remember from this lesson, because you're paying such close attention, that the correct answer is of course B. Okay, plasmids in prokaryotes are double-stranded DNA molecules. So you can immediately eliminate A and D since they're talking about RNA, and then you have to remember that it's double-stranded rather than single-stranded DNA. All right, so let's get some FRQ practice now. So this question says, scientists seeking to determine which molecule is responsible for the transmission of characteristics from one generation to the next knew that the molecule must, and there must be 3 features. 1, copy itself precisely. 2, be stable but able to be changed. And 3, be complex enough to determine the organism's phenotype. You are asked, your task verb is to explain how DNA meets each one of these 3 criteria stated above. This is a long question. Okay, so this was a long FRQ. This would be a 10-point question. It was an older question, so the scoring guidelines are a little bit different, but it is a 10-point question. So let's tackle the scoring guidelines. First of all, we just talked about copying. So the molecular properties for precise copying include the template concept. You could even talk about the replication being semi-conservative, that one of the strands is conserved and one is made brand new. You could talk about the molecular structure where we have complementary base pairing, A pairs with T, C pairs with G. DNA polymerase is the enzyme that actually adds those complementary nucleotides to the existing parental strand. The idea that those strands have to separate, you can even talk about helicase being the enzyme that separates, and then any elaboration of replication such as a proper sequence of steps, going into detail about leading and lagging strand, or discussing the enzymes involved in the process. The second part, you need to cover both stable and able. So what makes DNA stable, or the genetic material stable? Its shape. It's got a double helix. Okay. It's also stable because of its bonding. Remember that there's multiple hydrogen bonds between those nucleotides holding those strands together. There are silent errors. In other words, there may be some differences, but they're not always going to be in part of the DNA that is expressed. And then finally, it's able to be repaired through that proofreading that we discussed at the end of our discussion about replication. And then able, right? It's able to change mutations, right? That replication process is not 100% faithful. There's other ways to inject change. We talked about crossing over during meiosis. We talked about mutations such as base substitutions, gene rearrangements. I know you discussed these yesterday. Inversions and duplications and those kinds of things. And also DNA sensitive to mutagens such as ultraviolet radiation or harmful chemicals in the environment. And then finally, how is it complex enough to determine the phenotype? We know that it's the nucleotide sequence of the gene that determines the amino acid sequence in a protein. And actually tomorrow your lesson is going to go over exactly how that happens in the 2 steps in making a protein called transcription and translation. There are infinite base sequence combinations that can lead to the all of the different kinds of proteins that need to get constructed. And since proteins are responsible for the phenotype, there is infinite variety in the way that those proteins are produced. And then again, probably not today, but after you watch tomorrow's lesson, you can describe transcription and translation. So that was a long question. And if you wanted to try your hand at writing that, you should give yourself about 22 minutes. Okay, so a little bit more practice. This is a model of the molecules involved in DNA replication and their placement relative to each other. So you can see topoisomerase, you can see helicase, you can see RNA, and you can see the DNA double helix, and you can see the direction of those strands. The top strand and the bottom strand have different direction. And you kind of have to look at the unwound portion to really get that. Okay, so figure 1 is a model including molecules involved in DNA replication. Which of the following correctly explains where DNA replication will begin on the strand oriented in the 5' to 3' direction? We're reading from left to right. Okay, A, DNA replication will be randomly initiated along the unwound portion portion of the DNA strand since base pairing will occur. Okay, so you have to think, is there a random initiation sequence? B, DNA replication cannot occur since there is already RNA base pairing with the template strand. C, DNA replication will be initiated immediately to the left of the RNA since DNA polymerase requires an RNA primer. D, DNA replication will be initiated at the site of the topoisomerase since it is where DNA begins to uncoil. Okay, so that's just wrong on 2 counts. The helicase is where it begins to uncoil, creating the replication fork, so that one is just wrong. And we know that B is wrong because the RNA primer is necessary for the DNA polymerase enzyme to know where to start. So our correct answer, I hope you guys got that, is C. All right, so let's do a quick check for understanding for our science skill again. We want to be able to explain relationships in the process of DNA replication in a model that's represented visually. So here you have a model that is showing you the process involving nucleic acid. They don't tell you what it is, but it's a model of the process involving nucleic acid. Which of the following best explains what process is represented in figure 1? A, new DNA strands are being synthesized in the 3' to 5' direction from their DNA templates. B, new DNA strands are being synthesized in the 5' to 3' direction from their DNA templates. C, the new RNA strand is being synthesized in the 5' to 3' and from its DNA template. And D, 2 new RNA strands are being synthesized in both directions from their DNA template. So I think you know that we can eliminate C and D, and now we need to understand our directionality. So is it going to be going in the 3' to 5' or the 5' to 3'? Hopefully you know that DNA is always going to be synthesized in the 5' to 3' direction because the new nucleotides are always added to the free 3' end. So here's your debrief. You should now be able to describe the structures involved in passing hereditary information from one generation to the next. You should be able to describe the characteristics of DNA that allow it to be used as the hereditary material. You should be able to describe the mechanisms by which genetic information is copied for transmission between generations, and you should be able to explain the relationships between different characteristics of biological concepts, processes, or models represented visually in life context. And as has been our key takeaway for the last many lessons, heritable information provides for the continuity of life. All right, so let's take a look at a FRQ that was given on a recent AP Biology exam to look at some common errors and misunderstandings in this idea of DNA structure and replication. So first of all, figure 1 is showing you a COMET assay, which is an assay or a test that's used to detect double-stranded breaks in DNA. Okay? Before this question, nobody knew what a COMET assay was, and in fact, you don't really need to know because you're given all the information that you need in the question. So let's read it together. A COMET assay is a technique used to determine the amount of double-stranded breaks in DNA, which shows DNA damage in cells. The nucleus of an individual cell is placed on a microscope slide coated with an agarose gel. An electric current is applied to that gel, and that causes the DNA to move. And that process is called electrophoresis, and the DNA is stained with fluorescent dye. When viewed using a microscope, undamaged DNA from the nucleus appears as a round shape, which is the head, and fragments of the damaged DNA extend out from the head, which is called the tail. The length of the tail corresponds to the amount of damage in the DNA. And I'd like to point out one more thing. In this diagram, you've got a negative side and a positive side. Part A, to explain the movement of DNA fragments in the COMET assay, identify one property of DNA and provide reasoning to support how the property contributes to the movement during the COMET assay technique. And then in part B, in a different experiment, cells are treated with a chemical mutagen that causes only nucleotide substitutions in DNA. Okay? Remember what the COMET assay detects? It detects double-stranded breaks in DNA. It asks you to predict the likely results of a COMET assay for this treatment. Okay? So let's look at the scoring guideline for part A. You needed to identify that DNA has a negative charge, which means that when you place that electric current on it, it will move toward the positive pole. The other thing that needed to be identified is that DNA can be at different sizes, and we know that different sizes of DNA fragments will move at different rates in an agarose gel. The smaller ones move more quickly because the gel is porous, and the small fragments can travel through the pores at a different rate, at a faster rate, and the large fragments have a more difficult time getting through those pores. Okay, so that part was worth 2 points, and you needed to get both the identification and the reasoning to be able to get those points. And then you needed to make a prediction about what the likely result of COMET assay would be if you treated it with a chemical mutagen that simply causes a substitution. And again, you would have a prediction that there would be a head only or a head with no tail, and then the tail will be shorter than with a double-stranded break in DNA. So you could have said either of those predictions. Okay, so let's read a student response. That was a perfect 3-point response. DNA is negative. Therefore, when applying electric current, DNA will move towards a positive charge. Smaller pieces of DNA move through the agarose gel more easily than larger pieces, which is why the smaller damaged fragments of DNA are drawn out more towards the positive charge, or the tail. And in part B, all of the DNA will remain in the head, the nucleus, because with only nucleotide substitutions, there won't be any damaged fragments to travel outside. So what were some common misconceptions or knowledge gaps? So in part A, students often identify DNA as either a positive charge, or uncharged, or polar, or has both negative and positive ends. So there was general misunderstanding of the idea that DNA is negatively charged. Some students thought that the charge of DNA depended on whether it was damaged or not, and that negatively charged molecules were attracted to the negatively charged end of the gel. So be straight on DNA is negatively charged due to all those phosphate groups, and it will move toward the opposite charge, which is positive. So a response that demonstrates the understanding of the charge of DNA. DNA is negatively charged, therefore during electrophoresis, it moves toward the positive end of the gel. And then in part B, many students were unsuccessful at predicting the results of that COMET assay. Some students predicted that the results of the assay would be the same, or they would be different, but they did not give a comparison. Okay, so that's just remembering to complete your thought. A misconception commonly seen was the lack of a tail would mean the COMET assay is inconclusive. Okay, so a common misconception or gap in knowledge was that there were incorrect predictions, and a response that demonstrates understanding is there will be a head and no tail since the DNA is broken in nucleotide substitution. All right, so here's your homework. This is your homework for tomorrow. You'll be with Ms. Pfizer tomorrow, so she'll review this with you. And tomorrow's lesson will be on protein synthesis. So again, this is looking at 4 samples of nucleotide distribution in 4 nucleic acid samples. And the researchers were studying new viruses. They analyzed genetic material found in 4 different virus samples to determine the % nitrogen-based composition of each virus. The data are shown in the table. Your question is, which of the following samples most likely contains double-stranded RNA? Okay, so this is a little bit different than we've been talking about. So taking the information that we have learned in this lesson and applying it to a new situation. Okay, so I wanted to end today by giving you a little bit more information about some additional resources that you can find on AP Central. The first thing you need to do is log in at myap.collegeboard.org. Use your username and password to sign in. Oh, by the way, if you don't have an account, you can create an account. Then you're going to scroll over to where it says AP Classroom and click there. And also notice that it tells you when your exam day is and if you've registered for the exam or not. Then there's a little bit more information here. You can click here. This is where you can see your AP Live class that you're watching right now. And then this is where you can get some practice answering free response exam questions. So click there for optional practice questions. And what you'll see is all of these are unlocked for you. And you can look at what unit and then what the free response question is going to be going over. Is it concept analysis? Is it interpreting and evaluating experimental results? So you can look at what unit and what skill you want to practice. And then you click begin. Okay. So then you go ahead and answer your question. You can actually write in your response in the place provided. And then you're going to get a prompt that's going to say, are you sure you want to submit your assessment? Click yes. And then once you click yes that you want to submit, you'll get a box and it will ask you if you want to review your the responses, the scoring guidelines basically for this question on the AP exam. So if you want to review, check it out and then revise your answer, you click yes. And if you're good with your answer, you know you already got the best score you can get, and click no. And then what you'll see if you click yes is you'll see examples of what a response should look like to earn the maximum number of points. That's super useful. And then you can see how it scored on the AP exam. So click yes, review. or no, submit now. Okay. So you have a to-do list, and your to-do list is make sure that you have your device and internet connection all ready to go. Check to make sure your email address and phone number are in your AP account, and tell your teacher if you are testing. Thanks for watching. We hope you'll join us again. This is the last week of our review lessons, so make sure you do your practice question for homework. Ms. Pfizer will review it with you tomorrow when she gives her lesson on protein synthesis. So thanks for watching, and I'll see you again on Thursday.
Hi, I'm Gail Pfizer from Dartmouth Cookman Middle High School and Florida State College in Jacksonville, Florida. Lesson overview for today. We're in Unit 6, Gene Expression and Regulation. The topics include 6.3, Transcription and RNA Processing, and 6.4, Translation. By the end of this lesson, you'll be able to describe the mechanisms by which genetic information flows from DNA to RNA to protein, describe how the phenotype of the organism is determined by its genotype, and predict the causes or effects of a change in or disruption to one or more components in a biological system based on biological concepts. So grab your pen and paper. All right, for our warm-up, which was last night's homework, and it was from the DNA Structure and Replication from Ms. Katay. And here we have researchers studying new viruses analyze the genetic material found in 4 different virus samples to determine the percent nitrogen-based composition of each virus. The data are shown in the table. So we have a table based distribution in 4 nucleic acid samples. We have sample 1, 2, 3, 4, and then we have nitrogen-based adenine, cytosine, guanine, thymine, and uracil. And the question is, which of the following samples most likely contains a double-stranded RNA virus? Okay, so I'll give you just a moment if you're joining us for the first time, and if you did your homework last night, just sit tight and we'll go over the answer in just a moment. I'll get my clock going in the meantime. Okay, so we need to tear this table apart, and we can, if we're talking about RNA, then anything that has thymine is not going to be an option, right? Okay, and now we're down to 2. So let's look at the A's and T's and C's and G's and see if they are about the same. So here we have 21% adenine, and uracil we have 19%. Close, but maybe we could do better. And then cytosine, guanine 33 and 27. So we're looking at our A's and U's and our C's and G's. So here we have adenine 27, uracil 27, cytosine 23, guanine 23. So that's my choice is sample 1. And let's see if that's right. Yes, it is. I hope you chose A. All right, so before we get started with what we need to know, I need to give a shout out to Jack, who is in Ms. Deal's class in Camp Hill, Pennsylvania, and he asked me to tell his teacher that the students in her class appreciate all the hard work that she is doing to help them. Thank you, Mrs. Deal. All right, so genetic information flows from a sequence of nucleotides in DNA to a sequence of bases in a messenger RNA molecule to a sequence of amino acids in a protein. So that's a general overview. So we're going to go from, oops, still not what I want it to be. All right, so we're going to go from DNA, okay, and then we're going to transcribe to RNA, and then we're going to take that RNA, and we're going to then end up with a polypeptide chain and protein, okay? So that's the sequence that we'll talk about them. All right, so first of all, RNA polymerases use a single template strand of DNA to direct the inclusion of bases in a newly formed RNA molecule. This process is known as transcription. So what's going to happen is a enzyme RNA polymerase is going to unzip this DNA, and it's going to assemble a strand of messenger RNA, and it's going to add nucleotides to the 3' end. Okay, so it started at the 5' end, and it will add them at the 3 as it goes. All right, the DNA strand acting as a template strand is also referred to as a non-coding strand, the minus strand, or the antisense strand. Selection of which DNA strand serves as a template strand depends on the gene being transcribed. So let me share with you how I look at this. Okay, so the first thing that I can see right here, that's not what I want. All right, so the first thing I can see is that I have a 5' end, and there's my AUG. So if this is my messenger RNA, I know it's going to be almost identical to my coding strand, okay, of my DNA, for the exception of my U and T. Okay, so this would be there's an ATG and there's AUG. So this is my coding strand. Now my non-coding strand is then called my template strand. Okay, template strand. All right, so template. So my template then is also my minus strand, and this would be my plus, and then this is antisense. Well, I could go antisense, and this is sense. Okay, now depending on if there's where I don't want you to do this, because sometimes happens in my room. This question comes up. Is it so the top one is the coding strand, the bottom one? No, you can't look at it as top or bottom or left or right or whatever in the diagram. You have to look at the direction of the 5' and 3' ends of the molecule, and the gene can be on either side. It just depends on which gene is being transcribed at the time. All right, so the enzyme RNA polymerase synthesizes messenger RNA molecules in the 5' to 3' direction by reading the template DNA strand in the 3 to 5 direction. So like I just said, it's going to read this is your template strand, right? And so this will be your messenger RNA will be assembled to be complementary to that template strand. So here's T, A, U, etc. And it's assembled in 5' to 3'. So nucleotides are added at the 3' end. So anything that you're making new, okay, so 5' to 3' is your direction. Okay, everything starts with 5, ends with, and then is added on the back at the 3' end. Now that goes for messenger RNA. It also goes for when you're replicating DNA. All right, so this isn't the most ideal diagram, but I kind of took 2 from the test bank and kind of merged them together to say what I wanted to say with this. And eukaryotic cells, the messenger RNA transcript undergoes a series of enzyme-regulated modification: addition of a poly A tail, addition of a GTP cap, excision of introns, and splicing and retention of exons. So we're going to start here with, so here is our DNA, okay, and if you'll notice, there are different parts to it. They're the white bars that I just covered up. I covered those. Okay, those are the introns, and I tell my students the introns are going to stay inside of the nucleus, okay, and the exons are then going to be joined together to make the gene that's going to be, well, to make the messenger RNA that's going to go out and be read on the ribosome. So this has to happen. Introns have to be cut out, okay, but also we've got to put a 5' cap and a poly A tail on our, and there's many, okay, 300 of those A's, and we're going to, because we have to be able to get out of the nucleus, and we have to make sure that no enzymes degrade us on the way to our ribosome. So now we've cut out the introns, they're inside. We're only left with our 5' cap, our poly A tail, and our exons together, and some untranslated regions that won't be read into the, that won't make the polypeptide chain, won't be part of that. All right, so now after this is called messenger processing is what it's called. Okay, so when you have messenger RNA processing, I gotta get off of these pinks, they're not showing up. All right, messenger RNA processing. Okay, so it's not true mature messenger RNA until these steps have happened, and so now that's the processing. It happens inside the nucleus. All right, so and speaking of, this isn't ideally this image doesn't ideally match what I'm saying, but I think that you'll get it. Excision of introns and splicing and retention of exons can generate different versions of the resulting messenger RNA molecule. This is known as alternative splicing. I'm going to work backwards with this. So here we have this receptor. Okay, here we have this receptor, and it the ligand that will fit in it will be, here it is right here, and that will go right there. Now it was coded for by this strand of messenger RNA, which is different than this strand of our messenger RNA, but this messenger RNA will code for this receptor that will bind to this ligand. So now these are totally different, and they came from the same gene. But what's being cut out? See, we have segments 1, 2, 4, and 5 in this, so we use that one, those, and so those were the exons that were put together to make this messenger RNA, and 1, 3, 4, and 5 were the exons that were put together for this. So 2 was not an exon; it was actually became an intron to do this version. Is what I'm trying to say. All right, the sequence of the RNA bases together with the structure of the RNA molecule determines RNA function. Messenger RNA molecules carry information from DNA to the ribosome. Distinct transfer RNA molecules bind specific amino acids and have anticodon sequences that base pair with the messenger RNA. Transfer RNA is recruited to the ribosome during translation to generate the primary peptide sequence based on the messenger RNA sequence. Ribosomal RNA molecules are functional building blocks of ribosomes. So these are just the players. Okay, I'm going to go through the whole process on different slides, but these are the players involved. We're going to have our messenger RNA. Here's our 5' end and our 3' end. We're going to have our ribosome, which is made up of ribosomal RNA. Okay, Okay, so ribosomal RNA is going to interact with our messenger RNA. This is our transfer RNA. It's going to bring in our amino acid. It has anticodons that will be complementary to eventually codons on this messenger RNA on the ribosome. Okay, all right, so we're going to start from the beginning. The salient features of translation include translation is initiated when ribosomal RNA in the ribosome interacts with the messenger RNA at the start codon, and the sequence of nucleotides on the messenger RNA is read in triplets called codons. So here we have our small subunit, and we have our messenger RNA has engaged with this ribosome, the small subunit, and now the transfer RNA. This is AUG. This is our start, and this is UAC, which is the complementary A to U, U to A, GC, and this is going to bring in methionine. And now that all these players are together, then the large ribosomal unit will join them, and now we have our polypeptide factory in place. Okay, all right. Each codon encodes a specific amino acid, which can be deduced by using a genetic code chart. Many amino acids are encoded by more than one codon, and we'll talk about that a little bit later. All right, so let's start from the beginning. So here's our messenger RNA, and we can break this down into codons now. Okay, so there's AUG, CCG, CAA, UCU, GUU, CAC, GCA, CUC, AUG, and then I don't have enough to complete there, so we'll stop there. Now, so this will be our AUG, and this will be our CCG, and then we have CAA and UCU and GUU, and where are we? CAC and GCA. And I'll stop in a minute. GCA, CUC, and then AUG. Okay, so so we had AUG. So AUG, we went to our genetic code chart, and we can see that here's AUG. So this amino acid that was brought in, okay, was in fact methionine, and it would be this one right here. Okay, for the orientation of where the amino acids are on the strand. Okay, so then that was the first one, and then the second one, the CCG, we'll find it, CCG. Then we have proline, and that would be proline would be right here. Okay, so that one's done. Now CAA is glutamic acid, and so we can add this. Okay, that's done. And now presently in the P spot of the ribosome, okay, we have UCU, and we can make it that color. And now the next one that's coming in, now we can do a little bit more work with this. It's GUU, so now we have GUU, and there's valine. Okay, and so this is valine, and now this anticodon that was complementary of this would be CAA, right? Okay, so what I don't want you to do is try to find the amino acid with the anticodon. Okay, you look for it with the codon. Okay, that's enough on that one. I think that hopefully you got it with that if you are having trouble with the genetic code. All right, translation involves energy and many sequential steps, including initiation, elongation, and termination. Transfer RNA brings the correct amino acid to the correct place specified by the codon on the messenger RNA. The amino acid is transferred to the growing polypeptide chain. The process continues along the messenger RNA until a stop codon is reached. The process terminates by the release of a newly synthesized polypeptide, or soon to be protein. Okay, so here we have our ribosome, our large unit, small unit, everything that we needed to get started. Here's our first transfer RNA. It's going to take place. If you remember, we had spots. We had an arrival, a polypeptide spot, and an exiting spot. This comes in, 5' in, heads in this way. That's why it's over here on the side. Okay, so now we have this amino acid. The next one will come in. Let's go to step 2. So now we have 1, 2, 3, 4 on our fourth one. Now this empty transfer RNA will move out of the exit site. This one in the P site, the polypeptide chain will then covalently bond to the amine end of this amino acid, and you'll see that in the blue one this will move here, and then that will all be strung together like this right here. So as we come in, this attaches, this bonds with this moves over, and then the other one moves out. And now we have our polypeptide chain, and now we have a release factor that has been coded for with one of those 3 stop codons. There's no amino acid coming in with the stop, so this will be released and then go off for further processing. All right, translation of the messenger RNA to generate a polypeptide occurs on ribosomes that are present in the cytoplasm of both prokaryotic and eukaryotic cells and on the rough endoplasmic reticulum of eukaryotic. Okay, so here's some ribosomes, and these are in the cytoplasm. This is a eukaryotic cell, and then we have some on the endoplasmic reticulum also, and then so that's eukaryotic here. We have them in 2 places, endoplasmic reticulum and in the cytosol. And now here we have them in our prokaryotic cell. Okay, and I'll talk about this in just a second. All right, in prokaryotic organisms, translation of the messenger RNA molecules occurs while it's being transcribed. So there's no nucleus, there's not processing, but we do have transcription going on, and then immediately we can go into translation for that protein to be synthesized, and it happens on more than one ribosome at a time. All right, nearly all living organisms use the same genetic code, which is the evidence for the common ancestry of all living organisms. Whether I always tell my students, whether you are are a cat or a dog or a fish in our house, that we could be our genetic code. If we had a CUC, it's still going to be leucine. All right, genetic information in retroviruses is a special case and has an alternate flow of information from RNA to DNA made possible by reverse transcriptase, an enzyme that copies the viral RNA genome into DNA. The DNA integrates into the host genome and becomes transcribed and translated for the assembly of new viral progeny. So here we have our virus, and we see that the envelope plasma membrane is going to become part of the plasma membrane. We fuse with the plasma membrane of the cell, and then it's going to empty its contents. And then you can see that the virus has these protein capsids. The little black line is the RNA. The little green is going to represent our reverse transcriptase enzyme. And now let's look at that. Here's our RNA, reverse transcriptase, and with that reverse transcriptase enzyme, we can make a strand of DNA. And then we'll have a strand of DNA have a complementary strand of DNA. And now once we have a double strand of DNA, then we can be inserted into the genome, and then messenger RNA can be transcribed, because the point is to make more viruses. So now we need more membrane parts, we need more protein receptors, we need more capsids, we need more enzymes, we need more of everything before we can exit the cell and leave. But the point of the retrovirus part is that it started out as RNA, and then it made a strand of DNA, and then it made a strand of RNA again to make the protein. Right now, usually we start with DNA and we go to RNA, and then we have protein. So we did things a little bit backwards than usual, and that's where the retrovirus comes from. All right, so check for understanding. Here's a question. Figure 1 represents part of the process that occurs in eukaryotic cells. There are untranslated regions, UTR, in the sequence. So here's what we're looking at. We have a 5' cap, we have poly tail, we have some exons, we have all the parts we need, and we can see that introns have been cut out of this strand of DNA. Okay. All right. So which of the following best explains the process represented by figure 1? A, the synthesis of messenger RNA in the 5' to 3' direction. B, the modification of a protein to produce a functional form of that protein. C, the translation of messenger RNA molecule into a polypeptide. Or D, the enzyme regulated processing of pre-messenger RNA into mature RNA. So it had nothing to do. We're not synthesizing a new strand right now. We're already past that, and we're not at the modification part that's after translation, and then we're not translating. We're not a ribosome right now. So now this is our answer. That's what's going on. This was pre, and now we have mature, and we're ready to go to the ribosome. Okay. All right. So a lot of the questions today are going to have to do with changes. So this was the most appropriate science skill to talk about. Predict the causes or effects of a change in or disruption to one or more components in a biological system based on biological concepts. We're going to do some calculations. Very simple. Provide is a word that might come up. Predict. We're going to describe. Remember, describing. Just list the characteristics. And there's a certain describe that I want us to take us back to what describe really means. And then explain is how or why, and justify, claim, evidence, reasoning. Okay. All right. So Loeys-Dietz syndrome type 2B is a human genetic disorder associated with mutations in the LDS2B gene. Researchers have proposed that the LDS2B gene produces a growth factor receptor protein that regulates cell signaling pathways. The predicted product of the gene is a 565 amino acid protein with structural regions that bind to signaling molecules, span the plasma membrane, and have protein kinase activity. Protein kinases are cellular enzymes enzymes that transfer phosphate groups from ATP to target proteins. A representation of the LDS2B gene is shown in figure 1. So we can see that we have a start to our gene and a stop, and we have a transmembrane region and some kinase regions, and the dark shaded, and then the lighter is signaling molecule. Okay, figure 1, genomic structure of the LDS2B gene. The 7 exons, so that's what we're talking about right here. These boxes are the exons, and so the little lines would then be the introns. The 7 boxes, numbered rectangular boxes. The introns, thin lines between the boxes, and the start and stop signals for translation are represented. The portions of the gene that encode the different structural regions of the protein are shown, and the location of a methionine to valine substitution at position 425 of the encoded polypeptide M425V is indicated. So we're going to go, there's going to be a change. Okay, we're going from methionine, remember this because when I turn the page, methionine to valine. Okay, now we have our genetic code. Let's calculate, first thing we have to do, calculate the minimum number of nucleotides required in the coding region of the LDS2B. So minimum number of nucleotides, okay, to produce and terminate the 565 amino acid polypeptide. And I'll stop right there. So let's take, I'm going to just think out loud, 565, and that is how many amino acids we have, right? So how many codons did it take to do that? It took 3 each. There's 15, 18, 19, 16, all right, 16, 95. Okay, now it said a hint word here, and terminate. Even though we don't have an amino acid, right? We don't have, there's still something that's needed to code to stop. So let's add another 3 to that, okay? So let's keep that number, 1698. This is simple math. There still could be simple math on your exam. You don't need a calculator, you won't be graphing, but simple math could actually still be there. Provide 2 reasons that the number of nucleotides in mature messenger RNA may differ from the number of base pairs in the genes, okay? So we're talking, I'll give you just a second to think about that before I start adding in. Okay, so what did you think about? Did you think about when we cut out the introns, right? We cut them out. So there were introns in the DNA. So that would be a difference. We added a 5' cap. That would be a difference. And we also added a poly A tail. All right, that would be a difference. All right, so that's all I can think of, right? I think there's probably 6 or so options. I just can't remember them all. All right, one genetic change associated with the disorder results in methionine to valine substitution at amino acid 425, as shown in figure 1. Using the codon table in figure 2, predict a DNA point mutation that result in methionine to valine. So let's look at what did it take to make a methionine. It took this, right? AUG. And we ended up with valine. So valine could be any one of these, but what's the closest to it? That only one single point could be changed. It could be this one, right? So if it was GUG, then it would be valine. So this one letter. So we could have just a change from A to G, or if this was the DNA, and because this is the messenger RNA, remember? So this would have been a T to a C. Okay. All right. So let's finish up. I think we might be finished with this question. Let's see. Here we go. So here's the first part. Did we say 1698? Yes, we did. Response demonstrates understanding of the triplet code of the polypeptide, and response demonstrates understanding of the role of the stop codon in terminating translation. So there's our 1698, and there's 1698. All right. The next part, we did say that it was processed, and introns were removed, and that a 5' cap was added, and poly A tail adds nucleotides to the 3' end that are not encoded by the gene. We didn't talk about this, and we didn't talk about that. But what I wanted to make a point to say to you is this work that I do with you here. This is my planning work. So if I plan before I start writing, and then I put it into full sentences that are complete thoughts, okay, that bring all the pieces together. Like when we said that it has a 5' cap. Here's a 5' cap adds a nucleotide that is not encoded by the gene. You should say more than just 5' cap, okay? Because that would be more like an identified answer. Now let's look at the bottom. We have A to G because it was AUG to GUG, and also you could have said T to C, or you could have said a combination of the two, the A/G or the G/C. All right, now for a short FRQ. I know this one looks a little bit different than usual, and I got it right out of the CED. There's two FRQs in the back. There's a short one and there's a long one, and I put a copy of the short one is in I think maybe 2 lessons ago in one of the PowerPoint decks that I didn't have time to get to in the presentation. But anyway, they're both in the CED, so they're not a secret. They're available to everybody. All right, so in many countries, Anopheles gambiae mosquitoes are responsible for transmitting the parasite that causes malaria to people through their bites. A primary tool for mosquito control is the use of insecticidal nets sprayed with chemicals known as pyrethroids, which are relatively safe for people but toxic to mosquitoes. However, mosquito resistance to pyrethroids have now become widespread. Pyrethroids interfere with the function of the transmembrane sodium channel found in the cells of mosquitoes, and that's in figure 1. In one common mutation to the channel protein, a phenylalanine is substituted for a leucine at amino acid position 1014. Scientists hypothesize that this mutation is responsible for some cases of pyrethroid resistance. Figure 1 is a schematic drawing of the transmembrane sodium channel targeted by pyrethroids and other insecticides. The arrow points to the position of the amino acid 1014. To investigate pyrethroid resistance, mosquitoes were collected 4 times over a 2-year period from the following region. In region A, a southern vegetable growing region where large amounts of insecticides are, okay, and region B, a northern rice growing region where very little insecticide is applied. Okay, let's see what else. Scientists exposed the collected mosquitoes to filter papers soaked in 2 different pyrethroid insecticides, deltamethrin and permethrin, and the percent mortality of the mosquitoes was determined after 24 hours. The scientists simultaneously determined whether leucine or phenylalanine was encoded at position 1014 by each of the 2 copies of the sodium channel gene. All right, so now we have the table looks a little bit different because now we have our bars heading this way. Now look, here's percent mortality, and here is our regions. We had a region that had a lot of insecticide use, and then one that didn't, remember. And then we have our months. So figure 2, susceptibility of A gamma E mosquitoes from 2 regions to the pyrethroids, deltamethrin and permethrin. A mosquito strain that is acceptable to the insecticides displayed at least 95% mortality in all experiments. Mosquitoes exposed to untreated filter paper displayed less than 10%. Just giving you some additional details besides what they are. Error bars represent standard deviation, and ND means no data are available. All right, here's another table. Frequencies of leucine and phenylalanine at position 1014 of the sodium channel is looking to see if here it is region A, and it looks like most of them are homozygous for phenylalanine, and then B, it looks like there is a combination, but most are homozygous for leucine. Okay, here's our job. Describe the most likely cause of the amino acid substitution in the sodium channel protein. Okay, so A, think about what you think A is. Don't overthink it. Okay, in my planning, I'm probably going to think that there is now this is described, most likely cause. So there could be a mutation or a change in what? What am I talking about in my description? In the DNA, right? Okay. Explain how the substitution of a single amino acid in the channel protein could cause pyrethroid resistance in mosquitoes. Okay, so now if we had the substitution, what could happen? So if we had the substitution, we could have a change, right? We could have a change in our amino acid sequence, which could mean a change in our protein, which means a change in the way it folds, which means a change in the way something can bind or can't bind, which changes its function. Okay. Identify the dependent variable in the experiment whose data are graphed in the figure 2. So let's go back to figure 2 and find the dependent variable. So let's get rid of some of all this. So what do you think is the dependent variable here? Should be here. here, but that's what we're changing. So this is kind of flipped, right? So here is our dependent variable, and there is our, this would be what we're changing. The two is kind of flipped. All right, so if we just did it inside, it would be fine. All right, now what's the other? Whoops. Now identify the positive control in the experiment, and let's find that. It told us that. It said a mosquito strain that is acceptable to insecticide displayed at least 95% mortality. So that's what it looks like when it is acceptable, okay? And then the next thing, justify exposing some mosquitoes to untreated filter paper each time the experiment was performed. Remember, there's some things you should write about when we're talking about what the control is. The control, that's number one, and then there's no treatment, and then that whatever is happening in your experiment is happening because of something else, and not because of the it's happening because of the treatment, not because of the filter paper in this case. Okay, now let's check and see how we did. All right, a change occurred in the DNA sequence encoding the sodium channel polypeptide. Okay, now you should have taken that sentence that we talked about, and we said mutation change in the DNA. Okay, did a pretty good plan on that one, but I didn't go as far as saying what I was changing, what we ended up changing. Okay, explain how the substitution of a single amino acid in a channel protein could cause pyrethroid resistance by mosquitoes. The substitution could change the shape of the protein channel so that pyrethroids can no longer affect it, bind to it, or interfere with its function. Identify, we got that. Identify the dependent variable, percent mortality. Okay, identify the positive control, testing the strain that is acceptable to the insecticide. Justification of exposing some mosquitoes to untreated filter paper each time the experiment was performed. Okay, it says exposing mosquitoes to untreated filter paper confirms that any observed mortality is from the insecticides rather than the filter paper. Okay, or any other experimental condition. Okay, there were two other parts to that question in the back of the CED, but they had to do with evolution, and so they went into topics that were not included in the units 1 through 6, but you could go find them. All right, so here is one more. A processed eukaryotic messenger RNA lacking introns is introduced into bacteria so that the bacteria can produce a large quantity of the encoded polypeptide. Translation of the messenger RNA to produce a polypeptide involves the binding of large and small ribosomal subunits to messenger RNA. Each complete ribosome has 3 tRNA binding sites that align with 3 sequential codons in the messenger RNA. 3 steps in the process of translation, elongation, the addition of amino acids to a growing polypeptide chain are illustrated in figure 1. So this is figure 1. You've seen it. I've used it actually a number of times already through these videos. A schematic drawing of 3 steps in translation elongation. Sequential codons are numbered in AA, okay, right here, that represents an amino acid. So the first thing we need to do is we need to describe why translation of a messenger RNA can start before transcription of the messenger RNA is complete in prokaryotes, but not in eukaryotes. So I'll give you just a moment to think about that. Okay, and go ahead and jot some thoughts down. And then referring to figure 1, explain the process illustrated between step 1 and step 2. What happened in step 1? We had this polypeptide chain is going to be then covalently bonded to this next amino acid that's coming in. So there we have 1, 2, 3, 4. 1, 2, 3, and there's 4. And so when you explain that, you need to say amino acid 3 was then joined to amino acid 4 on the amine end covalently bond, and then this is going to move to the exit site, and this is now going to move to the polypeptide site, and then the next amino acid. Okay, do all those details when you explain what's going on here. All right, let's check. I think that I have C and D before we get to the scoring guideline. Okay, this is my favorite part. All right, fill in the boxes in the template to identify the nucleotide sequence from 3 to 5 of the transfer RNA anticodon that will recognize codon 5 and the transfer RNA anticodon that will recognize codon 510 of the messenger RNA template, and to identify the amino acids that will be added at these 2 positions. So I know we've already did this like 10 times, but we're going to do it 2 more. So here we have a CGG, that's our amino acid at location 5. We're going to go C and then G and then G. And what is that? It's arginine, right? Okay, and so that took, I had to bring that in. It had to be a GCC. Now at position 510, there is going to be a UAG. So now let's look at U, A, and G. And now we have a stop. So UAG is then going to have nothing, right? There's not going to be an amino acid. There's not one. And that anticodon then would have been what? It would have been the UAGs would have been A, and I can't think for some reason. There we go. All right, did I do that right? Okay, so there we have stop. That's the point. Stop's coming in there. All right, now let's check. The response indicates that the prokaryotes do not have a nuclear envelope that separates the genome from ribosomes in the cytoplasm of the cell, where eukaryotes do. I hope that you said that the nuclear envelope brain was an essential part of that sentence, separating the genome from ribosomes in the cytoplasm, and we in eukaryotes, but the prokaryotes do not have to do that. The response, and then B, the response indicates that the growing polypeptide chain attached to the transfer RNA aligned with codon 3 of the messenger RNA is transferred to the amino. Yes, that's what I said. You need to say all of those features attached to the transfer RNA aligned with codon 4 of the messenger RNA. And then 5 we said was arginine, right? And then 510, that's why I knew that. One of my little what do you call it? My indicator to stop. There's not an anticodon bringing in a stop. There's no stop. Okay. All right. And then D, the response indicates that the polypeptide produced by translation of a processed eukaryotic messenger RNA by a eukaryotic cell or a prokaryotic cell into which it has been introduced should have the same amino acid sequence because the genetic code is almost universal, and the same codon chart can be used for both prokaryotic and eukaryotic organisms. Okay. I hope you have time to go back and look at this question. I really like this question. I think it's a good learning tool to learn how to do protein synthesis and how to read the codon chart. One of my personal favorites. All right. Debrief and summary. The takeaway for the week: heritable information provides for continuity of life. I know that you're able to do all of the learning objectives for the week, and I know that we are constantly on this predicted causes or effects of a change, but that's super important. It's one of your more difficult science practices. Common errors and misunderstandings. Don't refer to DNA as the bottom and the top. Each has a role depending on the gene that is being transcribed. And then use the codon on the genetic code chart, not the anticodons. And now your homework for tonight, you have one question, and you're going to look at figure 1, model of process involved in gene expression. Which of the following best explains what strand X represents? And you have 4 options: A, a complementary RNA sequence because it contains thymine; B, the coding strand in this process because it's being read 3' to 5'; C, the antisense strand because it's serving as a template; and D, the pre-messenger RNA because it does not yet have a GTP cap. So you do that for your homework, and Ms. Kate will go over that first tomorrow. All right, so before I let you go, and look at this, I have a few things I want to say after the slide, so don't go. All right, so your to-do list: check your device and internet, okay? Check your email address, phone number. I have more to say about those, or you have more to read about those. So your teacher testing, practice questions, okay? That green button, and be sure to do your homework. And then Ms. Kate will be with you tomorrow evening for gene regulation. And now I want to go over just a few slides. I put this together this morning after this was released yesterday. So if you go to cb.org, you'll get to this page, okay? And then you click on this blue up-to-date information, and then that's going to take you to this page. And you can go either way depending on if you're a student. You're not banned from this updates for educators, okay? This information, if you're not able to contact your teacher, you need to be reading it yourself anyway. So updates for AP educators, and then learn more about taking the exam, and then click on this 2020 AP testing guide, and it is a fantastic, very clear, concisely written testing guide, okay? There's not even though it says that looks terrible, right? But it's not. It has simple information and diagrams on each page to walk you through. I walked through my students in 2 periods this morning. I put it on the presentation like you see me present on, and then we walk through each slide and let them ask questions. The most valuable thing that I can tell you in here is a May 4th practice, like a mock submitting a question. That's the day that is to me going to be the most important. But go find that file, read through it, and then I can answer all your questions. And I think that's it for today. All right, thank you.
Hi, welcome to today's AP Biology lesson. I'm Allison Kate, and let's get started with our warm-up today. So yesterday you were given a question for homework, and it is a visual representation of a process that is essential to gene expression. So it's showing you a few things to look at, and the question is, which of the following best explains what strand X represents? So the arrow is pointing here, strand X, and it's asking you to look at the strand, and I'm going to ask you to pay attention to the directionality. So here here's the 5' end, and here's the 3' end, and notice what nucleotides are making up strand X. Okay, so again, which of the following best explains what strand X represents? Is it A complementary RNA sequence because it contains thymine, B, the coding strand in this process because it is being read 3' to 5', C, the antisense strand because it is serving as a template, or D, the pre-mRNA because it does not yet have a GTP cap? Okay, so hopefully you have your answer, and the correct answer is C. It is the antisense strand because it's serving as the template. Let's also look at why the other ones are incorrect. So the first choice, RNA, because it contains thymine. Hopefully you recall from the lesson that I gave you on Tuesday that RNA does not contain thymine. Okay, this is not the coding strand because the coding strand is the DNA strand whose base sequence corresponds to the sequence of the RNA transcript, and it is not D again because it is not RNA. Strand X contains thymine. Okay, so hopefully that one didn't fool you. Okay, so today's lesson is on the regulation of gene expression and gene expression cell specialization, and this is a difficult topic, so I'm going to try to make this as understandable as possible. We're also going to be looking at Science Practice 2C, which is about explaining how biological concepts or processes represented visually relate to larger biological principles, concepts, processes, or theories. So today we're going to look at some visual representations of the regulation of gene expression. From yesterday's lesson, you should be familiar with transcription, messenger RNA processing, and translation. And today some important terms are going to be operon, promoter, and epigenetic. And don't worry if you're not familiar with these, I will be using them a lot today. So the objectives by the end of this lesson, you'll be able to describe the types of interactions that regulate gene expression. You will be able to explain how the location of regulatory sequences relates to their function. That's location of regulatory sequence relates to their function. To explain how the binding of transcription factors to promoter regions affects gene expression and or the phenotype of the organism, and explain the connection between the regulation of gene expression and phenotypic differences in cells and organisms. So here's what you need to know. First of all, let's talk about what a regulatory sequence is. And obviously we're looking at a eukaryotic piece of DNA. We're going to talk about regulation of gene expression in both prokaryotes and eukaryotes, but we're looking first here at a stretch of eukaryotic DNA, and regulatory sequences are stretches of DNA that interact with regulatory proteins to control transcription. So let's kind of dissect what we're looking at here. So first of all, each eukaryotic gene has a promoter, which is a DNA sequence where RNA polymerase binds and starts the transcription process proceeding downstream. Okay, so there's the promoter, and notice that it is labeled green. This is like green for go. This is the start of transcription. This is where RNA polymerase, which is the enzyme that facilitates the transcription of the mRNA transcript from DNA. That's where it will start. Downstream means to the right of that promoter, and upstream means to the left of that promoter. In addition, there are a number of control elements that are involved in regulating the initiation of transcription. These are DNA sequences located near or far from the promoter. The ones that are closest to the promoter are called proximal control elements, and the ones that are farther away are distal from the promoter. And distal control elements can be grouped together, and they're called enhancers. Okay, so you can see that there are several distal control elements grouped together as an enhancer, and that's farther away or farther upstream from the promoter. And then we've got our proximal control elements that are proximal to the control to the transcription site. Did I say distal? Okay, so distal is farther away, and proximal is closer. Okay, so at the other end of the gene, there is a poly A, which stands for many adenine signal sequence in the last exon of the gene that is transcribed into an RNA sequence. And this is the signal where the transcript is cleaved off and the poly A tail is added. I'll talk about what the poly A tail is in a minute. So look at the poly A sequence. It is in that last exon, and that's the signal for where we are going to stop that transcript. Right. So one of the things that's going to happen is once we get our primary transcript, it gets processed, and it will get processed in 3 steps. The first step will add a guanine cap at the 5' end of that mRNA. The next thing that will happen is there'll be an addition of a polyadenine tail at the 3' end, and both of these things are going to protect this messenger RNA transcript once it leaves the nucleus. And then there's some splicing that goes on. The introns, which are the non-coding segments, are going to be removed, and the exons will be connected together. Oops, sorry. And so I also wanted to point out, so we remove our introns, we connect our coding segment, which are the exons, and also here you're going to see this UTR, that's an untranslated region. And so we know that we're going to, when we finally get out to the ribosome, we're going to start translating this mRNA here and here. So there's also epigenetic changes that can affect gene expression through reversible modifications of either DNA or histones. So 2 things to unpack in that sentence. First of all, epigenetic means above the genes, literally. So this is not involving a DNA sequence that is going to be changed. So there's some other way of regulating gene expression. And so again, we're in eukaryotes, right? In eukaryotic DNA, the chromatin is packed using histone proteins. And so what we're going to do is we're going to see that there are some ways to either loosen or tighten the chromatin to either have greater access to those genes or less easy access to those genes. So the inheritance of traits transmitted by mechanisms that are not directly involving the nucleotide sequence is called epigenetic inheritance. And so if you look at the image, amino acids in the histone protein tails may be chemically modified. And I'm illustrating the acetylated histones. So acetyl groups are represented by these green balls, and they may be added to the chromatin to be less compact, which makes the DNA more accessible for transcription. And so you can see where those acetyl groups are, the DNA is less tightly packed together, which you can see in this unacetylated histone. The phenotype of a cell or organism is determined by a combination of genes that are expressed and the levels at which they're expressed. So I'm going to give you an example, and what you're going to see in this example is I've got 2 different types of cells, a liver cell and a lens and eye-lens cell. And we can see that on the actual piece of DNA, on the gene, we have both the albumin gene and the crystalline gene. However, the albumin gene will only be expressed in liver cells. It is a blood protein, and the lens cells will only produce the crystalline protein that's for the eye lens. And the way that happens is in each cell there are different specific transcription factors that the cell make that will determine which genes are expressed. And so if you look at my arrow here, I'm showing you that in this picture we've got just different colors representing the different transcription factors that are able to activate the transcription of this particular albumin gene. And so just to color code it, we've got a red, a gray, and a yellow, and they're going to actually, I'm going to talk about this bending that's going to happen in a future slide, but they are going to actually be able to activate, this is RNA polymerase to activate it to begin to express that transcribe that albumin gene. And that does not happen in the crystalline gene, even though those genes are both present in the liver cell. In the lens cell, there are different activators that are available, and these are color coded in gray and brown, and you can see that they are going to get activated. That's going to allow the RNA polymerase to transcribe the crystalline gene, but not the albumin gene. So that explains why you get different proteins expressed in different types of cells, even though the genes are present in both. Okay, so both prokaryotes and eukaryotes have groups of genes that are coordinately regulated. We're going to talk about prokaryotes first, and then I'll talk about eukaryotes. So in prokaryotes, groups of genes called operons are transcribed in a single messenger RNA molecule. So an operon is a group of functionally related genes that can be coordinately controlled by a single on-off switch, which is called the regulatory switch. This is a segment of DNA called the operator, which is usually positioned within the promoter. And I'm going to give you a graphic to illustrate this. So an operon is the entire stretch of DNA. That includes the operator, the promoter, and the genes that they control. There are 2 types of operons that I'm going to talk about today in prokaryotes. Repressible, which is a system where the gene is on, but you can turn it off or repress it. And the tryptophan or trip operon is an example of that, or an inducible operon, which is one that a system where the gene is normally turned off, it's not being expressed, but it can be induced to turn on. And the lactose or lac operon is an example of an inducible operon. So let's take a look at both of these. First, we're going to take a look at the lac operon, which is inducible. Again, it means that the system is not transcribing the gene needed to break down sugar, lactose, which is a disaccharide, but it can be induced to do so. So first, let's look at when lactose is absent. The repressor is active, which means that it is repressing the transcription of the genes. Okay, so the operon is off. And if you look at what's going on here, we've got our DNA. This is the promoter, the operator, and then we're going to have our structural genes here. This is RNA polymerase that is responsible for transcribing. So when lactose is absent, there is no need to break it down. So you don't need to make those proteins or enzymes that are going to break it down. And so the RNA polymerase is blocked by this repressor, which is sitting in this operator. In the second case, okay, so in the absence of lactose, the repressor switches off the operon by binding to that operator. In the second case, lactose is present. And when lactose is present, it is a nutrient for that bacteria. So it needs to break it down to digest it to get its energy. In this case, we're going to look at an isomer of lactose called allolactose, and it's being represented by this green ball here. And it is going to act as an inducer. It's going to bind to the repressor protein. So you can see that in this part. So it's going to hop right in this binding site, and it changes the shape of that repressor. When that shape is changed, it can no longer fit in the operator, and so the block for the enzyme RNA polymerase is removed. So RNA polymerase will hop on the gene, and it will begin transcription, and it will transcribe these three genes, lac Z, lac Y, lac A, making the enzymes that are necessary to digest lactose. Okay, and that makes sense because when there's no lactose around, you don't need to make those genes to digest it. But when lactose is present in the bacteria's environment, then they need to turn that system off. All right, let's look at the repressible type, which is represented by the trip operon. Tryptophan is an amino acid, so it's similar. It is a necessary nutrient for the bacteria. Again, repressible means that genes needed to synthesize tryptophan are being transcribed, but the system can be repressed. Okay, so in this case, we still have our piece of DNA. We've got our promoter and our operator, and this time we have 5 genes that are going to make the polypeptide subunits that make enzymes that are needed to make tryptophan. Okay, so when tryptophan is absent, the operon is on. That means that there's no tryptophan in the bacteria's environment, so it needs to make its own tryptophan. Okay, and the reason that works is that here's the repressor. It is not bound to the operator, so that operon is on. Okay, now when tryptophan is present in the environment, we no longer need to make it on our own, so we can turn this system off. And so what happens is tryptophan itself is the co-repressor. And again, if you look at the confirmation thing, here's tryptophan. The co-repressor is going to fit into the repressor, and it is going to change its shape. Okay, so now its shape is compatible to fit into the operator, and now when RNA polymerase tries to transcribe this gene, it can't fit on the operator or the promoter, so it's blocked. So that means that no tryptophan will be made, and that makes sense. Since tryptophan is present, the organism does not have to waste any resources making it. Okay, so now we're going to talk about eukaryotes. In eukaryotes, groups of genes may be influenced by the same transcription factors to coordinately regulate expression. Since operons are not used in eukaryotes, the genes that are co-expressed are scattered across different chromosomes. So for coordinated gene expression to occur, it depends on every gene in that dispersed group having a specific combination of control elements. It often occurs in response to a chemical signal from outside the cell. It could either be a steroid hormone or protein hormone that activate transcription factors. And as you might remember from your lesson on cell signaling, steroid hormones act directly to activate or repress transcription factors, whereas protein hormones are going to act via a signal transduction pathway to either activate or repress those transcription factors. So let's take a look at a few examples. So promoters are DNA sequences, again, upstream from the transcription start site where RNA polymerase and transcription factors bind to begin or initiate transcription. So now let's take a look at our DNA. Again, we've got our distal control element, which is farther away from that promoter, and these are our enhancers, and we have our activators, right? We also have our promoter, and this is the gene that we want to transcribe. Okay? So what's going to end up happening is to regulate the initiation of transcription, the DNA control elements in the enhancers are going to bind to these specific transcription factors. And that's going to facilitate the bending of this DNA, and that's going to enable those activators, okay, right here, those activators to actually contact the proteins of the promoter and will initiate transcription. So once the activators have attached to those mediator proteins, then we have a transcription initiation complex. And as you can see, RNA polymerase will be able to bind to the DNA and it will begin transcribing that gene. So there are some negative regulatory molecules that inhibit gene expression by binding to DNA and blocking transcription. And the main way that I'm going to talk about here is through DNA methylation, which greatly reduces transcription because it's going to prevent the RNA polymerase from actually getting into that gene. So what happens in DNA methylation is we're going to add some methyl groups to certain bases in DNA, and it's usually cytosine. So if you look at my image, you can see that there are methyl groups that are attached to those cytosine nucleotides. And what will happen is in this actual methylated piece of DNA, it actually restricts RNA polymerase from binding. Okay, and then without that methylation, you can see that RNA polymerase has more access to the DNA. So individual genes are usually more heavily methylated in cells where they're not expressed, right? That makes sense because it's harder to get to. Once methylated, genes usually remain so throughout successful cell divisions. And so after replication, enzymes methylate the correct daughter strand so that the methylation pattern is inherited. So gene regulation results in different gene expression and influences different cell products. So let's take a look at this is a gene for a muscle protein called troponin T. And we can see that it has 5 exons. So right, these are going to be introns and removed. So we're going to end up after we do our mRNA processing, we're going to use these exons to put together or splice, and we can put them together in different patterns. And this is called alternative RNA splicing. So if you notice in the first RNA, this is the transcript that we get after this DNA is transcribed. And the first one, mRNA molecules ended up with exon 3, which is shown in green, and the other one has exon 4, which is shown in purple. And you can see that these two mRNAs are translated into 2 different but related muscle proteins. So in this way, we can actually transcribe it once and then splice it 2 different ways to get different proteins produced. There are certain small RNA molecules that have roles in regulating gene expression. These are miRNA, which stands for a small single-stranded RNA molecule called a microRNA. Okay, so it's showing it here in yellow. And this is going to be our transcribed mRNA. And we're going to actually, those miRNAs actually degrade the messenger RNA. And so if the mRNA is degraded by these microRNAs, the match is not as complete when so you can see here, if we're looking at the hydrogen bonds that are holding these together, translation at the ribosome will be blocked. So the miRNA has a role in breaking down the mRNA to be translated. So a review. There are lots and lots of ways to regulate gene expression in both prokaryotes and eukaryotes. In prokaryotes, operons are used. In eukaryotes, we see a variety of methods. The first one is through chromatin modification. Okay, so normally in eukaryotic DNA, the genes are wound up around these histone proteins, and so we can either loosen or tighten that to give access through histone acetylation, right? That loosens up the chromosome structure to enhance transcription, or through DNA methylation, we are going to add those methyl groups, which blocks the ability of RNA polymerase to transcribe the gene, so that reduces transcription. And we can also look at what goes on in transcription. Remember to regulate the initiation of transcription. There's DNA control elements, the enhancers and activators, facilitate the bending of that DNA to connect the activators to the proteins at the promoter, and that will initiate transcription. Okay? Genes that are in a coordinately controlled group all share a combination of control elements. Then we also know that there's both mRNA degradation, which I just described through the process of those microRNAs. There's also protein degradation and processing that occurs after translation, and that can be post-translational modifications. And then finally, in the middle here, we have RNA processing, which you probably heard about yesterday. But again, you can, once you process your primary transcript, you can also do alternative RNA splicing, where you are able to transcribe one gene, but then splice those exons together in different ways. So let's review a whole signal. So let's say this is a signal, maybe it's a steroid hormone that is going to signal the activation of some transcription factors. So the signal arrives at the nucleus, where the DNA is going to, where it activates some transcription factors, and we are going to then transcribe the gene. We get our primary transcript, which is then processed. We get that cap and tail, which is protected as it leaves the nucleus for the cytoplasm, and then maybe those microRNAs degrade the mRNA, and so it is not transcribed. Or if it's transcribed, we get our protein. We get an active protein that may in fact be transported outside of the cell or used within the cell, or that protein could be broken down. So there are many places along this entire pathway where gene expression can be regulated. Right, so let's put this into some practice. So first of all, let's take a look at what happens in prokaryotes. And in this, bear with me because this is kind of a long question, but I'll be looking at these shapes. Maybe remember our skill is to look at some models that are represented visually. Okay? And so what you're looking at is the lac operon in E. coli. It consists of genes that code for enzymes necessary for the breakdown of lactose. When lactose is absent, the operon is inactive because a repressor protein binds to a specific site on the lac operon. When lactose is present, lactose molecules bind to the repressor protein, causing the repressor protein to dissociate from the binding site. In the absence of glucose, which is the preferred energy source for bacteria, the protein CAP or CAP binds to a regulatory site near the lac promoter to activate transcription of the lac operon. The following symbols represent macromolecules involved in the regulation of the lac operon. So you can see we've got a repressor that is a circle. We've got two 6-carbon rings representing the disaccharide lactose. We've got a triangle for CAP. We've got an oval for RNA polymerase, and then we have a squiggly line for the messenger RNA. In the diagrams below, the horizontal line represents the lac operon, and the bent arrow represents the transcription start site of the lac operon. Here's your question: which of the following diagrams best represents the scenario in which lactose is available to the cell and glucose is absent? So I want you to think about that. Glucose is absent, lactose is present. Which of those diagrams, A, B, C, D, best represents the scenario? Okay, so the correct answer is C, and that's because what you see is the lactose is actually bound to the repressor, so there is no repressor in the way of RNA polymerase, and it is actually making that messenger RNA. So it's the only one where you see the messenger, the transcription, the product of that transcription. What's wrong with D is it showing that the lactose is actually bound to RNA polymerase, and we know that that is not how the system works. Now let's take a look at a practice question in eukaryotes. We again practicing our visual representations. The figure above depicts the DNA protein complex that is assembled at the transcriptional start site of gene X when the expression of gene X is activated in liver cells. Previous studies have shown that gene X is never expressed in nerve cells. Based on the diagram, which of the following most likely contributes to the specific expression pattern of gene X? Okay, so we've got A, expression of gene X produces large amounts of transfer RNA, but undetectable amounts of mRNA. We got yesterday's last. B, the general transcription factors inhibit the activation of gene X in liver cells by blocking the activator from binding to RNA polymerase 2. C, the activator is a sequence-specific DNA binding protein that is present in some tissues but not others. And D, the enhancer is a unique DNA segment that is added to the nuclear DNA of some cells of an organism during the process of mitotic cell division but not others. So if you're paying close attention to today's lesson, you should have your answer. And of course, that answer is C. And that is because we are seeing that the activator is a sequence-specific DNA binding protein that is present in liver cells but not nerve cells. Okay? All right, so now let's get at FRQ. Practice. A difference between prokaryotes and eukaryotes is seen in the organization of their genetic material. Contrast the following activity in prokaryotes and eukaryotes. And what you are going to do here is contrast how genes are regulated in both prokaryotes and eukaryotes. Okay, so hopefully you have some ideas about how to approach this question. So let's take a look. Okay, so in prokaryotes we use operons, right? So we have one single on-off switch in a sequence in DNA that's all together. In eukaryotes there is no operon. In prokaryotes we have both positive and negative control. In other words, we can induce or repress those genes from being transcribed. In eukaryotes, if you think about it, our control is primarily positive control. In other words, the gene is not transcribed until that transcription initiation complex is in place. In prokaryotes, enhancers are rare, and as you saw, in eukaryotes, enhancers are commonly used. In prokaryotes, because there is no tightly packed with histone DNA, there is no other type of epigenetic regulation or above the gene sequence regulation. There's methylation, which you might remember will make it more difficult to transcribe genes, and there's acetylation, which will make it a little bit will make the DNA more accessible to be transcribed. Okay, so that's kind of a good slide to think about and remember. All right, so let's do a check for understanding, specifically for science skill 2C. Again, we're looking at a visually represented process, right? We're looking at gene regulation represented visually, and your task is to be able to explain how biological concepts work in this visual representation. So you're looking again at that lac operon, and we are looking at lactose being utilized for energy by the E. coli when glucose is not present. Allolactose is an isomer of lactose that's in the environment of these bacteria when lactose is present, and then of course the cap site prevents the binding of RNA polymerase when glucose is present in the environment. So then we see we have the lac Z, Y, and A genes, which code for the proteins that are needed for lactose metabolism. So what you are going to do is look at this, and again, if you look at allolactose, here's the repressor, here's the operator. We can see that RNA polymerase is here, and we can see an arrow. Here we see allolactose absent, and so the repressor is in the operator. Repressor is not in the operator here, and we can see that RNA polymerase is not transcribing. We've got a big X there. So this is the question: which is a scientific claim that is consistent with this information that is presented visually in Figure 1? And I'll come back to this. Let's look at our answer choices. So first of all, the presence of excess lactose blocks the functioning of RNA polymerase in this operon. B, when bound to the operator, the repressor protein prevents lactose metabolism in E. coli. C, the binding of the repressor protein to the operator enables E. coli to metabolize lactose. And D, allolactose acts as an inducer that binds to the operator, allowing E. coli to metabolize lactose. Okay, so here's the correct answer, which is B, and it's when bound to the operator, the repressor protein prevents lactose metabolism in E. coli. Okay, so let's go back. When bound to the operator, okay, the repressor prevents the RNA polymerase from transcribing that gene. Okay, if the gene can't be transcribed, it cannot make the enzymes necessary to break down lactose. All right, so here's your debrief in summary. Today, you now should be able to describe the types of interactions that regulate gene expression, explain how the location of regulatory sequences relates to the function, explain how the binding of transcription factors to promoter regions affects gene expression and/or the phenotype of the organism, explain the connection between the regulation of gene expression and the phenotypic differences in cells and organisms. And our key takeaway today is that differences in the expression of genes account for some of the phenotypic differences seen between organisms. So let's talk about some common errors and misunderstandings about gene gene expression and regulation of gene expression and cell specialization. This was an FRQ that was asked on the last year's 2019 AP Bio exam. And so what we're looking at is a visual representation of some data. So a researcher is studying the patterns of gene expression in mice. The researcher collected samples from 6 different tissues in a healthy mouse and measured the amount of mRNA from 6 genes. And the data are shown in this figure. First of all, you need to look at the legend. The open squares, no mRNA is seen. The gray squares, a moderate amount of mRNA is seen. And in the completely blacked out squares, a high amount of mRNA is seen. So across the top, you can see we have 6 genes called E, F, G, H, I, and J. And then we have tissues, liver, heart, brain, kidney, pancreas, and skeletal muscle. So you need to look at this data. And then in part A, you are asked to identify the gene that is most likely to encode a protein that is essential, that is an essential component of glycolysis, and then provide reasoning to support why you selected that gene. And then in part B, the researcher observed that tissues with a high level of gene H mRNA did not always have a gene H protein. Provide reasoning to explain how tissues with a high gene H mRNA level can have no gene H protein. So there are 2 different things. First of all, you need to identify the correct gene that is most likely going to encode a protein essential for glycolysis, and then figure out why if you have the mRNA, you don't have the protein for gene H. Okay, so let's look at the scoring guidelines. This was a 3-point question. The first point was identification, and you simply needed to identify gene G. Okay, and the reasoning behind that was also worth 1 point, and for either one of those bullet points, you would be awarded a point, and that is that gene G is the only gene expressed in all 6 tissues, and glycolysis occurs in all 6 tissues. So you needed to make that connection. Just saying that it was expressed in all 6 tissues was not enough. You needed to connect it to the fact that all 6 tissues undergo glycolysis. Or you could have said that mRNA is the only mRNA present in all 6 tissues, and glycolysis occurs in all 6 tissues. Okay, and then finally for your 3rd point, you needed to provide reasoning for why you have the mRNA from gene H, but you don't have the gene H protein. And any one of those bullet points would have earned your point. The mRNA is not exported from the nucleus. The gene H mRNA is not translated, or the those microRNAs could have prevented translation, or there could have been other post-transcriptional modifications to the mRNA that was transcribed, preventing that protein from being produced. Okay, so let's look at first let's look at a student response that earned all 3 points, and then we'll look at where the knowledge gaps and misunderstandings were. So here's a 3-point question. 3-point answer. Excuse me. Gene G is most likely to encode a protein that is an essential component of glycolysis because the mRNA is at least moderately present in all types of tissues. All tissues undergo glycolysis to get a small amount of ATP to function. Gene G is the only gene that has mRNA present in all tissues, so all of these tissues are able to code for the specific protein. Okay? And then for part B, tissues can have a high level of gene H mRNA, but not have a gene H protein because the mRNA was never translated. The gene H may have been transcribed from DNA to mRNA, but if the RNA and tRNA do not translate this particular strand, it will not code for the amino acid and therefore not become a functional protein. So excellent 3-point response. So where were the knowledge gaps? A lot of students had a difficult time correctly identifying gene G, and also not understanding that gene G is the only one that was expressed in all 6 tissues and that glycolysis occurs in all 6 tissues. So making that connection was crucial. So a response that demonstrates understanding of that is that gene G has a high to moderate amount of mRNA in every tissue on the table. Because glycolysis is the first step of cellular respiration, which is ATP making, gene G would most likely encode a protein essential for this energy bringing process, which is done throughout the body. So making that connection between gene expression and its function. And then in part B, students had difficulty correctly reasoning that gene H mRNA is not exported from the nucleus, or it's not translated, or that there's some type of RNA interference preventing translation, or that it undergoes those post-transcriptional modifications. So this was a difficult part of this question for students. Here's an example of a response that demonstrates understanding. Tissues with high gene H mRNA levels can have no gene H protein, as unsuccessful translation can lead to the lack of gene H protein production. Okay, so I'm going to give you your homework, but before I let you go, I'd like to give you just a few tips for getting ready for your AP exam on May 18th. So here's your homework, and this is due tomorrow. So Ms. Pfizer will be with you tomorrow, and she'll go over the scoring guidelines for this. Human cells divide every day for normal growth, replacement of cells, and tissue repair. And you know that if you joined our lesson on cell cycle and mitosis. The dividing cells proceed through 3 phases of the cell cycle, as shown in the figure. You see mitosis, cytokinesis, and interphase. When a person experiences a cut, damaged skin cells secrete a protein called epidermal growth factor, EGF. Epidermal, which are skin cells in the vicinity of injury, possess these proteins in their cell membranes called epidermal growth factor receptors, or EGFR. So there's your background. You can read that as many times as you need to understand kind of the background to help you with the intention of this question. Cancer can result from disruptions in the cell cycle control. Mutations that increase the production of EGFR have been associated with skin cancer of the head and neck. Describe, that's your task verb, how a mutation outside of the coding region of a gene that codes for EGFR could lead to the overproduction of the EGFR protein in the skin cell. So this is going to have you put together some lessons on cell cycle regulation and control, and also think about the expression and regulation of eukaryotic genes. Okay, so again, that will be your warm up tomorrow. So just a couple of slides here, just so you can kind of get a visual of what you're going to be seeing in the next couple of weeks. So you will need an AP Biology exam e-ticket. There are 2 ways that you'll get this. You'll either get this emailed to you 2 days before each test. This one happens to be for biology. But if you're taking multiple AP exams, you will get your test ticket 2 days before your exam. If you don't get the email, that's why we've been bugging you to go and make sure that College Board has your most current email on your AP account at MyAP. You can go to myap@collegeboard.org and make sure that your email is updated. Okay. Otherwise, you'll be able to get it at your MyAP at the College Board. Just a reminder about knowing when your test will be administered. Okay. Know your time zone. AP Biology exam will be given on Monday, May 18th at 2 p.m. Eastern Daylight Time. So I'm in California. My students will be sitting for the exam at 11 a.m. If you are in Madrid or in European time zone, check your time zone. You may be taking that test at night, so make sure you take it out during the day. Be prepared. Make sure you understand your time zone. If you miss, then you'll have to figure out how to get into that makeup exam. All right. And then 5 quick steps to prepare for your exam day. Again, review your contact information, check your tech. We've been asking you to make sure you have a device and internet connectivity. We are, and I'm going to talk to you more about this next week. Beginning on Monday, May 4th, there will be an exam demo. It will not just be for AP Bio, it'll be for all generic AP, and you can practice how to submit your response. So you're going to be walk through how to do that, and the 3 different ways that you're going to be able to write your response. Okay, and more on that later. Make sure that you gather what you need for each exam. You can consult collegeboardcb.org/exam-day docs. Download them, print them, gather your additional materials for your specific exams. For example, for AP Biology, you might want to have that AP Biology formula and equation sheet handy, and then make sure you receive your exam confirmation and e-ticket emails. Okay, so final words. During this challenging time, the AP program wants every interested AP student to have the opportunity to earn college credit. You've been working for it all year, so please stay safe and healthy, and remember, you got this. All right, so thanks for watching. Look at your checklist, complete your checklist. We hope you'll join us again tomorrow for the last review lesson. That will be with Ms. Pfizer. She'll go over your practice question, and tomorrow's lesson will review mutations in biotechnology. And I'd like to wish all of you best of luck on Monday, May 18th. Thanks for watching.
Welcome to AP Biology. Hi, I'm Gail Pfizer from Darnell Cookman Middle High School and Florida State College in Jacksonville, Florida. A lesson overview for today. Today is the last topics for Unit 6, Gene Expression and Regulation. I know we're all doing something different today than we would have thought months ago. Today, right this minute, I should be at the University of Florida watching my son graduate with his master's degree and playing a party, but where that happened anyway, but we're going to do it online. And I'm sure that you're not doing what you thought you would be doing today. But if you're listening and working and we're all safe, that's what's important right now. All right, topic 6.7, mutations. We've covered all of this already. So I'm just going to tell you whenever something comes up, I'm going to tell you where to reference that so we can spend our time on biotechnology. By the end of this lesson, you will describe various types of mutations, explain how changes in genotype may result in changes in phenotype, explain how alterations in DNA sequences contribute to variation that can be subject to natural selection, and explain the use of genetic engineering techniques in analyzing or manipulating DNA. So you'll need pen and paper for a few questions today. So our warm-up is from our homework yesterday with Ms. Katay, and it's from gene regulation. And this is a very rich question because it has a lot of reviewing other things in the question. So human cells divide every day for normal growth, replacement of cells, and tissue repair. So right there we have the function of mitosis, right? The dividing cells proceed through 3 phases of the cell cycle. We have interphase, mitosis, and cytokinesis. When a person experiences a cut, damaged cells secrete a protein called epidermal growth factor. So let's get, this can be our skin, and that is our ugly cut. Okay, I'm gonna draw some epidermal cells because I'm gonna need them in a moment. There's a very big epidermal cell. Okay, so when a person experiences a cut, damaged cells secrete a protein called epidermal growth factor, and we draw that in. Okay, all right. Epidermal skin cells in the vicinity of the injury possess proteins in their cell membranes called epidermal growth factor receptors. So here we have receptors on these cells. Okay, so now this protein and there's the protein and there's the protein receptor. Okay, cancer can result from disruptions in the cell cycle. Control. Mutations that increase the production of EGFR have been associated with skin cancer. So you know if we're talking about cancer, then we're going to be cells are going to be dividing out of control, and this cancer was the head and the neck region. So what are we going to do? We're going to describe how a mutation outside of the coding region. Now remember, just yesterday, this is a gene, a crooked gene, and here is outside. That was our coding region, and this is outside of our coding region. Okay, outside of the coding region of a gene that encodes EGFR could lead to the overproduction of EGFR protein in the skin cell. So if you're just joining us for the first time, I'll give you a few seconds. Let me turn my timer on anyway. And if you did your homework yesterday, then you should have this ready. All right, now we can start. All right, so here are some options for answers. Mutation in a regulatory sequence or promoter or enhancer could increase rate of transcription of EGFR gene. Okay, that's the answer that I would have written down. Here's some other options though. Mutation in transcription factor could increase production of messenger RNA, which would lead to production of more polypeptide. Gene duplication may increase production of EGFR. Gene translocation may inactivate repressor. Mutation in repressor gene may remove controls on cell division, and change in messenger RNA sequence may increase translation efficiency or messenger RNA stability. When I wrote this to prepare for today, this is the option that I wrote before I read the rubric. Okay, so what you need to know. Okay, so there are a lot of these slides that are just review information, and I'm just going to kind of skate quickly through those. And if I need to stop for a moment, I will. But if it's old information that we've heard before, then I'm going to refer you to where you should go to re-listen to that if you've been following the videos. All right, the function and amount of gene products determine the phenotype of organisms. The normal function of the genes and gene products collectively comprises the normal function of organisms. Disruptions in genes and gene products cause new phenotypes. This is a great, there's a question that follows this. So this is a great example. Example of a change in a genotype that results in a change of a phenotype. Okay, mutations in the CFTR gene disrupt ion transport and result in cystic fibrosis. A normal channel allows the transport of chloride out of the cell. The increase in the intracellular chloride concentration causes water movement into the cell due to osmosis. So here we're bringing osmosis into this along with protein transport. The mucus is dehydrated and sticky and builds up in multiple organs. So let's look at what should be happening. So here's our chloride channel. Okay, so here's a chloride. It should be able to move through to the extracellular fluid. But yet with this mutation in this protein, so here's our channel and it's mutated. Now this chloride ion cannot get through this channel. So it's not moving through. But what's happening is that the solute inside the cell is increasing, which is making the water potential lower. So the water is going to move from the outside to the inside through aquaporins. You will get through the phospholipid bilayer, but through aquaporins that water will move through. And then this mucus is going to, which had a great deal of water and was hydrated, is now going to be dehydrated and sticky and start affecting multiple organs. Okay. All right. So here's a question about CFTR protein. It is made up of 1480 amino acids linked together in a chain. Some humans produce a version of the CFTR protein in which phenylalanine, an amino acid, has been deleted from position 508 of the amino acid chain. Which of the following best predicts how the amino acid deletion will affect the structure of the CFTR protein? So let's take a look at these options. A, it will have no observable effect. Well, the structure of that CFTR protein. Well, I just showed you what was going to happen with this, so we already know that. It will affect the primary structure of the CFTR protein. That's true because this was amino acids, right? So we had a strand of amino acids and then our primary structure, and now one's gone. All right, or it's substituted out for the wrong one. But the other levels of protein structure will not be affected. That's not true. The properties of that amino acid then change the structure and the function of that CFTR protein. So that's not it. It will affect the secondary, tertiary structures, but the primary structure will not be affected. That's not true. We just said that at that position that there had been a deletion. Yes, a deletion. It will affect these, so that's not it. It will affect primary, secondary, and tertiary structures. Yes, it affects all of those levels of protein structure. So D is our answer. All right, so now we have another mutation. Alterations in DNA. I've seen this before. And the DNA sequence can lead to changes in the type or amount of protein produced in consequent phenotype. DNA mutations can be positive, negative, or neutral based on the effect or lack of effect that they have on the resulting nucleic acid or protein and the phenotypes that are conferred by the protein. So this again is in antibiotic resistance. I think I had the same diagram in natural selection the second night that we started the videos in natural selection. But anyway, you have bacteria in your body. Some of them are going to be resistant to antibiotics, and so you take the antibiotics, they kill all those that are not resistant, and then the resistant reproduce, make more resistant, and then they also can even exchange in a horizontal exchange to other bacteria. Okay, I guess that would be positive. I guess that perspective is probably a positive move for the bacteria, but a negative move for the human being. All right, so sickle cell is another mutation that we talked a lot about. In normal, remember that we had a T and we it coded for glutamic acid, and in the mutated hemoglobin we have an A and it coded for valine, and that was a problem because the glutamic acid was hydrophilic and valine is hydrophobic, and it ended up getting these hemoglobin molecules that stick together in this fiber form, and then they end up in this structure. Now that would be a negative effect is if we had two recessive alleles that do this. We'll talk about that. So if we have, we're just going to talk about this top part. If we had two recessive alleles, then that's negatively going to impact this individual because they're going to have sickle cell disease. If you have no sickle cell, if you're not a carrier, then you are subject to getting malaria. And then if you are a carrier, then you have the heterozygote advantage. You're not going to get malaria and you're not going to have sickle cell. So there's a positive experience or positive mutation in this case. All right, changes in genotype may result in changes in phenotype. Errors in DNA replication or DNA repair mechanisms and external factors, including radiation and reactive chemicals, can cause random mutations in the DNA. Ms. Katay just talked about this 2 days ago in DNA structure and function and replication. Whether a mutation is detrimental, beneficial, or neutral depends on the environmental context. Mutations are the primary source of genetic variation. If you need to review DNA replication or repair mechanisms, it's in topic 6.1 and 6.2. All right, errors in mitosis and meiosis can result in changes in phenotype. Changes in chromosome number often result in new phenotypes, including sterility caused by triploidy and increased vigor of other polyploids. So triploidy, we're talking what we could be talking about Down syndrome, and polyploidy that could be like strawberries. Changes in chromosome number often result in human disorders with developmental limitations, including Down syndrome, trisomy 21 that we just mentioned, and Turner syndrome. So if you need to revisit changes in chromosomal changes or environmental effects on phenotype, then go back to 5.5 and 5.6 or where those topics are located. All right, changes in genotype due to alterations in DNA DNA sequences may affect phenotypes that are subject to natural selection. Genetic changes that enhance survival and reproduction can be selected for by environmental conditions. So this is in Unit 6. Okay, this is talking about natural selection, and evolution, and especially natural selection, has been addressed in different parts, minor parts listed in Units 1 through 6. So I highly recommend that, for the sake of natural selection, that you go back and look at our first 2 videos. 7.1 and 7.2 are both about natural selection, and they could be helpful in understanding some of this biology. All right, so now we get to some topics that we haven't discussed in any previous video. So transformation is a method of horizontal acquisition. So what that means, so if I was a bacteria and then I had to binary fission, I made an offspring. Okay, that's not what I'm talking about. I'm not talking about DNA going from this to my offspring. Okay, that's not it. So what I'm talking about is a bacteria. In this case, we're talking about transformation. We're going to have a bacterial cell, okay, which is going, some genetic information is going to end up, that was here. Okay, let's make an example of this right now. Okay, so some genetic information that was in this bacteria, and it's going to be given to its neighbor that doesn't have it. Okay, so it was like that, and then it got the plasmid. We'll talk about a number of ways to make that happen. All right, so let's see what else. Let's talk about this is transformation. This is from students Petri dish in my classroom this year. Okay, so we'll talk through the process, and I drew this at Eastern Standard Time, and I drew this like 4 o'clock this morning. So it's not beautiful, but I think it gets the point. All right, so we have some E. coli. We have E. coli bacteria growing, has some colonies, and we're going to take a few small colonies, and we're going to mix it with calcium chloride. Okay, and we're going to keep this cold while we're working with this, and then we're going to add a plasmid. This plasmid is going to have genes on it, and so what this plasmid has, it has a gene that will break down arabinose. It has a gene that will turn it green with green fluorescent protein. It also has a gene that will give it ampicillin resistant. Okay, so now we're going to put this plasmid into the tube with our bacteria sample and our calcium chloride, and we're going to keep it on ice, very cold. Okay, now we're going to take it from the cold to the heat, and we're going to do what's called a heat shock. So we're going to heat shock it, and after we stay in the water bath for about 50 seconds, we're going to go straight back to the ice. Okay, now what happened during that time period? Let's say this is our phospholipid bilayer when we're very, very cold. Okay, we are very close together and it's cold. All right, and now we're going to get put into seriously warmer than we already are water. Okay, so let's get rid of this. And then what do you think is going to happen to this phospholipid bilayer? Okay, so now we're going into into warmer water, and now it's going to be not so tight. Okay, there's going to be some room. It's more fluid. Now this is the opportunity for the plasmid to get through this phospholipid bilayer, but the plasmid has what? It has a negative charge. So the calcium chloride, which has a positive charge, is actually going to be like the Secret Service and get kind of camouflage, make this not so negative, and be able to shuttle right through this membrane. Okay, so that's the hope that we're going to get it. We're going to expand the plasmid membrane, and then we're going to escort this plasmid through there, and then the plasmid will be inside the cell. Okay, so now that the plasmid's inside the cell, it can be expressed to make other proteins. All right, so how does this work? We have to have operons that we talked about yesterday, gene regulation. So this is their arabinose. Okay, and arabinose was in one of the plates, and arabinose is what's going to bind to this enzyme that's going to bind to this promoter area that's going to enable RNA polymerase to transcribe the messenger RNA that's going to go to the ribosome to build the enzyme that's going to break down the arabinose for a nutrient. Okay, now at the same time that all that's happening, we also have this GFP. So in the promoter area where this arabinose was binding to allow this RNA polymerase to transcribe, it's also going to transcribe for GFP. So we're going to break down arabinose, and we're going to be able to turn green in the process. Now it also has a gene on it for ampicillin resistance. So the ampicillin resistant gene is turned on, and it makes a protein that's going to then break down ampicillin so the bacteria can grow. All right, so now that we have all of that behind us, we're going to put on the plates. We're going to put two plates. This has plasmid, so there was some transformation that hopefully had occurred. So we're going to put this sample on the plate with plasmid, on an LB, which is lauric raw, and agar, and then ampicillin, which is an antibiotic. And the same thing here, LB, ampicillin, and then the arabinose sugar is in the petri dish. Now this has a plasmid also. We have here no plasmid on this tray, on this plate, and no plasmid on this plate. So the results in the next 24 hours, we have some isolates growing right here, and so that means that we have some transformation, right? But they're not growing green because the arabinose was not there that turned on these two. Okay, all right. So now we have in this plate we have arabinose, and so now we have this was able to be turned on. We transcribed GFP and the enzyme to break down the arabinose, and now we have the green glowing fluorescent protein. Okay, now we didn't have a plasmid in this, but we put it in ampicillin, which is an antibiotic, and so it killed all the bacteria. Now we put one without the plasmid in this plate, but it's only on LB. It's not in the ampicillin, so it grew all over the plate, and that's called a lawn. Okay, so that is transformation. We took non-green, okay, glowing, they were not glowing green, and now they are. So we transformed them. We gave them genetic information that was able to be transcribed for a new phenotype. Okay, all right. So this is from actually these are plates, so they're just beautiful. From I teach a college class. I'm adjunct professor for our Florida State College, and I teach Introduction to Biotechnology and Bioinformatics here. And these were plates I do this lab in both of those classes, my FSCJ classes and my AP classes at Bernard Cookman. And this particular mine looks crazy right here because I get the wonderful opportunity to do this a lot, and so I always change it up. And I think I put extra plasmid in my solution on that one. That's why it's so many. So in my AP classroom, I always follow up the lab with this particular lab because it's so the novelty of it, and it's so many steps, and you need to not only know how to do the lab and make the procedures go right, but to have understanding of what you were doing and why you were doing it at the end. That this is a set of questions that I always give them. So I'm giving to you about what we just talked about. In a transformation experiment, a sample of E. coli bacteria was mixed with a plasmid containing the gene for resistance to the antibiotic ampicillin, and it says AMP and a little r. Plasmid was not added to a second sample. Samples were plated on the nutrient agar plates, some of which were supplemented with the antibiotic ampicillin. The results of E. coli growth are summarized below. The shaded area represents extensive growth of bacteria, and dots represent individual colonies. So this is a lot of growth, and this is just a few colonies, and this is nothing. Okay. All right. In the second experiment, the plasmid contained a gene for human insulin as well as the AMP-R gene, which the following plates would have the highest percentage of bacteria that are expected to produce insulin. Okay. Now it's a trick question, kind of. Okay, because now the highest percentage of the bacteria that are growing. Well, 100% of these bacteria on this plate have been transformed. Okay. Now because they have in ampicillin and they have the plasmid, so they all have the gene for ampicillin resistance. So that they have the gene for ampicillin resistance, then they also have the insulin. Okay. So this would be 4 only, which would be C. Okay. Now the next one, plates 1 and 3, were included in experimental design in order to. Okay, here's 1 and 3. Both of them have growth all over them. Here they don't have ampicillin. So let's look at the right choices. They were in order to demonstrate that E. coli cultures are viable, demonstrate that plasmid can lose its AMP gene, demonstrate that plasmid is needed for E. coli growth, or prepare the E. coli for transformation. So I hope you chose A, right? That to make sure that E. coli could grow without the treatment. All right, plates that have only ampicillin resistant bacteria growing include which of the following? So only ampicillin resistant. So we have to have ampicillin. There's nothing growing here. So our only option is 4, okay, which is C. Which of the following best explains why there's no growth on plate 2? The initial E. coli culture was not ampicillin resistant. The transformation procedure killed the bacteria. Nutrient agar inhibits E. coli growth, or the bacteria on the plate were transformed. There's no growth here. So your answer, okay, is A. Yes, the initial E. coli culture were not ampicillin resistant. And then which of the following statements best explains why there are fewer colonies on plate 4 than on plate 3? So why are there fewer here than there? Okay, all right. So plate 4 is a positive control. Not all E. coli cells are successfully transformed. The bacteria on plate 3 did not mutate. The plasmid inhibits E. coli growth. So I hope you said that not all E. coli cells have successfully transformed. And that's transformation efficiency. efficiency. Remember we did transformation efficiency with something else just maybe a few weeks ago. Anyway, the amount that did divided by the amount that could, and then you could have your percent that were transformed. Okay, so here's another way of horizontal acquisition. Okay, so we're gonna take a virus this time. We're still going bacteria DNA to bacteria DNA. So here's our virus, and our virus has, let's give him pink. Okay, he has pink DNA, and he's inserting it into this bacterial cell. This is a bacteriophage. Anyway, inserting it into this bacterial cell, and he is using all the machinery. There's some ribosomes and the DNA. The whole cell is just getting destroyed and used. Anyway, let's make this purple. Here's some of the bacteria's DNA. And so now what is the purpose of this virus? His goal is to make more virus. Now, if we have in this mix, this happened, okay, and this virus is going to lead with bacterial DNA instead of viral DNA. And now this becomes incorporated into the genome of this bacteria. So we went from this bacteria to this bacteria, and that's a horizontal acquisition also, okay? And all of these increase genetic variation. Okay, here's a question, and we're adding in here's transduction, which we just talked about, plus fertilization. All right, which of the following correctly compares the two processes shown above? Both processes increase genetic variation. Yes, they do. Both processes represent aspects of sexual reproduction. No, this one does, but this one does not. Both processes require partitioning of genetic material and organelles. No, the bacteria does not have a nucleus and doesn't have compartmentalized partitioned organelles. So no. And the amount of genetic material per cell remains constant in both processes. No, it does here. Well, there's an addition here, and there's a 1N to a 2N condition here. So the answer here is A. All right, here's conjugation, and this is bacterial cell to bacterial cell. And so if we have, I'm going to put our one singular chromosome in here, and then I'm going to also add. I'm also going to add a. That's not really good. Can't see it. Okay, so here's my plasmid. Now this cell does not have a plasmid, but this plasmid is replicated from this cell. It can be put into this other bacteria through what's called a conjugation pilus that is formed with some small proteins between each of these two bacteria. That's conjugation. All right, here's some horizontal acquisition, and there's two ways you can do this. This is called transposition. And what happens is you can have a. No, let's do this one. This one is going to be cut and paste, and this one is copy and paste. So here we have this particular segment of DNA, and it's going to be copied, and then it's going to be inserted somewhere else in the genome. Okay, so now it's going to be there twice, and we're also this one in this case we can cut it out totally. It's not there anymore, and then insert it to somewhere else in the genome, and that's called transposition. It's also a horizontal acquisition of genetic material, and it also increases variation. All right, let's see. I cut out something, that's why I kind of paused. In the regular PowerPoint, there's a question that has to do with eukaryotic cells and transposition. I cut it out due to time, but it's there for you to work on when you have time. All right, related viruses can recombine genetic information if they affect the same host. So look at this. Here we have an avian influenza virus, and here's RNA from that virus, and here are the proteins on the envelope of that virus. And then we have a human influenza virus, and we have RNA molecules, and there are the proteins on the coat of that virus. Now, this pig respiratory tract cell has receptors for both the avian influenza virus and for the human influenza virus. So both of these are able to enter the cell. Now, this is the pig's DNA, which has nothing to do with what we're about to talk about. So now this new virus, this new strain of virus can be formed, and it has both. Look, it has the avian and it has the human. Okay, the human influenza, and then we notice that it does have the avian flu proteins on the outside. Okay, so this is how both of these have mixed together in this one cell and created a new strain. Very simplified version. All right, so here's a question about that. The cells lining the respiratory tract of pigs have receptors for both avian and human influenza viruses. Based on the model above, which of the following best describes the origin of the new strain of human influenza virus? The new viral strain inherited a mixture of genetic material from both avian influenza virus and human. Yes, it did. The new viral strain inherited RNA molecules from the RNA and packaged them inside the human influenza virus membrane. No. The new viral strain inherited a mutant DNA molecule from the pig. No. And the new viral strain inherited an RNA molecule that had been recombined with the DNA of the pig. No. It was both the avian and the human. So what I'd like to do with when I have this is if this is in a CED and I want to know what the people who wrote the CED, what the people who were writing the questions that support the CED, how all their minds are working together. Like what do these words not only mean to me, but what do they mean to the person writing questions or to the team writing questions? And so that's why I do a lot of this multiple choice work is to see where they're coming from with this information. So this was built from that statement in the CED. So I hope that's helpful. That's how I try to figure out what exactly they mean. If I feel like it is vague or maybe needs more details to tell me what I need to know, but that's how I figure it out. All right, reproduction processes that increase the genetic variation are evolutionary conserved and are shared by various organisms. So here we have a diagram crossing over conjugation and fertilization. Question asked: the processes illustrated in the models depicted above all result in which of the following? So transcription, an increase in genetic variation, yes. An increase in chromosome number, where is that happening? That's happening just in this one, right? And horizontal gene transfer, that's happening just in this one. So that's not it too. So I answer all of these that we've been talking about, increase in genetic variation. So let's talk, what did we talk about? We talked about transformation, we talked about transduction, we talked about conjugation, transposons. Okay, all of those work in the same way, horizontal genetic transfer. All right, so now let's talk about some biotechnology techniques, some genetic engineering techniques. Polymerase chain reaction, we'll talk about that first. It stands for PCR, and what it does is it amplifies a copy of DNA, which means it's going to take usually a gene of interest or a certain fragment of DNA and make a million copies out of it. This is my PCR in my room, and I have it has this 25 little slots here, and what these are 0.2 ml PCR tubes. And what's going to happen is that we need to make more copies. So we're going to do a synthetic version of what happens in replication. Okay, so what has to happen in replication? The two strands have to separate. So to do that, then we're going to have to denature it, and we're going to denature it at a high temperature of 94 to 96. So now we're going to get the strands to come apart of that double-stranded DNA, and then we're going to have to put a primer on. So we're going to have to cool it down for it to have the primer put on, called annealing, and then we're going to heat it back up just a little bit for the nucleotides to be put together on the new strand. So we go through a little cool down and a little warm up, and we have to do that through several cycles. This is a setting it had 35 cycles, so I could end up with about 1 million copies of particular DNA fragments when we got through with running the PCR. Okay, these are now we're going to talk about gel electrophoresis, and now we can, after we have amplified the sample, now we can separate it as far as fragment length, and we can do charges also. And so this is in my classroom, and this is a gel electrophoresis apparatus or tray. And what you see here is here's a negative end, and this red is a positive end, and then we're going to attach it to some power. And what's going to happen is that there's going to be hydrolysis occur, and then there's going to be a movement from I'll show you how in just a moment. If you put negatively charged molecules on one end of the negative end, then they're going to get attracted to the positive end, so they're going to move. Okay, this is just practice before you learn how to use DNA, which is expensive too with food coloring. Okay, so we have a lot of things to talk about on this too. If you've never done this before, never talked about this before too, I would do my best to make this quick, but it's thorough grow as possible too. So in order to separate fragments, we're going to need to cut DNA into fragments. And there are many restriction enzymes and are made from bacteria. Bacteria produces a restriction enzyme in order to destroy viral DNA that's invading its body. Okay, so if we were to cut with this restriction enzyme, this particular segment of DNA, it cuts out a palindrome. So you can see we have a GAATTC, we have a GAATTC. And so this came from Ecology, and it was restriction enzyme number 1. Okay, that was discovered. And so this is the site that this restriction enzyme always cuts out. So if you had, so we're going to be dealing with these 4 individuals. So if we had 4 individuals DNA, okay, and we were to cut it with restriction enzyme, we may cut this person, they have that palindrome there, there, and there. This one may be here. This one may be only there. And this one could be in many different places, all these different individuals. So what happens is these are cut into these fragments that are these links. Now what a fragment is a number of base pairs. So if you had 1, 2, 3, 4, 5, whatever, 8 I have there, that would be your base pair number. Okay, but they're in the hundreds and the thousands. So we could start off with fragments that are 30,000 base pairs long. So now we're going to load them into what is a well at the end of these gels. Okay, and what it is was made a little comb, and it's a depression. And you're going to put your DNA sample into it, and it's going to be, your DNA is negative, so we're putting this on a negative end, and then this is the positive end. And when this is turned on, the fragments are going to move. Okay, you're going to move towards the positive end. But there are different lengths of fragments. There's very long ones and there's very short ones, and they have different numbers of base pairs in them. So what's going to happen is, and I describe this to my students, if we all went to the mall shopping, and each one of us, of the 20-something of us in class, we all held hands and we tried to walk through the crowd in the mall. We're not going to get there very quickly because there's little places for us to get through, but we are not going to fit through there at any fast rate of speed. Okay, but if there were just 2 of us, then we would be able to move through the pores through to get to the other end quickly. So that's what's happening here. The smaller fragments, here's a smaller fragment. They are getting through this porous agarose and able to get to the other end faster. So this could be like 30,000 base pairs long here, and then 20 and 10,000, and then 1,000 and 500, and then 100, just making up numbers. Okay, all right. So I hope that helps with what restriction enzymes and gel electrophoresis stuff. So this is some UV transilluminators, and since the college that I work at also is like 5 miles down the road, I have the ability to jump in the car on planning and run down our samples so I can take pictures. And so this is a real huge desktop transilluminator, and I think we were working on PTC, that's the bitter taste lab when we were doing that, looking to see if we were homozygous or heterozygous for a trait or no trait. And this had to do with a food poisoning lab. This is one of my favorite things to do. It's the same thing on both of these slides, but I wanted to, instead of just showing you how these look when you separate these fragments, I wanted to address something that I have to give a shout out to Maya. She asked me to talk more about positive and negative controls. And this particular lab is where it all comes clear because there's so much other things that can go wrong. So a positive control receives treatment with a known result. Okay, we know what it's going to look like. And a positive control is a control for what we don't know can happen. So if let me see how we can do here. So I think, and I didn't go back and look because everything's at school, but I think that this open place right here where there was nothing, I think that was our negative control, which had no treatment at all. I'm pretty sure that was what that was. And then I believe this was our positive control. So our positive control is there to show us what a positive looks like. And then I believe this was the match for the positive control. So let's say that none of these, there were no results. Okay, that this was not there. Could we say that was negative? Yes. If we have this positive control to show us what a positive control, then we can say this is negative. But what if this positive control was missing? Could we say this is negative? No, we couldn't, because probably if this is missing and it should be showing a positive result, then there's been some type of operator error. And this is one of the labs where that's so easy for that to happen. And when you're working with DNA and pipetting and gel electrophoresis and all that. So if everything was blank and even our positive control was blank, then we knew something, one of these other things out of our true control that was happening. I hope that makes sense. All right, so let's do some work with this. All right, so we have a gel electrophoresis tray. Which of the following statements best explain the pattern seen on the gel with regard to size and charge of molecules of A and B? So this is, I've done this before, and put the wells in the middle and started with our DNA sample here, and then waited to see, like maybe with food coloring or some other type of something in my room, but not with DNA. Anyway, which of the following statements best explain the pattern seen on the gel with regard to the size and charge of molecules A and B? Molecules A and B are positively charged. If they were positively charged, would they be heading to the positive end? No, so we'll get rid of both of those. Molecules A and B are negatively charged. Yes, I agree with that. And then molecule A is smaller than molecule B. Yes, because where is it compared to the well? It got to the end quicker, right? So that would be yes. Okay, well, I guess that was not moving. All right, the diagram below illustrates results of electrophoresis of DNA sequences obtained from a family of 2 adults and 3 children and amplified using PCR. The bands represent short repeating sequences of variable length. Results for another female X are included for comparison. I forgot to mention when you're doing running a gel that you have a ladder, which is going to tell you how long your base pair fragments are. You don't have to guess that. Okay, so here's some DNA from Dad. Okay, let's look at this. Put some color on this so we can easily see what we're doing. So there is Dad, there's Dad, there's Dad, and now let's do Mom. Here's Mom and Mom and Mom, and not Mom and not Dad. Okay, well yes, there's Mom and there's Dad. Okay, so let's read. The banding patterns of the DNA fragments reveal that child 1 and 2 cannot be biological siblings, or they 1 and 2 are no, they are biological siblings because you can tell they have both Mom and Dad. And this is very simplified. Okay, we're making these statements based on exactly what we see. Child 1 and 3 probably look like the mother. We are not sure of what particular alleles are on these at this time. Okay, the mother cannot be the biological parent of all 3. Child 1, she is, but she is not. This child does not have a fragment here. Okay, so this is true. The mother's DNA has the same DNA sequence as the father's. Obviously not, because when they were cut with restriction enzymes and then amplified, they have different fragment lengths. And then child 2 and child 3 inherit all their DNA from their father. You can't do that. Okay, so the answer is C. Yeah, the mother cannot be the biological parent of all 3. Okay, so DNA sequencing is another genetic engineering technique, and it determines the order of nucleotides in the DNA molecule. It can be used to analyze DNA, identify organisms. They use it in forensics, and you can perform phylogenetic analysis with it. This is activity that I use in my room. It's quite. Let me see what my time. I'm going to just run right over this. It has to do with dideoxynucleotides and where they show up as far as in the Sanger sequencing. We have all the A's and cups, T's, C's, G's, and then we pick until we have a DDNTP. And when that ends, we know that our fragment is ending, and then it stops because a nucleotide cannot be added on to the hydroxyl in the 3' end of a DDNTP. So you go through this process, you figure out all of the nucleotides of the sequence. We were working with one that was 5 long, and then another thing. This is old school sequencing. They have NGS, they have Illumina, they have sequencing. All kind of things go on now depending on the number of the size of the genome, or whether you're doing a fragment, how quickly you need it, that type of thing. So here is old school. We put it in a polyacrylamide gel, and then separate these, and these will stop. And then you see that this one stops right here, and you know that is a C. Okay, and that this one was because it was an A, and then a G, and then a T, and then a C because of location in the tray. Okay, and then you can run an electrophoregram which says the genetic sequence also. But this is fun. If you email me, I will give you where I got this from. I've done it in both of my classes. All right, examples of genetic engineering techniques in analyzing or manipulating DNA. We have some glowing green fish here that has to do with the modified organism, and then we have some gene cloning. So gene cloning and cloning organism are 2 different things. So we have a vector, and then we have, which is the plasmid. That vector is what we use to get this DNA in. Here's our gene of interest. Both were cut with the same restriction enzyme. And then the restriction enzyme is going to move into this bacteria and through transformation, and then this will reproduce. And if this was reproducing, let's say this gene was for ampicillin resistant, and the offspring of this vector would make the epsilon resistant also. Now, as far as cloning an organism, you take the organism you want to clone, you take a somatic cell, which has all the chromosomes, double step 2 in, and then you take out of an egg, you're going to take out the genetic material out of that egg, you're going to put those two together, and then you're going to develop a organism that is similar to the first organism. All right, real quick on this, I'll skip that, you can read that at home. Okay, so cystic fibrosis is a genetic condition that's associated with defects in the CFTR protein. The CFTR protein is a gated ion channel that requires ATP binding in order to allow chloride ions to diffuse across the membrane. So the first thing we're going to do is identify the most likely site of location of mutant CFTR protein that has an amino acid substitution at the ATP binding site. So where would we most likely find this CFTR mutated protein? I'll give you a second. Okay, I hope you're thinking it's going to be in that plasma membrane, right? Okay, identify again the most likely site of location of the ribosomes that synthesize the CFTR protein. If we have to get a protein to the membrane, we're going to have to synthesize them on our endoplasmic reticulum, right? Okay, so there's the we'll do that on the ER. And then in the provided model of the cell, draw arrows to describe the pathway for the production of the normal CFTR protein from gene expression to final cellular location. This is where the tricky part is going to come in, okay? So we have to draw some arrows, and so it told us from gene expression, so where does it need to start? It's going to start from the nucleus, it's going to end up at the messenger RNA, is going to engage with the ribosome at the endoplasmic reticulum, but that's cool. Pinch off, take that protein to our Golgi apparatus, and then our Golgi apparatus will take it to the plasma membrane, right? Okay, that's what we say now. All right, so what do we in the plasma membrane? Yes, in the rough ER, and then we went from nucleus to the plasma membrane. Okay, I'm going to address this question one more time in common misunderstandings. All right, so debrief and summary, difference in gene expression, we talked about mutations, we talked about changes in genotype, we talked about a lot of genetic engineering techniques, one of my favorite things to do. All right, so we're back to this question. Now, because there's a mitochondria in this, I graded this question, it was in 2018, so it was one I'm familiar with. So when students saw this mitochondria, for some reason, there were a lot of this happening, and then to there, and then to there. So just because the picture's there, it doesn't mean it's part of it. I think they got confused on the ATP thing, but it said it binds. So a site on that channel actually was where ATP binds, but they got wrapped up in that mitochondria, okay? And then a lot of them said that it would, the lysosome would break it down also. Now, we've seen that it gets to the membrane and that it is just non-functional, okay? All right, I know this is busy. I had a million things to say at the last minute. I kept adding. So shout outs to kids and teachers that have talked to me over the past 2 weeks, I would say. Some of them I've had a couple conversations with Alexa, Sumi, Yuli, Sally, Kyle, Patrick, Marie, and Maya today. No, it was yesterday. All right, to-do list. You need to check your device and internet connection. You need to check your email and your phone number. Tell your teacher. Keep working on those student practice questions. Okay, keep practicing. There's that symbol. Read the 2020 AP testing guidelines. If you're on your own, your teacher will go over them with you, and we'll also go over them with you next week. But here is the email address to that testing guide, and here is where they practice question. There's the email address, the internet address, and here's the internet address for the exam demo that's working right now. You can start playing with that today, right now. If this is the first time you've heard this, next week we're going to have an exam practice week. I'm excited about this. It's going to be different than what we've usually been doing. So each day of the week we have something. Monday there'll be a time 45-minute window for 2 questions. I'm going to be here with you, but it's going to be like me being your test proctor. I'm going to give you the questions. You're going to write for that time period, and then you're going to save it to the next day. The next day we'll go over those FRQs. I'll tell you hints and tips about those particular FRQs that if there are any, and then we'll do I think there's a video on a demo video that we need to watch. And then on Wednesday you'll do the same thing, 2 new questions, and then you'll go over them on Thursday. And then Friday, Miss Kate and I will be together. We're going to give you some final test tips, and then we'll have a closing video to show you, and then send you off on your way. And then you're going to just do great if you've been following us on these videos and doing your practice questions. It's all going to be good. So that's all I have. I know I said a lot of things. I'm going 51. We'll probably more than that, about 3 minutes. Okay, I'll see you Monday. Bye.